So today we are going to revise the complete biochemistry. Okay, we are going to revise the complete biochemistry. So I believe that we can do this. Okay, let's take it as a challenge and start revising the complete biochemistry. Okay, so for some chapters I will write and show you, and for some chapters we will see from the notes. Okay, from the handwritten notes. Okay, and we have about 25 chapters. Okay, 25 units or chapters in which we have to discuss these things. Okay, so let's start from some interesting chapter. Let's start our discussion from the chapter which is called as your acid base balance. Okay, let's do this chapter first. So basically here we will talk about your blood buffers like your HCO3 minus, phosphate and proteins, which are the first line of buffer, and respiratory buffers like your ventilation and hemoglobin, which comprise of second line of buffers, and renal mechanisms like your H plus ion excretion, HCO3 minus reabsorption, acid excretion, and your NH4 plus excretion, which comprise of your third line of buffers. Okay, about disorders we'll see acidosis and alkalosis. Okay, just importantly remember, if the pH and the HCO3 minus change in same direction, we call it as metabolic acidosis, and if the pH and PCO2 change in opposite direction, we call it as respiratory acidosis or alkalosis. Okay, so SMOR, just remember SMOR. Okay, talking about acid base balance, pH is defined as the negative logarithm of H plus concentration. The body pH ranges from 7.35 to 7.45. we take the mean as 7.40. okay, and buffers are the substances which can resist the pH change. Suppose you put X gram of acid or alkali in the 1 liter of your buffer solution, and it changes the pH from changes the pH by 1 unit. Okay, so this X gram is called as the buffering capacity of the solution. Okay, what is alkali reserve? So alkali reserve is the HCO3 minus concentration in the body, that is about 24 millimole per liter. Range is about 22 to 28 or 22 to 26 millimole per liter. Okay, and this HCO3 minus overcomes the acid produced by our own body. Okay, this is called your alkali reserve. We have the Henderson-Hasselbalch equation, pH equal to pKa plus log of base by acid. pH equal to pKa plus log of base by acid. Okay, and for HCO3 minus buffer, okay, we have the equation as pH equal to pKa plus log of HCO3 minus by H2CO3. Okay, the acid is the carbonic acid H2CO3. Okay, and HCO3 minus is under metabolic regulation, while the H2CO3 is under respiratory regulation, putting the values of which we will get 7.4, which is the mean pH of the body. Okay, for the bicarbonate buffer, the HCO3 is to H2CO3 is about 20 is to 1. Okay, and the pKa value is 6.1. So it is the most potent extracellular buffer, most potent buffer in the extracellular fluid. Okay, it is the most potent buffer in the extracellular fluid. The reason is that it has increased alkali reserve and it is under your physiological control. Okay, and the phosphate buffers, they have the NaH2PO4 is to Na2HPO4 is to NaH2PO4, which is 40 is to 1. Okay, pK value 6.8. so it is the most potent intracellular buffer. Okay, and we have protein buffers comprising of the albumin and your hemoglobin. The pK value is 6.5. okay, after this so the most potent extracellular buffer was the HCO3 minus buffer, and intracellular buffer is the phosphate buffer due to the presence of 3 pK values. Okay, talking about hemoglobin, how it is doing the buffering action. Okay, so in tissues, in the tissues we have CO2 plus H2O combining, which in the presence of carbonic anhydrous form H2CO3 dissociates forming H plus and HCO3 minus. Okay, and this hemoglobin takes up the H plus, converting into HHB. Okay, converting to HHB. And in the lungs the opposite mechanisms occur. In the lungs the opposite mechanism occurs, that is your HHB. This combines with your O2, which is called Bohr's effect. Okay, it's forming HbO2. This HbO2 reacts with your H plus, and this HbO2, okay, this HbO2 plus H plus, okay, HbO2 plus H plus, and this H plus converts your HCO3 minus to your H2CO3, and in the presence of carbonic anhydrous is again converted into CO2 plus H2O. Okay, so this occurs in your lungs. After this, let's discuss the renal mechanisms. Your H plus excretion, HCO3 minus reabsorption, excretion of titratable acid, and your excretion of NH4 plus ions. Okay, so discussing about the renal mechanisms, first we take our blood, PCT and lumen. Okay, we take our blood, we take our PCT, and we take our lumen of this. Okay, in the PCT what happens, your CO2 plus H2O, this combines in the presence of carbonic anhydrous forming H2CO3. H2CO3 dissociates forming HCO3 minus plus H plus moves into the lumen, due to which a sodium moves into a PCT. Okay, and the sodium again goes to the blood, which will drag your HCO3 minus to the blood. Okay, so and this H plus comes here, so this H plus is excreted out. Okay, this is called as your H plus excretion. Okay, then how the HCO3 minus is reabsorbed. Okay, this HCO3 minus, okay, this HCO3 minus in your blood, this again combines into from your H plus forming a H2CO3. This H2CO3 again dissociates into CO2 plus H2O, which will replenish this reserve. Okay, understood? So this is called as your H plus excretion and your HCO3 minus reabsorption. And then you have the excretion of your titratable acid. Okay, excretion of your titratable acid. So let's take the blood. Okay, let's take the blood, then we'll take our PCT, not the PCT, the DCT or the collecting duct, okay, collecting tubule or the collecting duct, and the plasma. Okay, and in this what will happen is that in the DCT, CO2 plus H2O, the same thing, okay, H2CO3, and this H2CO3 dissociates HCO3 minus plus H plus. Okay, here in plasma we have a compound, this compound, we have this compound which is called as NaH2PO4. Okay, this NaH2PO4, this NaH2PO4 will give you sodium. Okay, this H plus will move here and the sodium will come into the DCT. Okay, and this H plus combines with your NaH2PO4. Okay, this combines here like this. Look at this. This combines with your this format. Okay, this NaH2PO4, it also changes into NaHPO4. Okay, this combines with NaHPO4 and forms again your NaH2PO4, and NaH2PO4 is a titrable acid which is excreted out. Okay, and the sodium as usual it comes into this, and HCO3 minus comes into your blood. Okay, so this is the mechanism of your excretion of your titrable acid. Okay, excretion of your titrable acid, and after that we will see another mechanism which is your an excretion of NH4 plus ions. So what happens, we have glutamine in the DCT, which in the presence of glutaminase changes into your glutamate and your ammonia. This ammonia comes into your plasma. Okay, and again the same mechanism, CO2 plus H2 in the presence of carbonic anhydrase changes to H2CO3. This H2CO3 dissociates into H plus HCO3 minus. HCO3 minus comes to the blood. Okay, and this H plus goes here, due to which the sodium comes into your blood. Okay, so and this NH3 and this H plus, this combine to form the NH4 plus. This NH4 plus is excreted out in the urine. Okay, understood? So this is excretion of your NH4 plus. Understood? So we discuss these mechanisms. Okay, we have an urinary anion gap which is equal to a sodium plus potassium minus your chloride. Okay, and then we'll discuss acid base disorders. If the pH is less than 3.5, this is acidosis. More than 7.5, this is alkalosis. Okay, HCO3 minus is normal, 22 to 30 millimole per liter, or 22 to 28 millimole per liter. Okay, if it is less than 22, metabolic acidosis. Okay, and if it is more than 30, it is called as a metabolic alkalosis. Okay, metabolic alkalosis. Understood? And PCO2, if the PCO2 is more than 45, we call it respiratory acidosis, and the PCO2 is less than 35, we call it as respiratory alkalosis. What is ABG analysis? So arterial blood gas analysis to evaluate the acid base status, monitoring the oxygen therapy, sleep disorder it is used, and detection quantification levels of abnormal hemoglobin also it is used. Okay, so it is the sample is collected by percutaneous needle puncture, where the radial artery, brachial artery, and femoral artery are more preferred arteries. Okay, and in infants also you can take from temporal artery. Anticoagulant is your heparin, lyophilized heparin is preferred. Okay, and the reference, these are the reference values given in Lippincott. pH is 7.35 to 7.45, we take the mean as 7.40. Then PCO2 is 35 to 45, mean is 40 mmHg. HCO3 minus 22 to 30, mean is 24 millimole per liter. PO2 is 67 to 104 mm of Hg. Okay, pH equal to pKa plus log of HCO3 minus by H2CO3. Okay, so let's say what is compensation. So if I try to explain compensation by this example. Okay, so this is a case, 2-year-old boy comes to neurology OPD, ABG analysis. So pH is 7.24 and PCO2 is 38. Okay, so what is happening, the pH is decreased. Okay, this pH is less, and the PCO2 which should be about 40, this is also less. Okay, so pH and PCO2 are in this these are changing in the same direction, and same direction we know SMOR. Okay, so same direction is called as metabolic, so this is called as a metabolic, and pH is decreased, so it is acidosis. Okay, we have the metabolic acidosis. Okay, we have the metabolic acidosis. And then how can you compensate it? What is the compensatory mechanism? Is that you have metabolic acidosis. We know the equation. Here, this pH equal to pKa plus log of HCO3 minus by H2CO3. We have metabolic acidosis. Okay, so as you have metabolic acidosis, this H2CO3, okay, this is under your respiratory control, and HCO3 minus is under your your metabolic control. Okay, so due to the metabolic acidosis, there is the decreased HCO3 minus. Okay, so to make this term constant, we have to decrease the H2CO3 also. Okay, we have to decrease the H2CO3, and this decrease of H2CO3 means your PCO2 must be decreased. Okay, PCO2 decrease means there must be hyperventilation. To decrease your PCO2. So this is the respiratory compensation. Okay, so for metabolic acidosis, we will see your hyperventilation, increased PCO2, and decreased H2CO3. Okay, for metabolic alkalosis, we will again see respiratory compensation, but this time hypoventilation. For respiratory acidosis, okay, where there is increased PCO2, okay, we will see your metabolic compensation. Okay, where the renal mechanisms will work, the renal conservation of HCO3 minus will occur. Okay, so as to increase the HCO3, the kidney will try to conserve it. Okay, for respiratory alkalosis, we will see again renal mechanism, where there will be renal excretion of HCO3 minus. Okay, for decreasing of HCO3 minus. Understood? This is the thing. So we have the causes now of metabolic acidosis. Metabolic acidosis, we can have high anion gap and low anion gap. Okay, high anion gap metabolic acidosis and low anion gap metabolic acidosis. Did I talk about your anion gap? Okay, let's talk. Anion gap is the indicator of unmeasured anions. Okay, so plasma anion gap according to United States Convention, it is sodium plus potassium minus your chlorine plus HCO3 minus. Okay, measured cations sodium potassium and your measured anions, that is your chlorine and HCO3 minus, or you can write unmeasured anions minus your unmeasured cations in the opposite form. Okay, unmeasured anions means protein, phosphate, sulfate and organic acids. These are unmeasured anions, and the reference interval is 12 plus minus 4 millimole per liter. What is the reference? 12 plus minus 4 millimole per liter. Understood? So after this, let's talk about metabolic acidosis. So if the anion gap is higher, so it called as high anion gap metabolic acidosis or your HAGMA. Okay, and if it is normal, it is called as a NAGMA. Okay, this NAGMA. Okay, what are the causes? High anion gap, we have Dilaro causes. Okay, we can remember from a mnemonic, that is Dilaro. Okay, what is Dilaro? Your diabetic ketoacidosis is one of the cause. Lactic acidosis. Okay, your aspirin drug, amino aciduria, your renal failure. Okay, where there is decreased excretion of sulfur, phosphorus and organic acid. Okay, and organic aciduria, this also causes your high anion gap metabolic acidosis. You can remember normal anion gap metabolic acidosis by the mnemonic DAR. Okay, so it is diarrhea, and the drug called as acetozolamide. Acetozolamide inhibits the carbonic anhydrase, which will decrease your H plus secretion. Okay, and which will impair your HCO3 minus reabsorption. Okay, then you have another cause, that is your renal tubular acidosis. For metabolic alkalosis, causes are you can remember voltage gated calcium channels. Okay, you can remember it. So we have the causes: voltage, he is vomiting, GI obstruction, Cushing syndrome, and your adrenal tumors. Okay. For respiratory acidosis, you can remember all the disorders, the disorders of respiratory tract: pneumonia, asthma, COPD. Okay, and using of narcotics and paralysis of your respiratory muscles also. This can cause your respiratory acidosis. For respiratory alkalosis, you can remember the mnemonic MASH, MASH. Okay, MASH. So MASH stand for your meningitis, septicemia. Okay, meningitis, septicemia, altitude, high altitude. Okay, and H for hyperventilation, hypoxia and hysteria. Okay, and the compensation is via renal mechanism. Okay, so here we complete our acid-based disorders. Okay, So let's move to the next topic. Let's discuss about another topic, which is called as nutrition. Okay, it is a short chapter, not a big chapter. Okay, so let's discuss nutrition. So we have protein energy malnutrition, which is of three types: your kwashiorkor, your marasmus, and your marasmic kwashiorkor. Okay, kwashiorkor is the protein deficiency, while marasmus is the caloric deficiency. Okay, and kwashiorkor occurs in 1.5-year-old child, while marasmus occurs 1 to 5-year-old child. Okay, not 1.5, and marasmus occurs in less than 1-year-old child. Okay, in infants. Okay, and in kwashiorkor there is edema due to protein deficiency. Okay, because the albumin levels, are less. Okay, and here the albumin levels are more. Okay, so there is edema in your kwashiorkor, but there is no edema in your marasmus. Okay, and there are sparse hairs and there is normal hair in your marasmus. You have moon-like face in kwashiorkor and old man type of face in your marasmus. Okay, like a moon-like face. Okay, and you have this old man-like face in your marasmus. Okay, and dermatitis will be there in your kwashiorkor. Your fatty liver will occur in your kwashiorkor, but there is no liver involvement in case of marasmus. Marasmic child will have poor appetite, while your kwashiorkor child will have the poor appetite, and the marasmic child will have voracious, will be the voracious feeder, because there is caloric deficiency. So, okay, so the child needs to eat more and more. Okay, so as to get the glucose. Okay, there is no muscle wasting in your kwashiorkor, but there is muscle wasting present in your marasmus. Okay, you can see the skinny child. Okay, and albumin is here 0.5 to 2 gram per deciliter in this, and in your marasmic child it is about more than 2 gram per deciliter. Understood? So what is the cause of edema? That if there is decreased albumin in the kwashiorkor, okay, the oncotic pressure will decrease. This will cause a retention of fluid in the interstitial space. Okay, this will cause a retention of fluid in the interstitial space, and this will cause your edema. Okay, this is the cause of edema. How can you assess the PEM disorders? Okay, one is the weight by height ratio. You can measure the weight by height ratio, or you can measure the mid upper arm circumference. Okay, this was asked in one of the Olympiads, ESB Olympiad. Okay, and let's talk about your BMR, basal metabolic rate. It is the amount of energy required by an individual in awake state at complete physical and mental rest in post-absorptive phase to maintain the vital activities. This is called as your BMR, measured by direct method or indirect method, directly by heat evolved and indirectly by your O2 consumption per unit time. This is done by Benedict-Roth apparatus. Let's talk about the Benedict-Roth apparatus. This is the Benedict-Roth apparatus. Okay, So here the patient will breathe oxygen from a 6-liter old gas 6-liter gas reservoir for 6 minutes. Okay, the patient breathes oxygen 6-liter gas reservoir 6 minutes. Okay, and the patient will release the CO2 which is absorbed by the soda lime present over here. Okay, it will absorb by the absorber containing soda lime, and the amount of O2 used, okay, amount of O2 will be recorded. Okay, the O2 will be recorded. Like in 6 minutes, if Y liters of O2 are consumed, okay, the heat generated will be calculated by a 4.8 Y. Okay, 4.8 Y, where 4.8 is the calorific value of 1 liter of oxygen. Okay, calorific value of 1 liter of oxygen. So heat generated can be calculated by 4.8 Y kilocalories. Okay, the normal BMR values in basal metabolic rate in males is your 33 to 38 kilocalorie per meter square per hour, while in females it is your 32 to 35 kilocalorie per meter square per hour. Understood? So these are your BMRs. It is the amount of energy required by an individual in awake state at complete physical and mental rest in post-absorptive phase to maintain its vital activities like digestion, absorption, respiration and all. Okay. What are the factors which affect the BMR? So genetics affect the BMR. Okay, the cardiac output affects the BMR. Increased cardiac output will increase your BMR. Surface area will affect your BMR. Okay, it is directly proportional to surface area. Age will affect. Age increase age, then the BMR will decrease. If the age increases, the BMR decrease. Okay, 2% per decade of life. Your muscle mass, increase BMR, as there will be increased metabolism. So males have greater BMR than the females. Okay, and climate, cold climate, there will be increased BMR, and BMR decreases during sleep. It decreases during starvation. Okay, the thyroxine will increase the BMR, and in fever, 1 degree change in temperature will increase the BMR of the body by 10 to 12%. Let's talk about the specific dynamic action of food, SDA. This is called as the thermogenic effect of food or the calorigenic effect of food. Okay, so proteins have specific dynamic action of 30, lipid has about 15, and carbs have about 5. Okay, so it is the amount of energy expenditure above the BMR, so as to process the food for its use and storage. Okay, it is above the BMR so as to utilize the thing for any other purposes. Like proteins have more SDA to meet the energy requirements for deamination, urea synthesis, and your protein biosynthesis. This extra amount of energy for lipids, 15%, it is used in your storage, mobilization and oxidation of your fats, and this in carbs it is used in your glucose to glycogen conversion. Okay, and for mixed diet it is about 10% of your total calories. Let's talk about the calorific value of food. So it is the amount of heat required to raise the temperature of 1 kg of water by 1 degree Celsius. Amount of energy required to raise the temperature of 1 kg water by 1 degree of Celsius is measured by your bomb calorimeter. Calorific value for carbohydrate is 4, lipids is 9, and protein is 4. Understood? So these three are your calorific values. Let's talk about the RQ quotient. RQ quotient is the volume of CO2 produced by volume of O2 use. Volume of CO2 produced by volume of O2 use. For carbohydrate it is 1, for fat it is 0.7, for proteins it is 0.8, for mixed diet it is about 0.825 or 0.85. Okay, then about nitrogen balance, there are positive and negative. So positive nitrogen balance, there is more protein intake as compared to your output, and in negative there is more output as compared to your input of nitrogen. Okay, positive balance is seen in your pregnant women, growing children, and steroid therapy. Okay, and negative balance is seen in fever, infection, malnutrition and all. So talking about balanced diet, okay, so a man requires about this 2600 kilocalories, okay, the calories per day, okay, and protein about 60. Okay, the female woman will require protein about 55, less than the male. Okay, about fats you can sit 30. Okay, if the man is doing moderate work, assuming it. Okay, and in the females it is about 5 less, 25. Calcium is about 600, but women in a pregnant woman, lactating woman, you require more and more your calcium. More requirement of calcium is there. Okay, more requirement of calcium is there. Okay, well, it's gone. Okay, yeah. Then the iron, man will require 17 milligram per day of iron, but the woman will require 21 milligram per day of iron because there is menstrual iron loss, so as to compensate it. Okay, then in the ICMR food pyramid, you can see the ICMR food pyramid here. Okay, you should eat the fresh vegetables and all more, but if I have sweets less. Okay, you can see the ICMR pyramid here. Afterwards, let's talk about dietary advice for optimal health in pregnancy. What will advise in pregnancy? Okay, so you should take 350 more calories, okay, and about 30 gram per day fat, okay, and calcium must be taken more, like 1200 milligram per day. Iron must be taken more, like 35 milligram per day, okay. And the patient should eat vitamin rich food, green leafy vegetables, legumes, okay, and 15 minutes sunlight exposure is important. No alcohol, no tobacco. Iron and folic acid supplementation must be given, like iron 100 milligram per day, folic acid 500 milligram per day. Okay, this much supplementation is required. Folic acid is required for the anemia and neural tube defect prevention. Okay, and vitamins and minerals must be taken adequately. Dietary advice for optimal health in your diabetes mellitus. Okay, so fresh fruits and vegetables are recommended. No fruit juice. your skimmed milk and your whole milk, you should avoid the whole milk, and you should take skimmed milk. There should be no red meat intake. Okay, you should eat the nuts, seeds, oil, oat and sprout. Okay, regular plasma glucose must be checked, lipid profile must be checked for triglyceride, cholesterol, HDL, LDL, VLDL. Okay, and the patient should take high fiber diet. Okay, and increased omega-3 fatty acid consumption that must be there. Okay, and the patient should do weight loss and exercise for maintaining his health. Okay, and taking care of the feet and eyes, because there is the infection in the feet. Okay, the diabetic foot syndrome is there. Okay, and eyes are also damaged, cataract formation is there. Okay, in diabetes mellitus, snowflake cataract we will see in your carbohydrate metabolism and all. Okay, the patient should manage his stress, and regular dental checkup is mandatory, and the patient will take sugar free. Okay, sugar free tablets. Okay, dietary advice for coronary artery disease patients. Okay, fresh fruits and vegetables are compulsory. Then the patient should take whole grains, which are excellent source of fibers, and this fibers will reduce the absorption of cholesterol from intestine. Okay, reducing the blood cholesterol level. Okay, then fat alpha linolenic acid is important for reverse transport of cholesterol by the HDL. Okay, so alpha linolenic acids must be taken. Then the patient should eat decreased saturated fat like coconut oil, butter oil and palm oil. These oils must be avoided, these are not good. Okay, and the patient must not take trans fatty acids. Okay, this trans fatty acids, this increases his atherosclerosis risk. Okay, there must no smoking, no tobacco, no alcohol. Okay, the patient should can take healthy proteins like egg white, soya bean, peas, lentils and fish. Okay, and the patient should avoid your sodium food like your potato, or pickles, meat and canned juice, and your like that cup noodles and all. Okay, the patient should avoid this. Lipid profile must be checked at regular intervals, healthy weight management, and the patient should do yoga and meditation. Okay, so this completes our chapter, that is nutrition. Let's move on to the next chapter. Let's discuss another small chapter, that is cancer. Okay, so cancer is the second most common cause of your death worldwide. It is the first one is your cardiovascular disorders. Okay, oncology is the study of cancer. Talking about neoplasm, these are abnormally new growth of tissues. Okay, which can be benign or malignant. Benign don't spread, but malignant types of spread. But the properties of cancer cells is that they proliferate rapidly. They have diminished growth control. Okay, the growth is not controlled. Okay, cell cycles are disturbed. Okay, mutations are more. Okay, and there is the loss of contact inhibition. Contact inhibition, contact inhibition, if there is a cell, if it is dividing. Okay, if there are like cells around the dividing cell, okay, then it stops dividing. But in cancer cell, this property is lost. Okay, and they invade the local tissue, which is called a metastatic property. Okay, they are self-sufficient in growth signals, so they can evade your apoptosis. Okay, because your survival signals are they provide the survival signal themselves. Okay, they stimulate local angiogenesis. Okay, increase the vasculinity. Okay, so they have self-sustaining proliferating growth signals, evasion of growth suppressors. They do metastasis, limitless replicative potential. Okay, they have resistance to apoptosis, and they stimulate your angiogenesis. Okay, what biochemical changes you can see in your cancer cells? So they show mutations, abnormal cell cycle, chromosomal abnormalities. Okay, and inactivation of TSGs and activation of oncogenes. They have epigenetic changes, alteration in gene transcription. Okay, and they have increased telomerase activity. This is important, increased telomerase activity. They show the reappearance of the fetal antigens, which are carcinogenic. Okay, so carcinofetal antigens, which are used as the tumor markers. Okay, appearance of abnormal sugar chains are there. Decreased adhesions. Okay, increased property of metastasis. They release exosomes, they release biomarkers. Okay, they release angiogenic factors, they release enzymes which act on the ECM, and they alter the metabolism, causing aerobic glycolysis. They cause aerobic glycolysis, which is called as a Warburg effect. Okay, so carcinogenesis, okay, it is of clonal origin. Single abnormal cell will give multiple cells. Okay, and the tissue microenvironment significantly affects the tumor formation. And what are the causes of a genetic damage? So there are acquired mutation and inherited mutations. Okay, acquired mutation may be replication mutation, your environmental mutation, and the hereditary mutation, R, E, and H. Okay, and your inherited mutations may be due to familial conditions. Okay, radiations means your UV rays, X rays, and gamma rays, which will induce the production of your ROS, reactive oxygen species. Okay, and this UV rays, these rays will form the pyrimidine dimers. Okay, create apyridinic or apyrimidinic sites. Okay, and will form double or single strand breaks or cross linking of your DNA strands. Okay, so these are the problems. And UV radiations may cause skin cancers. Okay, and we have chemical carcinogens which covalently modify the DNA and form a variety of nucleotide adducts. Okay, like your chemical carcinogen, polycyclic aromatic hydrocarbons like your paraminobenzene, okay, benzopyrene, and your dimethyl benzanthracene. Okay, benzopyrene, dimethyl benzanthracene. We have aromatic amines like your 2-acetyl aminofluorine and N5 N-methyl N-amino azobenzene, MAB. Okay, nitrosamines like your dimethyl nitrosamine and diethyl nitrosamine. Okay, and then you have various drugs. various drugs like a cyclophosphamide and diethylstilbestrol, cyclophosphamide and diethylstilbestrol, and your naturally occurring compounds like your aflatoxin B1. Okay, this another drugs are there like methachloramine and beta propiolactone, which interact directly with your DNA. Okay, and the carcinogens are generally electrophiles, which will attack on the nucleophilic sites in the DNA and affect them. Okay, azodie causes bladder cancer, nitroso compounds cause gastric cancer, and asbestos and nickel cause lung carcinoma. Okay, so chemical carcinogens work by the initiation and promotion methods. Okay, Ames assay is used to test the ability of chemical carcinogens to induce the mutations. Okay, where Salmonella typhimurium bacterium is used. Okay, so first you have to add bacterium which requires a histidine for the growth. Okay, in the agar medium. Okay, then add the suspected mutagen, then incubate it. Okay, if there is if chemical is a mutagen, many colonies of his positive will be formed. If the chemical is not a mutagen, then fewer colonies will form. Okay, then talking about oncogenic viruses, these are dysregulating the cell cycle. This will inhibit the apoptosis and will cause abnormal cell signaling pathways. Okay, so abnormality in cell signaling pathways. Let's talk about some oncogenic viruses like Epstein-Barr virus, which will cause your Burkitt's lymphoma, nasopharyngeal cancer, and B-cell lymphoma. Hepatitis B virus will cause hepatocellular carcinoma. C virus again hepatocellular carcinoma. Human herpes virus 8 HHV8 will cause Kaposi sarcoma. Okay, so human herpes virus 8 HHV8 will cause Kaposi sarcoma, and human papilloma viruses like type 16 and 18 will cause cervical cancer, and human T-cell leukemia virus will cause adult T-cell leukemia. Okay, about the genes, oncogenes are altered genes which will stimulate your cell growth. The proto-oncogenes are normal genes which are stimulated and form oncogenes. What are the stimulating factors? Mutation, promoter insertion, and enhancer insertion. This can stimulate as it occurs in the cause of Rose sarcoma virus. And chromosomal translocation, as in your Burkitt's lymphoma and Philadelphia chromosome, this will stimulate proto-oncogene, forming an oncogene. Okay, and gene amplification also can stimulate this process. Okay, Philadelphia chromosome is between your chromosome number 9 and chromosome number 22, where the chromosomes break. Okay, leading to your BCR and ABL gene together, which will lead to a cancerous condition. And your Burkitt's lymphoma, in the Burkitt's lymphoma, there is a reciprocal translocation of your chromosome number 8 and 14. Okay, 922 is your Philadelphia chromosome and 814 is your Burkitt's lymphoma, where the Ig gene and your MYC oncogene, Ig gene and MYC oncogene, this stayed together causing your cancer. Okay, afterwards let's discuss tumor suppressor genes like your P53, RB gene, WT gene, FAP gene, and BRCA gene. These are tumor suppressor genes. Okay, which suppress your cell growth or division. Okay, and inactivation of tumor suppressor genes will lead to your cancer. So TSG has a dual function. They have gatekeeper function also, which will control the cell proliferation, and they have caretaker function. Okay, which will preserve the integrity of your genome. Okay, and let's discuss the difference about oncogenes and tumor suppressor. Okay, in oncogenes, if there is mutation of only one allele, it is sufficient to produce cancer, but mutation in both alleles are required to produce your oncogenic effects in TSGs. Okay, oncogenic effect is to gain a function, because oncogenes when stimulated cause cancer, and tumor suppressor gene when they are inhibited cause cancer. Okay, and the mutations in oncogenes arise from the somatic cells, but your mutations in your tumor suppressor genes may be present in your somatic or your germ cell. Okay, and your oncogenes usually don't show tissue preference. Okay, with respect to the type of cancer, but this TSG shows strong tissue preference. Okay, example, your RB gene mutation. Okay, this will cause a retinoblastoma in your retina only. Okay, so P53 is also called as a guardian of the genome, and which when activated cause the cell cycle to arrest apoptosis, senescence, and your DNA repair mechanisms to be activated. Okay, so it is called as a guardian of the genome. These are some important facts. You can just read this. Carcinogen is a multi-step process. Okay, and mechanism. What is the mechanism? P53. It is a phosphoprotein. And it is also called as a molecular policeman, or your gatekeeper gene, or your guardian of the genome. Understood? So if there is mutation in P53, okay, so there will be P53 induced transcription, which will lead to the production of P21 protein. This P21 protein will inhibit the CDK, inhibiting the cell division, or this P21 protein will inhibit your PCNA, proliferating cell nuclear antigen. Okay, which will inhibit your replication. Okay, inhibit CDK or inhibit PCNA. What is the mechanism of retinoblastoma gene? Is that retinoblastoma gene is unphosphorylated, and the eukaryotic transcription factor 2F is bound to it. Okay, as we know that it is bound to your tightly to your RNA polymerases. Okay, so if there is phosphorylation, the ETF2F will be free, and this will initiate your transcription and cell division will occur. Okay, so phosphorylation will stimulate your cell division growth. Okay, understood? After that, we have oncomyRS. Okay, onco-miRNAs. Okay, these are oncogenic miRNAs like your miRNA MIR 1792, MIR 21, MIR 155, MIR 15, and MIR 16. Okay, MIR 16 and 15, this lead to chronic lymphocytic leukemia. Understood? After that, it is a fact that 5 to 15% cancers have hereditary etiology. Okay, these are hereditary etiology. Okay, and cancer cells also exhibit genomic instability. They have microsatellite instability, and they can have CIN instability, which may be 2 types, like aneuploidy and copy number variation. Okay. And how apoptosis is related to cancer is that the apoptosis is in the cancer cells. They are evading the apoptosis, okay, by generating the intrinsic growth signals themselves, okay, the survival signals themselves. Understood. And another fact is the tumor microenvironment plays a critical role in carcinogenesis, like T cells and macrophages. Okay, this activates the necrosis factor KB. Okay, it's promote the tumor growth. Okay, the mesenchymal cells also form cancer stem cell nicks, which will promote your cancer, which will promote your tumor growth. These adipocytes, these have growth factors which will stimulate your tumor growth. So presence of these cells in the nearby, in the periphery, will cause your cancers. Okay, Warburg effect is that cancer cells take up the glucose and metabolize it to lactic acid. Take up glucose, metabolize it to lactic acid in the presence of O2. Note that it is in the presence of O2. Okay, so there is a switch from aerobic to anaerobic glucose metabolism, which is the driver of tumorigenesis. Okay, this is called as the Warburg effect. Understood? Let's discuss the tumor markers again. Tumor markers are the substances produced in tumor cells in response to tumor tissues and detected in the body fluids. Okay, these are specific for a cancer. These are sensitive, and these have increased concentration in normal individual. And this should tell about a staging of cancer. Which stage cancer is that? It should tell it. Okay, the uses are in a screening, diagnosis. Okay, your staging of cancer, prognosis, monitoring, and a follow-up of cancer. Okay. So we have 3 types. We have your oncofecal antigen, okay, or your carcinofecal antigen. Okay, then we have carbohydrate antigens and hormones. Okay, and we have enzymes markers too. We have enzyme markers too, and some other markers also. Let's discuss the oncofecal antigen. Are of 2 types. Your AFP, okay, your AFP, which will show your hepatocellular carcinoma or your germ cell tumors. Okay, then your carcinoembryonic antigen, which will show colorectal cancer, breast cancer, and your lung cancer. Okay, and your carbohydrate antigen, which are of 3 types. CA 15.3, which will show your breast cancer, CA 19.9 pancreatic cancer, CA 125 your ovarian cancer. Okay, so you can remember BPO and this numbers 15.3, 19.9, and 125. Hormones, beta HCG will show your choriocarcinoma, okay, the carcinoma of the chorion, and the calcitonin of thyroid carcinoma, gastrin of gastrinoma, okay, insulin of insulinoma, ACTH of ectopic lung cancer, serotonin of carcinoid tumors, catecholamines of pheochromocytoma. Okay, we have the enzyme markers alkaline phosphatase, which will indicate liver and bone metastasis, neuron specific enolase, which will indicate your neuroblastoma, prostatic acid phosphatase indicates your prostate cancer, and lactate dehydrogenase, which will indicate your testicular and germ cell cancer. And others like a PSA, prostate specific antigen, okay, HER2, which will indicate the breast cancer, and ER and PR also there, which will also indicate the breast cancer. Okay, and monoclonal antibodies and your Benz-Jones proteins, this will indicate your multiple myeloma. So here we complete our cancer topics. Let's move to the next topic. So let's discuss the protein targeting and sorting chapter. This is a very small chapter, okay, but it is like important, okay, not important for your this prof point of view, but let's discuss it at it is in your syllabus. So we are discussing each and every chapter which are there in a syllabus. Okay, so the proteins can be directed from the cytosol to mitochondria, your nucleus, and the endoplasmic reticulum. Okay, the proteins can be directed from this sites, okay, directed to this sites. And protein targeting was major contribution was by the scientist called Blobel. Okay, and the proteins contain a in their amino terminus, the protein contains a sequence which is called your signal sequence or your coding sequence, okay, which targets them appropriately to the designated location. Okay, and this delivery process is based on the information contained in the protein itself. Okay, understood? So what is the point? That this is the protein, this is a signal sequence. Okay, so you have translocation channels. Translocation channels will cleave your signal peptide. This translocation channels will have signal peptidases attached to them, okay, which will cut your the signal sequence and will give your protein inside the cell. Okay, and the signal sequences are again regenerated. Okay, they're again recycled. Okay, this is called as a signal hypothesis. Okay, so what are the signal sequence? We have N-terminal signal peptide. In case of your endoplasmic reticulum, we have carboxy terminal KDEL sequence, which is organelle targeted is a lumen of ER. Okay, KDEL sequence, which have lysine, asparagine, glutamine, and leucine. Okay, lyse, asp, blue, leu. Okay, and we have diacetyl sequences containing asparagine, X, and glutamine. Okay, for diacetyl sequences for Golgi membrane. Okay, we have amino terminal sequence for mitochondria matrix. Okay, we have NLS, the NLS for your nucleus. Okay, and we have PTS, peroxisomal targeting sequence for your peroxisomes. Okay, for your peroxisome. Understood? And then we have mannose 6 phosphate for your lysosome. Understood? So the signal sequence it is removed when the protein reaches its final destination. So what are the mechanisms of a protein sorting? Okay, so first mechanism is that protein is synthesized in cytoplasm and then delivered to its intracellular location post translationally. It is delivered post translationally after translation. Okay, and the proteins for nucleus, chloroplast, mitochondria, and peroxisomes are delivered by this process. Okay, then 30% proteins go by another mechanism which is called a secretory pathway. Okay, this directs proteins into an ER, endoplasmic reticulum, co-translationally when translation is occurring. Okay, so these proteins are there. Okay, talking about cytosolic protein targeting, protein synthesis in the cytosol are targeted to your mitochondria, your nuclei, and the peroxisome. Okay, this is called a post-translation translocation. Okay, in case of your mitochondrial protein, this mitochondrial protein have at least two signal sequences. These have at least two signal sequences on their N-terminal ends. Okay, this mitochondrial protein must be unfolded before the transport. Okay. Talking about the nuclear proteins are RNA, DNA polymerases, histones, topoisomerases, etc. Okay, these proteins are targeted to the nucleus and have targeted to the nucleus. They have a specific sequence which is called as nuclear localization sequence (NLS). Nuclear localization sequence (NLS). Okay, this NLS is made up of 4 to 8 amino acids. Okay, this NLS is made up of 4 to 8 amino acids. Okay, and plus consecutive basic residues. Okay, consecutive basic residues of arginine and lysine. These are your arginine and lysine residues here. Arginine and lysine residues in addition to your 4 to 8 amino acids. Okay. Talking about a peroxisomal protein, the signal sequence is present at the carboxyl terminus. Okay, it has a signal sequence containing 3 amino acids, your serine, lysine, and leucine. Okay, you have your serine, lysine, and leucine. Okay, it contains of 3 amino acids only. Okay, so how a peroxisomal protein enters? Okay, this peroxisomal protein enters by a PEX protein complex. Okay, so this is a peroxisomal protein containing the PTS, the peroxisomal targeting sequence. Okay, this PTS binds to a PEX5 protein. Okay, and this PEX5 again, this by binding this PEX5, okay, it will be ready to bind with the PEX14 protein, which is in your intrinsic membrane protein. Okay, and by this if it binds to the PEX14, then it can be taken in by your PEX2 10 12 complex, which is also your intrinsic membrane protein. Okay, this is taken in by your PEX2 10 12 complex. Okay, and then your signal sequence and the your PEX5 is now recycled. Okay, understood. So this is there. Okay, for your endoplasmic reticulum, we said that the peptide chains are transported co-translationally. Okay, and the translocation is the steps like this. Okay, for the ER protein at the N terminus, okay, we can see about 9 to 12 amino acid residues, okay, which comprises of its signal sequence. It comprises of 9 to 12 amino acid residues. Okay, understood. So what is the mechanism in which the proteins targeted for the ER goes into it? Okay, protein targeted for ER goes into it. So first what happens is the SRP. Okay, we have this protein. Okay, and the 9 to 10 amino acids attached to this. Okay, the signal peptide. So SRP, there is a signal recognition particle. Okay, this will recognize your sequence. Okay, and the SRP will bind your ribosome and the signaling sequence. Okay, it will bind it. Okay, and by this binding, okay, this binding of the ribosome as well as the SRP, okay, the protein synthesis is halted, and this whole complex, it goes to your ER membrane. It goes inside your ER membrane. Okay, after it goes inside the ER membrane, the SRP ribosome complex, there is an SRP receptor in the ER membrane, okay, which will recognize your SRP, the signal recognition particle, okay, and the ribosome attached to it. This SRP receptor, okay, okay, will recognize and will transfer it to the translocon channel. Okay, translocon channel, which is a protein conducting channel. This translocon channel has a signal peptide. The signal peptide will cut your signal. Signal peptidase will cut your signal peptide and then transmit the protein inside. Okay, and it will release your SRP, signal recognition peptide, which is recycled. The protein comes in. Okay, protein is folded by your chaperones. Okay, and modified by N-linked carbohydrate addition. Okay, and there is sorting and transport now. Understood. And then the role of what is the role of Golgi apparatus? So Golgi apparatus is used in the post-translation glycosylation of the proteins. Okay, it is helping in the sorting of proteins. Okay, so Golgi apparatus is two types. Two function: post-translational glycosylation. Okay, post-translational glycosylation, and another function is the sorting of proteins. So this first function is done by all parts of the Golgi, but sorting is done by the trans-Golgi network specifically. Okay, and it forms the transport vesicles from the ER, okay, which will carry the protein to the Golgi. Okay, and the Golgi, the vesicles fuse and deposit the cargo inside the Golgi. This cargo is your that protein which it's carrying. Okay, and the cargo proteins are further modified by attachment of your carbohydrates. Okay, from the Golgi, the transport vesicles arise. Okay, this will carry the protein to their final destinations. Okay, understood. Now let's discuss the disorders associated with protein targeting and sorting. This is important. Okay, so we have many disorders like a protein targeting disorders, defect in the mitochondrial target sequence, MTS, defect in the mitochondrial target sequence. We have primary hypoxaluria type 1. Okay, so in this disorder what happens, your AGT deficiency is caused. AGT is a glyoxylate aminotransferase 1. This is deficient, and this is caused by its mistargeting to the mitochondria. This AGT is mistargeted to the mitochondria, but it had to be targeted. This AGT had to be targeted to the peroxisome, but it is mistargeted. Okay, due to which the glyoxylate is now not converted into glycine, because the AGT converts the glyoxylate to glycine. It is now not converted. This glyoxylate will oxidize, okay, to oxalate, and this oxalate will deposit as calcium oxalate in kidney, and it will lead to kidney stones and kidney failure. Okay, then we have pyruvate dehydrogenase deficiency, PDS deficiency. Okay, this is also MTS defect. The mitochondrial target sequence is defect, due to which there is a mutation in your PDS gene. Okay, that's PDS deficiency, and the most common cause is the primary lactic acidosis in children, which will cause your pyruvate dehydrogenase deficiency. Understood? Then we'll discuss about the disorders due to peroxisomal abnormality. So we have 5 disorders, okay, but we'll see here 3 disorders which are important. So we have a Zellweger syndrome, we have neonatal adrenoleukodystrophy, we have infantile Repsum disease, okay, we have hyperpipecolic acidemia, and rhizomelic chondrodysplasia punctata. Okay, so first let's discuss the your Zellweger syndrome. Okay, so Zellweger syndrome is due to the defective oxidation of VLCFA, very long chain fatty acid metabolism is altered. Okay, because there is here the correct address is not printed on the protein packet. Okay, so it cannot be delivered to your correct locality. Okay, peroxisomal enzymes are produced. Okay, these enzymes are produced, but their entry is now the address is now wrong. Okay, it won't enter into a peroxisome. Okay, so there is insufficient oxidation of VLCFA, because VLCFA are oxidized in your peroxisome only. Okay, this accumulation of VLCFA and CNS will cause neurological impairment and death. Okay, let's discuss the infantile Repsum disease. It is the phytanic acid oxidase deficiency, which will impair your alpha oxidation. Alpha oxidation won't occur. Okay, alpha oxidation won't occur. This is also a protein targeting error. Okay, because the wrong address is printed. Okay, the patient will present with polyneuropathy, retinitis pigmentosa, nerve deafness, and cerebellar ataxia, all the neurological symptoms. Okay, and how you can treat it? You can tell the patient to avoid milk, because milk is source of a phytanic acid. Okay, as the phytanic acid oxidation, that the phytanic acid is not oxidized. Okay, so that's accumulation of phytanic acid. So to reduce the phytanic acid, okay, so you have to restrict the dietary intake of your phytanic acid. Understood? Let's discuss the neonatal leukodystrophy, neonatal adrenoleukodystrophy. Okay, this is also due to a deficient oxidation of very low chain fatty acids, okay, via peroxisomal enzyme. Okay, here the VLCFAs are accumulated and myelin sheath are destroyed by the VLCFAs. Okay, and this is also a X-linked condition. Okay, so this completes our chapter, that is your protein targeting and sorting. And now let's move on to next chapter. So another short chapter, which is your extracellular matrix, okay, or your ground substances. So this is a gel-like matrix, and ECF has structural part, your specialized part, glycoproteins, and your proteoglycans present. The structural part has 3: collagen, elastin, and fibrillin, which you have to read. The specialized glycoproteins are fibronectin and laminin. This fibronectin has the function of cell adhesion and cell migration, and the laminin is present in the basal lamina of glomerular membrane. Okay, and this is also helping in your this is also specialized for some functions. Okay, and proteoglycans, which are combination of proteins and your GAGs. Okay, these are your beta glycans, perlecan, versican, decorin, fibromodulin. Okay, these are your beta glycans, your perlecan, versican, your fibromodulin, and your decorin. Okay, so talking about collagen, let's read about collagen. Collagen is the most abundant protein. It has a triple helical structure. It is comprised of a XY repeats. Okay, so non-collagen collagens are some there, like your C1q, your surfactant SPA and SPD. These are your non-collagen collagens. Okay, non-collagenic collagens or non-collagen collagens. So what is the method of synthesis? We have the pre-pro collagen. Okay, where the signal peptide is cleaved, it will form a pro collagen. This pro collagen, there will be hydroxylation, glycosylation, SH bond formation, and triple helix formation. Hydroxylation, glycosylation, SH bond formation, and your triple helix formation, you can remember HDST, okay, and which will form a trophocollagen. This, that will be in the trophocollagen, there will be removal of NMC terminal polypropeptides, okay, by peptidase, okay, and these are assembled into a fibrillar structure forming a collagen, and collagen is stabilized by the cross linking by the lysyl oxidase, which is a copper dependent enzyme. There is a clinical that a lysyl oxidase is inhibited by the toxin in the sweet pea, okay, inhibited by the toxin in the sweet pea Lathyrus odoratus, okay, and this will cause a lethyrism, but there will be skeletal as well as vascular problems due to your decreased cross-linking of your collagens, okay. And proline and hydroxyproline are present in your collagens, which provide your rigidity to your collagen, okay. And this hydroxylation of proline and hydroxyproline requires a vitamin C, and deficiency of vitamin C causes your scurvy, okay. So in scurvy there is decreased hydroxylation of proline and your hydroxyproline, okay. And this proline and proline, sorry, this is found at Y position of GlyXY sequence. GlyXY repeats I told, so in Y position your proline and hydroxyproline are present, okay. So this is the whole procedure, how it is done, okay, how it is done. This is given in your Lippincott textbook, okay, understood. So there is a self-assembly of trophocollagen which will form the fibrils, and the cross-linking of fibrils will form your mature collagen fibers. Structure of collagen. Collagen is a fibrous protein, you know. There is a triple helical structure containing 3 alpha chains. There is 3 alpha chains in your collagen, okay, and glycine is present at every third position of the triple helix. This glycine is the smallest amino acid, so it induces some turns, okay, bends, okay, it can be accommodated in the smaller space. Okay, so if GlyXY repeats in where X is the proline and Y is the hydroxyproline most commonly, okay, but in Y it can be hydroxylysine in place of your hydroxyproline also, okay, and this proline and hydroxyproline are providing your rigidity to your collagen molecule, okay, and they are providing rigidity to collagen molecule. So first what happens, there are GlyXY repeats are there, okay, then these are linked like this, forming an alpha chain. This alpha chain will form a triple helical structure, okay, having the diameter of 1.4 nanometer, and this triple helix will form triple helix molecule of 300 nanometer, and this will form a fibrils, and the fibrils are then cross-linked forming the mature collagen, okay. So what are the fibril forming collagen? The type 1 and type 2, these are the fibril forming collagen. This form your fibrils, okay. What are multiplexins? Multiplexin are collagens with a multiple triple helix domain and interruptions, okay. These have multiple triple helix domains, okay, like a type XB collagen and a type 18 collagen. 15 and 18 are called as your multiplexins, okay. 15 and 18 are called as your multiplexins, okay, understood. So this is the general sequence. This is the structure of your basic collagen, okay, just see. Then let's discuss the type of collagen, okay. So we have about 28 types of collagen as per Harper, or more than 25 types as written in Lippincott, okay. So we have type 1, type 2, type 3 and type 4 collagens present, okay. So type 1 collagen is present in your connective tissue, bones and tendons. Mutation of type 1 collagen will cause your osteogenesis imperfecta, where you will see your brittle bones are there, blue sclera will be there, and you will also see your deafness, okay. Talking more about your osteogenesis imperfecta, just look at this radiograph. There is the bowing of your hand, of your bones, okay, brittle bone disease it is called. In 80% of cases, it is the mutation of alpha 1 or alpha 2 of type 1 collagen only, okay. So you have type 1 osteogenesis imperfecta and type 2. Okay, type 1 is the most common type, and there is bone fragility, hearing loss and blue sclera as I talked earlier. Type 2 is the lethal part, okay, but there are pulmonary complications also, okay. And there is a dentiogenesis imperfecta which generally accompanies your OI, where there is the tooth development is disordered, okay. Then talking about another part, which is your type 2 collagen. This is present in your cartilage and vitreous humor, and also in your IV disc, okay. Just remember cartilage and vitreous humor. Type 3 collagen is present in your skin, lung and vessels. Type 4 collagen is present in your basement membrane, okay. Mutation of type 4 collagen will lead to your Alport syndrome or your hereditary nephritis, okay. So these syndromes I am talking about are all collagenopathies. Collagenopathies means collagen forming disorders, okay. So this Alport syndrome is due to type 4 collagen mutation. This is called as a hereditary nephritis, okay. So there are ocular lesions are there. Your hearing loss will be there. Hematuria will be there. Why hematuria? Because the basement membranes are ruptured, leading to your bleeding, okay, leading to your hemoglobin that comes in the urine. Not hematocrit specifically, which comes in the urine, okay. And then you have renal damage, because the renal basement membranes are also damaged, okay. And we have fibril forming collagen. I said 1 and 2, just remember 1 and 2, 1, 2 and 3 you can remember, okay. Network forming collagens are 4, 8 and 10. This type 8 collagen is present in your corneal and vascular endothelium. So if there is deficiency of type 8 collagen, it will cause your ocular lesions as well as your endothelial ruptures. Okay, we have beaded filaments, type 6 collagen, anchoring fibril, type 7 collagen. Your transmembrane domain is 13 and 17. 13 and 17 form your transmembrane domain forming collagens. Okay, fibril associated collagens are 9 and 12. Fibril associated collagens with interrupted triple helix. Okay, these are called your FACIT, FACIT. Okay, fibril associated collagen with interrupted triple helix. Okay, and the collagen genes have the prefix col. Okay, collagen genes have the prefix col. You can get a difference between collagen and elastin. So talking about this, collagen has many genetic types, while the elastin has only one genetic type. Okay, and collagen is of triple helical structure. Elastin has no triple helix. Random coiling is there. Gly XY and repeats are there, and in elastin no gly XY repeats. Okay, hydroxylysine is present in a collagen. Hydroxylysine is absent in a elastin. Okay, and collagen have carbohydrate in them, while elastin have no carbohydrate. Okay, aldol cross links are formed in collagen. The cross linking I told is your aldol cross links, but in elastin your desmosine cross links are present. This is important. Okay, and in collagen there is the presence of extension peptides, and the no extension peptides are present in your elastin. Okay, this is given in your Harper. Okay, and degradation of collagen are called as your is dependent upon the action of a collagenases which belong to a metalloproteinases. Okay, and collagenopathies, first one is your Ehlers-Danlos syndrome. Okay, this is due to mutation in gene for type 1 as well as type 3 collagen, type 1 and type 3. Also type 5 is written in few books. Okay, so there is defect in lysyl hydroxylase enzyme, due to which there are hypermobile joint, hyperextensible skin. Look at this, the skin is hyperextensible. There is bruising and vesicular rupture also. Okay, bruising and vesicular rupture. Just see that the type 3 collagen, where it is present, is skin, lung and vessels. So there is vessel rupture in your Ehlers-Danlos syndrome. Okay, vessel rupture in your Ehlers-Danlos syndrome. Okay, there are six subtypes also. Your hypermobility, classical, vascular, kyphoscoliosis, okay, anthracoalasis and dermatosparaxis. This is not much important. This is 6 types. Okay, subtypes. The classical and vascular, these are autosomal dominant inheritance. Classical type is due to type 5 collagen defect. Okay, and vascular type is the most serious one, where there is the rupture of blood vessels and your internal organs often start to rupture. Okay, internal organs also start to rupture. So if you understand this, if you understood this, we talked about osteogenesis imperfecta. Let's talk about the Menkes disease. So Menkes disease is due to the ATPase 7A mutation. Okay, ATPase 7A mutation, where the intestinal deabsorption of copper is impaired. Okay, as lysyl oxidase requires copper. Okay, so there is neurodegeneration, there is kinky hair and your skeletal abnormalities. Okay, and your skeletal abnormalities, because lysyl oxidase requires copper and there is deficient of copper, so lysyl oxidase won't work. So there is no cross linking present. Then you have epidermolysis bullosa, which is a mutation in type 7 collagen. This is a mutation in type 7 collagen. Did I have talked about type 7 collagen earlier? Okay, yes, type 7 collagen, which is present in your anchoring fibrils, which is present in your anchoring fibrils. Here also it is written anchoring fibrils. Okay, so that is the defect in anchoring fibrils. Okay, manifestations will be that the skin will break and there will be blisters formation, petechiae and ecchymosis will be present. Okay, like this, this petechiae and ecchymosis will be there. Okay, the red spots. Scurvy, vitamin C deficiency I have talked about it. Talking a little bit about elastin and another thing about fibrillin. Okay, we'll finish the chapter. Okay, elastin, talking about elastin. So elastin is found in your lungs, wall of large arteries and your elastic ligaments. Okay, this will cause your extensibility and recoil property of lungs. Okay, blood vessels and your ligaments, the recoil property. And elastin from trophoelastin, in post translation modifications we get your elastin. Okay, elastin has no triple helix, no gly XY repeats, no hydroxylysine, rich in proline and lysine rich. Okay, and desmosine crosslinks are present. Diseases you can remember by a mnemonic. William is the assistant superintendent of police. Okay, William means William Bruren syndrome. Okay, we have the William Bruren syndrome. So William Bruren syndrome, there is the deletion of elastin gene. Okay, where there is supravalvular aortic stenosis. Importantly remember, SAS, we have SAS, supravalvular aortic stenosis. Then you can remember your scleroderma, there is increased elastin. Okay, then we have pulmonary emphysema, where there is increased elastin degradation. In fact, what happens in emphysema is that in emphysema, due to any stimulus, okay, your trypsin is released, and trypsin will, it's a protein digesting enzyme, so it will accelerate your elastin degradation. Okay, and then in the aging of skin you will see decreased elastin. Okay, so these 4 are your diseases. This aging is your physiological change. Okay, this is not can be caused as a disease. Okay, so these are your things. Just see that if you give alpha-1 antitrypsin, okay, it will inhibit your elastase, and prevent your trypsin from degrading. Okay, it will prevent your trypsin from degrading. Okay, so you can give your treatment of alpha-1 antitrypsin in your emphysema patients. Then talking about fibrillin is a myofibril component. Okay, it is a large glycoprotein distributed in the lens, periosteum and aorta. can remember lap. Okay, you can remember your lap, lens, periosteum, and your aorta. Okay, lap to remember the distribution. Disease is called as a Marfan syndrome. Marfan syndrome. Probably Abraham Lincoln had this syndrome. Okay, so in Marfan syndrome, what happens? There is a mutation in gene on chromosome number 15 for fibrillin 1. Okay, there is fibrillin 1 gene mutation in chromosome number 15, where there is the structure abnormality of fibrillin 1. Okay, and the sensory ligament of the eye. Okay, as it is present in your as I told earlier. Okay, it is present here. Okay, it is present in your lens and periosteum, and in your ligament also, sensory ligament. Okay, and the periosteum, media of the aorta, this all are affected. Okay, and this will cause your ectopia lentis. So ectopia lentis is also seen in Marfan syndrome. Okay, arachnodactyly is the spider like veins. Okay, and dilation of your ascending aorta dilatation. Okay, ascending aorta dilation is seen in a Marfan syndrome. Just look at the patient. It has ascending aorta dilation, so the chest is shaped like this. Okay, we have long digits. Okay, ectopia lentis, tall stature, and ascending aorta dilatation. Okay, and this causes the chromosome 15 mutation in which your fibrillin 1 gene is present. Okay, so we finished our extracellular matrix about collagen, elastin, and fibrillin all. Okay, so we talked about extracellular matrix. Let's move on to the next chapter. Okay, so let's discuss your organ function tests. Okay, so first we are going to liver function test. Liver function test, you can remember the mnemonic A, B, C, D, E, F, G, H, I. Okay, so from A we have albumin and Ag ratio and ammonia levels and alpha 1 antitrypsin, alpha L antitrypsin. Okay, and from B we have bilirubin level, the total direct and the indirect bilirubin, and bile pigments and bile salts. Okay, and for C you have cholesterol, coagulation profile, and your ceruloplasmin. Cholesterol, coagulation profile, and your ceruloplasmin. D, if I have the bromosulfathalin. Okay, bromosulfathalin. And E, you have enzymatic markers for liver function test. Okay, enzymatic markers for liver damage. To assess the hepatic function, you have enzymatic markers SGOT, okay, SGPT, your LDH4 and your LDH5. Okay, and to assess the obstruction, you will see obstructive tumor markers. Okay, the ALP, the GGT. Okay, and the 5-dex nucleotides. Okay, F stands for ferritin and your fetoprotein level, alpha fetoprotein. Okay, ferritin and alpha fetoprotein. G stands for globulin, your galactose tolerance test, globulin and your galactose tolerance test. H for hippuric acid excretion. Okay, hippuric acid excretion, your haptoglobin and your HBS antigen. Your hippuric acid excretion, your haptoglobin and your HBS antigen. Okay, and I stands for the immunological tests. Okay, so let's go one by one. Let's discuss the albumin. So albumin, it has a half-life of 20 days. It indicates as chronic liver damage. Okay, and normal values are 3.5 to 5 gram per deciliter. Just remember, albumin is 3.5 to 5 gram per deciliter. In Ag ratio, the normal Ag ratio is about 1.5 to 2 is to 1, 1.5 to 2 is to 1. Okay, and it is altered or reversed in chronic liver damage. It is altered or reversed in chronic liver damage. Ammonia, it is important in assessing your encephalopathy, liver cirrhosis, urea cycle disorders also. Okay, and alpha 1 antitrypsin, its deficiency will indicate your liver damage. Okay, deficiency will indicate your liver damage. And serum bilirubin, talking about serum bilirubin, it is most common in assessing a liver function in jaundice. Okay, in case of jaundice, the serum bilirubin levels are very important. You have the total bilirubin, the direct bilirubin, the indirect. Total bilirubin is up to 1.2 milligram per deciliter. Direct is 0.2 to 0.4. Indirect is 0.2 to 0.6 milligram per deciliter. Okay, total is just the sum. Okay, direct is 0.2 to 0.4 and indirect is 0.2 to 0.6. Okay. And in hemolytic jaundice, there is increase in indirect bilirubin. There is increase in your indirect bilirubin. Okay, and this gives a Vandenberg reaction after 30 minutes. Okay, this is a Vandenberg reaction after 30 minutes, which is called the indirect positive Vandenberg reaction. In case of a hepatic jaundice, there is an increase in indirect bilirubin plus your direct bilirubin. In hepatic jaundice, indirect plus direct. Okay, this gives a biphagic Vandenberg reaction, that is the immediate color, and this color intensifies on your addition of your intensifies on solubilization or solubilizing agents addition. Okay, in case of obstructive jaundice, there is only increase in your direct bilirubin. Okay, obstructive jaundice, there is increase in your direct bilirubin. Okay, you can remember the mnemonic ODHI, OD. Remember mnemonic OD, because in obstructive jaundice there is direct bilirubin increase, and in the hemolytic jaundice you have your indirect bilirubin, and in hepatic both are increased. Okay, so bilirubin, what is the Vandenberg reaction? The bilirubin is added with the diazotized sulfonylic acid, diazotized sulfonylic acid. Okay, which forms an azobilirubin, and this azobilirubin is a purple colored. Okay, which gives a positive Vandenberg test. Okay, understood. So bile pigment, we have the Fawcett's test for bile pigment. Okay, presence of bile pigment. Okay, where the pista green color is seen. It is done in a hepatic and your obstructive jaundice, hepatic as well as your obstructive jaundice. Okay, And for your hemolytic jaundice, you use urobilinogen, which appears in urine in your hemolytic jaundice only, okay. And then talking about cholesterol is the indicator for chronic liver damage or chronic liver cirrhosis, okay. Liver cirrhosis, where there is decrease in your cholesterol. Okay, normal is your less than 200 milligram per deciliter, okay. Let's talk about coagulation profile. Is a prothrombin time, okay. Prothrombin, which has a half-life of 6 hours, okay, which increases in your liver damage, okay, because the plasma proteins are not produced, okay. So there is increase in your prothrombin time, okay. That is increase in your clotting time, the prothrombin time, okay. So it will indicate your acute liver damage. Talking about ceruloplasmin, okay, which will be decreased in your Wilson's disease, okay. Wilson's disease is your cerebrohepatolenticular disorder, okay, and the ceruloplasmin decreases in your Wilson's disease. Dye test, we have the bromosulfonamide dye, okay, bromosulfaphthalin dye, okay. So we have the BSP excretion test, which will indicate the excretory function of liver. What happens is that you inject the BSP via the intravenously, okay, and you measure the serum BSP, okay. After 45 minutes, okay, it is 5% is retained by the normal liver, okay. 5% is retained by the normal liver, okay. If there is increased retention of your BSP, so the liver is not performing its excretory functions, okay, and this indicates your liver damage, okay. Then enzymatic markers, SGOT or your AST, and SGBT or your ALT. SGOT, let's talk about SGOT. If this is the normal values are 5 to 40 international unit per liter, normal SGOT. Normal SGBT is about 5 to 35 international unit per liter, okay. Increase in SGOT will indicate your alcoholic liver damage, your cirrhosis, and your myocardial infarction too. This is important. It also indicates a myocardial infarction, AST. But at ALT, this only indicates your infective hepatitis or your liver disorders only, okay. So ALT is more specific than your SGOT, your AST. So there is the specificity of ALT is more than your AST, because the AST also elevates in your myocardial infarction, okay. And this SGOT, this increases up to 100%. This increases up to 100% in case of your infective hepatitis, okay. This both increase up to 100% in infective hepatitis, okay. And LDH 4 and 5, the levels will indicate your liver damage, okay. And the cholestatic marker is there, the biliary obstruction. So biliary obstruction, you have ALP levels, okay. So it increases up to 10 to 12 times of normal in your cholestasis, and increases up to 20 to 25 times, okay, in cancer ALP levels, okay. And your gamma glutamine transferase, this is specific for your alcoholic liver damage. So alcoholics are generally screened for your GGT, okay, gamma glutamine transferase. Your 5-nucleotidase is increased in your cholestasis, okay. Then your serum ferritin, which is decreased in your ammonia and increased in your hemosiderosis, okay. And the serum ferritin indicates your storage status of iron in your liver, okay. And the alpha fetoprotein, this slightly increase in your liver cirrhosis, but highly increases in your liver cancer, okay, alpha fetoprotein. And alpha fetoprotein levels, if they are much more higher, okay, this will indicate a neural tube defects. The globulin level, normal globulin is 2 to 3 gram per deciliter. Your normal albumin was 3.5 to 5 gram per deciliter, okay. And the normal globulin levels are your 2 to 3 gram per deciliter, okay. These values are important, so I am revising it. So this increases in your chronic liver disorders. Okay, globulin levels increase chronic liver disorders. Then galactose tolerance test, it is used to assess the metabolic function of your liver. The test used to assess the excretory function was your BSP diet test, okay. To assess your excretory function was that, okay. To assess your storage function of liver, we had what test? Your serum ferritin test to assess the storage status of iron, serum ferritin test. And then you have galactose tolerance test to measure your metabolic function of your liver, okay. In which the excretion of galactose is measured, and retention of galactose, if the galactose is retained in your liver, okay, it will indicate your liver damage. And HbAg, okay, hemoglobin antigen, this will indicate liver damage by hepatitis B virus. Then we have haptoglobin is also affected in liver disorder, okay. The immunological tests are there, okay, many immunological tests are there, which will indicate hepatitis A, B and C viruses, okay. Then we have a small topic of the function of liver. Synthetic parameters like your albumin, cholesterol and prothrombin are measured, okay. In metabolism, carbohydrate glycogen metabolism, gluconeogenesis, blood glucose regulation, all these are done by your liver, okay. And protein catabolism is done by liver. Your fat, triglyceride, ketone body, cholesterol synthesis, okay. Detoxification of ABCD. What is ABCD? Your ammonia, bile pigment, cholesterol and drugs. Detoxification of ammonia, bile pigment, cholesterol and drugs. This is also done by your liver. And the storage function like a vitamin A, D, E, K, okay, A, D, K, and your vitamin B12 and your glycogen, this is stored in your liver, okay. and liver function test. This indicate all these disorders we discussed earlier. Okay, there is a classification of your liver function test. Okay, if there is a question that classify the liver function tests, classified based on the biochemical parameters: the excretory function for measuring the excretory function, for assessing the enzymatic function, for metabolic function, for synthetic function, and special tests. Okay, for excretory function, we use urobilinogen, bile salt, bile pigment, and bilirubin tests. Okay, for enzymatic function, we have SGOT, SGPT, and all. For metabolic function assessment, we have the galactose tolerance. This is not a glycogen tolerance. This is galactose tolerance test. Sorry, galactose tolerance test. Okay, and then you have synthetic functions. For assessing the synthetic functions, you have total protein, albumin, globulin, Ag ratio, and special tests like ferritin, alpha 1 antitrypsin, AFP test. Ferritin is for your storage assessment. Based on clinical aspects, we have the liver dysfunction test. Okay, like bilirubin, BP, bile salts, urobilinogen. Okay, and albumin, Ag ratio, these all. And another test is based on enzymatic markers like your SGOT, your ALP, your SGPT, and then your 5-desynucleotidase, your GGT, and all. Okay, so this completes your liver function tests. Okay, let's now move to your renal function tests. So here we are discussing the renal function test to assess the kidney diseases. We have the renal function test, and first to assess the kidney diseases. Okay, and to assess the renal function. Okay, renal function tests are there. Okay, so to assess the kidney diseases, we use serum parameters like urea, creatine, sodium, potassium. These are your serum parameters. Okay, these values are very important of this all. Urine analysis, we do functions of urine analysis. You have read in your practical classes, urine analysis, where the urine is checked for your pH and your turbidity, and the color of urine is checked, and all will see. Okay, and then to assess the renal function, okay, many tests are there, like your assessment of glomerular function. We have the clearance test. Okay, for assessing the permeability, we have the protein estimation test. For assessing the tubular pH, we have the concentration tests and the dilution tests. Okay, so functions of kidney is to regulate the acid base balance. Okay, to regulate the acid base balance, BP regulation, water balance, and electrolyte regulation. Okay, kidney does the synthesis of vitamin D and erythropoietin. Okay, let's say this is the filtration, reabsorption, and secretion. Okay, you must have known this. Okay, like about 180 liters per day are filtrated, reabsorbed, 178.5 are reabsorbed. Okay, understood. And urine that is secreted out, that is 1.5 liters per day only. Okay, and what things are filtrated? Your glucose, amino acid, potassium, HCO3 minus is your filtrated and reabsorbed and all. Okay, so test for your glomerular filtration. Okay, first you have the clearance test. Okay, what is clearance of a substance? So definition is important. This is the amount of plasma. Okay, just note, this is the amount of, not your substance. This is the amount of plasma that would be completely cleared up a substance in unit time. Your okay, the clearance of insulin is more than your creatine, and it is even more than your urea. Okay, insulin, creatinine, and urea. Okay, insulin is the best. It is used in the best because it is not reabsorbed and not secreted also, so the levels remain constant. So insulin is like it is the best reagent for your measuring your clearance. Okay, and it gives value the nearer to the GFR. It gives value exact to the GFR, like 125 ml per minute. GFR is 125. Okay, so insulin is the best, but disadvantage is that it has to be administered IV, so it is a little bit invasive procedure is there. Talking about creatinine is freely filtered, not reabsorbed, but a little bit is secreted. Okay, so in your males, the creatine values are 90 to 115 ml per minute. In females, it is about 85 to 110 ml per minute. Okay, and these are little less than the this will give values little less than the GFR, little less than 125, but this is also preferred. So this is also widely used. Okay, this creatinine clearance is like widely used. And for the urea is freely filtered, but there is 40% reabsorption, and it is also affected by protein diet, so it is not used for clearance. Okay, but you can measure the urea clearance if you want. Okay, so this is there. After this clearance, the formula is UB by P, where U is the urinary creatinine, and V is the volume of creatinine, okay, in ml per minute. Okay, and P is the plasma creatinine. So CCR, the creatinine clearance, is UCR by V by PCR. Okay, so UCR is the urine creatinine, V is the volume of creatinine in ml per minute, and P is the plasma creatinine. Okay, and we have another equation which is called as Cockcroft-Gault equation. Okay, so this states that the creatinine clearance is 140 minus age into weight by 72 into PCR. Okay, what is PCR? Your plasma creatinine in milligram per deciliter. Okay, and this is multiplied by 0.85 in case of female. So what is a Cockcroft-Gault equation? It is a creatinine clearance which is equal to 140 minus of age into divided into your weight by 72 into PCR. Okay, in case of females, it is multiplied by 0.85. Okay, understood? So this is a Cockcroft-Gault equation. Okay, if the creatinine clearance is more than 90, if it is more than 90, then the normal renal function is indicated. If it is 60 to 90%, then mild renal impairment. 30 to 60%, moderate renal impairment. Less than 30% is dangerous, severe renal impairment, and less than 15% will indicate renal failure. Okay, so creatinine clearance must be always more than 90%. Must be always more than 90%. Okay, understood? And let's talk about the test for the glomerular permeability. For the glomerular permeability, we use urinary proteins. Okay, we have the tubular function test. Okay, which will assess the specific gravity of urine. Normal specific gravity is 1.010 to 1.025. This is normal specific gravity of a urine. So you have the concentration test. Okay, and you have the dilution test. Okay, in dilution test, 1200 ml of water is given over 30 minutes. Okay, and 4 samples are taken and assessed for your dilution. Okay, and in your concentration test or your fluid deprivation test, as it is called. Okay, it is also in this manner. Okay, it is also measuring your specific gravity of your urine. Okay, and if it is lesser, so it will indicate your diabetes insipidus or your ADS deficiency diabetes insipidus. Okay, understood? So concentration tests and dilution tests. Okay, after your kidney function tests, let's move on to your thyroid function tests. So talking about thyroid function test, so thyroid gland is an endocrine gland, bilobe gland, situated in front of your trachea, which releases your hormone T3, your T4 and your calcitonin. Okay, the C cells release calcitonin. T3 is called as a triiodothyronine and T4 is called as a tetraiodothyronine or a thyroxine. And calcitonin is also a hormone which is reduce the plasma levels of your calcium. Okay. So let's start discussing. The function of thyroid hormones is to increase the BMR, stimulate protein synthesis, increase the blood glucose level, okay, and maintain the water and electrolyte balance, and mature and develop the body, okay, and will facilitate utilization of your lipids. Okay, so iodine molecules get attached to your tyrosine residues of your thyroglobulin. So in the release process, there are many mechanisms of release, like if you must have read, there is iodine trapping. Okay, then it is organified to I2. Okay, then I2 is now coupled. Okay, the coupling reactions are there. The two molecules of DIT and MIT. Okay, DIT plus MIT will form your T3, and your DIT and another DIT will form your T4. Okay, so stimulated by your TSH. Okay, and this all are stored in the colloid form. And this colloid is bound to your thyroglobulin. Colloid is bound to your tyrosine residue of your thyroglobulin. Okay, so basically, if there is a TRS, if the TRS is reduced, okay, it is called as a tertiary disorder. Okay, if there is a secondary disorder, so there is the deficit at the level of your anterior pituitary. Okay, and if the T3 and T4 are deficit, then it is a primary disorder. Okay, then it is your primary disorder. Okay, understood? Then talking about a hypothyroidism, this is your decreased T3 and T4 hypothyroidism, but increased TSH levels. Okay, because TSH is reduced. Okay, TSH is reduced, but the T3 and T4 are not produced. Okay, so this is most commonly seen in iron deficiency goiter. Okay, hypothyroidism, and Hashimoto's thyroiditis is there. Secondary hypothyroidism is there. The tertiary hypothyroidism is there. Okay, and iron deficiency goiter is the most common hypothyroidism disorder. In hyperthyroidism, your T3 and T4 are elevated, but the TSH levels are decreased. Okay, like in your Graves' disease, your nodular goiter, and in your MN goiter. Okay, so normal T3 and T4 levels, but TSH levels are affected. This is a subclinical thyroidism. Okay, normal T3 and T4, but TSH levels are affected. This is called as a subclinical hypothyroidism. Okay, talking about a Graves' disease or a Paris disease or a Basedow disease or a thyrotoxicosis, it has many names. Okay, or the exophthalmic goiter. It has the symptoms. It has the symptoms of hyperthyroidism. Okay, which symptoms? So symptoms of hyperthyroidism can be seen in this symptom. Goiter. Okay, the gland enlargement is there, edema is there, irritability, tremors, restlessness, okay, excitability, and heat intolerance is there. Okay, heat intolerance is there. So thyroid hormone is normally present in a bound form with albumin. It is bound with the albumin, but in blood. Okay, but in some in your blood it is somewhat present in a free form also. Like total T3 is a bound T3 plus a free T3. Total T4 is a bound T4 plus free T4. Okay. Talking about TSH is sensitive and specific test. Sensitive and the specific test for assessing the thyroid function. Normal levels are 0.5 to 50 mu international unit, micro international unit per liter. This is the normal level, 0.5 to 50. Okay, micro international unit per liter. Free T4 normal levels are 0.7 to 1.8 milligram per deciliter. 0.7 to 1.8 milligram per deciliter. Okay. Okay, let's talk what will be the approach to a patient with your thyroid disorders. Okay, so thyroid function tests are just simply you're measuring your TSH, your free T3, and the total T3, your total T4, and all. Okay, so first, if a patient comes with a hypothyroidism, you will know the patient suffering from hypothyroidism from the symptoms. Okay, so ask the patient to do the TSH and the free T4, TSH and the free T4. If the TSH is increased and the free T4 is decreased, okay, so it is primary hypothyroidism. Your TSH and is increased, but your free T4 is decreased. Okay, your free T4 is decreased. Okay, so it is your primary hypothyroidism, as the disorder is not in your TSH, disorder in your T3 and T4. Okay, and if there is primary hypothyroidism, ask the patient to do a anti-thyroid peroxidase antibody, okay, TPO antibody test. TPO antibody test. If the TPO antibody is positive, okay, then the autoimmune disorder Hashimoto's thyroiditis must be there, and if it is negative, then iodine deficiency goiter is simply there. Okay, and if the TSH is high and the normal free T4 is there, TSH is high and normal free T4, so it is called as subclinical hypothyroidism. And if your TSH is decreased, TSH is also decreased, and free T4 is also decreased, so this is your secondary or tertiary disorder. Okay, ask the patient to do the TSH stimulation test. Okay, if the TSH is normal, okay, if the TSH is normal, then the pituitary causes can be like suspected. If there is delay in increase in TSH, then hypothalamic causes can be suspected. Okay, then let's say a patient comes with a hyperthyroidism. Okay, ask the patient to do TSH and free T4. Okay, if there is decrease in TSH and increase in your free T4, okay, then there is primary hyperthyroidism. There is decreased TSH but increase in free T4, primary hyperthyroidism. Okay, ask the patient to do anti-TRH antibody test. Okay, if it is positive, then it will also be the autoimmune causes may be suspected. Your Graves disease must be there. Okay, if it is negative, anti-TRH antibody is negative, ask the patient to do radio iodine uptake study. Okay, and this will cause a suspicion of a toxic nodular goiter if it is positive, or a multinodular goiter. Okay, if the TSH level is decreased but FT4 is normal, so prescribe FT3, okay, free T3. If the free T3 is also normal, then there is subclinical hypothyroidism. If there is increased free T3, okay, so there is decreased TSH, normal FT4 but increased FT3, okay, then you will suspect your T3 thyrotoxicosis disorder. Okay, and if there is increased TSH and increased FT4, okay, increased FT4, so TSH is also released. Okay, so there is resistance to thyroid hormone, okay, or the pituitary causes or your resistance to thyroid hormones. Okay, so you can prescribe your TRH and TSH tests. Okay, other tests are there, like your ultrasonography is there, your CT scan is there, your FNEC is there. Okay, and the calcitonin level is a marker for your medullary carcinoma of the thyroid gland we saw from the cancer topic. Okay, and thyroid blinding globulin, the TBG levels should be assessed for more details, and radio iodine uptake studies are also there. Okay, so this completes your organ function tests. Okay, let's move on to the next chapter. Let's discuss another small chapter, that is your free radicals. Okay, free radicals. So free radicals, free radicals have more than or one unpaired electron. They are highly reactive, shorter lifespan, and cause damage to the tissues. Okay, so this O2 minus radical is the superoxide anion. Okay, HOO radical, that is your hydroxyperoxyl radical. Okay, OH radical is your hydroxyl radical. NO radical is your nitric oxide radical. Okay, ONOO radical is your peroxynitrite radical. So this former three are your reactive oxygen species, and nitric oxide and peroxynitrate, these are your reactive nitrogen species. And other two, like H2O2 and IO2 singlet oxygen, these are your free radicals, but unpaired electrons are absent here. Okay, what are the sources of your free radicals? So you can have endogenous sources from the body. Free radicals are generated from your ETS, your NO synthase, your respiratory burst, xanthine oxidase, and your prostaglandin synthesis also your free radicals are generated. The lipid peroxidation free radicals are generated, and auto oxidation of metals, okay, heavy metals also produces your free radicals. So ETS, NO synthesis, respiratory burst, xanthine oxidase, prostaglandin synthesis, lipid peroxidation, and your auto oxidation of metals. And in endogenously you have alcohol, which will produce the free radicals, the radiations like UV rays and gamma rays and X-rays, the drugs and the smoking. Okay, these all will are the exogenous agents, okay, which will produce the free radicals. So how respiratory burst produces free radicals? If you ingest a bacteria, the macrophage ingests a bacteria, okay, and this will what it will do, that the O2 will be converted in the presence of NADPH oxidase to O2 minus radical. Okay, O2 minus radical. This O2 minus radical is called as your superoxide anion. Okay, so this O2 is reduced to your superoxide anion by NADPH, which is changing to NADP. Okay, and this O2 okay, superoxide anion in the presence of superoxide dismutase, the SOD enzyme, which is a chain breaking antioxidant. Okay, this will convert it into H2O2, and this H2O2 will be converted into HClO, and this HClO will kill the bacteria. Okay, so respiratory burst is the sudden demand of your oxygen. When sudden oxygen demand increases, okay, which will produce your free radicals. Okay, this free radicals are produced, O2 minus radical. Okay, and by using an antioxidant, you can kill the bacteria. Then lipid peroxidation, there is an initiation, elongation in the termination steps. How it is initiated? First, the RH in the presence of RH is a PUFA, any PUFA, okay, polyunsaturated fatty acid. In the when it reacts with the OH minus radical, it is changed to R minus R radical and H2O. Okay, so RH in the if it reacts with OH minus radical, R plus H2O. This R radical converts then again binds with your O2, okay, to form ROO minus radical, which is called as a lipid peroxyl radical. This ROO minus radical again reacts with RH, okay, to form ROOH plus again another lipid peroxide and a free radical. Okay, so free radicals are generated by this process. How it is terminated? If ROO minus radical reacts with another ROO minus radical, then you form ROOR plus H2O. Okay, in this process no free radicals are generated. So lipid peroxides can be measured by Fierce oxidation in xylenol orange. Okay, xylenol orange is the reagent for your lipid peroxide measurement. Okay, which is called as a FOX assay, FOX assay. Okay, so disorders reacted to reactive oxygen species. So you can have inflammation, acute inflammation. You can have Alzheimer's disease, multiple sclerosis, Parkinson's, cataract. Okay, then your diabetes mellitus and myocardial infarction, atherosclerosis, and cancer. Okay, so you can remember the trick ABCDMP, where A stands for atherosclerosis, your Alzheimer's, and your acute inflammation. B stands for your bronchoalveolar disorders. Okay, C is for your cataract, D is for diabetes mellitus, M is for your myocardial infarction and multiple sclerosis, and your P will stand for your Parkinson's disease or your paralysis agitans. Okay, this atherosclerosis, in this the LDL will be increased. Okay, and the free radicals will favor the formation of oxidized LDL, which will form the foam cells. Okay, foam cell plagues are seen in atherosclerosis, so will form foam cells. Okay, and this diabetes mellitus also, the free radicals are damaging your beta cells, okay, which is called as insulin dependent diabetes mellitus. Okay, so these are the disorders which are related with your free radicals, okay, or your reactive oxygen species. Okay, these are some of the complications seen in the diagram. Okay, you can see what are the oxidative stresses is causing. Okay, done. So let's move on to the next chapter after free radicals, a chapter related with the free radicals, that is antioxidants. Talking about antioxidants, these are compounds which decrease or prevent the oxidation of compounds. These are of two types, your enzymatic and your non-enzymatic. Okay, so enzymatic, you can remember the mnemonic SC Gupta. Okay, so S stands for your superoxide dismutase, SOD. This SOD is formed in two isoforms, your cytosolic part and mitochondrial form. Okay, the cytosolic part requires your copper and zinc, and mitochondrial part requires manganese, okay, for its use. So what it does, it converts your O2 minus radical, okay, your superoxide radical, okay, this radical, and if the radical reacts in the presence of H plus and superoxide dismutase, it is converted into H2O2, and this H2O2 in the presence of catalase is converted to H2O. Okay, next we have catalase. The function of catalase is to convert the H2O2 to H2O. Okay, convert the H2O2 to H2O, and this G stands for glutathione peroxidase. Okay, glutathione peroxidase, let's see the function. The H2O2 is there. Okay, H2O2 in the presence of glutathione peroxidase, it is changed to 2H2O plus O2. Okay, it is detoxified. Okay, this H2O2 is detoxified in the presence of your glutathione peroxidase enzyme. Okay, and then we have glutathione reductase this is a glutathione reductase enzyme. Okay, so the glutathione peroxidase also converts your reduced glutathione GSH to GSSG, or oxidized glutathione. Okay, this is again reduced. This oxidized glutathione is again reduced to a reduced glutathione by this enzyme, which is called as a glutathione reductase enzyme. Okay, glutathione reductase enzyme. Glutathione peroxidase is another function, that is it converts ROOH in the presence of 2GSH. Okay, it converts into the ROOH into a hydroxy fatty acid, okay, H2O, and this reduced glutathione into your oxidized glutathione. Okay, oxidized glutathione. Then you have non-enzymatic antioxidants. You have exogenous and you can have endogenous. Okay, exogenous, you can have vitamins and minerals. Okay, so vitamins, we have three vitamins: vitamin C, vitamin E, and your vitamin A, or your beta carotene form. Okay, and minerals, we have your Zn, Sc, Cu, Mn. Okay, so you can just remember this mnemonics which I have made. Okay, Janam se Buraman. Okay, Zn, Sc, Cu, Mn. Okay, And you, let's talk about your vitamin E. So vitamin E, your active form is alpha tocopherol. Okay, so ROO radical in the presence of if it reacts with the reduced vitamin E, it is converted into ROOH. Okay, so it converts ROO minus to ROOH. Okay, and the reduced vitamin E is converted to the oxidized vitamin E. As you can see, it is a redox reaction that is occurring here. Okay, and the oxidized vitamin E is again converted to reduced vitamin E in the presence of a vitamin C. So what are the reactions of vitamin C? Vitamin C, as you can see here, it is also an exogenous non-enzymatic antioxidant. Okay, so you can see that the vitamin C, it is doing the reduction of oxidized vitamin E, of course. Okay, the reaction saw earlier. The vitamin C also converts this O2 minus radical into H2O2 and your monodihydroascorbate. Okay, and the vitamin C, the ascorbic acid, okay, reacts with OH minus radical, converts it into H2O plus monodihydroascorbate. Okay, it gets reduced into monodihydroascorbate there. Okay, vitamin C also prevents a lipid peroxidation. Lipid peroxidation was producing a free radical, so it prevents a lipid peroxidation and also prevents a cataract formation. Okay, cataract formation. Then endogenously we have this, you can just remember this mnemonic. Okay, you can just remember this mnemonic. You can just take a screenshot here. Done. Okay, so you can remember this mnemonic, that is up from C you have your ceruloplasmin. Okay, from U you have your uric acid. Okay, from TE you have your transferrin. Okay, you have your transferrin, ceruloplasmin, uric acid, transferrin. Okay, from B you have bilirubin. From F you have ferritin. Okay, from P you have phytochemicals. Okay, from U you have ubiquinone, phytochemicals. Okay, you have phytochemicals, you have then ubiquinone, and from Li you can have lipoic acid. Okay, and a question is asked, what are the chain breaking antioxidants? So you can remember SUV. S stands for your superoxide dismutase. Okay, U stands for your uric acid, and V stands for your vitamin E. Okay, SOD, uric acid, and your vitamin E, these are your chain breaking antioxidants. Okay, so this was all about your antioxidants. Okay, this was all about your antioxidants, and now let's move on to the next chapter. So now let's talk about your xenobiotics metabolism. Okay, so xenobiotics refers to any foreign compounds, okay, which are there to be detoxified in our body, like your drugs, chemical carcinogens, your polychlorinated biphenyls, insecticides, pesticides. Okay, these all are xenobiotics, like drugs, like antibiotics. Okay, and your heavy metals, like your AS and PB. Okay, invalpaints, your hormones like estrogens, excess estrogens. Bacterial degradation products like histamine, cadaverine, putrescine, tyramine, and tryptamine. These are bacterial degradation products that should be removed from the body. Plastics like PVC, insecticides and pesticides like ODTs, like the DDTs, polychlorinated biphenyls, PCBs, and dioxins. Okay, these all are your xenobiotics. Okay, so xenobiotics should be converted into less toxic materials, but sometimes it may be converted to more toxic or non-toxic materials. This process of conversion is called as biotransformation. Okay, which incorporates detoxification and intoxication. Okay, and this all substances are then eliminated. So xenobiotics metabolism may be advantageous or disadvantageous. Advantageous in the case if this activation of prodrug to active compound is occurring, then it is advantageous. But if the formation of carcinogen from inert precursors are occurring, then it is disadvantageous. Okay. Now, what are the types of reactions? So we have the phase 1 reactions as well as phase 2 reactions. So phase 1 reactions render the compounds more reactive. Okay, these are the reactions which can render the compounds, the xenobiotics, more reactive. Okay, and for detoxification of phase 2 reactions, these are the conjugation reactions generally. Okay, these are your conjugation, acetylation, and methylation. Okay, so phase 1 reactions, we have the main reaction, that is hydroxylation. Okay, using the cytochrome P450 and all. Okay, and deamination, dehalogenation, desulfuration, epoxidation, peroxygenation, reduction, hydrolysis, and some non-cytochrome P450 reactions. Okay, these are your phase 1 reactions. Okay, and among them your hydrolysis, reduction, and your hydroxylation and hydrolysis, this three, hydroxylation, reduction, and your hydrolysis, these three are important to discuss. Okay, so let's talk about your hydroxylation, or which is also called as your oxidation. Okay, so here cytochrome P450 participates in phase 1 xenobiotic metabolism. Okay, we have about 57 cytochrome P450 genes, and of the major cytochromes are called as CYP1, CYP2, and CYP3. Okay, CYP1 has about 3 subfamilies, CYP2 has about 13 subfamilies, and CYP3 has about 1 subfamily. Okay, so cytochrome P450 is a heme enzyme. Okay, it is a mixed function oxidase enzyme. Okay, a monooxygenase. Okay, belongs to your class 1 of enzymes. Okay, it possesses overlapping substrate specificity. Okay, the substrates are wide ranging for your cytochrome P450, and the cytochrome P450 isomers form a superfamily. Okay, all the isomers form a superfamily. Like one of the members is CYP1A1. Okay, how it is named? Look at the this part. Look at this part here. So CYP denotes the cytochrome. Okay, CYP denotes the cytochrome. P450, and 1 is the member of family 1. Okay, member of family 1, and this A refers to a subfamily A, and this 1 again refers to the first member of the subfamily. Okay, the first member of the subfamily. And cytochrome P450 is found in the liver and your enterocytes. This hang, like dangle to the SCR membrane. This dangle to the SCR membrane like this. Okay. So we have a genobiotic XH. Okay, this reacts with oxygen, NADPH and H plus. Okay, in the presence of cytochrome P450, it is converted. The genobiotic is converted into XOH, which is a more water soluble compound. Okay, water soluble form. Okay, so look, now it has become more reactive. Okay, due to the unpaired electrons of O. Understood? This O2 and H plus combine to form your H2O. Okay, and this NADPH is giving your NADP. Understood? So this is the overall reaction. Just remember the overall reaction. Okay, cytochrome P450 is named so because it showed absorbance at 450 nanometer when it was first isolated. Okay, it is an inducible enzyme. Okay, like we have important examples like a phenobarbital drug. The phenobarbital drug. Okay, it induces a CYP29. It induces a CYP29, which helps in the biotransformation of warfarin drug. Okay, warfarin is an anticoagulant. Okay. Then we have an ethanol drug, okay, which induces your CYP2E1. Okay, and the CYP2E1 metabolizes your tobacco smoke, releasing dangerous metabolites which are pro-carcinogens. Okay, so ethanol will increase your carcinogenic risk by inducing a CYP2E1, which will metabolize your tobacco smoke. Okay, causing dangerous pro-carcinogens. Okay. Major site of detoxification is your liver for all that is. Then we have the grapefruit. Grapefruit has furanocoumarins. This furanocoumarins inhibit the cytochrome P450 and affect the metabolism of many drugs like statins, omeprazole, antihistamines, and benzodiazepines. Okay, this 4 drugs metabolism are altered by a furanocoumarins which is present in a grapefruit. Okay, and this CYP2A6 has 2 alleles. Okay, 2 alleles, a wild type and an inactive type. Okay, this inactive allele can't metabolize nicotine to ornithine. Okay. So these are some examples which are given in a Harper textbook. Okay, so I have written these examples. Then talking about what other things. Okay, talking about what other things are done. Hydroxylation. Some other examples of your hydroxylation. So I have methanol being converted into your formic acid, then ethanol converted into your acetic acid, benzoyl alcohol into benzoic acid, benzaldehyde into benzoic acid, chloral into trichloroacetic acid, aliphatic amines into aliphatic acid plus urea, aniline into aminophenol, and benzene into your phenol, sulfur into H2SO4, meprobromate drug into hydroxymeprovamate. Okay. Then we come to reduction reactions, in which the nitro compounds are reduced to your amines. Okay, and the aldehydes and ketones, these are reduced to your alcohol. Like picric acid is reduced to your picramic acid, paranitrophenol is reduced to your para-aminophenol, chloral is reduced to your trichloroethanol. Okay, here it is, the chloral was becoming trichloroacetic acid. Okay, in the presence of your cytochrome P450, the hydroxylation reactions. Okay, but here the chloral is changing into a trichloroethanol. Okay, coming to hydrolysis reactions, A or B in the presence of H2O becomes AOH plus BH. Okay, this is called as a hydrolysis reaction, addition of water. Okay, which splits the compound. What compounds are the substrates for your hydrolysis? Okay, so you have anesthetics, you have amines, you have hydrazines, you have amides, you have glycosidic bonds containing compounds, carbamates, COX inhibitors, like your aspirin. The drug aspirin is hydrolyzed. Okay, this is acetyl salicylic acid, which is a non-selective NSAID COX inhibitor. Okay, which inhibits the cyclooxygenase 1 and cyclooxygenase 2. Okay, this is also an example of your hydrolysis. Okay, and aspirin is an analgesic, antipyretic, and anti-arthrogenic drug. Okay, and we can have the hydrolysis of acetylanides also. Coming to phase 2 reactions, we have glucuronide conjugation first, which is important. Okay, phase 1 reaction will give you a new metabolite which is more reactive than the former one, and then this new metabolite is added with UDP glucuronate, okay, and transported out. So this glucuronide conjugation helps in the transport. Okay, so first we have your bilirubin. Bilirubin is conjugated with UDP glucuronate in the presence of UGT, UDP glucuronate transferase. Okay, it is converted to bilirubin monoglucuronide plus UDP. Understood? and this deficiency, if deficiency occurs, okay, there will be a syndrome which will see in your heme metabolism, which is called as a Crigler-Nazar syndrome. Okay, and this UDP glucuronidase transferase, okay, this is present. Okay, this is present in cytosol and ER. And bilirubin monoglucuronide again gets conjugated with your glucuronide, okay, conjugated with glucuronide to form bilirubin diglucuronate and UDP. Okay, this is also a reaction. Then have a carcinogen 2-acetyl aminofluorine, which can be also undergone your conjugation with glucuronide, aniline, okay, benzoic acid, phenol, and steroid hormones. These all can be excreted as glucuronides. Okay, understood. The next reaction is a sulfuration. Okay, sulfuration where PAPS is present. PAPS is adenosine 3-phosphate 5-phosphosulfate. Adenosine 3-phosphate 5-phosphosulfate. Okay, this will donate the active sulfur group. Okay, so PAPS is gonna donating the sulfur group for your sulfuration reaction. Okay, and this PAPS, like what for which compound sulfuration is done. So we have steroids in which you can do the sulfuration. We have GAGs, okay, we have glycolipids, and have glycoproteins for which sulfuration can be done. Okay, like phenols and indols, XOH. PAPS is there, so it will donate your sulfur group to the XOH, forming XOS. Okay, in the presence of that H, you will get S. Okay, this will form phenyl sulfates or indoxyl sulfates depending upon which phenol or indol it is. Okay, and PAP will be released. Okay. Then we have conjugation of glutathione. Okay, what is glutathione? Glutathione is a tripeptide, a sulfur containing tripeptide, okay, containing glutamate, cysteine, and glycine. So it is also called as a glutamyl cysteine glycine. Okay, enzyme is your glutathione S transferase. Glutathione S transferase, also called as your ligandin. Okay, ligandin, or the king of ligands. Ligandin, or the king of ligands. Okay, so it is a cytosolic enzyme. It has two classes. It may be a homo or heterodimer with 7 subunits. Okay, two classes it has. Cytosol it is present, and it has 7 subunits. So bilirubin is conjugated with the glutathione. Okay, glutathione binds to it at other than its active site, not at its active site. Okay, then it is transported to the blood. From blood it is transported to the liver, then to the ER, and at the ER it is then conjugated with the UDP glucuronate. Okay, so first conjugation of bilirubin is with the glutathione. This is a recent concept which is discussed. And this glutathione S transferase is an early marker of liver damage also. Okay, this is also an early marker of liver damage. Okay, then we have the conjugation with the glycine. Okay, glycine is also an amino acid. So benzoic acid plus glycine, this will form hippuric acid. Benzoic acid plus glycine gives you hippuric acid. Okay, cholic acid plus glycine will give you glycocholic acid. Then quinodeoxycholic acid plus glycine will give you quinodeoxycholic acid. Then conjugation of glutamine. So phenyl acetate is converted into phenylacetyl glutamine. Okay, in this process, these are not important reactions, so I'm just discussing it in brief. Okay, then have conjugation with the thiosulfate. Like a cyanide is converted into its excretable form on the conjugation with the thiosulfate. Okay, and okay, these were your conjugation reactions. Another thing is that the xenobiotics can produce toxic effects by 3 ways. Okay, what are the 3 ways that can be implemented by xenobiotics to produce toxic effects? Okay, so first, what can happen, that may be covalent bonding with DNA, RNA, or protein. Okay, covalent bonding may occur. Okay, this will lead to cell injury. Okay, because DNA repair, if it damages the DNA, it will repair your ADP ribose. Okay, DNA repair requires ADP ribose. This ADP ribose this comes from your NAD. Okay, this ADP ribose comes from NAD. Okay, so NAD levels will decrease. Due to decrease in reduction NAD level, this will decrease the ATP, and decreased ATP will lead to your cell death. Okay, so ADP ribose transferase will be activated for your DNA repair. Okay, so we will require ADP ribose, and due to this, your deficiency of ATP is occurring, which will lead to cell death. Okay. Then second is that the reactive metabolite that is produced in the phase 1, okay, it will bind and alter the antigenicity of the protein. Okay, so autoantibodies may be produced, and it may cause your autoimmune disorders also. Okay, so we have a risk of seeing of autoimmune disorders. Okay, then we have the activated xenobiotic. Okay, this will act with the DNA. Okay, and the activated xenobiotic will act with the DNA and damage the DNA. Okay, like a benzopyrene. Okay, which induces your CYP14. Okay, and which activates the metabolite to carcinogen. We saw the reaction above. Okay, and the enzyme called epoxide hydrolase, this prevents it. Okay, this is called your activated xenobiotics when it acts with your DNA. Okay, so that's all in xenobiotics. Let's move on to the next chapter. So look here, we completed some of the chapters. Okay, so we have completed our organ function tests, nutrition, free radicals, antioxidants and xenobiotics, cancer, tumor markers, ECM, okay, and any other chapter? Yes, acid base balance. Okay, so we completed this much of chapters. Okay, and now let's do our next chapters or other chapters. Okay, let's move on to the next chapter. Let's start with revising our molecular biology. Okay, it is a huge chapter, but let's revise it in a quick span. So talking about the RNA. Okay, the RNA is your single stranded and 3'5' phosphodiester linkages are present. Base is your adenine. It forms adenosine. Nucleotide is your AMP. Guanine, guanosine, GMP. Cytosine, cytidine, CMP. Uridine, uracil, uridine, UMP. Okay, and RNA polymerases we have three types: RNAP1, RNAP2, and RNAP3. Okay, RNA polymerase 1 we have rRNA, which can form your rRNA, 5s 5.8s RNA, okay, 18s RNA and 28s RNA. RNA polymerase 2 can form your snRNA and mRNA, and RNA polymerase 3 can form your tRNA and 5s RNA. Okay, other RNAs are snRNA, which is used for RNA modification, siRNA for RNA silencing, microRNA for gene expression regulation, okay, and small cytoplasmic RNA for protein targeting. Talking about mRNA, it is 5 to 10% of total RNAs. It is the coding RNA. It carries information from DNA to ribosomes for your protein synthesis. Okay, and this hnRNA, in the presence of your RNA polymerase 2 post-transcriptional modifications, it is converted into your mRNA. Okay, understood. What is the function of your mRNA? So the function is to transport the function of some component like a 5-methylguanosine cap and this poly-A tail. Okay, these are your post-transcriptional modifications. We will discuss about them later. Talking about tRNA, it is called as the adapter molecule or soluble molecule. It is the smallest. Okay, 15 to 30% of the total RNA is your tRNA, and it reads the message of your mRNA. Okay, it is about 75 to 90 nucleotides in length. Okay, structure, secondary structure is clover leaf-like, and the tertiary structure is your L-shaped structure. Okay, and it has 5' to 3'. We can remember data for the arms. Okay, 5' to 3'. So first we have the DHU arm, and the D arm or the DHU arm here. Okay, from starting from 5', so the DHU arm first. Then we have the anticodon arm, okay, which is complementary to the mRNA. Then we have the TCIC arm, okay, thymidine, pseudouridine, cytosine arm, okay, TCIC arm, which is the variable TCIC arm. And then we have the acceptor arm, okay, acceptor arm, and acceptor arm has your sequence ACC in the 3' end. Okay, unusual bases are present like your thymine, dihydrouracil, and hypoxanthine. Okay, and the unusual nucleoside present is your pseudouridine. Unusual nucleoside is your pseudouridine. rRNA is the site of protein synthesis. Okay, we have 70s in prokaryotes and 80s in eukaryotes. 70s we have 50s and 30s. 80s we have 60s and 40s. Okay, 70s the 50s subunit comprises of a 23s rRNA and 5s rRNA and about 34 proteins, and a 30s subunit contains 16s rRNA and about 21 proteins. Okay, and the 80s, the eukaryotic ribosome, okay, has 60s and 40s. The 60s, okay, it has 28s rRNA, 5.8s rRNA and 5s rRNA. Okay, about 50 proteins are there. Okay, and the 40s, it has 18s rRNA and about 35 proteins are there. Talking about DNA synthesis, DNA has a right-handed helix. Okay, antiparallel DNA strands are present. It is called as a Watson-Crick model. Okay, Watson-Crick model. We have the major groups and the minor groups, and about 3.4 nanometer is this length. Okay, and the length is 3.4 nanometer for 10 bases. Okay, the purine are adenine and guanine, pyrimidines are cytosine and thymine. Okay, nucleotides, Discussing about the DNA replication in prokaryotes, so it is bidirectional. Synthesis is in 5' to 3' direction. It is semi-discontinuous and semi-conservative because in one old strand and one new strand is present in each daughter DNA. Okay, and it is single origin of replication. What are the proteins or enzymes required? We have DNA A protein. Okay, we have helicase enzyme, SSB, single strand binding protein, topoisomerases 1 and 2. But the topoisomerase 2 is called as a DNA gyrase, and it is inactivated by a norfloxacin drug, which is a bactericidal drug. Okay, primase, DNA polymerase 3, DNA polymerase P1, okay, DNA polymerase 1, DNA polymerase 3, and your ligase. Okay, so starting with the DNA replication, first there will be AT region, which is called the ORI-C region. This is recognized by the DNA A molecule. Okay, and it causes a local unwinding. So few nucleotides are unwound, forming the replication bubble. Due to the after replication bubble, helicase acts on it. Okay, this helicase will form the replication fork. Okay, and it's open up the strands, forming the fork. And the supercoils, the DNA is like coiled in this manner. They are called the supercoils due to histone packaging. These are removed by a topoisomerase 1 and 2. This topoisomerase 1 removes from single strand and 2 for the double strand, the DNA gyrase. from the double strand. Then we have SSB proteins to stabilize the unwound strands and release the tension. Okay, there is tension due to which the strands tend to coil again. To release it, we have the SSB proteins. Okay, and we have two type of strand. One the leading strand, one is the lagging strand. Okay, leading strand and lagging strand. So in leading strand, just see in diagram, this is your leading strand. Okay, this is synthesis in your leading strand is like continuous synthesis is occurring, and in the lagging strand discontinuous synthesis will occur. Okay, synthesis will occur by joining up some fragments which are called as your Okazaki fragments. These fragments have about 50 to 250 nucleotides in length. Okay, and the synthesis occurs in leading strand and lagging strand in 5' to 3' direction in both the strands, but in leading strand it occurs in your 5' to 3' direction, okay, towards the fork, but in lagging strand it occurs in 5' to 3' direction away from the fork. Okay, away from the fork. Understood? So after this we require your primase. Okay, primase is the DNA dependent RNA polymerase which synthesizes the RNA primer required for the DNA replication, and the DNA polymerase 3, okay, this elongates the RNA primer, okay, and forms the phosphodiester linkage. DNA polymerase 3 has 5' to 3' polymerase and 3' to 5' exonuclease activity. This is special for your DNA polymerase 3, and the DNA polymerase 1 then acts which removes the RNA primer, okay. It has also 5' to 3' polymerase as like your DNA polymerase 3, but it has 5' to 3' exonuclease instead of your 3' to 5'. Okay, and then you have DNA ligase, which is the Nick sealing enzyme and requires ATP. Okay. And the termination of DNA replication occurs on the arrival of TER sequence. Okay, this TER sequence arrive, and the TER-TUS complex result in the termination of DNA replication. We don't need to go into much details. Then let's talk about DNA replication in eukaryotes. Okay, so speed is about 50 nucleotides per second. We have the multiple origin of replications, which are called as your ARS, autosomously replicating sequences, or your replicators. Okay, replicators or replicons, which are recognized by ORS, origin recognition complex, ORS. So ARS are the recognized by the ORS, and this will form your multiple replication bubbles. Okay, then helicase will come to unwind the DNA strand and unwind the DNA strand from the fork. Okay, then we have the RPA, replication protein A. This is special to keep the strands together. Okay, so as to prevent much separation. And then you have topoisomerase 1 and 2. Okay, and they will unwind the supercoils as in the prokaryotic DNA replication. Okay, in the leading strand there will be continuous synthesis, and the lagging strand discontinuous synthesis. Then primase comes to synthesize the RNA primer. Okay, then DNA polymerase alpha acts, okay, which forms a short DNA strand linked to the RNA. Okay, so primase synthesizes the RNA primer, then forming a short DNA strand is done by DNA polymerase alpha. Okay, because in your eukaryotes you have DNA polymerase alpha, beta, gamma, epsilon and delta. Okay, after DNA polymerase alpha forms a short DNA strand, we will see the use of a PCNA, proliferating cell nuclear antigen. Okay, so PCNA generally forms a clamp around. If this is the DNA, the PCNA will form a clamp like this. Okay, this PCNA will form a ring. Okay, this will form a ring, but due to the action of RFC or replicating factor C, okay, this will open up the PCNA ring. Okay, this will open up the PCNA ring like this, due to which the DNA polymerase can attach to here. Okay, so PCNA and replicating factor C, these are utilized in case of your DNA replication. Okay, so PCNA and replication factor C is there. After that, we will require DNA polymerase epsilon, okay, for synthesis in the leading strand, and DNA polymerase delta for the lagging strand. Epsilon for leading and delta for your lagging strand. After that, let's talk about another protein, that is the RNase H1. RNase you know, so it removes your RNA primer. Then we have the flap endonuclease 1. Flap endonuclease 1 removes the last nucleotide of your RNA primer, and DNA ligase will seal the Nick sealing enzyme. Okay, so eukaryotic DNA replication requires this all nucleotides for your energy purposes. Okay, so see, this is the PCNA and the RFC which opens up the PCNA, the DNA polymerase epsilon, okay, and the delta which is attaching, and the delta will cause the DNA polymerization in the lagging strand. Okay, so this is not like your epsilon, this is not your delta, this is your epsilon. This is your DNA polymerase epsilon, because it is causing a DNA polymerization in the leading strand. Okay, for the lagging strand you have DNA polymerase delta. Okay, understood? So this completes your DNA replication. Okay, let's talk about any other things like your telomeres. Okay, let's talk about another concept, that is your telomeres and histones, about histone proteins. Okay, so telomeres, these are short TG recepts. Okay, these are short TG repeats. Okay, and there are 6 nucleotide sequence at the end of the chromosome. It is called your telomere. Like the sequence is TTAGGG. Okay, 5' TTAGGG 3'. Okay, about 1500 base pair in length and maintained by a telomerase enzyme. Function is to correct the end replication error. If there is replication error, it corrects it, prevents loss of DNA molecules, prevent fusion of your two chromosomes. Okay, it prevents the DNA fusion or chromosomal fusion. It protects from the exonuclease activity and gives stability. Okay, telomerase is absent in somatic cell but present in the germ cell and stem cell, and you can see increased activity of telomerase in cancer. We saw that in cancer chapter. Okay, so it is used as a target for chemotherapy. And aging as telomerase, this has like inversely proportional. Okay, due to age the telomerase activity decreases. Okay, length of telomere decrease. Then you have histones. Histones have H1 histone, is least tightly bound. Okay, H2 and H2A, H2B, H3 and H4. Okay, H2A, H2B, H3 and H4 are called as core histones. Okay, and this histone octamer is formed like this, and the two molecules of H2A, H2B, 2 molecules of H2B, 2 molecules of H3, 2 molecules of H4, like this. Okay, and then 1.75, okay, 1.75 turn of your DNA will be there. 1.75 turn of your DNA will be there, and H1 histone is there. H1 histone is also there. H1 histone is called as a linker histone. Okay, H1 histone is called as a linker histone. Okay, and about mitochondrial DNA, this is double stranded circular DNA which has heavy and light chains. Okay, it has about 16,569 base pairs, encodes 13 protein subunits, and forms few untranslated regions, also UTRs. It has a high mutation rate. Okay, it is human mitochondrial DNA, circular in nature. Okay, and transmitted to the next generation by maternal inheritance. Okay, just remember, mitochondria is transmitted from your mother. Okay, so it is called as a maternal inheritance. Okay, so after this telomeres and histones, let's talk about your DNA damage and repair. Let's talk about your DNA damage and repair here. Okay, talking about your DNA damage and repair mechanisms. Okay, talking about DNA damage and repair mechanisms. Okay, so DNA repair is important because the DNA can be damaged. Okay, DNA can be damaged by many causes like free radicals. Okay, and physical agents like UV rays and X-rays, and chemical agents like your drugs, like your toxins, and alkylating agents, your hydrazine and your ozone. This can damage your DNA. Free radicals, physical agents, and your chemical agents. Okay, types of DNA damage. What are the types? We have single base alteration. One base is altered, like your deamination is there, depurination is there, deletion is there, insertion is there. Okay, in which only one base is altered. Then you have two base alterations like your TT dimer formation. Okay, thymine dimer formation is there, and alkylating agents also induce two base alterations. Then you have chain breaks. Chain breaks is due to the ionizing radiations and radioactive damage. Okay, and we have cross linking between bases of the same or opposite strand. Okay, cross linkage is one of the types of DNA damage. Okay, so we have at least 5 repair mechanisms. Okay, one is called as a mismatch repair. So let's discuss the mismatch repair. So assume here you have a DNA damage here. Let me show you. This T is correct, but it is connected to a C, which is incorrect. Okay, so first what happens is that there will be methylation by the methylase enzyme. Okay, the CH3 gets added to this part. Okay, this part, and this due to methylation there will be recognition of this mismatched base by proteins called as MSH and alpha MLH. Okay, by MSH and MLH proteins. Okay, MSH and MLH proteins. This will remove the damage sequence. These proteins like they slide like this, and they come and remove the mismatch sequence. Okay, this is removed. Okay, and from DNA polymerase the sequence is formed, which is correct only. Okay, your with T, A should be there, so it forms the A, new DNA synthesis. And for sealing the nick, we have the DNA ligase acting. Okay, what are the steps? First there will be methylation. Okay, then recognition by MSH and MLH. Okay, then there will be exonuclease activity in which the base will be removed. Then DNA polymerase activity, new base synthesized. Then your DNA ligase, which will seal the nick. Okay, seal the nick. Afterwards, there is if there is faulty mismatch repair, we will get hereditary non-polyposis colon cancer. Okay, hereditary non-polyposis colon cancer, which is also called as a Lynch syndrome. Okay, so the autosomal dominant disorder, and there is mutation in your MLH and MSH proteins. Okay, let's talk about base excision repair. Base excision repair, which is caused due to spontaneous deamination and depurination, like a cytosine is converted into uracil. Okay, so there is spontaneous deamination or depurination. So let's say you have GC, but this C due to some defect, okay, spontaneously, okay, this is converted into U. Okay, this is converted into U. So what happens is first there will be recognition by the uracil DNA glycosidase. Okay, the enzyme used will be uracil DNA glycosidase. Okay, this will recognize your mismatched base. Okay, in the base excision repair, this will excise the base. Okay, this will cleave the base, this will cleave the uracil and form an A basic site. Okay, a site in which there will be no bases. And then this A basic site will also be removed by apurinic or apyrimidinic endonuclease. Okay, this is removed, and after this is removed, there will be synthesis by the DNA polymerase. Okay, the strand will be again formed, the strands will be again formed. Okay, and then a ligase will come, DNA ligase, and this DNA ligase will just join the nick. Okay, it will form the nick, and your GC is now correct. Okay, so there will be recognition by uracil DNA glycosidase, there will be removal by apurinic or apyrimidinic endonuclease, synthesis by your DNA polymerase, ligation by your DNA ligase. Understood? After that, we have the nucleotide excision repair, which is induced by the UV light. Okay, UV light induces your nucleotide excision repair. Okay, like UV light will induce a dimer formation, TT dimer is formed. Okay, this TT dimer will be recognized by your XC nuclease activity, the XC nuclease enzyme, which will bind to the lysine. Okay, XC nuclease will bind to the lysine and remove the segment will be removed, and if the segment is removed, then DNA polymerase will come, will synthesize the correct base. Okay, and there will be ligation by the DNA ligase, same properties. Okay, so enzyme, just remember the enzyme, that is XZ nuclease. Okay, and faulty nucleotide excision repair is called as a xeroderma pigmentosum. Okay, xeroderma pigmentosum. Just a fact, remember that if there is TT dimer formation, okay, the person will be sensitive to the sunlight. Okay, and there will be freckles on the exposed part of the body, like this. The freckles are there in the exposed part of the bodies. Okay, and let's talk about a double strand break repair. If there is the breakage of the 2 strands, okay, in place of 1 strand. Okay, so this may be caused due to damage due to ionizing radiations, your oxidative stress, or your bleomycin drug. Okay, bleomycin drug, and this causes a double strand break repair. Okay, double strand breaks. Okay, not the repair. Okay, so here the 2 strands are broken. This will be first recognized by Ku proteins, Ku proteins. This Ku proteins bind to this ends like this. Okay, then DNA dependent protein kinase will come, which will use ATP and approximate the strands. Okay, this will cause the strands to come together like this. This approximation will favor the activity of helicase. Okay, helicase, and helicase will generate sticky ends here. Helicase will generate the sticky ends here. Okay, and will pair the base, and excess nucleotide will be removed by the helicase, and this DNA ligase will favor the nick sealing. Okay, so this is the complete DNA synthesized. Okay, so here just remember your Ku protein and your DNA dependent protein kinases, and approximation and helicase activity and DNA ligase activity. Okay, so 3 disorders are there due to faulty double strand break repair. We have the ataxia telangiectasia. Okay, ataxia telangiectasia, your Fanconi's anemia, and a Bloom's syndrome. Okay, Bloom's syndrome, Fanconi's anemia, your Bloom's syndrome, and your ataxia telangiectasia. Okay, then we have the direct repair, which is helpful in the repair in the case of pyrimidine dimer if it is formed, or an alkyl eating agent, or your modified bases. Okay, like your guanine, let's say guanine is modified to methyl guanine. Okay, this is a modified base. Okay, methyl guanine. This is the methyl guanine. Okay, how it is repaired? Your methyl transferase, which is active. Okay, this will transfer the methyl group and become inactive. Methyl transferase inactive will remove the methyl group specifically, and this methyl guanine will be converted into your guanine again. Okay, so this is about your DNA damage and repair. Okay, and let's move on to the core topics of molecular biology now, like your transcription. Okay, let's discuss about the transcription prokaryotes first. Okay, so from DNA your RNA is synthesized. This is called as your transcription. Specifically your mRNA, your hnRNA is synthesized. Okay, main enzyme your RNA polymerase. Okay, no primer is required in the prokaryotic your transcription. Okay, let's take the structure of RNA polymerase. So you have a sigma subunit, 2 alpha subunits, okay, a beta subunit, a beta dash subunit, and a epsilon subunit, okay, or an omega subunit. So you have the core enzyme. Okay, core enzyme is composed of beta, beta dash, alpha, alpha, omega, and your sigma factor is there. Sigma factor is the cofactor, okay, which will make the whole holoenzyme. Okay, holoenzyme. Beta subunit has polymerase activity, beta dash has helicase activity, alpha will recognize the DNA to be transcribed into RNA, and your sigma will scan the DNA and recognize the promoter region, okay, which will favor the binding of RNA polymerase to the promoter. Okay, and this beta is inhibited by your rifampicin or your rifampicin. Okay, talking about what are the different sites and boxes. Okay, so we have the +1 site, transcription start site. Okay, then upstream, if you go upstream means in negative direction, and downstream means your positive direction. Okay, if you go upstream in at -10 basing, okay, you will get a Tata box. Okay, the sequence will be TATAAT, and at -35 region you will get a consensus sequence. Okay, which is called the TTGACA. Okay, and we have 2 strands. You know, this is the coding strand, this is a coding strand, and this is your template strand, this is your template strand. Understood? Or your nonsense strand. Okay, and the coding strand and the mRNA sequence are same except in uracil. Okay, if you get a sequence of the coding strand, okay, so the mRNA form will be just similar to the coding strand sequence, but just remember to change all the thymines with your uracil, because in your RNA thymine will not be present, your uracil will be present. Okay, so minus 10 reason is a Tata box or your Pribno box. Okay, just called as your promoter element, and minus 35 will be called as your consensus sequence. Okay, 5' to 3' TT GAC, which is the initial point of contact for the holoenzyme. What are the steps? So first we'll form the pre-initiation complex, then open complex will be formed, then 5 to 10 nucleotides will be synthesized, and then we'll have the promoter clearance. Okay, these are the steps in your initiation. Okay, so sigma factor, this will scan the DNA and recognize the consensus sequence. Okay, the sigma factor will come like this and will first bind to your minus 35 sequence, consensus sequence. Okay, and binding with induce a open, this will form the open complex. Okay, open complex formation. Okay, then it will slide and bind to the Tata box. Okay, Tata box is the site of melting or unwinding, which will just melt about 14 nucleotides. Okay, about 14 bases. Okay, and this will form the formation of a bubble like structure, which will be called as your transcription bubble as will proceed. Okay, so sigma factor will recognize the consensus sequence, binds to it, form the open complex. Afterwards the holoenzyme will slide to the next sequence, the Tata box at minus 10. Okay, and this is the site of melting or unwinding, but 14 base pairs of DNA will unwind, forming an open complex or a transcription bubble. Okay, so there is a formation of your transcription bubble here. Okay, and the RNA polymerase will then synthesize a stretch of RNA, okay, of 5 to 10 nucleotides, okay, which will occur in a 5' to 3' direction. Okay, afterwards the RNA polymerase will move a bit away from the initiation site, and this is called as the promoter clearance. After promoter clearance, this promoter clearance will mark the end of your initiation and starting of your elongation. Okay, so in elongation there will be polymerization. RNA polymerase will start synthesizing a transcript of your DNA strand. Okay, this will form a transcript, and the sigma factors will be released, and core enzyme will move away from the template strand, forming the RNA strand. Okay, so we are just now adding the nucleotides and forming your RNA strand in the elongation process. Then termination, we have the intrinsic termination, or you can have the row dependent termination. Two mechanisms are there. In intrinsic termination, you get a sequence like this: GC, GC, CG, CG. Okay, due to this there is increased affinity between C and G's, okay, which will start forming hydrogen bonds and form the GC rich stem. Okay, this will form the GC rich stem, which is called as a hairpin loop. Okay, this will called as a hairpin loop formation. Okay, and another intrinsic signal is a string of uridines at the string of uracils at the 3' end. Okay, this use. Okay, this will start from binding with the A's. Okay, and this will also prevent your transcription to occur. And the row dependent termination, in which the row protein is there, a row not the row, you have the you have the row. Okay, what I'm telling. Okay, you have the row factor, which will which is a rotating protein. It's a rotating protein. This is not like this. Okay, this is shaped like this. This is a hexameric protein. Okay, this is a hexameric protein, and this row factor is a RNA stimulated ATP dependent helicase. So basically it is a helicase which will just stop your transcription. Okay, by blocking it here, it will rotate and stop your transcription like this. Okay, it is a hexameric protein and separates the DNA RNA hybrid helix, causing the release of your RNA. Clinicals to rifampicin I told, which inhibits transcription initiation by binding to a beta subunit, is used in a TB treatment. Okay, it is an anti-tubercular drug. Now dactinomycin D or actinomycin is an antibiotic drug. Okay, it intercalates between the DNA bases. It remains between your DNA bases like this. Okay, DNA bases, and will inhibit the transcription initiation and elongation steps. Okay, so after this, let's move on to the transcription in your eukaryotes. Talking about the transcription in eukaryotes, it is a bit more complicated than in your prokaryotes. Okay, so we have a fidelity reason. Fidelity is the +1 reason, like we had the transcription start site. Okay, so in eukaryotes it is called as the fidelity. Okay, and RNA polymerase in eukaryotes, it differs. It is of 3 types and differs on the basis of sensitivity. On the basis of your sensitivity to alpha amnitin. On the basis of sensitivity to the alpha amnitin. So we have the RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay, RNA polymerase 1 is insensitive to your alpha amnitin. Alpha amnitin is basically a toxin from your Amanita muscaria muscrum. Okay, and this RNA polymerase 2, this is highly sensitive. Okay, RNA polymerase 3 is intermediate sensitive. Okay, and RNA polymerase 1 will catalyze the formation of your rRNA, 28S rRNA, okay, 18S rRNA, and your 5.8S rRNA. Okay, this RNA polymerase 2 will catalyze the formation of mRNA and your snRNA. Okay, and this RNA polymerase 3 will catalyze the formation of your tRNA and your 5S rRNA. Understood? After this, which are the factors involved? So we can remember A, B, C. Okay, activators, basal transcription factors, and co-regulators. Activators, basal transcription factors, and co-regulators. So activators like SP1, ATF, and CTP. The basal transcription factors like your Tata binding protein TBP, and your transcription factor ABFEH. Okay, and the co-regulators like your TF2D, okay, transcription factor 2D, okay, which comprises of your Tata binding protein and 14, okay, and 14 transcription activating factors. Okay, this Tata binding protein will recognize the Tata box, and this TAF transcription activating factor will bend the DNA. Okay, so the transcription in your eukaryotes occurred in a bent shape of DNA. We'll see now. Okay, so in your prokaryotic, just look at the sequences. Okay, what are there? We have the +1 region as usual. We have in -25 region we have the Tata box or the Hogness box, and then -70 region we have the CAAT box, and we have a GC box about in -100 or -110 region. Okay, so the Tata box or Hogness box, to which the Tata binding protein first binds. Okay, this Tata binding protein has with it present transcription factor 2D. Okay, this Tata binding protein has a transcription factor 2D, and this will bend the DNA like this. This will bend the DNA like this. Okay, afterwards there will be there will be recruitment of your transcription factor 2A. Then A comes. Okay, A comes and binds to here. Then B comes. Then B comes and binds to here. Function of A and B are to stabilize the DNA complex, the bent structure. Okay, they are to stabilize your bent structure. This transcription factor 2B, this binds to your Tata binding protein specifically and recruits transcription factor 2F. Okay, transcription factor 2F. This F is important. This transcription factor 2F is tightly bound to the RNA polymerase. Okay, this is tightly bound to the RNA polymerase. Then it will recruit transcription factor 2E. 2E will bind and recruit transcription factor 2H. Okay, this H has the main function. H has the helicase activity, which we are not discussing. It will phosphorylate. This is important. It will phosphorylate the carboxy terminal domain, CTD, carboxy terminal domain of RNA polymerase. Okay, by phosphorylating the carboxy terminal domain of RNA polymerase, it will activate the RNA polymerase. Okay, so the RNA polymerase having the carboxy terminal, it will phosphorylate it and activate the RNA polymerase. And then you have the nucleotide excision repair. The TF2H will help in the nucleotide excision repair also, which we are not discussing now. Okay, so these are the functions of your TF2H. Okay, and after this, so you now know the sequence of recruitment. First of all, your TF2D is there. Okay, which is bound to your Tata binding protein. Then it will recruit your TF2A. Then it will recruit your B. Okay, then it will recruit your F. After that will recruit the E and your H. Okay, so DAB FEH is the sequence. DAB FEH is the sequence. Then afterwards, all the factors will dissociate except your TF2B. Okay, and this will start the formation of your RNA, and the promoter clearance again occurs like in your prokaryotes. It was there. Okay, then elongation will there, the 3 to 5 phosphodiester linkage formation will be there. Okay, and there will be more addition of your nucleotides will be added more and more. Okay, and for the termination we'll see the use of three proteins specifically. Okay, one is called as a CPSF. Okay, CPSF, this binds to the sequence, okay, which is called as AAUAACA. Okay, this will bind to a sequence AAUAACA. Okay, this is the sequence to which CPSF binds. CPSF is a cleavage and polyadenylation specific factor. And CSF is there, which is the cleavage stimulating factor. This both factors, this will bind to your AAUACA. Okay, AAUACA, and after this they will recruit another factor, which is cleavage factor, CF. Okay, and this CF has the exonuclease activity, which will terminate your transcription in eukaryotes. Okay, so this is the whole process of the transcription in eukaryotes. Okay, next we'll talk about some post-transcriptional modification after this mRNA is synthesized, your hnRNA is synthesized. Okay, from your transcription, okay, you synthesize your hnRNA. Okay, hnRNA, but we require your mRNA, and for this mRNA we require post-transcriptional modifications. Okay, let's discuss the post-transcriptional modifications now. Discussing about the post-transcriptional modifications, we have 5' capping. Okay, it is the attachment of the 7-methyl GTP cap. Okay, 7-mg cap. Okay, not the 7-methyl guanosine GTP cap, specifically 7-methyl guanosine GTP cap to the 5' end of the mRNA. It is called as a capping. It is attached by the triphosphate linkage. So 7-mg cap is attached at the 5' end. This is called as your 5' capping. It will prevent your RNA from 5' exonuclease activity. It will give stability to the mRNA. It will help in its translocation from nucleus to cytoplasm, and it will help in the binding of mRNA to the ribosome also. Okay, then you have the poly-A tail, 3' poly-A tail formation. Okay, the enzyme used will be polyadenylate polymerase enzyme, okay, which will join your about 350 to 250 nucleotides. Okay, you are having poly-A tail. Okay, AAA are there. Okay, and it starts after the intrinsic signal AAUAA. Okay, once you get the intrinsic signal AAUAA, and the polyadenylate polymerase enzyme will start the synthesis of a poly-A tail. Okay, This what is the importance? It will prevent the activity of your 3' exonuclease. It will also give stability and helps in binding of the PAB protein. Okay, this will help in the binding of the PAB protein, which you will see in your translation procedure. Okay, and then we have the splicing process, which is the joining of consecutive exons. Okay, the exon 1 and exon 2 are there. Okay, introns should be removed. Okay, introns are the non-expressing sequences. This should be removed, and the exons should be joined with one another. Okay, so for splicing, this is called as splicing. Okay, we have two mechanisms. We have intron-mediated splicing and your spliceosome-mediated splicing. Okay. For intron mediated splicing, which do not require ATP or protein or factors, okay, first what will happen will there will be a recognition of specific sequences, then there will be a 5' cut, okay, and the loop formation will be there, then the 3' cut and the joining of the exons. Okay, so let's say we have the 5' cap here and the poly A tail here, 3' poly A tail here. Okay, and this is the exon 1, this is the exon 1, this is the exon 2. Okay, we have the 5' splice site here and the 3' splice site here. Okay, and the branch point where adenine is present. Okay, this adenine will attack upon the 5' splice site. Okay, 5' splice site, and this will turn like this, the intron will turn like this. Okay, and then this 3' OH will again attack. Okay, this is called as a 3' attack. This 3' will again attack on this site. Okay, and this will cause the removal of your this bent structure, that is your lariat. This is called as a lariat. Okay, and by this your exon 1 and your exon 2, these are both joined to each other. Okay, this is called as the intron mediated splicing. Then we have the spliceosome mediated splicing, which will require 5 snRNAs, 5 small nuclear RNAs designated from 1 to 5, U1, U2, U3, U4, U5. Okay, and about 60 proteins. Okay, about 60 proteins, and this will form the SNRPs, small nuclear ribonuclear proteins. Small nuclear ribonuclear proteins, this is called as your SNRPs. Okay, what are SNRPs? About 5 snRNAs and about 60 proteins. Okay, what happens is that to the 5' splicing site, the U1 is attached, U1 snRNA is attached, and to the branch point U2 is attached. Okay, U1 is attached, and afterwards U2 is attached. This will cause the formation of inactive complexes of U4, U5 and U6, okay, due to which your U1 and U4 are removed. Okay, and there will be loop formation around the complex, which is called as a lariat. Okay, the lariat is removed and the 2 exons are now joined. Okay, and we have another post-transcriptional modification called as RNA editing. Okay, which we will discuss in your eukaryotic gene regulation and gene control. Okay, so after post-transcriptional modifications, let's move on to our topic, which is called as your translation. Okay, let's move on to the prokaryotic translation. Talking about translation in prokaryotes, so basically we have our mRNA now after post transcriptional modifications. Okay, and we have codons now, which would be coded into your amino acids, different amino acids. Okay, and so requirements are your mRNA, it should be there, tRNA, which is the carrier for the amino acids, ribosomes, like 70S ribosomes in the prokaryotes, and amino acids. Okay, so 70S have 50S and 30S. We know 50S about 23S and 5S and about 34 proteins. This 30S have only one 16S rRNA and about 21 proteins. Okay, what are the factors required for initiation? Is the initiation factor 1 to 6. For elongation, we have elongation factor Tu and elongation factor G. These both are there, but the elongation factor G will provide the energy for your translocation. We'll see. Then for termination, we have release factors 1, 2 and 3. So what are the steps? First, there will be a charging of tRNA. Where the amino acid should be activated. So amino acid combines with the ATP in the presence of amino acyl tRNA synthetase enzyme, forming your enzyme AMP AA complex, which is called as your activated amino acid. ENZ AMP AA complex, which is called as your activated amino acid. And again, this reacts with the tRNA. Okay, tRNA, and forms tRNA. tRNA has a CCA end, I told, at the 3' end. So CCA, to which your OH of amino acid is bound. Okay, so this forms your tRNA CCA OH AA. Okay, which is your charged tRNA. Okay, this is called as your activation of your tRNA. Okay, so we activated our amino acid, we activated our tRNA. Okay, and formed the charged tRNA. After that, we'll see initiation. So first what happens, there will be a ribosomal dissociation. The 70S ribosomes breaks down into 50S and 30S. Okay, and initiation factor 6, IF6, is responsible for dissociation of your 70S. Okay, then the 30S. Okay, then the 30S, this combines with the initiation factor 1 and your initiation factor 3. Okay, 30S combines with the initiation factor 1 and initiation factor 3. Okay, then there will be the formation of your ternary complex. So ternary complex is formed from your N formyl methionine tRNA, okay, and formyl methionine tRNA carrying the N-formyl methionine as it is equivalent to the methionine, okay, in your eukaryotes, okay, but in prokaryotes N-formyl methionine is present over here, and the initiation factor 2 is there, and your GTP is there, which will form the ternary complex, okay. And then there will be formation of 30S initiation complex. So what happens, the ternary complex attaches to the mRNA and the 30S subunit, okay, which are combined with the IF1 and IF3 to form the 30S initiation complex. So here you will see one mRNA is there, okay, having the your N5 formyl methionine tRNA, okay, and your initiation factor 1 which was attached to it, initiation factor 3, and your initiation factor 2 also, which was in the ternary complex, and a GTP must be bound to it, which I have not run. This is a GTP, okay. Then there will formation of the 70S initiation complex, okay, 70S. So 30S initiation complex combines with the 50S, forming the, in the presence of initiation factor 5, forming a 70S initiation complex, okay. Okay, so here we can see the creation of three sites. One is the A site or your acceptor site, and the P site or the peptidyl transferase site, and the E site which is called as your exonuclease site, okay, not the exonuclease, the exist site, okay, not the exonuclease site, just note. Then we have elongation factors EFTU and EFG catalyze them, okay, where there is the attachment of tRNA to the A site first, okay, the tRNA comes and attaches to the A site, okay, and attachment of tRNA to A site, after that the mRNA will move away. This is called as a translocation. Energy is provided by EFG, okay. Afterwards again another amino acid comes to the A site, okay. When another amino acid comes, this also moves a little away, okay, and this amino acid moves to your P site, and this P site amino acid, first one, will move to your E site and be exited, okay, and again when another amino acid comes, okay, this will move to your P site, and the proteins will be just go on adding, okay, the amino acids just go on like adding in the P site, okay, due to the peptidyl transferase enzyme, okay, there will be the catalyzing the formation of your peptide bonds, okay, and translocation is occur and peptide bond formation is going on, okay. There are many peptide bonds, okay, so we need our termination now. On the arrival of stop codons, what are stop codons? Your UAA, UAG and UGA, these are three are your stop codons, okay, and due to this arrival of a stop codons here, UAA, okay, your release factor will bind. Release factor, just note that release factor is the analog of your tRNA, okay. This release factor will catalyze the hydrolysis of this amino acid chain, okay, and the protein will be released outwards, okay. This protein will be folded by proteins called as chaperones, okay. Chaperones, these are proteins which will fold the protein that is released, okay. Then we have inhibitors of protein synthesis in prokaryotes. First one is a tetracycline, which will bind to a 30S subunit and prevent the attachment of the first amino acid tRNA to the A site, okay. So it will inhibit your initiation step, okay. Tetracycline is usually a broad spectrum antibiotic, okay. Example is the doxycycline is the example of your tetracycline group of drugs, okay. Then we have chloramphenicol, which will bind to your 50S and will inactivate inhibit the activity of a peptidyl transferase enzyme, okay. This is the antibiotic for enteric fever, typhoid fever and meningitis, okay. So this chloramphenicol will block the peptide bond formation. Then we have erythromycin is used in upper as well as lower respiratory tract infections. This also binds to your 50S and will prevent the attachment of tRNA to the P site, okay. The first one, the tetracycline, was preventing the attachment to the A site, but here the erythromycin, which will bind to the 50S and will prevent the attachment of tRNA to the P site, okay, to the P site. Then we have aminoglycosides. So for example, you have streptomycin for TB treatment, gentamicin, neomycin, amikacin. These are your aminoglycosides which will bind to a 30S, okay, and will inhibit the formation of your initiation complex, okay, and will misread the mRNA and will also inhibit your translocation, okay. This act as a variety of steps, okay. So this completes your 4 drugs: tetracycline, chloramphenicol, erythromycin and aminoglycosides, okay. With this we complete our translation process in prokaryotes, okay. Let's see the transcription process in eukaryotes afterwards, okay. Let's start the translation in your eukaryotes. Okay, so translation in eukaryotes, the first step is your tRNA activation, okay, the same step as in your prokaryotes. Then there will be initiation, okay. In initiation, the extra step is your mRNA activation, okay. mRNA is then activated due to the binding of your 4F and 4B and your some proteins are there like your PAB, okay. This we'll see how the mRNA is activated. Then there will be formation of a ternary complex, ribosomal dissociation, 43S pre-initiation complex formation, 43S PIC formation, then 48S initiation complex formation, and your 80S initiation complex formation. Okay, this comprise of your initiation process in your translation in eukaryotes. Okay, for elongation we will see the same things, tRNA attachment to the A site, peptide bond formation, and your translocation. Okay, this is a translocation, not a translation. Then there will be termination. So first, activation of tRNA. So amino acid in the presence of ATP and amino acyl tRNA synthetase, your amino acyl tRNA synthetase enzyme forms your activated amino acid enzyme AMP-AA complex. This enzyme AMP-AA complex in the presence of tRNA reacts with the tRNA, okay, and forms a tRNA CCA-OH amino acid activated tRNA. Okay, same step as prokaryotic one. Then we have the initiation of translation. Okay, then first we have the mRNA activation. Okay, so let's say here you have the mRNA, here you have the AUG codon, this is the start codon. Okay, so first ATP, with the help of ATP, the 4F subunit binds to the 5' end. The 4F subunit binds to the 5' end. This 4F subunit comprises of your 4G subunit, 4A subunit, and your 4E subunit. Okay, it comprises of your 4G, 4A, and 4E. This 4A subunit, this will remove the secondary structure of mRNA. Okay, 4E subunit will help in the recognition of your cap. Okay, cap recognition. Okay. So what happens after this all? Okay, 4F binds, then there will be recruitment of your 4B and PAB. What is PAB? Or poly A binding protein, which will bind to the AAA. Okay, we talked about this in the post-transcriptional modifications. Okay, the AAA, the tailing was favoring the binding of your PAB. Okay, so 4F is bound and PAB is now bound. Okay, 4B is now bound. Okay, this 4B will recognize the AUG sequence, okay, and start forming a small RNA, which is called as a 18S rRNA. Okay, this sequence between your 4F and your 4B, this sequence is called as a Kozak consensus sequence. Okay, in prokaryotes we have the Sine del Carnot sequence between your this subunits. Okay, so we have the Kozak consensus sequence, whose sequence is complementary to the 18S ribosomal subunit. Okay, and this PAB, what is the utility? So it recruits the 40S subunit in the ribosome to the mRNA. Okay, it will recruit the 40S subunit. Let's see. Then there will be ribosomal dissociation, the 80S ribosome in the presence of your eukaryotic initiation factor 6. Okay, in prokaryotes there was initiation factor 6 in prokaryotes. In eukaryotes we have eukaryotic initiation factors, just like write EIF. Okay, EIF 6. Okay, and what this will cause? This will dissociate the ribosome into 40S and 60S. Okay, 40S comprises of your 18S and about 34 proteins, 60S about 5S, 5.8S, 28S, and about 50 proteins. Okay, after that there will be formation of the ternary complex, again methionine tRNA. Okay, the tRNA carrying methionine amino acid, EIF2 and your GTP, these are forming a ternary complex in eukaryotes. Okay, then the 40S subunit will be recruited by your PAB. Okay, this poly A binding protein will recruit your 40S subunit, and this 40S will bind with the eukaryotic initiation factor 1 and 3. Okay, like in your prokaryotes it was happening. Okay, in your prokaryotes it was happening, your 30S was binding with your initiation factors 1 and 3. Okay, and the ternary complex then attaches. Okay, forming a 43S pre-initiation complex. 43S pre-initiation complex. This will have your methionine tRNA. Okay, and your 40S subunit, your eukaryotic initiation factor 1, 3, and the 2 of the ternary complex. This will have a GTP attached to it. Okay, then there will be formation of your 43S initiation complex. This 43S, not the 43S, this is the 48S. Sorry, I have made a mistake here. This is the 48S. Okay, this is the 48S. The 48S initiation complex. This 43S pre-initiation complex, which is formed pre-initiation complex. Okay, this will react with your mRNA. This will combine with your mRNA and form the 48S initiation complex. Okay, here we have the 5', 3' and methionine tRNA, your 40S subunit, your 1, 3 and eukaryotic initiation factor 2 having the GTP bound to it. Okay, then we'll have the active 80S initiation complex formation. Well, all the subunits, all the initiation factors dissolve will dissociate. Okay, so eukaryotic initiation factor 5 will catalyze the formation of your active 80S initiation complex, in which the large ribosomal subunit will bind to your 48S initiation complex, forming the 80S initiation complex. Okay, and EIF5 will facilitate the attachment of 40S and 60S subunits, and all the other factors will dissociate. Okay, Then the elongation step, is similar, just your attachment of tRNA to the A site is catalyzed by your EEF1. This will provide the energy, GTP also, and the peptide bond formation is catalyzed by your EEF2 and your GTP. Okay, this will provide the energy. EEF2 is inhibited by your diphtheria toxin, just remember. Okay, the same process, the same translocation will occur, 1 amino acids will be added. Okay. And here we have the for termination we have release factor 1, which will bind your release factor 3, and this both complex release factor 1 and 3 will hydrolyze your peptide bond. Okay, hydrolyze your peptide bond. These are your differences between the eukaryotic and prokaryotic translation, which you can take a screenshot. Okay, done. Okay, let's move ahead. So first, and then we have to discuss the inhibitors of protein synthesis in your eukaryotes. Okay, so you have the diphtheria toxin, which will inhibit your eukaryotic elongation factor 2. Okay, by Corynebacterium diphtheri, which causes your nasopharyngeal infections. Okay, it causes your ADP ribosylation of your EEF2 and inactivates it. Okay, so EEF2 is required for your translocation, so it inactivates your EEF2 and prevent your translation procedure. And your ricin is there. Ricin binds to a 60S ribosomal subunit. It is a protein from your castor beans. Okay, and removes the adenine nucleotide from the 28S rRNA. Okay, 60S has your 5.8S, 5S and 28S. Yes, so it removes the adenine from your 28S rRNA and it blocks the translation. Okay, then your cycloheximide will bind to a 60S. Okay, and inhibit your peptidyl transferase activity, like in your prokaryotes. Okay, which was inhibiting your peptidyl transferase activity, your chloramphenicol. Okay, here you have the cycloheximide, which will be inhibiting your peptidyl transferase activity by binding to the 60S subunit of ribosome. Okay, so in this all your eukaryotic translation inhibited by your binding to the 60S ribosomal subunit, mainly 60S. Okay, then you have the puromycin. Puromycin is a structural analog of your tyrosinyl tRNA, structural analog. Okay, it causes a premature termination of your protein synthesis. It terminates the protein synthesis prematurely. Okay, so puromycin is special because it is blocking your translation in eukaryotes as well as your prokaryotes. Okay, so afterwards let's talk about the post-translational modification. So talking about post-translational modifications, we have the proteolysis and your covalent modifications. Okay, proteolysis is we have the 3 types: removal of your initiator methionine. Okay, activation of proteins, like in your digestive system you might have seen activation of trypsinogen into trypsin. Okay, and chymotrypsinogen into chymotrypsin. Okay, this is catalyzed with enterokinase, proelastase to elastase, pepsinogen to pepsin and all. Then we have the cleavage of your signal peptide. The signal peptide is cleaved. Okay, signal sequence, we saw in the protein targeting how the signal sequence was cleaved by the signal peptidases linked to your translocon channels. Okay, then we saw then we have the covalent modification. Okay, we have proteolysis and covalent modifications. In covalent modifications, we can see the addition of carbohydrates. Okay, addition of prosthetic groups like your biotin and heme, phosphorylation, dephosphorylation. Okay, your SS bond formation as in insulin. Your OH group is added. Okay, formation of hydroxyproline and hydroxylysine. The formation of hydroxyproline and hydroxylysine, OH group is added. Your gamma carboxylation for vitamin K and osteocalcin, gamma carboxylation is there. And your acetylation, your isoprenylation in cholesterol synthesis, isoprenylation is there. Okay, and the activation of P21 RAS protein, also isoprenylation is there. So addition of carbohydrates, talking about it a little later, a little bit more. So Golgi, the cis Golgi removes the mannose. The medial part of Golgi is adding a fucose and N-acetylglucosamine. And the trans Golgi is catalyzing your galactose and sialic acid addition. That's your galactose and sialic acid. Just remember this all. So these are your mug of fats only. And addition of carbohydrate increases the antigenicity of the protein. Okay, and also helping the cell to cell recognition. Enzymes which are activated by phosphorylation, dephosphorylation. So phosphorylation, many enzymes are there, dephosphorylation, many enzymes are there. I'm giving one example, your glycogen phosphate, not the phosphate, this is a phosphorylase, glycogen phosphorylase, okay, which will be activated by your phosphorylation. And your glycogen synthase, we'll see in the glycogen metabolism today only. Okay, this is the example of a dephosphorylation activation. Okay, this is activated by dephosphorylated state. Understood? So after post-translational modifications, let's discuss the regulation of your gene expression in your prokaryotes, and let's talk about an important concept, which is your lac operon concept. Okay, let's talk about this. Okay guys, talking about gene regulation, we have positive regulation and your negative regulation. Positive regulation is the increasing of gene expression, negative regulation is the decreasing of your gene expression. Okay, then we have positive regulators which are called as enhancer or activators. Okay, and negative regulators are called as silencers or repressors. Okay, we have two types of genes, your housekeeping genes and your regulatory genes. Okay, the housekeeping genes and regulatory genes. Housekeeping genes, they express at a constant rate to maintain the basic cellular functions. They express at a constant rate. Okay, these are also called as your constitutive genes. Constitutive genes. Okay, also called as your constitutive genes. Okay, yeah. And we have the regulatory genes. We have the regulatory or regulated genes which express only in certain conditions. Okay, we have the tissue specific expression, like the certain gene may be expressed in a particular cell. Okay, in particular cell, this tissue specific expression is seen in your eukaryotes mainly. Okay, then the site of control of gene expression, at which levels it is controlled. Okay, in prokaryotes it is controlled in a transcription level only, but in eukaryotes it may be controlled in a transcription, your post transcriptional level, your post translational level, also at your DNA level. Okay, all of this, but transcription level is the most common site of control of your gene expression in both prokaryotes and eukaryotes. Okay, then you have some regulatory sequence and molecules like a cis acting substance. Okay, these are your regulatory DNA sequences. Cis acting sequences are your regulatory DNA sequences, okay, to which your trans acting factors bind to which your trans acting factors, which are your regulatory molecules, this bind to your cis acting sequences. Okay, so this is a trans acting factor here. Look at the diagram from your Lippincott textbook. Okay, this will bind to a cis acting element, and this binding will facilitate the action of your RNA polymerase. This will facilitate the action of your RNA polymerase, and transcription will go on in this direction. Okay, this is a simple thing. Okay, don't get confused. Then the binding of proteins to DNA is through structural motifs. There are about 3 to 4 structural motifs. Okay, your Lippincott says that there are 3 motifs. Okay, but in other textbooks it are given 4 motifs. The zinc finger motif. For example, a binding of transcription factor 3A to the 5S RNA. Okay, 5S RNA, we haven't seen the transcription of your 5S RNA. Okay, because this is not in our syllabus. Okay, so zinc finger motifs promotes the binding of your transcription factor 3A to the 5S RNA. Okay, your transcription factor 2A to the DNA. Okay, to the DNA. And a leucine zipper motif, this is seen in your Foss and June regulatory proteins, how they are bound to the DNA. In the helix turn helix motif, how the CRO protein is binding to the DNA via its alpha 3 helix. Okay, this is the example. CRO protein is responsible for the lysogenic and the lytic cycles. Okay, we'll see it in your molecular switching in your bacteriophage lambda, which is also a control of gene expression in your prokaryotes. Okay, after this, let's talk about the lactose operon or lac operon. You must have read the lac operon in your smaller classes also in your 12th standard. So lac operon, your lactose operon, and operon means a linear array of genes involved in a metabolic pathway. Okay, this was described by your Jacob and Monod. It contains 3 genes which code for 3 proteins. Okay, 3 proteins. Your lac Z gene, okay, lac Y gene, and your lac A gene. Okay, 3 genes are there. Lac Z gene codes for your beta galactosidase, which converts your lactose to galactose. Or glucose. Okay, lac Y gene codes for permease, which will increase the permeability of cell for the lactose, and lac A gene is catalyzing your translation of your thiogalactose transacetylase enzyme. Okay, this enzyme which will acetylates the lactose. This we don't know even the exact function of this acetylation procedure or the nature as written in your Lippincott. Okay, then let's talk about this the linear array about the operon. Okay, so we have a lac I gene, which is the repressor gene. This is a constitutive gene. It expresses at all times. Then we have a cap site here. We have a P region here, which is the promoter region where RNA polymerase binds. We have a O site or the operator site. We have a lac Z gene. We have a lac Y gene and a lac A gene. Z, Y, A are arranged in this sequence. Okay, lac Z will synthesize one mRNA, lac Y another mRNA, lac A another mRNA. Okay, the mRNA will called as the polycistronic mRNA, and the cistron is the smallest unit of gene expression. Your cap region is catabolite activator protein, sometimes called as a CRP, your camp regulatory protein also. Okay, cap region is a catabolite activator protein, also called as a CRP, camp regulatory protein. Okay, we'll see the function of your cap. Okay, when only glucose is present, we will take 3 cases. When glucose is present, lactose is absent. Okay, and another case will be when lactose is present but glucose is absent. Okay, and another case will be when both are present. Okay, only when glucose is present, lactose is absent, the bacteria will obviously use glucose, will prefer glucose over the lactose. Okay, so we don't need the lac operon. This is switched off. Operon is repressed. It's called a negative regulation. How it occurs? Okay, so how the negative regulation is occurring, just see what happened here. Yeah, how the negative regulation is occurring. The lac I will constitutive gene, so express it, transcription will occur, RNA is formed, repressor protein will be formed. This will be active due to absence of lactose. Repressor tetramer will be formed. This tetramer will bind to your O region. This will prevent your RNA polymerase to be bound to the P region, and due to this not binding of RNA polymerase, the genes won't be transcribed. Okay, genes won't be transcribed. Okay, and this will prevent the transcription of the structural genes, and the proteins beta galactosidase, permease, and thiogalactose transacetylase will not be formed. Okay, and here if glucose is present, in the presence of glucose, your adenyl cyclase is inactive in the presence of glucose. Okay, so cap protein remains in unbound state in the plasma. Okay, cap protein remains in unbound state. Okay, it doesn't bound to the capsid, doesn't bind to the capsid, and this unbound cap protein is called as a catabolite repressor. Okay, repression of your cap protein is called as your repression of your adenyl cyclase is called as a catabolite repression. Okay, only when lactose is available. Okay, when only lactose is present, the bacteria will obviously use lactose now, because it will have preferred glucose, but glucose is absent now. Okay, only lactose is available, so we need the operon. So operon will be induced. It will be induced. It is called as a positive regulation, as the operon is induced. Okay, what happens? The lac-I gene will synthesize your mRNA. This mRNA will form repressor protein, but in the presence of lactose, the repressor protein are bound to the lactose and they can't bind to the O region. Okay, this if they can't bind, your RNA polymerase can easily bind to your P region. Okay, P region. Just understand, if the repressor protein are already bound to the lactose, they can't bind to the O region. This will favor the binding of your RNA polymerase to the P site, and if RNA polymerase is bind, obviously the transcription will start occurring. Okay, the polycistronic mRNA is produced, and this will form your beta galactosidase, permease, and transacetylase. Understood? As glucose is absent, your adenyl cyclase is active. Okay, adenyl cyclase is active. Okay, so CAMP binds to your cap, or your CRP. Okay, and this CAMP and cap complex binds to your cap site. Understood? So this is the whole process if only lactose is available. And let's talk if both glucose and lactose both are available. Okay, so bacteria will prefer the glucose in place of lactose. Okay, both are available, bacteria will prefer the glucose, so operon will be off, like the first case. Okay, and catabolite repression will be also seen. Understood? Then the operon is there, which is a tryptophan operon. It has 5 structural genes about TRPE, TRPD, TRPC, TRPB, and the TRPA. These are 5 genes. Okay, 5 structural genes. Okay, and you can see it in E. coli bacterium, where the chromosome for E. coli bacterium. Okay, and TRP operon is subject to negative control. How it is controlled? If the TRP is plenty, okay, the bacteria won't need TRP. Okay, TRP is plenty. So operon is switched off by the formation of an attenuator. Okay, the mechanism by which the TRP operon works is called as your attenuation. What is attenuation? Attenuation is the formation of a hairpin structure in your RNA, like you saw your GC stem in your termination. So attenuation is also like formation of a hairpin structure like this in your RNA. Okay, and due to this attenuator, due to this formation of this hairpin loop like structure, your RNA polymerase can't go to the next site. Okay, and transcription won't occur. So there is no transcription, and as TRP is plenty, there is the formation of your attenuator, and attenuator will now prevent your transcription process. Okay, and these genes are not transcribed. Okay, but when TRP is scarce, the operon is switched on. Okay, and the lack of TRP, due to the lack of TRP, the ribosomes stall at the 2 TRP codons present at the 5' end. So in the 5' end, okay, in the 5' end of your DNA, the 5' end, okay, there are 2 TRP codons. There are codons coding for the TRP, 2 TRP codons, where the ribosomes are stalling here. The ribosomes can't move here. Okay, and this stalling of ribosomes will cover the region for attenuation. Okay, so attenuation won't occur. Okay, so these ribosomes are basically covering the region of your DNA, which is covering the region of your, not the DNA, mRNA, which is required for your attenuation, and this will prevent your attenuation, and transcription will continue as usual. A genetic switch in bacteriophage lambda, which is done with CRO protein. Okay, bacteriophage lambda has a lytic cycle and a lysogenic cycle. This is not in your course, but I am just explaining that it is given in a Lippincott textbook. So teachers may ask you in 5 also. Okay, or question may come in worst cases and worst cases. Okay, so in unfavorable conditions, what happens when the nutrients and sunlight are not available? Okay, the gene for repressor produces an mRNA, which produces repressor protein, and this reversibly bind to the ORI1 site, which is a CRO promoter region, and this binding to the ORI1 site will inactivate your gene for CRO, and this gene for crow is inactivated, no crow protein. Okay, so bacteria will lie dormant because the bacteria requires crow protein for synthesizing its viral machinery. Okay, so bacteria will lie dormant, and this dormant phase is called as a lysogenic phase. And if nutrients and sunlight are available, there will be no gene for repressor won't code. It is inactive, it is inactivated. Okay, so crow protein can easily be synthesized, which is required for your replication, your lytic phase. Okay, lytic phase. So bacteria can alter its phases according to your climate by altering the crow protein, and by doing like this, the crow synthesizing and inactivating the gene coding for the crow proteins. Okay, after this, let's talk about the control of gene expression in your eukaryotes. Okay, let's talk about this. Talking about the regulation of gene expression in eukaryotes, it is regulated at various levels. Like at the DNA levels, you can have modifications like epigenetic modifications. Okay, epigenetic modifications, which comprises of acetylation and methylation of histones. Okay, acetylation and methylation of histones. So what happens is that if there is acetylation of lysine H tails of the histones, okay, lysine H tails of the histones, so there will be decreased positive charges, and this decreased positive charges will decrease the interaction between your histones and DNA. And okay, and decreased interaction, so DNA will not be available for your transcription. Okay, DNA will be DNA will be available for your transcription as histones and DNA have decreased interaction. Okay, and this will increase your genetic expression. Sorry, okay, so DNA is available for transcription. Okay, this will increase your gene expression. Acetylation of lysine H tails will increase your gene expression. Then another epigenetic modification, that is a methylation of histones, which will decrease your gene expression. Okay, which will decrease your gene expression. Okay, methyl group addition in histones decreases gene expression. Then you have a process called as gene amplification and gene rearrangement. Let's talk about them. So see the gene amplification, it is the production of copies of gene. Okay, let's take the gene for the DHFR, okay, dihydrofolate reductase enzyme. Okay, and we have A and B regions here. We have A and B, we have A, and here we have B. Okay, so gene amplification in A and B, like this, it occurs like this. Like there are many copies of genes produced here, like this, many copies of genes are produced here. So this DHFR is basically inhibited by a methotrexate drug. This is a clinical here. Okay, which converts the fatty acid to the dihydrofolate and dihydrofolate to the trihydrofolate. Okay, so tetrahydrofolate. Okay, not the tetrahydrofolate. This is done by DHFR, the dihydrofolate reductase. Understood? This is called as a gene amplification. Now gene rearrangement, so it is seen in your immunity parts. Okay, let's say this is your IgG. Okay, your antibody. Okay, so you have a variable region and a constant region in your antibodies. Okay, so you have about 300 genes, okay, coding for your variable regions from V1 to V300. Okay, and 5 genes coding for the J region, that is hinge region. Okay, J region is a hinge. Okay, this is your hinge region present. J is your hinge region, and you have 5 genes coding for the hinge region also, 5 genes coding for your hinge region also. Okay, and again you have your D region. Okay, for your D region there are about 10 genes coding for your D region. Okay, so you can have various combinations like a V2 J3 D3. Okay, this is one rearrangement which is occurred, and V5 J3 D1, this is another combination. Okay, different combinations are producing different light chains. Okay, this is called as a gene rearrangement. So I hope you understood the gene rearrangement. Okay, let's talk about other levels. So at the DNA level you have epigenetic and your gene amplification and rearrangement. Okay, at the second level, this is the transcription level, which is the main level of regulation of gene expression in eukaryotes. Okay, you have promoter, enhancer. Okay, and you have transcription factors and DNA binding motifs. Okay, to regulate your transcription and regulate gene expression at the transcription level. Okay, so talking about this, we don't have to discuss much. We have regulatory elements like enhancers and depressors are there, you know. Okay, your TTA AGG region, your -25 region, we have talked about this. Okay, promoters like your Tata box, your INR is called as an initiator element. Okay, and DP is again there. DP is the downstream promoter element. These are promoters. Okay, Tata box, your INR, and your DPE, these are the promoters. And then the proximal promoters like your CAAT and your GC. Okay, CAAT and your GC, these are called your proximal promoters. Okay, and you know about the transcription factors. We talked about a lot in your transcription procedure in your eukaryotes. Okay, what are this? Like your Tata binding protein, this recruits your D, it recruits your A, it recruits your B, F, E, H. Okay, we talked about the sequence DAB, FEH. Okay, and DNA binding motifs, we talked about DNA binding motifs also, like a zinc finger motif. Okay, your leucine zipper motif, your helix turn helix motif, and another motif is written in few books, your helix loop helix motif. Okay, these are attaching a transcription factors to the DNA, And talking about regulation at the post-transcriptional level, we talked about your splicing. Okay, we discussed two splicing, that is intrinsic splicing and a spliceosome-mediated splicing. Okay, here we'll talk about another two splicing, which is selective splicing and alternate splicing. Okay, selective splicing is just your common splicing. Exons, introns are removed, okay, selectively, and these exons are added. Okay, exons are added. Selective splicing is there. Okay, and in the alternate splicing, what happens? You have 5' or 3' splicing. Like every exon is a 5' or 3' region. Okay, so it can produce different combinations. Like the 5' of one will be connected with the the 5' of another will be the 5' of another. Okay, this will form another combination, and this 3', this if they combine, this will form another combination. So they produce different mRNAs. Okay, understood. So then we will talk about a concept called as the mRNA editing. So the famous example is the ApoB48 and ApoB100. Okay, ApoB48 will be produced by the intestine, where the protein synthesis has stopped at the 48%. Protein synthesis has stopped at 48%. In ApoB100, 100% protein synthesis has been occurred. Okay, why it stops? Because the CAA converts into UAA. Okay, the CAA encounters, the CAA is like going on, it is produced by the CAA. Okay, but at the 666th position, of your chain, okay, this encounters the UAA. This encounters your UAA, and the protein synthesis will stop at your 48%. Okay, and ApoB100, there is whole protein synthesis occurring. Okay, talking about another concept, which is mRNA stability. So in iron deficiency anemia, what happens? We will see some production of this hairpin loop like structures, which are called as your iron responsive elements. These iron responsive elements, okay, these iron responsive elements, if there is decreased iron, okay, then they will stimulate the IRP, the iron responsive proteins. Okay, the IRP, iron responsive proteins, to bind to the iron responsive element. Okay, this IRPs bind to the iron responsive element, and this binding will increase the stability of your mRNA, and due to the increased stability of mRNA, your transferrin receptor synthesis is increased. This transferrin receptors, it binds the iron. Okay, so transferrin receptors will bind more and more iron, and your iron production in body again goes up to the normal level. Okay, so this is seen in iron deficiency anemia. Okay, this is the body's intrinsic mechanism, the mRNA stability, which will increase the transferrin receptor synthesis. Okay, so we talked about a few things. Okay, let's go to the levels back. Okay, so we talked about at the transcription level, we talked about promoter and the transcription factors, DNA binding motifs, and we talked about at the post transcriptional level, we talked about splicing, your alternate splicing, your selective splicing, your okay, then mRNA editing and your stability. Okay, at the translation level, we have the eukaryotic initiation factor 2 alpha and the eukaryotic initiation factor 4e. We have talked about this in your translation procedure. Okay, and then we have the stability at the gene expression regulation at the post translational modification, post translational level also. Okay, so okay, let's see what we have. Okay, so EIF2 at the translational level, we have EIF2. Okay, which if phosphorylated will inhibit your initiation process. This is important for initiation. And your eukaryotic initiation factor 4e, okay, it recognizes your cap on mRNA. We have talked about this, and if this is phosphorylated, it becomes active. Okay, so here it is also example of your, and this is regulated by a phosphorylation, dephosphorylation. Okay, understood. Next we have in galactose circuit. This galactose circuit is not in your syllabus. This is given in the Lipin code. Okay, operons are absent in your eukaryote, so there is regulation by coordinate regulation. But the galactose circuit and hormone receptor circuits are two circuits. Okay, so this is the, if the GAL80 blocks the transcription if galactose is absent. Okay, if galactose is absent. Okay, so the TAD. Okay, the TAD is your transcription activation domain is blocked by your galactose 80, and this will prevent your transcription. Okay, and if there is galactose present, okay, so GAL80 will bind to the GAL3. Okay, this GAL80 will bind to the GAL3. Okay, GAL3 gene, and this then the TAD will recruit proteins needed for a transcription. This will induce the TAD to recruit protein for its transcription, and the transcription of the mRNA will occur. Okay, so this is your galactose circuit. So in absence of galactose, your circuit is off, and in the presence of galactose, your circuit is on. So this is about your galactose circuit. Let's move on to another parts, the miscellaneous parts of your molecular biology, that is your recombinant DNA technology. Okay, Talking about recombinant DNA technology or genetic engineering, okay, or gene cloning or gene manipulation, okay. So let's talk about the steps. We have a cell, okay, treat it with enzyme detergent, your phenol, proteases, ribonucleases, okay, do the ultra centrifugation, okay, then precipitate it with chilled ethanol and obtain the desired DNA, okay, and treat the DNA with restriction endonucleases and separate the fragments by electrophoresis method, okay. Identify the segments by Southern blotting, okay. You know the snowdrop phenomena, okay. Snowdrop as SNOW and your DROP. So Southern blotting is for DNA, Northern blotting for RNA, and Western blotting is for your proteins, okay. So snowdrop is there, Southern blotting identified by the Southern blotting, and the desired DNA is got with sticky ends present, okay. So you have a prokaryotic vector, okay. Treat the prokaryotic vector with EDTA and lysozyme, okay. EDTA and lysozyme. EDTA is for your chelating agent and lysozyme is for the breakage of your cell wall of the prokaryotic vector, okay. Then treat with sodium lauryl sarcosynate, do ultracentrifugation and electrophoresis, okay, and obtain the plasmid. Then treat the plasmid with the same restriction endonuclease as you treated the DNA with endonuclease, okay, which will generate complementary sticky ends. The plasmid will generate sticky ends, and the sticky ends will this plasmid and DNA, this will have sticky ends, desired DNA and the plasmid have sticky ends, okay. DNA ligase will act upon by DNA ligase, ATP will be converted to ADP and PI, and a recombinant or chimeric or a hybrid DNA is got. This is a recombinant, hybrid or a chimeric DNA, okay. And by gene transfer, you transfer the chimeric DNA into the host cell, and this DNA is integrated into the host cell genome. When the host cell multiplies, there will be the expression of gene to produce the desired product. You get the desired product from here, okay. This is the whole method. What are the enzymes? Your nuclease enzymes, the exonuclease and the endonuclease, okay. You use restriction endonuclease, which is called as your molecular scissors or your scalpels of molecular biology. So 1, 2 and 3, some books write 1, 2, 3 and 4, okay. Restriction endonuclease 1 and 3, these are not much used, okay, because this cut the DNA away from the restriction sequence, okay. But so we need restriction endonuclease 2. This is most widely used in genetic engineering, okay, because it cuts the DNA within the restriction sequence, okay. Restriction sequence is basically a sequence of 4 to 6 nucleotides, and this is palindromic, very important, palindromic, just same in the both direction. You can see example like madam, right, MADAM, okay. Just you can read from both the direction, it is same, okay. So palindromic sequence is like that, like your GAATTC and CTTAAG, okay. This is a palindromic sequence. If you read from the 5' to 3', this is your GAATTC, and from 3' to 5' again it is your GAATTC, okay. Same in both directions, okay. Understood? So restriction endonucleases, for example, you have EcoR1. How it is named? Eco means the E. coli, Escherichia is the genus, okay. Coli is the species, so Eco, and R means the rough strain, okay. Rough strain, and I is the order of discovery, okay. This is Eco R1, okay. This is not I, 1 you can write, okay. So this is the order of discovery. It is first isolated, which will restrict the growth of virus, okay. And this act like this. You have the GAATTC here, GAATTC here, okay, and CTTAAG here, okay. So the Eco R1, this cuts at this level, at this point and this point, okay, and this generates some sticky ant like GAATTC and CTTAA and G in this direction, okay. The sticky ends are generated like this. The sticky end will favor the integration with the plasmid, okay. So this sticky ends are generated like this, okay. And DNA ligase is called the molecular glue, which requires your ATP, and forms the phosphodiester linkages, okay. Then you have the polymerases and alkaline phosphatases and reverse transcriptases. For this procedure also, this recombinant DNA technology, we require all these enzymes, okay. Talking about vectors, what are vectors? So there are many vectors like your plasmid, your bacteriophage, your cosmid, YAC, BAC, MAC. Okay, these are your vectors. Okay, plasmid is double-stranded extra chromosomal genetic material. Okay, and it has independent replication. It can replicate independently. It doesn't depend upon the host genome. Okay, example, it inserts about DNA of about less than 10 kb. Okay, example is your PUC and PBR 322 with plasmid Bolivar Rodriguez 322. Then your bacteriophage, which will insert it, which will allow the insertion of a DNA segment of your 10 to 20 kb. Okay, 10 to 20 kb. And after this, we have your cosmid. Which will allow about 35 to 50 kb. YAC and MAC have 1 MB, and BAC has a little less, less than 300 kb. YAC is the yeast artificial chromosome, B is a bacterial artificial chromosome, MA is the man artificial chromosome. Okay, and if you just look at the structure of your PBR 322, so you have an ampicillin resistance gene here. The purple one, the tetracycline resistance gene, a ORI region. It has about 4361 base pairs. Okay. and these are your sites where the restriction enzymes are acting. Okay, the BAM-H1 acts in a tetracycline resistance gene region. Okay, your PBU1 acts here near your tetracycline resistance. Okay, your EcoR1 acts here between your ampicillin and tetracycline resistance gene, and your PST1 acts in the ampicillin resistance gene region. Okay. Talking about host is generally prokaryotic hosts are preferred because they have the faster rate of replication, like E. coli, Bacillus subtilis. Eukaryotic hosts like fungi, plant cell, and mammalian cells are also used, but prokaryotic hosts are preferred. Okay, talking about gene transfer techniques, we have many gene transfer techniques. We have physical methods, chemical methods, and biological methods. Okay, physical methods like electroporation, like electric shock is given to the cell due to which pores are created, and this will favor your easy transfer and transmission of your genetic material. Okay, then thermal assisted transfer, your biolistics, micro injection, okay, and laser assisted, your opto injection, and these all are advanced procedures. Okay, then chemical methods, we have your calcium phosphate method, okay, your cationic lipid mediated method, your liposome method, okay, nanoparticles, your the DNA fragments are coated with gold or silica and given, okay, and transferred. Biological methods like a transduction using viral vectors. Okay, transduction is a procedure by which like viral vectors like a bacteriophage lambda are used for transduction. Then we can use adenoviruses, retroviruses, and lentiviruses for the gene transfer also. Okay, and another thing which is called erythrocyte ghosts are there, and exosomes. Okay, gene gun, you see, okay, you have a plastic disc, and you put your DNA coated gold particles over the plastic disc. Okay, and here is a screen where you put some target plant cells. Okay, you fire your helium gas through it, which will cause your falling of this plastic disc along with the DNA coated gold particles, and this DNA coated gold particles will be inserted into your target cells. So this is what a gene guns look like. Okay, understood. So after gene transfer techniques, we finish our recombinant DNA technology. Let's discuss some of the applications of your recombinant DNA technology. Okay, talking about the applications, first one is the production of therapeutic agents or drugs like insulin. Okay, production of insulin, recombinant vaccines. Okay, in COVID we saw the many companies are producing the recombinant vaccines. Okay, like Sputnik and all. Okay, so insulin production, let's see. If there is a okay, this is a plasmid, okay, having I, P, and O genes. Okay, this has an A gene, okay, and a B gene in another of the plasmid. Okay, this both are transferred into E. coli. Okay, this contains your A chain of insulin, this contains your B chain of insulin, specifically the genes which are coding for the A chain and your B chain. Okay, so E. coli is transformed with a recombinant DNA vector. Okay, and the transgenic cells are selected based on their antibiotic resistance. Okay, and this B gal, okay, there is infusion of beta galactosidase, insulin A fusion protein. There is a beta galactosidase, insulin A fusion protein, and we put here beta galactosidase, insulin B fusion protein. Okay, in these two culture medias. Okay, this cause fusion, and your fusion proteins are purified, and the beta gal are removed from this too. Okay, and you get the A chain and you get the B chain. A chain has about 21 amino acid, B chain has about 30 amino acid. Okay, this A chain and B chain are combined, disulfide bonds are formed to form your functional insulin. Okay, talking about hepatitis B vaccine, which is a DNA virus. Hepatitis B virus is a DNA virus. Okay, so hepatitis B antigen producing gene, a gene is taken and the plasmid is cut by restriction endonucleases. Okay, the recombinant DNA is formed, which is introduced into your yeast. Okay, and the yeast in response to your antigen, the yeast will produce your antibodies. Okay, the yeast will produce your antibodies. Here AG is written, this is not AG, this is your antibodies. Okay, the HBV, okay, HBV antibody. Okay, HBV antibody. Okay, the yeast will produce antibody, and this antibody will be used by us as a vaccine. Okay, understood. So DNA vaccines are produced by the recombinant DNA technology by this method. Many substances are produced which are transgenic, like a transgenic erythropoietin for anemia treatment, filgrastim for neutropenia treatment, factor 8 and 9 for your hemophilia treatment, interferons for viral infection and cancer treatment, interleukins for your renal cell carcinoma treatment, epidermal growth factors which are required for a burns treatment, tissue plasminogens and activators like alteplase, retaplase, tenecteplase, which are thrombolytic agents. Okay, the tissue plasminogens and activators are thrombolytic agents. This activates thrombolytic pathway. Okay, anticoagulants, transgenic anticoagulants like your lepirudin. Okay, heparin is your intrinsic anticoagulant of the body. Okay, we can produce transgenic ones like lepirudin. Okay, hormone production like FSH, LH, GSH are also produced transgenically. Okay, it is also used in recombinant DNA is also used, technology is also used in the human genome project. Okay, in gene therapy, in DNA fingerprinting, in construction of a cDNA library, a complementary DNA library. Okay, production of transgenic animals like a knockout mice is there. The Dolly sheep, which was cloned in 1986. In Dolly sheep, what was that? Like adult Scottish blackface ewe was taken, who was the egg cell donor. Okay, the egg cells were removed from the Scottish blackface ewe. Okay, and the nucleus is removed. Nucleus is removed by a micropipette. Okay, and then the nucleus. Another one is a nucleus donor, that is an adult Finn Dorset ewe. Adult Finn Dorset ewe, which has white face. Okay, and this had black face. So from white face sheep, the nucleus is taken. From the nucleus donor, the donor cells are removed from the mammary gland. Okay, the donor cells are starved and growed in low culture media. Okay, and which must have arrested growth cycle, and this both cells are given electrical sparks, pulse, and fused. Okay, cell fusion. The cell fusion, after they are fertilized cell division will occur, embryo formation will occur. Embryo is again implanted into adult Scottish blackface ewe, which also acts as a surrogate mother, and from this surrogate mother, your Finn Dorset lamb, your Dolly, was produced. Okay, this is the Dolly. Okay, you can see the beautiful picture of your Dolly. Okay, so these are the applications of recombinant DNA technology. Let's now discuss a technology which is called as a polymerase chain reaction. Let's discuss the polymerase chain reaction. Okay, talking about polymerase chain reaction, in polymerase chain reaction we use DNA polymerase, okay, for extension of nucleotides to DNA. Okay, there is a chain reaction specifically occurring which proceeds in exponential manner. Okay, this is an amplification technique. Okay, we amplify your DNA to produce several copies. Okay, what are the requirements? We require a PCR machine which is called as a thermal cycler or a thermocycler. Okay, this is a template DNA strand. Okay, which is required for a new strand synthesis. Your primers of 20 to 30 nucleotides, which are called your proofs, which must be complementary to the ends of the target DNA. Okay, you can have forward primers or your reverse primers. Okay, and then we can have your tag DNA polymerase, which only helps in your extension procedure obtained from a Thermus aquaticus bacteria, which is a thermophilic bacteria. Okay, a bacteria which can survive in extreme temperatures. Okay, we also required DNTPs like DATP, DGTP, DCTP, and DTTPs. Okay, this is see the thermocycler machine. This is thermocycler machine. Okay, what are the steps? So 3 steps: denaturation, annealing, and extension. Denaturation is done at 95 degrees Celsius. From 15 seconds to 2 minutes it is done. Annealing 56 degrees Celsius. Okay, for 30 seconds to 2 minutes, and extension at 72 degrees Celsius for 30 seconds to 2 minutes. Okay, denaturation at 95, annealing at 56, extension at 72. The time is like on an average we do about 1 minute, 15 seconds to 2 minutes, 30 seconds to 2 minutes, 30 seconds to 2 minutes. So for one cycle we require about 3 to 5 minutes. For 25 to 30 cycles we will require about 3 to 6 hours. Okay, so this is a DNA strand. Okay, you do denaturation, the strands will separate like this. Okay, then you add the primers, which is called as annealing. Annealing is done at 56 degrees Celsius. So the primers are attached like this. The primers must be complementary to the ends of DNA we told earlier. Then you attach the tag DNA polymerase, which helps in your extension procedure. Okay, this tag DNA polymerase attached, which will catalyze the formation of catalyze the extension, and will form your 2 DNA molecules. This strand and this strand are made up of by your tag DNA polymerase. Okay, and the steps 1, 2, and 3 are repeats to produce multiple copies. What are the applications of PCR? So it is used in the detection of viral infections. Okay, like SARS, your HIV, and all. Detection of your tuberculosis, detection of your cancers, your prenatal diagnosis of your diseases like your sickle cell disease and thalassemia. Okay, in recombinant DNA technology, gene library or cDNA library construction. Your forensic applications like a paternity testing also it is used. Okay, then how you detected your COVID-19 virus in your COVID times or pandemic times? RT-PCR was most commonly used. Okay, so here we involved one extra step also. One extra step was there in your when your COVID-19 was there, because for DNA viruses we don't need the extra step, but COVID-19 is a RNA virus. The SARS-CoV-2 is a RNA virus. Okay, so when taking the swab, okay, you must have seen the pictures of or you may have experienced the swab collection. This is a little bit of discomfort is there and all. Okay, which is transported via viral transport media, transported via VTM, viral transport media, taken to the lab. Okay, in the lab there is RNA extraction. RNA extraction is done, and this is the extra step. It is treated with a reverse transcriptase enzyme. Okay, reverse transcriptase, which will form your complementary DNA formation. Okay, as you don't have your DNA beforehand, so we will have to produce DNA from the RNA. Okay, then after the DNA is formed, just do the steps of your PCR, denaturation, annealing, and extension. Okay, and produce multiple copies. Based on the time taken, based on the number of cycles required to produce multiple copies, you can estimate the viral loads. Okay, if the viral load is less, there will be more cycles required. Okay, those cycles is inversely proportional to your viral load. Okay, in real-time PCR, your RT-PCR or real-time PCR, we required 2 extra things like a Tachman proofs, TAQMAN Tachman proofs, and your SYBR dye, which is a fluorescent dye. Okay, which is a fluorescent dye, and this can be detected by your fluorometer, and a graph can be formed. Okay, so Tachman proof and SYBR dye, these are used to quantitatively estimate the viral load in real time. Okay, in real time. Okay, so what happens in the Tachman proof? This is a Tachman proof. We have a Q region and a R. Okay, a Q marker and a R marker. Okay, Q is called the quencher of fluorescence, R is called the reporter of fluorescence. Okay, and this is your DNA polymerase. Okay, so when Q and R are together, okay, when Q and R are present together, there is no fluorescence. Okay, there is no fluorescence. But when the DNA synthesis starts occurring due to the action of the DNA polymerase, this DNA synthesis will start occurring. Okay, and R and Q will be separated due to exonuclease activity of the polymerase. Polymerase has exonuclease activity, you know. Okay, due to the exonuclease activity of the polymerase, the R and Q separate, and due to this R and Q separation, there is the fluorescence which can be detected by a fluorometer. And this fluorometer will tell you how much is the viral load. Okay, so this is the artificial, is the advanced technique and the improvisation over the PCR. Okay, so here we complete our molecular biology. Here we completed our molecular biology. Let's move on to another chapter. Okay, let's move on to another chapter. Okay, so see that we have completed these chapters, and we have completed the molecular biology, recombinant DNA technology, PCR now. Okay, which was a big unit. Okay, and let's move on to the next chapter. Let's take which chapter we should learn now. Let's read about cell. Okay, let's read about our cell. Okay, let's read about our cell. You know many things about the cell. You have read in physiology, you have read in previous classes in 12th and in detail. Okay, so cell is everyone's favorite chapter, like that. Okay, so I will try to just cover up the important facts which are needed, which should be written for your prof exam as well as the PG is concerned. Okay, and MCQs are concerned. Okay, so let's discuss about the cell. Okay, talking about a cell, which things are important. Your fluid mosaic model, this is important topic of cell, and there is an important diagram of your mitochondria too. Okay, mitochondria, you should be able to draw and label the diagrams of your mitochondria. Okay, and transport, and the transport across cell membrane, okay, this can become this can come as a long question, and you can get short questions on the organelles too. Okay, so there are organelles and marker enzymes which are important. So mitochondria, okay, we are talking about enzymes and the marker enzymes. Okay, marker enzymes. Okay, for the mitochondria, the marker enzyme is a ATP synthase. Your marker enzyme is a ATP synthase, and for your lysosome, the marker enzyme is cathepsin. Okay, cathepsin, and for your cytoplasm, the marker enzyme is a LDH. An important fact is in your cell is that about the Zellweger syndrome. Okay, we discussed the Zellweger syndrome where in the protein targeting also. Okay, Zellweger syndrome. So it is a cerebrohepatorenal syndrome. Okay, it is the biogenesis disorder of your peroxisomes. Okay, so just remember this there is the absence of your peroxisomes are absent. The peroxisomes are absent, due to which the oxidation of a very long chain fatty acids, this is affected. Okay, understood. So let's talk about your fluid mosaic model. Okay, let's talk about your fluid mosaic model. Okay, let's discuss the fluid mosaic model. Okay, before fluid mosaic model, there was another model which was called as unit membrane model. Okay, which was called as a unit membrane model. Okay, which was called as a unit membrane model given by Robertson. Okay, but this was this model has very defects. Okay, and the most accepted model is the fluid mosaic model given by Singer and Nicholson in your year 1972. Okay, and the fluid mosaic model is the most accepted model currently. Okay, and talking about the fluid mosaic model, let's see the diagram of the fluid mosaic model. So this is your fluid mosaic model. Okay, and your fluid mosaic model is composed of your lipids, proteins, and carbohydrates. If you have your lipids, you have your proteins, and you have your carbohydrates, you have your carbs. Okay, lipids are present majorly as your phospholipids. Okay, phospholipids are majorly present. Okay, and you have some cholesterol also. Okay, cholesterol, what is the function of cholesterol? Cholesterol maintain the stability of membrane, act as the membrane buffers. Okay, this are responsible for the fluidity of the membrane. Okay, the membrane make the membrane fluid, less and more fluid. Okay, according to the needs of the cell. Okay, this maintain the fluidity of the membrane. Okay, and you have the phospholipids. Phospholipids are arranged like this. It has a hydrophilic tail. This is the hydrophilic end, which loves your water. Okay, and you have the two tails and a kink here. Due to the double bond present, there is a kink present. Okay, and this is your tail, which is a hydrophilic tail. Okay, so how this is arranged? Okay, let me tell you how it is arranged. let us add some pages. Okay, how it is arranged. So the heads are like this, heads are like this over the outer side of the membrane, and the tails are present in the inner side of the membrane. Okay, so it is a lipid bilayer, it is a lipid bilayer with some proteins in between. Okay, look at this, we have some proteins, the protein channels are present. Okay, globular proteins are present. Okay, we have glycoproteins too, which are your proteins attached to your carbohydrate chains, you can see. And you have an integral membrane protein, you have integral membrane protein and peripheral membrane proteins. Okay, integral membrane protein which span the whole membrane. Okay, and your integral membrane protein, look at this, this is also an integral membrane protein, the spiral protein, also called as a alpha helix protein, which is also integral membrane protein. Okay, and your carbohydrates are present over the proteins, which are your glycoproteins, and glycolipids are also present. Okay, if that the carbohydrates are there here over the lipids, okay, which are called as your glycolipids. Okay, glycoproteins and glycolipids are present over the cell membrane, and glycoproteins and glycolipids has the function of giving antigenicity to the cell membrane. Okay, and help in the cell reception also. Okay, act as receptors for binding of various substances. Okay, and look at this, as cholesterol is present between the cell membrane and all. Okay, so you know a lot about the cell membrane, so I'm not discussing it in detail. Okay, so afterwards let's talk about the transport across the cell membrane, how the transport occurs. Okay, transport across cell membrane. So you can have a passive transport, you can have active transport, and you can have vesicular transport. Okay, passive transport is by simple diffusion, it may be facilitated diffusion, and you can have osmosis. Okay, and active transport, it may be primary active transport, it may be secondary active transport. Okay, so what is primary active transport is that this is important, just focus. Primary active transport is that the direct energy is derived from your ATPs, and in the secondary active transport, that is indirect energy utilization, indirect energy utilization. Okay, primary active transport, for example, you have your sodium potassium ATPase. Okay, sodium potassium ATPase, you have your calcium ATPase. Okay, you have your calcium ATPase, and you have your H plus K plus ATPase, as seen in gastric cells. Okay, then you have indirect energy utilization, you have the secondary transport. For example, you have a uniport mechanism in which one substance only goes by the protein. You have symport mechanism, you have the symport, in which two things move across the membrane in same direction, and you have an antiport too. In which the two substances move in opposite direction. Okay, we have an antiporter, symporter, uniport proteins. For example, your fructose goes via the uniport mechanism. Okay, fructose goes via the uniport mechanism. Okay, and vesicular transport, you have two types, like your exocytosis is there and your endocytosis is there. Okay, you know all this. Okay, in your physiology also there is a chapter, transport across cell membrane, in your diagram textbook. Okay, you must have read. Okay, active transport, what do you need about active transport? This occurs against the concentration gradient. From low concentration you go to the higher concentration, and active transport is always carrier mediated. We have a carrier protein which mediate this. Okay, primary active transport and your secondary active transport, like a sodium potassium ATPase, you know. Okay, there is a sodium potassium pump, and here is the membrane. Okay, so the sodium potassium pump, it transfers your 3 sodium out. Okay, 3 sodium out and 2 potassium in. Okay, 2 potassium in, and it requires a breakdown of ATP, changing it into ADP plus PI. Okay, you have a calcium ATPase as well as H plus K plus ATPase. Okay, in secondary active transport, what happens in secondary active transport? You have a uniport, symport, and an antiporter. Okay, you have your like uniport by fructose, how fructose is moved. Okay, you have your symporter. Examples of a symporter are your sodium glucose symporter, sodium glucose symporter. Okay, sodium glucose symporter, and you have your sodium amino acid symporter, Okay, these are your symporters, and these symporters are this sodium amino acid symporter, this is affected in your syndrome, which is called as a cystinuria. And your heart nerve disease, this is affected. The sodium amino acid symporter is affected. Okay, understood. Then talking about antiporter, you have two types. You have your sodium H plus antiporter, exchanger. Okay, exchangers are basically your antiporters only, and your calcium H plus exchanger. Okay, and this antiporter, look at this cell. Okay, the cell, this is the sodium H plus exchanger. Okay, which will transfer your sodium inside the cell and will transfer the H plus outside the cell. Okay, and another antiporter is your sodium K plus antiporter. Okay, this sodium which comes inside the cell is transmitted out. It's transferred out by the sodium K plus antiporter, due to which the K plus comes in. The K plus comes in. Okay, so these are your antiporters present in the membrane. Okay, you know about the passive transport. Let's discuss the passive transport. You know about the simple diffusion. It is the down the concentration gradient from high concentration to low concentration. Okay, here the energy expenditure is 0, no energy required. Your carrier is not needed. Also, your carrier is also not needed. Okay, you have a simple diffusion. Simple diffusion has two mechanisms, like your lipid soluble compounds which are transported directly. Okay, you have your direct mechanism, direct simple diffusion, and you have your ion channels too, ion channels. These are also your simple diffusion. You have voltage gated ion channels, okay, which are based upon your charges, like your sodium voltage gated, potassium voltage gated channels and all. Okay, then you have ligand gated channels, like ligand gated, okay, which which use your neurotransmitter and neuropeptides for the opening and closing. Okay, which use your ligands, okay, which use your ligands like acetylcholine neurotransmitters like GABA and glutamate. Okay, acetylcholine and glutamate. Okay, understood. Then about the facilitated diffusion, you know about the facilitated diffusion. Okay, energy expenditure is not there. Your energy is not needed, but what is needed? Your carrier protein is needed. Okay, carrier protein is needed, like and it acts by the ping pong mechanism. You know about the ping pong mechanism. Okay, ping pong mechanism, what happens if is the membrane. Okay, if there is a transporter, okay, if there is a transporter here, okay, it will bind some the substances which it has to get into the cell. This is the inside of the cell, this is the out of the cell, this is the out of the cell. Okay, and this transporter, this turns by your 360 degree. Okay, like this, not a 360, sorry, 120 degree, 180 degree. Okay, and the substances which it had received is transported inside the cell. Okay, this is called as a ping pong mechanism. Okay, understood. After this, your fructose, which moves by antiporter. Okay, another example of an antiporter, fructose moves by uniporter. Another example of antiporter is seen in your stomach and RBCs, in which there is a HCO3 minus chlorine exchange. Okay, you must have read about your hamburger phenomenon and all. Okay, so antiport, 3 examples. You can see the 3 examples here. Okay, understood. Then afterwards, let's talk about the osmosis. There are clinical your applications like edema and your saline infusion. Okay, your mannitol is there. Mannitol has an osmotic effect which helps in the treatment of cerebral edema. So just remember the clinical facts. Okay, and osmotic fragility test is done in your hereditary spherocytosis also. Okay, osmosis, what is osmosis? It is the movement of your solvent, specifically solvent or water, of any water, okay, towards higher solute concentration. Means if there is this present and these are connected, okay, this is less solute concentration and this has more solute concentration. Okay, so the fluid that is present in the less solute concentration, this moves towards your high solute concentration. Okay, understood. And about the cellular organelles, you know about the cell organelles. You have a nucleus. Okay, nucleus is the genetic material, contains the genetic material, DNA. Okay, you have an endoplasmic reticulum. Okay, just remember that the marker enzyme of ER, where we were talking about the marker enzymes. Okay, the marker enzyme for the ER is a glucose 6-phosphatase. Okay, glucose 6-phosphatase. Okay, just remember the marker enzymes. For mitochondria it is ATP synthase, for lysosome it is cathepsin, for cytoplasm it is LDH, for ER it is glucose 6-phosphatase, which is the marker enzyme. Okay, then we will talk about the lysosome. You know about the lysosomes. It is a bag of your digestive enzymes. Okay, the bag for digestive enzymes. It is single membrane bound. Okay, and the lysosomes will have the function of your autophagy. Okay, autophagy of the cell and digestion of your degradation of your bacterial toxins. Okay, bacterial toxins and all your worn out cell organelles, and all. Okay, then talking about your mitochondria, you know about the mitochondria. Mitochondria is the powerhouse of cell. What are the pathways that are occurring in our mitochondria? So your ETC is occurring, your ATP synthesis is occurring in the mitochondria, your TCA cycle is occurring, your fatty acid oxidation is occurring. It is very important biochemically. Your ketone body production is occurring, your heme synthesis is occurring. Then your urea cycle, and about your gluconeogenesis, and the pyrimidine synthesis and all. Okay, pyrimidine synthesis and all. These are occurring in your mitochondria. Okay, mainly a part of your heme synthesis, not the whole heme synthesis. Okay, and you know how to draw the mitochondria. So draw a sausage shape like structure. Okay, and the inner membrane is folded. The foldings are called as your cristae. Okay, cristae are your infoldings in the mitochondria, and cristae have your F0 F1 particles, F0 F1 ATPase particles. Okay, these are called as your ATP synthesis. ATP synthesis, this F0F1 particles or your Fernandez-Morans particles. Okay, and intermembrane cleavage, the outer membrane. Okay, it's the matrix which is present inside. Okay, you just know how to draw. And mitochondria has the ribosomes. Your 70S ribosomes are present in mitochondria. Mitochondria also has its own DNA. Okay, so your mitochondria has your own DNA, which is called as mtDNA, and mtDNA, so maternal inheritance. Okay, maternal inheritance. About your okay, so the marker enzyme which I was talking about, your ATP synthesis is the marker enzyme. Okay, and talking about next the Golgi apparatus, the marker enzyme for the Golgi apparatus is your galactosyl transferase. Is your galactosyl transferase. Okay, galactosyl transferase, which acts as the marker enzyme for your Golgi apparatus. Marker enzyme for your Golgi apparatus. Understood? And Golgi apparatus is involved in protein packaging, sorting, and secretion, as you saw. Okay, then talking about the ribosomes, you know about a lot about ribosomes, you can write. Okay, so, and cytosol, it is a gel like matrix surrounded by a cell membrane. Okay, in cytosol, what are the pathways that are happening in the cytosol? Okay, so cytosol, it is a gel like matrix. Okay, surrounded by a cell membrane. It is the site of your protein synthesis. Your protein synthesis is occurring in the cytosol. Your glycolysis is occurring in the cytosol. Okay, your glycogen metabolism is occurring. Glycogen metabolism, okay, is occurring. And fatty acid synthesis, your fatty acid synthesis is occurring in your cytosol. Okay, and a part of your heme synthesis, your urea cycle. Okay, a part of this all are occurring in the cytosol. Okay, so this completes your cell topic. Okay, and after the cell topic, we have completed the cell topic in such less details, okay, which are needed for your net profs. Okay, just in a revision format, but I have covered each and every point. Okay, and said each and every point. Okay, so now we have completed our cell. This cell is now completed. Okay, so now let's move on to our next topic. Okay, let's move on to our next topic. Okay, so let's start with the chemistry of your carbohydrates. Okay, carbohydrates, these are the substances which are providing about 4.1 kilocalorie or 4 kilocalorie per gram of energy. Okay, and this form a nucleotide component, DNA and RNA, metabolic intermediates like a glucose 6-phosphate. Okay, and the precursors of the glycogen, galactose, ribose, glycolipids, and the proteoglycans. Okay, carbohydrates are like simple and complex. Okay, found naturally in plants like fruits. Okay, veggies and vitamin plant sources. Animal sources like milk and cheese, these are present. Simple carbohydrates are sweet in taste and are used as a fuel for rising dough and preserving jams and jellies. Okay, you know this. Okay, so these are simple sugars, just one unit is there, and these are complex sugars, the branchings is there, branching and the complex chain is there. Okay, and added sugars which are not good for health are present in the cereals and cornflours. Okay, so cornflakes are not much good for health. Okay, monosaccharides, we have monosaccharides, oligosaccharides, and polysaccharides. So monosaccharide, we have one unit like a glucose, galactose, and fructose. Oligosaccharides, we have 2 to 9 units. Okay, disaccharides and trisaccharides and tetrasaccharides. Okay, disaccharides like glucose plus glucose is maltose, glucose plus galactose is your lactose. Okay, and glucose plus fructose is your sucrose. Okay, so that's the correction here. This is not galactose. This is your lactose. Okay, this is your it got very thin. It is your lactose. Okay, it is your lactose. Understood? So after that, your sucrose, which is called as a table sugar. Okay, we have trisaccharides, we have tetrasaccharides, pentasaccharides, hexasaccharides, and so on. Okay, polysaccharides have 10 or more units of glucose of your that sugars. Okay, understood? So after this, this starch, important fact is that it lowers the absorption of sugar and increases the bulk of stool. Okay, so starch is prescribed as in addition to dietary fibers. Okay, so lactose is broken down by lactase to glucose and galactose, sucrase to glucose and fructose, maltose by maltase to glucose and glucose. Okay, so these are broken down. Okay, if you eat food, and starch digestion starts from the mouth only. Okay, important fact about fructose is that it is higher constituent in honey and maple sugar. In honey, about 50% sucrose is present, and in maple sugar, okay, it is more preferred and just as compared to honey, because less than 1% fructose is present. Okay, less than 1% fructose is present. Okay, and insulin, it moves glucose out. Okay, and stimulates your liver and promotes your fat and protein synthesis. Okay, so these are your introduction, just your introduction to your carbohydrates. Let's move on to the base parts. So now we'll read in detail about the carbohydrates, okay, classification and all. So talking about glucose, glucose is one of the most important carbohydrate, okay, and it is a major metabolic fuel of mammals, universal fluid of the fetus, and precursor of glycogen, ribose, deoxyribose, galactose, and etc. Study of sugars is called as glycobiology. Okay, glycome is the entire glucose content of an organism. Study of glycome is called as a glycomics. Okay, classification, we have monosaccharides, we told about trioses, tetroses, pentoses, and hexoses, and we have aldoses and ketoses of this all. Aldoses and ketoses of this all. Like aldoses and ketoses form of some important sugars, okay, like your trioses. Okay, trioses, we have the aldose from glycerose and ketose from dihydroxyacetone. Okay, glyceraldehyde and dihydroxyacetone. Tetroses, we have aldose form is erythrose, ketose form is your erythrolose. Pentoses, we have aldose from ribose and your ketose form is a ribulose. Okay, hexoses, we have aldose form of glucose, galactose, mannose, and the ketose form is a fructose. And then heptoses, we don't have any aldose form here, but we have the ketose form, that is pseudoheptulose. Okay, this is given in a Harper. Then about disaccharides, disaccharides is comprises of monosaccharides and monosaccharides. Okay, condensation of two monosaccharides like lactose, maltose, isomaltose, sucrose, and trehalose. Okay, trehalose is present in hemolymph of your insects. Okay, and oligosaccharides, you have the condensation of 3 to 10 monosaccharides which are not digested by the human enzymes, and polysaccharides. Okay, more than 10 monosaccharides are condensed, like your hexoses and hexosans and pentosans. Okay, if monosaccharide is hexose, then the hexosans are formed. The monosaccharide is pentose, then the pentosans are formed. Okay, what are the examples of your polysaccharides? Your cellulose, insulin, starches, dextrins, and all. Okay, this is the straight chain formula of the your glucose. Okay, the straight chain formula of the glucose. How to draw? Just you see, you have the 6 carbon atoms here. Okay, 1 2 3 4 5 and 6. Okay, just do CH2OH here and just write CHO here. Okay, and just you have to remember right, left, right. Okay, so you have to do like this. Okay, just put OH, H, and OH. Okay, and then fill up it with OH and H. Okay, this is your glucose. This is your glucose. This is a straight chain form of the glucose, and glucose can be represented in other forms, like a Howard projection is there. In our Howard projection, the hemiacetal form of glucose is there. Okay, the hemiacetal form is thermodynamically more favored form. The hemiacetal form, the thermodynamically more favored form, and a chair form is also there. A chair-like structure you must have read in your stereochemistry and all. Okay, a chair form is also there. Okay, so this is the representation, the Howard representation of glucose. Okay, this is dependent on alpha and beta. Okay, just I'm drawing the structures of your alpha glucose and the beta glucose. Okay, so talking about the alpha and beta form and how you can identify it, just look at this HOH, HOH, OH, HOH, and H and CH2OH. Okay, this form where the O and the OH are a bit far, this is the alpha form, alpha D glucose. This is alpha D glucose, and where your this O is there and your OH, H is there, OH, H, and HOH, these are all normal. Okay, HOH and CH2 OH and H. Okay, this where the O and the OH, these are in close proximity. Okay, this is called as a beta D glucose. Okay, it's called as a beta D glucose. Okay, so alpha form and beta form, this both, the alpha D glucose and beta D glucose, these are called as your anomers. Okay, they're called as your anomers. So what are anomers, we will see in the next part of your carbohydrate chemistry. Okay. Okay, so see, so glucose has about 4 asymmetrical carbons. So 2 to the power 4 is 16 isomers. So glucose has about 16 isomers. Okay, so we're talking about isomerism. So D and L isomerism is that, D and L isomerism. The parent compound is the glycerose, glyceraldehyde, which is the simplest carbohydrate. So in D and L isomerism, this is taken as the base. Okay, this is L glycerose, this is D glycerose. Okay, this is L glyceraldehyde, this is D glyceraldehyde. How you said L and D? With if it is left, present in the left, OH is present in the left, L glyceraldehyde, and OH is present in the right, it is D. This is capital D and L. Don't confuse it with small d and L. Okay, so this carbon, okay, which is next to your CH2OH group, the carbon next to your terminal alcoholic carbon, the carbon next to the terminal alcoholic carbon is called as the penultimate carbon, which is your C5 in your glucose, the C5 carbon in your glucose. Okay, and this penultimate carbon determines your D and L. If D is on, OH is on the right, it is D, and if OH is on the left, it is L. Okay, the most naturally occurring monosaccharides are basically D sugars. Okay, so this is D glycerol, L glycerol, this is L glucose, this is L glucose, and which we are drawing is a D glucose. Okay, which, and the trick I said to draw the glucose. Okay, this was your D glucose. Okay, and the L form just reverse the OH group. Okay. And the C5 position. Talking about optical isomerism, if there are 4 different groups attached to the chiral carbon, okay, then the 4 different groups are attached to the carbon is called as a chiral carbon or asymmetric carbon, which confers optical activity. So it rotates the rotates the plane of polarization to the right or left, depending upon them. We say the small d and small l. If the right twist is there, then it is called a dextrorotatory plus or a small d. Don't confuse it with large D, okay, capital D. And if it is the left twist, we call it levorotatory. Negative sign is there, all small l, okay. And rotation is independent of the stereochemistry of the sugar, okay. So fructose in nature is your D negative isomer, okay, D form, but levorotatory isomer, D negative isomer, okay. So don't confuse it. And glucose solution is dextrorotatory, so it is called as a dextrose. So glucose is also called as a dextrose because majorly D form is present, okay. Then you have the pyranose and furanose form. Pyranose means a 6-membered ring. This is from your pyran, okay. Pyran is a 6-membered compound, okay, and the 6-membered rings are thus named pyranose, okay. And the furanose form is a 5-membered ring. This is furan, a 5-membered compound, okay. So all the glucose structures which have 5 members are called as furanose structures, okay. So glucose in solution about 99% is a pyranose form only, okay. This is in a 6-membered ring, okay. And see at the pyranose and furanose, this is alpha glucopyranose and the alpha glucofuranose structure. Just you have to see the structures. These structures are not much important, okay. And these are the pyranose and furanose form of glucose. The alpha fructopyranose, okay. This is a beta fructopyranose. Alpha and beta I said. This is far, so it is alpha, and if it is near, okay, it is near, it is your beta, okay. Then alpha your furanose why it is a 5-membered, okay, and alpha because it is far, and as it is near, it is a beta fructofuranose, okay. And glucose 19% is a pyranose form. Then we have anomerism, alpha and beta anomers. The ring structure of aldose is a hemiacetal. The ring structure of ketose is a hemiketal, okay, hemiketal, okay. So we have the alpha D fructopyranose and the beta D fructopyranose. I said how to determine your alpha and beta, okay. So this completes our isomerism. Let's move on to the next topic. So this was all about your monosaccharides. So just know that all your monosaccharides, okay, there are few tests for the monosaccharides, okay. There are a few tests, okay. Let's talk about the Molis test. Molis test is a group test for all carbohydrates, okay. Then you have the Benedict test, okay. This is a general test. Molis test is a general test. Benedict test is only given by reducing sugar, okay, and all monosaccharides are reducing in nature. Then you have the Barfoed's test. Barfoed's test. Barfoed's test is for monosaccharides only, okay. This is specific for your monosaccharides. And you have your Sullivan's test, okay, Sullivan's test, and this is given by your ketose sugar only. Okay. Let's talk about, after monosaccharides, let's talk about the disaccharides, okay. So disaccharides, we have reducing disaccharides, okay, and the non-reducing disaccharides. So reducing disaccharides, we have your lactose, okay, and your maltose, okay. Just remember in mnemonic your mala sutre, okay. These are the disaccharides. So this mala, maltose and lactose, these are reducing, and the sucrose and trehalose, these are non-reducing, okay. So lactose is formed by glucose and galactose, you know, okay. This is formed by your glucose, and galactose, and the bond is your in your lactose, the bond is your beta 1,4, okay, beta 1,4 glycosidic bond. In your maltose, you have your alpha 1,4, okay. You have the combination of glucose, and you have alpha 1,4 glycosidic bond, okay. And in non-reducing, we have a sucrose is formed by glucose and fructose, okay. And the glycosidic linkage is a alpha 1 beta 2 glycosidic linkage, okay, in which the carbon 1 and 2 both are involved, okay. As the both carbons are involved, it won't form your ozazone, okay. So sucrose, ozazone, okay, it won't form your ozazone, okay. And about your which carbohydrates form ozazones are formed, okay. If the carbohydrate derivatives, okay, so ozazones, what are ozazones, okay, when your carbohydrate derivatives, when you're reducing sugars, this is a property of reducing sugars, and the reducing sugars are treated with the phenylhydrazine. These are treated with the phenylhydrazine at boiling temperature, okay. They form your ozazones. Like what ozazones are there? The ozazone of lactose, okay, these are called as a lactosazone. Ozazones of maltose is called as a maltosazone, okay. The ozazones of your glucose, fructose and mannose, this form your same ozazone, your glucose, your fructose and your mannose, okay. This form is same ozazone, because the ozazones this all, okay, differ only over your carbon 2, okay. And this is masked because the phenylhydrazine, okay, this only get attached to your C1 and C2, okay. So as there is no difference now, as they were differing in your C2, and the phenylhydrazine also attacks your C1 and C2, okay. So this all form a same type of ozazones. So what how is the shape of the ozazones? Let's see. Okay, just see what are the shapes of the oxazones. So needle shaped glucosazone crystals are there. Okay, so the oxazone, that is glucose. Okay, this shape like needle shaped crystals like this. Okay, the glucose, the fructose, and your mannose. Okay, this form your needle shaped structures, needle shaped crystals. And your galactosazone. Okay, your galactose form rhombic plate like structures. Okay, this form your rhombic plate like structures, plate like. Okay, like this, these are called as rhombic plates. Okay, rhombic plates. Okay, and your maltosazone forms sunflower shaped structures. Okay, like your sunflower shaped structures are there. Okay, it is also called as your petal shape. Okay, petal shaped structures, and your the lactosazone, your lactose forms your powder puff or hedgehog shaped structures, your powder puff or the hedgehog shaped crystals. Okay, so your lactosazone is forming your powder like. Okay, powder puff shaped. Okay, and your glucosazone, your fructosazone, and your mannozazone, these are forming the needle shaped or the broom shaped. Okay, and another one is your galactosazone is not much important. Okay, and the maltosazone. Okay, this is forming your petal shape, or your sunflower shape, sunflower shape. Okay, so this is all about the disaccharides. Let's talk about your polysaccharides. So polysaccharides can be of two types: your homopolysaccharide. Okay, okay, which have the same sugar units, and your heteropolysaccharides. Okay, and your heteropolysaccharides. Okay, what are the homopolysaccharides? So just note the homopolysaccharides. What are there? These are your starch. Okay, these are your starch, and your glycogen, starch, and your glycogen. Okay, and your cellulose. Okay, we'll discuss this also, cellulose. Okay, and your dextrin. Okay, and afterwards we'll talk about the dextran is also there. Dextran. Okay, I'm writing what I'm writing. Dextran. Okay, dextran. And your inulin, not insulin, it is your inulin. Okay, inulin, and again your chitin, inulin, and again your chitin. Okay, these are your homopolysaccharides. So starch, glycogen, cellulose, dextran, insulin, your inulin, and your chitin. Okay, so these are your homopolysaccharides. Okay, and you can get short note on your important homopolysaccharides and about the functions. Okay, so starch is a storage form of carbohydrate. Okay, it is comprises of your two parts, which is your amylose, and your amylopectin. Amylose is the straight chain. Amylopectin are the branches. Okay, it is the branch chain. Okay, it has the branches present. Okay, and it comprises of about 20% of starch. Amylopectin is of 80% of your starch. Okay, amylose at this straight chain and simple structure, so it is water soluble. But your amylopectin, it is water insoluble due to complex structure. Is water insoluble. Okay, and which bonds are present in amylose? Alpha 1,4 glycosidic bond, alpha 1,4 glycosidic linkages are present. And in your amylopectin, alpha 1,4 bonds are present, of course, but alpha 1,6 are present at the branch points, your branching points. Okay, so this is about your starch. Okay, this is about your starch. Okay, so you have two forms, amylose and your amylopectin forms. About your glycogen, so glycogen is also multi-branched polysaccharide. Okay, it is the main storage form. It is the storage form of your glucose in your liver, in case of your liver and your skeletal muscles. Okay, skeletal muscles. So it is the homopolysaccharide of your glucose with which bonds? Your alpha 1,4 bonds plus your alpha 1,6 bond at the branching points. Okay, and it makes the glycogen more globular. Okay, it makes the glycogen more globular and compact structure. Okay, and less space consuming. Okay, talking about cellulose is a polymer of glucose with beta 1,4 glycosidic bonds, beta 1,4 glycosidic linkage. Okay, it can't be digested by humans can digest it. Okay, humans can digest it due to the enzyme, due to the lack of your enzyme, that is cellulase. Okay, cellulase, which acts as a dietary fiber. Okay, and we'll talk about the dietary fibers also. Important thing is to note that the cellulose decreases your glucose, decreases. Okay, this decreases the absorption of your glucose and your cholesterol, so it is important in the management of your diabetes mellitus and obesity. Okay, so it is important. Okay, management of your diabetes mellitus and obesity, decreases glucose and decreases cholesterol absorption. Okay, absorption from intestine. Okay, talking about dextrin, dextrin is the partial digestion product of starch. Okay, if you just keep on digesting your starch, okay, you will get dextrin. And about dextran is a complex branched structure. It is also branched. Okay, it has many important uses, like it is used as a plasma volume expander. Okay, this is important, plasma volume expander, and it is used in the treatment of hypovolemic shock. Okay, this is used in the treatment of your hypovolemic shock. Okay, it has also anti-thrombolytic activity, antithrombolytic activity. Okay, it is also used in eye drops. It is also used in the eye drops as a lubricant. Okay, lubricant. And it also provides parenteral nutrition. Parenteral, okay, across the placenta it provides nutrition. Okay, parenteral nutrition. Okay, these all are important functions of your dextran, so just remember this is important. Okay, and inulin, about inulin is a polymer of fructose. It is a polymer of fructose, not of glucose, the polymer of fructose. Okay, it important statement, it is used in the assessment. It is used in the assessment of your GFR, glomerular filtration rate. We saw today in your organ function tests. Okay, the assessment of GFR was also, inulin was also used. Okay, it is a better marker over your urea and creatinine. Okay, because it is filtered through the glomerulus, it is neither reabsorbed nor secreted by the renal tubules. Okay. Then we will talk about chitin. Chitin is present in the exoskeleton of insects. Okay, so insects have exoskeleton. Okay, over which the chitin is present, and the cell wall in the fungi, cell wall of fungi is also made with your chitin. Okay, we know this. This is a polymer of N-acetylglucosamine. This is a N-acetylglucosamine polymer, and this N-acetylglucosamine is derived from a glucose itself. Okay, understood. So after this we will talk about a heteropolysaccharide. We talked about the homopolysaccharides. Okay, so heteropolysaccharides have more than 10 sugar units. Okay, there are many important heteropolysaccharides. So these are mainly divided into 4 classes. One is your GAGs, okay, glycosaminoglycans, or which are also called as the mucopolysaccharides. Mucopolysaccharides, we have disorders related to them also. We have agar. Okay, we have agar, we have agarose, and we have pectin. Okay, we have pectin. Agar is made up of glucose and galactose. Okay, glucose plus galactose will give you agar. Glucose and galactose, not a disaccharide. Okay, and it is used in the bacterial culture media. You know agarose, agar, and agarose is made up of your galactose plus anhydrogalactopyranose. Agarose is made up of your galactose plus anhydrogalactopyranose. Okay, this is important. Just they can ask in your viva that what is agarose made up of? Agarose made up of galactose and anhydrogalactopyranose. Okay, and it is all used in the protein and nucleic acid electrophoresis. Okay, pectin, you know, pectin is the main content is the galacturonic acid. Okay, it is used in the food as a gelling agent. This is the food in the gelling agent. Okay, and it is also a dietary fiber. Understood. So you got an idea about pectin. Okay, let's talk about the glycosaminoglycans. You can get anything about a long question, or you can get a short note. Okay, this can be a long question itself. Okay, GAGs is a very huge topic, or the mucopolysaccharides. Okay, so heteropolysaccharides are composed of repeating unit disaccharide. Okay, so it has two parts. It has an amino acid. It is not amino acid. It has an amino sugar. Okay, and a uronic acid. Amino sugar and a uronic acid. Okay, amino sugar. It may be glucosamine. It may be glucosamine, or it may be galactosamine. Okay, and uronic acid, it may be D-glucuronic acid. Or it can be L-iduronic acid. Okay, and this GAGs, this contain negatively charged groups. Negative groups are there in your GAGs, like your COO minus group is there, SO4 minus group is there, acetyl group is there, etc. Okay, which cause repulsion from each other, and this repulsion is beneficial biochemically. Okay, because this will be responsible for the slippery action of them. Okay, this glycosaminoglycans, these have a slippery action, due to which they are used as a lubricants. Okay, like your hyaluronic acid and mucus, these act as lubricants, and this also act as a shock absorbent in your synovial fluid, in extracellular matrix and all. Okay, so let's classify the GAGs. So first one is your hyaluronic acid. Okay, hyaluronic acid is made up of your N-acetylglucosamine. Okay, N-acetyl, I am writing Na, and glucosamine, N-acetylglucosamine plus D-glucuronic acid. Okay, and it is the only one which has no sulfate group. Okay, no sulfate group is present. Okay, this is important. So hyaluronic acid, N-acetylglucosamine, and D-glucuronic acid, and no sulfate group. Okay, it is present in a synovial fluid. Okay, it's present in a synovial fluid and a vitreous humor. Okay, and it is acting as a lubricant, and your shock absorbent, and also in a cell migration and wound repair. Okay, there's various uses. Okay, second one is your chondroitin sulfate. After hyaluronic acid, we have the chondroitin sulfate. So chondroitin sulfate, it is made up of N-acetyl. N-acetyl is constant. Okay, we have the galactosamine in this time. Just change your glucosamine to in your hyaluronic acid to galactosamine. Okay, and your D-glucuronic acid is again present. Okay, D-glucuronic acid. Chondroitin sulfate is present in your cartilage. Okay, in your bone, in your tendon. Okay, cartilage, bone and tendon, and it gives you shape and structure. okay, to your body tissues and all, okay, to your cartilage, bone and tendon. Then we have dermatan sulfate, okay, dermatan sulfate, okay. This is made up of just don't change the N-acetyl galactosamine. So N-acetyl galactosamine is constant, okay, plus you will change the D-glucuronic acid will be converted to your L-iduronic acid, L-iduronic acid, okay. This is your dermatan sulfate, and this is present in your skin, okay, this is present in your skin, and this is also giving shape and structure. Talking about the fourth one is heparin. Heparin is an anticoagulant, okay. It is made up of another substance which is called as a glucosamine, not N-acetyl, it's just your glucosamine, okay. And any acid may be present, like your glucuronic acid may be present or your iduronic acid may be present, okay, it depends. And heparin is secreted from your lungs, it's secreted from your mast cells, it is present in the blood, skin, okay, etc. It is the anticoagulant, and it also causes the release of LDL. Let's say this is a capillary, so it will also cause the release of LDL from the capillaries, okay. Fifth one, after heparin we will talk about the heparan sulfate. Heparan sulfate is composed of your glucosamine. The same glucosamine is there, okay, and the same iduronic acid or glucuronic acid. So the heparin and heparan sulfate have your same composition, okay, and this is present also in your skin, kidney, okay, mainly in basement membrane, okay. The negative charge of basement membrane is due to this, okay. This is responsible for the negative charge on your basement membrane. This is responsible for your negative charge on the basement membrane. After this, let's discuss the keratan sulfate. After your heparan sulfate, we will have your keratan sulfate. Keratan sulfate comprises of your N-acetyl glucosamine, N-acetyl glucosamine, plus your galactose, okay. Just remember this constituent, N-acetyl glucosamine, and galactose is present in keratan sulfate. This is present in your CC region, your cornea and your cartilage, okay. Keratan sulfate, K is just sounding like C and C, okay. So it is present in your cornea and your cartilage, okay. This is responsible for your transparency of your cornea, okay. Understood? Okay, so we discussed about all the glycosaminoglycans, okay. We have two syndromes, we have two disorders related with the glycosaminoglycans, okay. Let's discuss these two syndromes. We have two syndromes called as your mucopolysaccharidosis, okay. We have your Hurler syndrome and your Hunter syndrome. So let's discuss them. So here we have mucopolysaccharidosis 1H, which is called as your Hurler syndrome, okay. MPS 1H is your Hurler syndrome. You can get a short note on this. It is a double heterozygous nonsense mutation in your IDUA gene. What is IDUA gene? In chromosome number 4p you have a gene, okay, which is alpha L iduronidase, okay, which will encode the enzyme alpha L iduronidase, okay. Here you will see multiple organ and tissue involvement, usually death by the age of 10 years. What are the manifestations? We have hepatosplenomegaly, coarse facial features, you can see corneal clouding, large tongue, skeletal dysplasia, nasal discharges, and ear infections, okay. And the treatment is via the stem cell transplantation, okay. So you will have skeletal abnormalities, hepatosplenomegaly, okay, and your ear, nose, throat, ENT problems also, okay. Then you have a sky disease, okay, which is similar to your MPS 1H, okay. This is not much important. Manifestations, you will have joint stiffness, aortic valve disease, corneal clouding, mild diastosis, okay, and your normal intelligence and stature will be there, okay. The mental retardation is not present in the sky disease. And then you have mucopolysaccharidosis 2, which is called as a Hunter's disease, MPS 2H, which is the deficit of your iduronate L sulfatase enzyme, ILS enzyme, okay. ILS enzyme is like your deficit, okay. Iduronate L sulfatase, okay. This enzyme is deficit, and the manifestations exclusively in males, okay, and the symptoms are same as MPS 1H, but lack of corneal clouding. So if you can find the corneal clouding, it will be your MPS 1H, but if it does lack of corneal clouding, okay, then there will be your Hunter syndrome, okay, your Hunter's disease, okay. And there will be slower CNS deterioration and group skin papules, okay. Papules will be present in the skin. You can treat this with stem cell transplantation, but this therapy is under research now, okay. So these two were the mucopolysaccharidosis, okay. Let's talk about another important concept, which is your glycemic index. So we are talking about your glycemic index. So glycemic index, it is a value to measure how much specific food increases your blood sugar level, how much a food is increasing your blood sugar, okay. This is the measure, okay, blood sugar level, okay. It is a system assigning a number to a carbohydrate containing the foods according to how much the food increases your blood sugar level, okay. What is the importance is that we should check the glycemic index of food before eating the food, so as to lose weight, okay. And it is also useful in the management of your disorders like your obesity and your diabetes mellitus and coronary artery diseases, okay. For the management of this, glycemic index is important, okay. So foods like low glycemic index, okay, low GI. Okay, we have food having medium GI, medium glycemic index. Okay, and we have foods having high glycemic index. Okay, which food have low GI? We have the green vegetables. Okay, green vegetables, green veggies, and you can eat eggs. Eggs having the low GI. Medium GI are present in sweet corn, okay, sweet corn and bananas, oats. Okay, sweet corn, bananas, oats, these all have your medium glycemic index. But high glycemic index is present in white rice, which is a staple food of India. Okay, so in India, obesity and diabetes mellitus are becoming common. Okay, potato and bread. Okay, bread and your sugary foods, or sugar items, and your drinks, your canned juice. Okay, this all. Okay, the glucose has a GI value of your 100. Okay, and the white rice you are eating have a GI value of 87. Okay, your ice cream, okay, this also has a higher glycemic index, about 51. Okay, your kidney beans have a GI index of less, that is 24. Your soya bean, this is also good. Okay, it has about 16. Kidney beans have 24, soya bean has 16. Okay, so you can eat this items, okay, and avoid this glucose containing foods and your ice creams. Okay, understood? So this can come as a short note. Your glycemic index can be asked as a short note. We discussed it, and let's now discuss the dietary fibers also. Dietary fibers. So what are dietary fibers? These are plant components that are not broken down by human enzymes. You can't break them. Okay, dietary fibers, there are many dietary fibers, like a cellulose. Like your cellulose is there, hemicellulose is there. Okay, hemicellulose is there. Your pectin is there. Your lignin is there. Okay, your gums. Okay, then starch. Your oligosaccharide, lactulose. This all. Okay, what are the sources of the dietary fibers? How you can get the dietary fibers? The fruits are good sources of dietary fibers, all the fibrous fruits and all. Okay, green veggies are also good sources. Okay, grains, you should eat more and more grains. Okay, grains, legumes, oats. Okay, these all are rich in dietary fibers. Oats are very good sources of dietary fibers, which are commonly now people are eating more and more oats. Okay, rye also, RYE. Okay, root tubers, almonds and all. Okay, what is the mechanism of action of the dietary fibers? This can cause a bulking. Okay, this can cause a bulking of stools. Okay, bulking of stools means the water content is absorbed, the stools are bulky. Okay, this can cause the viscosity, increase in viscosity. Okay, and have fermentation activities also. So what is the importance? The importance is what it can be asked in the short note. Okay, importance. So the short note can be define dietary fibers and enumerate the importance of the dietary fibers. Okay, so it can increase your bulk of stools. Okay, it can relieve your constipation. Constipation is helpful in your weight loss. Okay, it decreases the absorption of glucose from intestine. Okay, and so it is helpful in the diabetes mellitus and obesity. Okay, this decreases also absorption of your cholesterol from your intestine again. Okay, and it reduces the risk of colon cancer. Okay, it reduces the risk of colon cancer, reduced risk, and it is also helpful in your GI disorders, gastrointestinal disorders. Okay, so you can be asked about the dietary fibers also. Okay, after this let's learn about your metabolism of carbohydrate. Okay, let's go on to the carbohydrate metabolism. Okay, after the carbohydrate chemistry, let's move on to the carbohydrate metabolism. Okay, so welcome to carbohydrate metabolism. Okay, first we're talking about your glycolysis. So glycolysis, the pacemaker enzyme is your phosphofructokinase, PFK. Okay, and in this process the glucose is converted to glucose 6-phosphate in the presence of hexokinase or glucokinase. This is inhibited by insulin, and this is stimulated by insulin and inhibited by glucagon. So insulin will stimulate your glycolysis, reducing the blood glucose level. The glucagon will inhibit your glycolysis, the glucokinase or hexokinase. Okay, increasing the blood glucose level. Okay, difference between hexokinase and glucokinase is that hexokinase acts on all hexose, where glucokinase is specific on glucose. Hexokinase is low Km value, so it has high affinity. Okay, glucokinase is high Km value, so it has low affinity. We'll see about the Km values in the enzyme chapter. Okay, hexokinase is not inducible, but glucokinase is inducible. Okay, hexokinase is present in all tissues, but glucokinase is present only in your liver and your pancreas. Okay, this glucose 6-phosphate in the presence of isomerase get converted to fructose 6-phosphate. Okay, and this fructose 6-phosphate is acted upon by phosphofructokinase 1, PFK1, in the presence of ATP, where ATP is utilized. ATP is utilized in the first step also. From glucose to glucose 6-phosphate is glucose 6-phosphate, and from fructose 6-phosphate to fructose 1,6-bisphosphate also, where ATP is utilized. Okay, and your phosphofructokinase PFK1 enzyme is active upon this. Okay, this is the rate limiting step for glycolysis. Okay, and this PFK1 is activated by your insulin and AMP, inhibited by your glucagon, citrate, ATP, and your CAMP. Okay, And if the fructose 6 phosphate is acted upon by PFK2, phosphofructokinase 2, okay, it has another isoform PFK2, then it is forming a fructose 2,6 bisphosphate, not fructose 1,6, it's forming fructose 2,6 bisphosphate, which will again activate your PFK1. So the major product will be fructose 1,6 bisphosphate only. Okay, and the aldolase, the fructose 1,6 bisphosphate aldolase will act, which will convert it into DHAP and glyceraldehyde 3-phosphate, which are interconvertible by the phosphotriose isomerase enzyme. Okay, in the glyceraldehyde 3-phosphate, again the glyceraldehyde 3-phosphate dehydrogenase acts, which converts the NAD plus to NADH plus H plus. Okay, and forms a 1,3-bisphosphoglycerate. Okay, 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate again enzyme will act 1,3-bisphosphoglycerate kinase. Okay, it will form a 3-phosphoglycerate and ATP is synthesized. Okay, so this is called as your SLP, substrate level phosphorylation. Okay, this step is called as your SLP. And after this 3-phosphoglycerate, the mutase will act. Mutase will convert the 3-phospho-3-PG, okay, into 2-phosphoglycerate, 2-PGA. Okay, 2-PGA, which enzyme acts? Enolase acts. Enolase will convert it into PEP. Okay, phosphoenol pyruvate. And on this phosphoenol pyruvate, where pyruvate kinase will act, which will convert it into pyruvate. And on this pyruvate, if there is anaerobic conditions, then LDH, the lactate dehydrogenase will act and convert into pyruvate into your lactate, where NADH plus H plus is converted into NAD plus. Okay, in this step also, in PEP to pyruvate, your ATP is synthesized because kinase enzyme is there. Wherever there is kinase enzyme, the ATP will be synthesized. Okay, and this pyruvate kinase is again activated by your insulin and AMP, and deactivated by your glucagon, ATP, and your CAMP. Okay, important statement is that this enolase this is inactivated by fluoride, so it is used in glucose estimation. Okay, use the gray top vial. Okay, the vial, the vial, the container. Okay, this container, this will have a gray top. Okay, the gray top will be there. And what is there in the gray top vial is that it contains your sodium fluoride. It contains your sodium fluoride is to potassium oxalate in 1:3 ratio. Okay, in 1:3 ratio. And this sodium fluoride, and this has a fluoride ion which will inhibit enolase, so glycolysis won't occur in this glucose estimation vial. So as to prevent glycolysis, we use sodium fluoride and potassium oxalate. Potassium oxalate is a precipitating agent. Okay, it will precipitate it. Okay, understood? Okay, so energetics of your glycolysis, aerobic glycolysis, we have 2 NADH, okay, which will produce 5 ATP. 1 NADH produces 2.5 ATP, so 2 NADH generation, that will produce your 5 ATP. Okay, 2 substrate level phosphorylations have occurred, which will generate 4 ATP. Okay, then we have your utilization of 2 ATP here. Okay, like 1 ATP is used here, 1 ATP is used here. Okay, utilization of 2 ATP, so you have 7 ATP as the net gain. And in anaerobic condition, there will be no NADH formation, so this 5 ATP must be taken out. So 4 ATP from SLP and 2 ATP from your 2 ATP are your lost. Okay, so you get a net gain of 2 ATP. Okay, so at what steps ATP are utilized? At here, ATP is utilized at here, step number 1. Step number 3, ATP is utilized. Okay, and NADH is NADH converts into NADH plus H plus in this step, and glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. And when the PEP is converted to pyruvate, okay, here ATP is synthesized. Okay, and from your pyruvate to lactate, okay, this NADH plus H plus changes to NAD plus. Okay, in anaerobic conditions. So SLP, this step is also an SLP step where ATP is synthesized. Okay, and this step is also an SLP step where ATP is synthesized. Okay, 2 SLPs. Talking about another cycle, which is a Rappaport-Lubering cycle, or RL cycle. What happens? Just focus on whichever part I am highlighting. Okay, Rappaport-Lubering cycle. I'm talking about your Rappaport-Lubering cycle or your RL cycle. Okay, so talking about your RL cycle, follow this blue highlighter. Okay, this 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate was there. Okay, this is acted upon by a mutase enzyme, which will convert it into 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate or 2,3-diphosphoglycerate. We have read in physiology also that 2,3-diphosphoglycerate it has an effect on your oxygen hemoglobin dissociation curve. Okay, so mutase acts 2,3-bisphosphoglycerate, and this 2,3-bisphosphoglycerate again in the presence of phosphatase converts into 3-phosphoglycerate. One phosphate group is broken down. Okay, it forms your 3-phosphoglycerate. Okay, and so this cycle is called as RL cycle, Rappaport-Levering cycle. Okay, it is due to the it is forming the generation of your doing the generation of your 2,3-bisphosphoglycerate. Okay, it is occupying the central pocket of hemoglobin and helps to unload the oxygen. Okay, this 2,3-bisphosphoglycerate acts as the buffer in your RBC. Okay, and it increases in anemia, high altitude. Fetal hemoglobin has increased 2,3-bisphosphoglycerate. Okay, so increased level of 2,3-bisphosphoglycerate will cause your left shift of your hemoglobin, oxygen hemoglobin dissociation curve. Okay, it also increases in your COPD. Okay, afterwards let's talk about a PDH complex. Your PDH complex, so we have a link reaction which is linking your glycolysis as well as a Krebs cycle. Okay, so we have a PDH complex which is helping in the link reaction. Okay, so here first 3 enzymes are there and your 5 coenzymes are utilized. Okay, 3 enzymes and 5 coenzymes. 3 enzymes, we have first enzyme your pyruvate dehydrogenase, which is active as dephosphorylated state. Then your dihydrolipoyl transacetylase DTA and your dihydrolipoyl dehydrogenase, your DDH. Okay, 3 enzymes are there. 5 coenzymes, we have NAD plus, FAD, TPP, CoA and lipoic acid. NAD plus, FAD, TPP, CoA and lipoic acid. Okay, so here pyruvate is converted in the presence of E1, your first enzyme, to CO2. Pyruvate is converted into your CO2. Okay, due to this your TPP is converted to your hydroxyethyl TPP. This hydroxyethyl TPP is again converted into TPP in the presence of another enzyme which is called as a dihydrolipoyl transacetylase E2. Okay, due to this enzyme also your oxidized lipoamide is converted to your acetyl lipoamide, which in turn converts into reduced lipoamide. Okay, this acetyl lipoamide, when it converts to the reduced lipoamide, the CoASH is converted into acetyl CoA. Okay, so here so acetyl CoA is generated. Okay, we require the acetyl CoA and here it is generated. Okay, and this reduced lipoamide again in the presence of the third enzyme, that is E3, that is your dihydrolipoyl dehydrogenase. Reduced lipoamide converts into your oxidized lipoamide again. And due to this enzyme also the FAD is changed into a FADH2. Okay, this gets back again to FAD, and due to this NADH plus H plus gets converted into a NAD plus. Okay, this NAD plus again goes back into NADH plus H plus. Okay, and due to this NAD plus when changes into NADH plus H plus, okay, the NADH it will go into ETC and produce 2.5 ATP. So 2.5 ATP is produced in your link reaction also. Okay, and we have a syndrome here. When there is TPP deficiency due to deficient vitamin thymine. Okay, thymine is deficient. Okay, due to thymine absorption, due to decreased thymine absorption, the TPP is decreased. Okay, due to TPP decreased, as the TPP is decreased. Okay, so this hydroxyethyl TPP, this is also not formed. Okay, so it is called as a Wernicke-Korsakoff syndrome, Wernicke-Korsakoff psychosis, or your Wernicke-Korsakoff syndrome. Okay, you can remember your the features by the mnemonic GOAMP. Okay, GOAMP. Okay, you can remember this. It is seen in mainly in chronic alcoholics. Okay, chronic alcoholics. G stands for global confusion. O stands for your ophthalmoplegia. A is for ataxia, the drunken gait. M is for your memory loss, and P is for your psychosis. Okay, these all symptoms are due to a decreased ATP synthesis because the TPP is not there. Okay, so this whole pathway will be affected and this 2.5 ATP generation will not be there. Okay, understood. After your link reaction, let's move on to your Krebs cycle or your TCA cycle. Okay, Krebs cycle or your TCA cycle. So what happens is that first of all, okay, we have your, okay, let's start. We have your acetyl CoA, which combines with the OAA and forms in the presence of a citrate synthase. It forms a citrate. Okay, citrate. In the presence of citrate synthase, it forms a citrate. Okay, and this acetyl CoA, it's converted back to a CoASH. Okay, here the CoASH was converted into acetyl CoA, but here the acetyl CoA your joins with your OAA in the presence of citrate synthase converts into a citrate. Okay, this citrate in the presence of aconitate, enzyme, okay, it's converted into your isocitrate. This isocitrate in the presence of your isocitrate dehydrogenase for NAD plus is converted into NADH plus H plus. Dehydrogenation reaction is occurred. Okay, isocitrate, it is converted into oxalosuccinate. In this oxalosuccinate, isocitrate dehydrogenase is again acting. Okay, and it is converted into alpha ketoglutarate. The CO2 is released. Okay, the CO2 is released. This alpha ketoglutarate is changed into a succinyl CoA. Okay, what changes are occurring in this step is that your NAD plus is changed into NADH plus H plus. Enzyme is the alpha ketoglutarate dehydrogenase enzyme. Okay, the NAD plus is changing into NADH plus H plus, and the CoASH is changing into your O2. Okay, this is changing into your O2. Now you have the succinyl CoA. Okay, now there will be the utilization of GTP and there will be GTP synthesis. Okay, GTP synthesis. Okay, and this GTP will again convert into a GDP. This is called as a coupled reaction. So succinyl CoA in the presence of your thiokinase enzyme converts into a succinate, okay, and there will couple reaction will occur in this step. This succinate, in the presence of succinate dehydrogenase again, where FAD is changed to FADH2. Here one step you got where FAD is changed to FADH2. Okay, in the presence of succinate dehydrogenase, succinate is converted into a fumarate. Okay, and the fumarate, in the presence of a fumarase enzyme, fumarate in the presence of a fumarase enzyme, H2O is added. It is converted into a malate. Okay, and malate, in the presence of a malate dehydrogenase again, malate is changed into your oxaloacetic acid. Malate is changed to oxaloacetic acid. Malate dehydrogenase again, the NAD plus is changed to a NADH plus H plus. Okay. So this is the whole Krebs cycle. Just practice drawing this cycle again and again. You can take a screenshot if you want. Okay, I have written many things here. Okay, which will be easy for your revision purpose. Okay, let's talk about the energetics of your Krebs cycle. Okay, energetics. So you have the generation of 3 steps where there is a generation here. One step you saw, and here one step there is the NADH generation. Okay, and here also you have the NADH generation. So you have 3 steps, 3 NADH, which will give 7.5 ATP. One FADH generation, one FADH gives one FADH generation. Okay, one FADH2 generation, which will give you at least 1.5 ATP. Okay, and one GTP generation, also one GTP generation, which will give you 1 ATP. So if you do the total, you get 10 ATP. So net gain of your Krebs cycle is your 10 ATP. Understood? After that, we have very important things which are given in your Krebs cycle. Okay, so Krebs cycle is a catabolic cycle as an anabolic cycle. Okay, so it forms a few substances. Okay, like from the acetyl CoA, your on subsequent steps your CO2 is formed. Okay, so it is also called as your catabolic role. So it has a catabolic role. Okay, and what is the anabolic roles? Okay, from a citrate, acetyl CoA can be formed. This is the anabolic role. From alpha ketoglutarate, your glutamate and GABA can be formed. These are your anabolic role. Okay, and the succinyl CoA. Okay, the enzymes for the heme synthesis and ketolysis. Enzymes for the heme synthesis and ketolysis are formed. Okay, and these are your anabolic roles. And from OAA, your aspartate is formed. Okay, OAA aspartate is formed. These are your anabolic roles. Okay, then to replenish the contents of your Krebs cycle, we have anaplerotic reactions. These anaplerotic reactions are mainly replenishing reactions, which are replenishing the intermediates of your Krebs cycle. So anaplerotic reactions. So anaplerotic reactions are generally replenishing your intermediates of your Krebs cycle. Like your OAA, if it is finished, your OAA is again formed by aspartate. Is again formed by aspartate. You know about the transamination reactions. Okay, then the OAA can be formed from your aspartate. And another thing, the pyruvate can form a malate as well as oxaloacetic acid. Okay, malate as well as oxaloacetic acid. Okay, so pyruvate, your phenylalanine, tyrosine, and from your urea cycle intermediates, you can replenish your fumarate. Okay, fumarate can be formed from your phenylalanine, tyrosine, and from your urea cycle also you can form a fumarate. Okay, you'll see when you discuss the urea cycle. Okay, propionyl CoA will be able to form a succinyl CoA. Propionyl CoA can form a succinyl CoA, and valine, methionine, this also can form a succinyl CoA. Your valine and methionine can also form a succinyl CoA. And your glutamate, this glutamate can again, your glutamate, arginine, proline, histidine. Glutamate, arginine, proline, and histidine, this can again form your alpha ketoglutarate. So these are your anaplerotic reactions. Then we'll talk about what are the inhibitors of your Krebs cycle. Okay, 3 inhibitors are there. Like just follow the green highlighter which I am marking with. Okay, the fluoroacetate, this inhibits your aconitate enzyme, which will prevent your conversion from citrate to isocitrate. Okay, fluoroacetate inhibits the aconitate enzyme. And your another is your arsenite. Another is arsenite, which is inhibiting your alpha ketoglutarate dehydrogenase enzyme. Arsenite inhibits alpha ketoglutarate dehydrogenase. Okay, and malonate is competitively inhibiting the succinate dehydrogenase enzyme. This is an example of a competitive inhibition. Okay, so these are your inhibitors of your Krebs cycle. Okay, these are inhibitors of your Krebs cycle. So after the Krebs cycle, we'll go to your gluconeogenesis. Okay, the formation of glucose from other compounds like a lactate, glycerol, propionyl CoA, and glucogenic amino acids. So we'll see the formation of glucose from lactate, glycerol, propionyl CoA, and glucogenic amino acids. Okay. Talking about gluconeogenesis, it is the reversal of your glycogenesis, not the exact reversal, but you can just get an idea of how to write the pathway of your gluconeogenesis. Okay, it is the source of glucose after depletion of your glucose stores. Okay, so we can form glucose from lactate, glycerol, propionyl CoA, and glucogenic amino acids. So let's start from the lactate. Okay, so lactate, you can convert the lactate into your pyruvate. Okay, using the enzyme lactate dehydrogenase. In its NAD plus is changed to NADH plus H plus. Okay, you can form your pyruvate. Okay. And if you have propionyl CoA, you can use propionyl CoA carboxylase to change it into methyl malonyl CoA. Okay, and this methyl malonyl CoA This propanol CoA carboxylase acts in the presence of ATP, biotin, and CO2. Can change this propanol CoA to methyl malonyl CoA. This in the presence of methyl malonyl CoA mutase, okay, and vitamin B12. It is changed to succinyl CoA, and you can use the succinyl CoA in TCA cycle, generate OAA, okay, and this OAA can be converted into your pyruvate, okay. So you can get the pyruvate from here also. And if you have your amino acids like your isoleucine, valine, and methionine, okay, isoleucine, valine, and methionine, this amino acids can be converted directly into a succinyl CoA, which in TCA cycle generate OAA and pyruvate. So you can convert this amino acids to pyruvate, okay. And if you have glycerol, okay, then you can use glycerol kinase enzyme to convert it into glycerol 3-phosphate. This glycerol 3-phosphate can be converted to DHAP by glycerol 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase converts the glycerol 3-phosphate to DHAP. You can use isomerase to convert the DHAP into PGAL, and this PGAL, after subsequent steps of glycolysis, you can convert it into pyruvate, okay. Pyruvate. So we got the common thing pyruvate from these three pathways, okay, and the pyruvate can be converted in the presence of pyruvate carboxylase enzyme into OAA. This OAA can be converted in the presence of PEP carboxykinase enzyme to PEP. Again, just now reverse the glycolysis. PEP in the presence of enolase 2-PGA, 2-PGA in the presence of mutase 3-PGA, 3-PGA in the presence of 1,3-bisphosphoglycerate kinase. ATP is changed into ADP here. Just look at this. ATP step has been done reversal here. Okay, in glycolysis 1,3-bis-PGA was converting into 3-PGA in the presence of kinase, and ATP was being formed. But here 3-PGA is reverse. That is a reversal of the steps. Okay, this 1,3-bis-PGA in the presence of glyceraldehyde 3-phosphate dehydrogenase converts into 3-phosphoglyceraldehyde. Okay, which in presence of isomerase can convert into DHAP also. This 3-PGL in the presence of aldolase is fructose 1,6-bisphosphate. To convert the fructose 1,6-bisphosphate into fructose 6-phosphate, okay, and now you cannot use your phosphofructokinase enzyme. Now you have to use another enzyme, that is your fructose 1,6-bisphosphatase, okay, fructose 1,6-bisphosphatase, which uses H2O and gives out PI. Okay, this fructose 6-phosphatase, you can use your isomerase again to change it into glucose 6-phosphate, and the glucose 6-phosphate can be changed into glucose by another enzyme. Okay, you can't use your hexokinase or glucokinase. You will use glucose 6-phosphatase. Okay, so what are the special enzymes here? All the enzymes are the glycolytic enzymes only, but the special enzymes are your pyruvate to OAA, which is catalyzed by your pyruvate carboxylase, then OAA to PEP, which is catalyzed by your PEP carboxykinase, not carboxylase, PEP carboxykinase. Okay, and then your fructose 1,6-bisphosphate to fructose 6-phosphate by your fructose 1,6-bisphosphatase, and your glucose 6-phosphate to glucose by glucose 6-phosphatase. Okay, understood? So this is the whole pathway of your gluconeogenesis. So you can write the pathway if it is written, write how you can make your glucose from your lactate. So you have to write this whole pathway. Okay, you don't have to write all this propanol CoA and all. Okay, so you can write the pathways like that. Okay. Then what are the important things again? Okay. What are the important things? Then you have another pathway. Just see, you have the glucogenic amino acids. Okay, this glucogenic amino acids can also give you glucose. Okay, so we saw about the isoleucine, valine, and methionine. Okay, these were directly converting to succinyl CoA, which can enter the pathway like this. Okay, then another amino acids like alanine, glycine, serine, and cysteine, AGSC. Okay, these are converted directly into your pyruvate. Okay, then your aspartate and asparagine can directly convert into your OAA. Okay, and this phenylalanine, tyrosine, asparagine, and aspartate can convert into fumarate, and the fumarate can go into TCA cycle, okay, and give you OAA. Okay, this is another pathway of your glucogenic amino acids giving us glucose. Okay, and another thing is your glutamate, glutamine, arginine, histidine, proline. Okay, this can convert into alpha ketoglutarate. This alpha ketoglutarate will again go into TCA cycle, generating your OAA, and it will enter the pathway like this. Okay. Another question is that why the ethanol causes hypoglycemia. So ethanol the ethanol is changed into acetaldehyde by alcohol dehydrogenase. Okay, which converts the NAD plus to NADH plus H plus. Okay, when the detoxification of ethanol is occurring, this acetaldehyde again uses acetaldehyde dehydrogenase to convert into acetate. Okay, acetate, and this will also require NAD plus changed into NADH plus H plus. Due to this NADH are generated. Okay, this NADH will favor the conversion of pyruvate to malate, and this malate converts into your oxaloacetate. Okay, and this pyruvate also changes into a lactate, which will inhibit your gluconeogenesis. Okay, if gluconeogenesis is inhibited, then you will suffer from hypoglycemia. Okay, so alcohol causes hypoglycemia like that. Okay, After talking about your gluconeogenesis, let's talk about your glycogenesis. Glycogenesis is a synthesis of glycogen. Okay, so we are now into your glycogen metabolism. Glycogenesis, synthesis of glycogen. Okay, this is occurs in the cytosol of your liver and muscles, in which glucose, in the presence of glucokinase, is converted to glucose 6 phosphate. Okay, ATP is utilized to ADP. ATP is utilized. Okay, this glucose 6 phosphate. There is no enzyme like the glycolytic pathways is not there. Okay, so you have glucose 6 phosphate mutase, okay, which will convert it into glucose 1 phosphate. This glucose 1 phosphate is acted upon by an enzyme UDP glucose phosphorylase, UDP glucose phosphorylase, okay, which will activate the glucose and form the UDP glucose. Okay, and this UDP glucose, again, this UDP glucose is acted upon by glycogen synthase, which is the key enzyme of your glycogenesis. Okay, we are talking about the glycogenesis. And this glycogen synthase is active in dephosphorylated state, as I stated earlier. Okay, this UDP glucose is active upon glycogen synthase, which will add the activated glucose to your glycogen primer, to your glycogen. Okay, which will add your glycogen, which will add your glucose to the glycogen primer or your glycogen. Then it is acted upon by glycogen synthase, which will again cause your addition up to 10 to 12 glucose residues. It will add 10 to 12 glucose residues to the glycogen primer. Again, it is acted upon by branching enzyme. This branching enzyme will form the branching points. Okay, it will form the branching points, like this glucose chain is there, and it will form your branching points here. Okay, and by the alpha 1,4 linkages. Okay, alpha 1,6 linkages. Sorry. Okay, this form the branching points. Okay, branching enzyme. And if there is deficiency of branching enzyme, we will suffer from Anderson's disease. Anderson's disease, also called as amylopectinosis, which is a glycosomal your glycogen storage disorder type 4. Okay, understood. So you can have one, that is a von Gerke's disease, two is the Pompish disease. Okay, and Anderson's disease is there. Okay, and Cory's disease is there, and McArdle's disease. These are your glycogen storage disorders. We'll see about them. Then branch points are formed, alpha 1,6 linkages. Okay, then elongation and branching occurs by glycogen synthase and your branching enzyme, and you get your glycogen. Understood. Then we discuss about glycogenolysis, that is a breakdown of glycogen. It occurs in the cytosol, your liver and muscles. Okay, this was occurring in your cytosol of liver and muscles, and this again will occur in your cytosol of liver and muscles. What happens is that glycogen, in the presence of your glycogen phosphorylase, which is the key enzyme of your glycogenolysis, okay, which is active in a phosphorylated state, glycogen, from the glycogen, glucose 1 phosphate residues are being removed. Okay, which will lead to the formation of limit dextrins. Okay, this is a limit dextrin. Okay, limit dextrin. This limit dextrin, in the presence of transferase enzyme, that is your alpha 1,4 and alpha 1,6 alpha 1,4 glucan transferase, also called as alpha 1,4 glucan transferase. Okay, a tripeptide, sorry, not the tripeptide, the 3 residues of glucose. Okay, these 3 residues of glucose are transferred into the straight chain. Okay, these 3 residues are transferred into a straight chain. So these 3 residues have come here in the straight chain. Okay, so we have this. Then you have the debranching enzyme, which will remove the branch point. Okay, and again acted upon by alpha 1,6 glucosidase. Okay, which is also called as a lysosomal acid maltase, which has the same function as the debranching enzyme. Okay, which will form a linear chain. And in the linear chain, again glycogen phosphorylase will act. Your glucose 1 phosphate residues are removed. Okay, and your glucose 1 phosphate is produced at the end. Okay, glucose 1 phosphate is produced at the end, and this glucose 1 phosphate in the presence of mutase converts into glucose 6 phosphate. This glucose 6 phosphate is acted upon by glucose 6 phosphatase enzyme. Okay, which will form your glucose again. So here we have some disorders. Okay, like your okay, the glucose 6 phosphatase deficiency causes a von Gerke's disease. Okay, von Gerke's disease, which is a type 1 glycosomal storage disorder. Okay, it sets in 3 to 4 months of birth. Okay. And so glucose 6 phosphatase is deficient, where there is accumulation of glucose 6 phosphate. Okay, and glucose is less. The glucose is not formed, so there is fasting hypoglycemia. Okay, and there is uric acid accumulation as the glucose 6 phosphate goes to your uric acid pathway. Okay, production of your nucleotide metabolism and all. Okay, uric acid production increases. So triglyceride and cholesterol also increases, and lactic acid also increases. Okay, lactic acid also increases. Okay, why uric acid is being formed? The glucose 6 phosphate goes into an HMP shunt. HMP shunt forms it into ribose 5 phosphate. This ribose 5 phosphate is the precursor of synthesis of IMP, AMP, GMP. Okay, which one degrade? IMP, AMP, GMP, which one degrade? Produces uric acid. Okay, so this is called a hyperuricemia. Hyperuricemia occurs. Okay, And the glucose 6 phosphate, if it is accumulated, also it is forming an acetyl CoA. Is forming acetyl CoA, and this acetyl CoA is the product from which the cholesterol synthesis and the fatty acid synthesis is stimulated. Okay, this acetyl CoA, the substrate upon which cholesterol and fatty acid synthesis will occur. Okay, and the patient will suffer from increased cholesterol and fatty acids. Okay. And in the von Gurkish disease also, we have the clinical features like a doll-like faces. Okay, the face is like a doll-like face. Okay, and we have thin extremities, protuberant abdomen. Abdomen is large, short stature, and hepatomegaly is there. Okay, you don't have splenomegaly in this syndrome, and kidney disorders are also ruled out. Okay, splenomegaly and kidney disorders are not there in your von Gurkish disease, which are of diagnostic importance. Okay, we have hepatomegaly but not splenomegaly. Treatment is a protein-rich diet. You have to give protein-rich diet. Allopurinol. This will reduce the level of your uric acid. Okay, and your statin drugs, which will reduce your cholesterol level. Okay. Then another disorder, which is due to deficiency of this alpha-1,6 glucosidase or lysosomal acid maltase. Okay, this is called as a Pompe's disease, which is both a lysosomal storage disease as well as your glycogen storage disorder. So it can be asked in your viva that which disease is both LSD as well as GSD. This is the Pompe's disease. This Pompe's disease is the GSD type 2. Okay, and deficiency of alpha-1,6 glucosidase or your lysosomal acid maltase. Clinical feature are, first, it occurs in the first few months of birth. Okay, also the patient also hypotonia, loss of tone of the muscles, myopathy. Okay, the floppy baby. Okay, so floppy baby syndrome is also due to Pompe's disease. Okay, failure to thrive and poor feeding, and deafness will also occur. How can you diagnose in the lab? Lab diagnosis is assay. Okay, as acid maltase assay and your biopsy also. Okay, and treatment is by recombinant enzymes, myozyme and lumizyme. Your myozyme and your lumizyme. Understood? This was your Pompe's disease. And if you have debranching enzyme deficiency, we will have Cory's disease, which is your glycosomal storage disorder type 3. Okay, and if you have glycogen phosphorylase deficiency, that is your muscle glycogen phosphorylase deficiency. It is due to your muscle glycogen phosphorylase deficiency. Okay, you will suffer from McArdle's disease, which is your GST type 5, Okay, McArdle's disease. And the clinical features are exercise intolerance will occur. Okay, the person, if he does a few exercises also, he will feel your fatigue. Okay, second wind phenomenon is there. Okay, like delayed starting of any exercises. Okay, second wind phenomenon. And your myoglobinuria, so patient will present with red urine. Okay, myoglobinuria. Okay, and the treatment is diet and exercises. Okay, the symptoms will be treated by your diet and exercises. Understood? So this is this. Regulation of glycogen metabolism. So glucagon and epinephrine, this will stimulate your adenyl cyclase. This adenyl cyclase will cause conversion of ATP to CAMP and PI. Okay, and the CAMP and PI, this will stimulate your protein kinase inactive form to protein kinase active form. This active protein kinase will stimulate your phosphorylase kinase inactive to phosphorylase kinase active. This phosphorylase kinase active form will stimulate your glycogen phosphorylase. Okay, and convert it into glycogen phosphorylase active form. So glycogen will be converted into your glucose. Okay, glycogen phosphorylase converts the glycogen. It will favor your glycolysis. Okay, it will prevent your glycogenolysis. Okay, glycogenolysis, not glycolysis, glycogenolysis. Okay, and the glycogen will be converted into glucose. So there is increase in level of glucose. Okay, so that's why your glucagon and your epinephrine. Okay, glucagon and epinephrine will increase your blood glucose level. Okay, and again you have insulin, which will stimulate your phosphodiesterase, which will convert your CAMP to 5-AMP, which will break down the CAMP to 5-AMP. Okay, and due to this your CAMP concentration decreases. Due to decreased CAMP, your PK, your protein kinase is inactivated. This inactivated protein kinase will stimulate your glycogen synthase. And activate the glycogen synthase. So glucose will convert it into a glycogen, and again it will inhibit your glycogen phosphorylase, due to which your glycogenolysis stops. Okay, so there is formation of glycogen. So insulin, it will stimulate the formation of your glycogen and decreases the blood glucose level. Okay, then you have another metabolism, that is galactosemia, about the galactose metabolism specifically. Okay, so what is the galactose metabolism? So galactose, in the presence of your galactokinase, converts into galactose 1-phosphate. Okay, this galactose 1-phosphate, in the presence of galactose 1-phosphate, uridyl transferase, it converted into UDP galactose. Okay, galactose 1-phosphate is converted into your UDP galactose. This UDP galactose, in the presence of your epimerase, is converted into your UDP glucose. Okay, epimerase, as the galactose and glucose are epimers. Okay, this UDP galactose is converted to UDP glucose by the presence of epimerase. Okay, And here the galactose in the presence of aldose reductase is converted into a galactitol. Okay, if you reduce the galactose, you will get galactitol, which is also called as a dulcitol. Understood? And this dulcitol accumulation in your the lens, okay, this will cause a cataract. The dulcitol will cause a cataract. So talking about if galactose 1 phosphate uridyl transferase is deficiency, okay, this enzyme is deficient, then you will suffer from galactosemia, which is type 1 or classical galactosemia. Okay, the clinical features will be hypoglycemia, okay, vomiting, diarrhea, and cataract will occur. Why cataract will occur? Because the galactitol will be accumulated. Okay, hepatomegaly, jaundice, and mental retardation. Okay, mental retardation will be also seen in the galactosemia. Okay, and talking about your what will happen? Your autosomal recessive disorder. Okay, and due to which glucose 1 phosphate is accumulated. This glucose 1 phosphate, sorry, not your glucose, is galactose 1 phosphate. Okay, this is your galactose 1 phosphate which will be accumulated, not glucose, sorry, there's a correction here. Galactose 1 phosphate, this will accumulate, and this increased galactose 1 phosphate will cause your decrease in bilirubin uptake and conjugation. Okay, and this will lead to increase in unconjugated bilirubin, which will lead to your jaundice. Okay, so there is in galactosemia, there is jaundice also. Okay, increase in unconjugated bilirubin. Okay, and you will suffer from hypoglycemia, of course, and your dulcitol accumulation in lens causes cataract. Okay, and this is autosomal recessive disorder. And you have type 2 galactosemia also, which is the deficiency of the galactokinase enzyme. Okay, in this only cataract is occurring. Okay, in this only cataract will occur. Okay, and you have the type 3 galactosemia also, which is asymptomatic. Okay, doesn't have any symptoms. Let's talk about another pathway, which is called as HMP shunt, also called as a pentose phosphate pathway, or a phosphogluconate pathway, or a monophosphate pathway. Okay, this generates your NADPH as well as your pentose sugar, NADPH and your pentose sugar. Okay, so talking about your HMP shunt, it is a little bit bigger pathway. Okay, let's discuss this. So first you have your glucose 6 phosphate, in the presence of glucose 6 phosphate dehydrogenase. Don't confuse between glucose 6 phosphatase and glucose 6 phosphate dehydrogenase. Okay, this will convert the glucose 6 phosphate into a 6-phosphogluconolactone, and the 6-phosphogluconolactone in the presence of hydrolase will convert into 6-phosphogluconate. Okay, 6-phosphogluconolactone in presence of hydrolase, 6-phosphogluconate. This 6-phosphogluconate in the presence of your phosphogluconate dehydrogenase, converts into ribulose 5-phosphate. Okay, where NAD plus is changed into NADH plus H plus. Understood? So we have G6PDH. Okay, you have hydrolase, then you have phosphogluconate dehydrogenase, and here also NAD plus changes to NADH plus H plus. Okay, understood? This is your oxidative phase. Up to this is your oxidative phase, which occurs in your liver, adipose tissue, and RBCs. Okay, this is an oxidative phase which occurs in all of this liver, adipose tissue, and RBCs. Next, we'll talk about the non-oxidative phase. Okay, this occurs in all your cells. Okay, after your ribulose 5-phosphate, the non-oxidative stage will occur. Okay, this ribulose 5-phosphate changes into, in the presence of epimerase, converts into xylulose 5-phosphate, and in the presence of isomerase converts into a ribose 5-phosphate. Okay, ribose 5-phosphate. This xylulose 5-phosphate and ribose 5-phosphate. So you can just see if you just sum the phosphates, you will get about 10 phosphates. Okay, so in each step you will see is pseudoheptulose 3-phosphate, glyceraldehyde 3-phosphate. Okay, so pseudoheptulose is 7 plus 3 is 10. Okay, then your erythrose 4-phosphate and fructose 6-phosphate. Again, your 4 plus 6, that is 10. Okay, this will occur. Okay, so just see epimerase, xylulose 5-phosphate, and your ribose 5-phosphate. Okay, and in the presence of your transketolase, enzyme, okay, and which requires a TPP as a cofactor. Okay, this is converted into a pseudoheptulose 3-phosphate, and your glyceraldehyde 3-phosphate, pseudoheptulose 3-phosphate and your glyceraldehyde 3-phosphate. Okay, the pseudoheptulose and glyceraldehyde 3-phosphate in the presence of transaldolase, is converted into your erythrose 4-phosphate and your fructose 6-phosphate. Okay, and this erythrose 4-phosphate again combines with your xylulose 5-phosphate. This erythrose 4-phosphate combines with your xylulose 5-phosphate, and in the presence of TPP and transketolase again is converted into your fructose 6-phosphate, and your glucose 3-phosphate. Okay, this glucose 3-phosphate is converted again into your fructose 6-phosphate. Okay, fructose 6-phosphate. We get your fructose 6-phosphate from here. Okay, we get fructose 6-phosphate from here also. Okay, so you get about 5 for fructose 6-phosphate, which can be converted into your 5 glucose 6-phosphates. Okay, so this is the net reaction. Net reaction will be 6 into glucose 6-phosphate plus 12 NADP plus 6 H2O. This is the input. Output is your 5 glucose 6 phosphate. Okay, 1 glucose 6 phosphate is lost. Okay, 12 NADPH. Okay, from NADP plus we get NADPH, 12 NADPH. So NADPH is generated, 12 H plus and 6 CO2, 12 H plus and 6 CO2. Okay, this is the net reaction. What is the function of NADPH? So NADPH is used in a fatty acid, cholesterol and steroid hormone synthesis, steroid synthesis. Okay, it is a free radical scavenger. It maintains the RBC membrane integrity because NADPH, okay, it by the glutathione reductase, it activates the glutathione reductase, which in glutathione converts into a reduced glutathione. Okay, maintenance of the RBC membrane integrity occurs. And it is responsible for a lens transparency and also a cytochrome P450 detoxification. Okay. Let's talk about the regulation of blood glucose, which may come as a long question. Okay, so basically what you have to do, it's a trick that you have to just draw the pathways which I have taught for the regulation of your glycerol metabolism regulation. Okay, this hormonal effects, your glucagon, epinephrine, this whole pathway. Okay, what is happening, how it is increasing your glucose, and this insulin mechanism. Okay, how it is decreasing your blood glucose. Okay, you just write this two pathways. Okay, these two pathways will you'll get some marks, and I will tell how to write. Okay, the long question. So normal blood, first write the values. The normal fasting blood glucose level is about 70 to 110 milligram per deciliter. Your post meal is about 110 to 140 milligram per deciliter, and random is about 70 to 140 milligram per deciliter. Okay, this is the normal blood glucose levels. For regulation, to increase the blood glucose. Okay, this is the whole pathway. The effect of hormones, glucagon and epinephrine. Okay. This glucagon also inhibits your PFK and your PK, which inhibits your glycolysis, and there is no formation of your pyruvate. Okay, so glucagon is also helping by this process. And again the insulin, which will decrease the blood glucose level. So this pathway you have read from your glycogen metabolism I have taught. Okay, and insulin also inhibits your pyruvate carboxylase. Okay, pyruvate carboxylase, which will inhibit your gluconeogenesis. Okay, which will reduce your glucose synthesis. Okay, blood glucose levels. Insulin also activates your PFK and PK, which will favor your glycolysis. Okay, and glycolysis will also decrease your blood glucose level. Understood? Then utilization of glucose in different states. So in well-fed state, when you are getting the good food, okay, all cells will utilize your glucose. Okay, and the glucose will be deviated towards glycogen and fat synthesis also. In post-absorptive stage, the glucose source is a liver glycogen. Okay, the glucose source again is a liver glycogen. Okay, so glycogenolysis will occur up to 4 to 6 hours after meal. Okay, the liver glycogen can produce a glucose. And if you starve, okay, starvation, okay, if you have not eaten for 16 to 18 hours, okay, so glycogen stores are also depleted. Okay, so the glucose source is now gluconeogenesis in liver and kidney. So in liver and kidney, gluconeogenesis will occur. Okay, the four pathways, lactate from propionyl CoA, from your that glycerol, from your short chain fatty acids and all, gluconeogenesis in liver and kidney. And brain will depend upon the glucose and ketone bodies, glucose as well as ketone bodies. And if there's prolonged starvation, you have not eaten for several days, so muscle proteins will start to break down. Muscle proteins will break down by gluconeogenesis and give you glucose, and brain will only depend upon the ketone bodies as the fuel. Okay, so prolonged starvation is dangerous. Okay, renal threshold of glucose is 180 milligram per deciliter. It is asked as in your viva. Okay, it is asked in your viva. And at hypoglycemia versus hyperglycemia, your causes are insulinoma, insulin overdose, and Cushing syndrome. Hyperglycemia have causes like diabetes mellitus. Okay, you have three glands like adrenal gland, pituitary gland, and thyroid gland. If they are if there is hyperadrenalism, or hyperpituitarism or hyperthyroidism, then there is hyperglycemia. If there is hypoadrenalism, hypopituitarism, hypothyroidism, then there is hypoglycemia. Okay. Talking about another disorder, which is a diabetes mellitus. This is an important case-based question. This can be a case-based question. Okay, so I have to read in detail about your diabetes mellitus. So diagnostic criteria, what is the diagnostic criteria of diabetes mellitus is that your fasting blood glucose level, if it is more than 126 milligram per deciliter, your 2-hour blood glucose is more than 200 milligram per deciliter, your random blood glucose is more than 200, and if your HB1AC, glycated hemoglobin, is more than 6.5%. This is the hemoglobin for average of 6 to 8 weeks. Okay, so HB1AC is the glycated hemoglobin, so the average your blood glucose level over 6 to 8 weeks. Okay, what are the cardinal features? You have polyuria, glucose in urine, which will drag your H2O by osmotic effect, and there will be frequent urination. Your polydipsia, due to loss of H2O in urine, so there is dehydration and excessive thirst will occur. Your polyphagia, excessive eating tendency, excessive hunger. To decrease glucose, the person tries to fulfill his glucose demands, and the hunger centers are stimulated, producing hunger. Metabolic changes due to decreased insulin, the increased gluconeogenesis will occur, or increased glycogenolysis will occur, and increased lipolysis will occur, which will increase your blood glucose level. And increased lipolysis will deviate it to fatty acid and glycerol synthesis. Okay, so ketone body and triglyceride levels are also elevated in diabetes mellitus. Okay, complications, we have acute complication and chronic complications. Acute, we have severe dehydration, diabetic ketoacidosis, and increased blood glucose level. Okay, and your chronic complications like a retinopathy, diabetic foot, like your delayed wound healing, and your feelings are affected. Your diabetic foot is tested by some bristles like thing, okay, which is present over your that apparatus. Okay, it is used for treating diagnosing a diabetic foot. Okay, your heart attack, your nephropathy, your neuropathy. Okay, snowflake cataract is seen in diabetes mellitus due to accumulation of your sorbitol. Snowflake cataract is occurring. Glaucoma and sexual problems, these are common in your chronic complications of diabetes mellitus. Then we have two types, type 1 and type 2 DM. Type 1 is called insulin dependent and type 2 is called the non-insulin dependent. Type 1 is basically seen in children like below 20 years. Okay, type 2 in more than 30 years patient. Okay, more than 30 years subject. Okay, 10 to 20% cases are type 1 and 80 to 90% type 2. Insulin production decreases here due to pancreatic destruction. But in type 2, what happens? Decreased production of resistance to insulin has been developed. Okay, in type 1, there is no obesity. In type 2, there is obesity. Okay, type 1 has non-genetic causes, but type 2 has genetic causes, but type 1 is autoimmune. Okay, type 1 DM is autoimmune disorder. Okay, autoantibodies are present due to against the pancreatic cells, your islets of Langerhans, which is causing destruction to your pancreas. Okay, and decreasing your insulin production. And treatment is only insulin, obviously insulin injections, but you can treat your type 2 diabetes mellitus by exercise and diet plans, also insulin also. Okay, talking about diabetic ketoacidosis, which is an important case, in your carbohydrate metabolism, which is an acute complication of your type 1 diabetes mellitus. Okay, lab diagnosis, you will see increased plasma glucose level, Benedict's test positive, Rothera's test for ketone bodies positive. Metabolic acidosis, we learned about metabolic acidosis. Okay, we know about the mnemonic, that is Dilaro. Okay, you know about the mnemonic Dilaro? Yes. The Dilaro about the metabolic acidosis. Okay, the high anion gap metabolic acidosis specifically. Okay, and the hyperkalemia is also seen. Okay, lab diagnosis. If there is decreased insulin, there is increased lipolysis, increase in fatty acid and glycerol, which leads to increased ketone body synthesis. To neutralize, there is a SCO3 minus buffer. Okay, so acidic nature of ketone bodies will decrease the pH, causing a metabolic acidosis. Okay, and to neutralize, we have the HCO3 minus buffer. So HCO3 levels will also decrease. Okay, so there is decreased HCO3 levels also. And to compensate the metabolic acidosis, the respiratory mechanisms are there. A deep rapid breathing will occur, which is called as a Kussmaul's respiration. Okay, so breathing is like deep and rapid breathing. Okay, deep and rapid breathing like this. This is called a Kussmaul's respiration, which is seen in a diabetic ketoacidosis. Okay, clinical features are confusion, loss of consciousness, dehydration, vomiting, abdominal pain, low blood pressure, Kussmaul's respiration. Understood? This is a diabetic ketoacidosis. A little bit about fructose metabolism, and then we finish our carbohydrate metabolism. Your fructose in the presence of your fructokinase, okay, this is changed into, okay, this is fructose in the presence of fructokinase. This is changed into fructose 1-phosphate. What is happening with my tab? Okay, fructose 1-phosphate, and this fructose 1-phosphate in the presence of your aldolase B, this is converted to glyceraldehyde. This glyceraldehyde in the presence of triose kinase is again converted to your glyceraldehyde 3-phosphate. Okay, so deficiency of this fructokinase, if there is a fructokinase deficiency, okay, will suffer from fructosuria. Lab diagnosis, you will see Benedict's and Sellew-Venop positive. Okay, both are reducing sugars and all. And this aldolase B deficiency will also hereditary fructose intolerance, HFI, hereditary fructose intolerance, which is an autosomal recessive condition. Okay, in this fructose 1-phosphate accumulates due to this enzyme deficiency. This fructose 1-phosphate will accumulate. Okay, it will inhibit your glycogen phosphorylase and cause hypoglycemia. So there is hypoglycemia in your hereditary fructose intolerance because fructose 1-phosphate is inhibiting the glycogen phosphorylase. Okay, and there is jaundice and hepatomegaly as well as death. Jaundice, hepatomegaly, which will lead to your death. Okay, so fructosuria and hereditary fructose intolerance. So here we finish our carbohydrate metabolism. Okay. Okay, after finishing carbohydrate metabolism, so like you must be wondering that why I'm doing such in scattered form, because if you just go on revising the reactions, you will get saturated by reading the biochemistry. Like if I discuss your protein metabolism or fat metabolism just after your carbohydrate metabolism, okay, then you can get frustrated after so many reactions, so many enzymes, so many your ATP utilizations where they're occurring and all. Okay, so I'm taking up scattered chapters, like I'll cover all the chapters, but I am just like taking up some of the topics. Okay, topic was not in sequence. Okay, I'm not talking things in sequence. Okay, so we'll talk about the chromatography. So chromatography is a biochemical technique. Okay, the substance which has more affinity towards stationary phase, which is retained in the stationary phase. Okay, we have a stationary phase and a mobile phase in a chromatography. Okay, which has more affinity towards mobile phase, this is retained. Okay, on the basis of this property, we'll separate two components. Chroma means color and graphy means writing. Okay, so I have a mobile phase and a stationary phase. Okay, mobile phase, like your solvent. Okay, solvent or your butanol. Okay, solvent butanol, or this is your mobile phase. Okay, this is your mobile phase, and here you have the paper, your Whitman's paper or the filter paper, your Whitman's filter paper, which will act as your inert media. And what is the stationary phase? The water that is adsorbed on the paper. The water that is adsorbed on the paper is the stationary phase. Okay, so stationary phase, it is more affinity towards stationary phase, will be retained on the stationary phase. Okay, so the dots that you have made in your color, okay, this has more affinity towards the stationary phase. Okay, it will get retained here. Okay, then you have another example of alumina packed column, which is your liquid solid chromatography, example of liquid solid chromatography. Okay, in this the mobile phase is a solvent. Okay, solvent which moves down. Okay, and your alumina is there, which acts as a stationary phase. Okay, alumina is there, which acts as a stationary phase. Understood? So by this, your less soluble parts remain here, and the more soluble part they come down. Okay, so it is a technical procedure for separation of similar compounds by continuous redistribution between two phases, between the stationary and mobile phases. Okay, so let's classify the chromatography based on the mobile phase. We have liquid chromatography and your gas chromatography. Okay, on the basis of a stationary phase, you can have solid type of solid liquid chromatography, or the liquid solid chromatography, liquid chromatography, gas solid chromatography, and gas liquid chromatography. Okay, for solid chromatography, the column with alumina, this is an example of liquid solid chromatography. And we saw the paper chromatography above. Okay, that is the liquid chromatography. Paper chromatography, it is liquid chromatography. Okay, for gas chromatography, we have solid and liquid. So we have gas solid chromatography and I have gas liquid chromatography. Okay, what are the properties that are involved in a chromatography? First is the adsorption. Okay, adsorption is the one component is adsorbed over another component, like alumina. Okay, alumina was being adsorbed. Then partition is that more soluble moves fast and less soluble is retained. Okay, in the paper chromatography and thin layer chromatography, these are based on the partition. These are also called as the partition chromatographies. Okay, then molecular sieving mechanism, in which the columns are packed with gel, so and beads and pores are present. Okay, and separation is based upon the size and shape of the particles. Okay, like sieve like action, smaller particles are retained and larger particles are eluted out. Okay, this is important. You must be thinking otherwise, but here smaller particles are retained, the larger particles are eluted out. Like a gel filtration chromatography is a technique utilizing a molecular sieving. Okay, then ion exchange resins, which are used in the purification of water and all. Okay, then affinity based packed with ligands and antigens for antibodies. Okay, so affinity. So chromatography is also based upon an affinity. So you have a paper importance of chromatography, like your paper chromatography. This is helping in your separation of amino acids and reducing sugars, your purification of carbohydrate, proteins and lipids, enzymes, vitamins and drugs. Okay, purification of all this. Okay, the HPLC is the gold standard method for measuring your measuring analytes. Okay, analytes like your drug, vitamins, hormones, enzymes. HPLC is the gold standard method. High pressure liquid chromatography. Okay, and your screening of inborn errors of metabolism also. So paper chromatography, it is a liquid type partition chromatography. Okay, based upon the partition property of your chromatography. So you have three types here: ascending, descending, and the radial paper chromatography. So in ascending, the solvent moves in the upward direction. And in this descending, the solvents moves in a downward direction, because the paper is bound. The solvent reservoir lies up. Okay, and radial, you just take a filter paper and make a design by making a hole in it. Okay, and you just turn it in a radial fashion. Okay, so the solvent system will move radially, and this will cause a chromatography. So you can see how the radial chromatography is being done. Okay, descending and ascending, you can take a screenshot of these diagrams if you want. Okay, done. Okay, then two dimensional. Okay, you take a paper and put three dots. Okay, you take 5 cm above, you put 3 dots here. Okay, and then you rotate it 90 degree. Okay, and this system will be formed. Okay, this system will be formed, okay, by turning it by 90 degree. Okay, and you can just absorb it like the your chromatogram. Okay, steps for amino acid separation. So we have to saturate the chamber by solvent system. Solvent, you will take butanol is to acetic acid is to distilled water, 4 is to 1 is to 5. 4 is to 1 is to 5. Butanol, acetic acid, distilled water, BAD, bad 415. You can remember bad 415. Okay, then cut in size, wash with your cut the filter paper in size and wash it with your 0.1 normal HCl. Dry the workman's filter paper. Okay, then you mark a line from your 5 cm from the lower edge. Okay, mark a line with a pencil, not with a pen, because pen ink will flow. Okay, then apply the sample. Apply about 2.25 milliliter of sample here. 2.25 milliliter of sample here. Like you applied the sample. This red ones are the sample. Okay, this red one, look at these red ones, these are your sample. Dip the lower edge in the chamber and hang it to the chamber. Okay, like you have hanged it to the cover up a chamber. Allow run for 10 to 12 hours. Okay, this is left for 10 to 12 hours. Okay, then dry the paper in oven. Okay, then dry it in oven and spray with ninhydrin solution to visualize it. Ninhydrin solution, and by ninhydrin solution, the spots of Ruheman's purple color. You must have read in your biochemistry practicals that ninhydrin gives a Ruheman's purple. Okay, on protein exposure. So Ruheman's purple color spots can be visualized. Okay, we have a term called as retardation factor, or a ratio of fronts, or a partition coefficient. So retardation factor is equal to distance traveled by the solute by distance traveled by the solvent, solute by the solvent. Okay, this is specific for a particular solvent. So the protein can be recognized by the this retardation factor. Okay, by measuring all the distances you can get your retardation factor, and by this retardation factor you must have a list of proteins and the retardation factors, so you can identify which protein is this. Okay, so it is used in the identification of protein also. Uses, I said, separation of amino acids and reducing sugars, purification of different compounds, and detect inborn errors of metabolism. Then we have a high pressure liquid chromatography, HPLC. This is an improvised procedure. Okay, in which the solvent reservoir is this. A pump is there. A sample is put here. Okay, sample is put here. Then you have the HPLC column. Okay, HPLC column which will give you a reading. Okay, this sample is pumped by this. Okay, then HPLC column passes the HPLC column and it will be read by the chromatogram. Okay. Advantage is that it is very fast and it has better resolution, and its use in the measurement of all analytes. Okay, measurement of analytes like vitamins, drugs, hormones, enzymes, and amines. Then another technique is thin layer chromatography. Same procedure as the paper chromatography, but the chamber is like lined with thin layer adsorbent material. Glass chamber is used and lined with a thin layer adsorbent material, which will act as a catalyst. Okay, and it will fast the process and it will give better resolution also. Okay, so this is a thin layer paper chromatography. Okay, so this is all about your chromatography. Let's move on to the next chapter. So let's see, we have completed our carbohydrate metabolism as well as the carbohydrate chemistry. Okay, and also we discussed our chromatography technique. Okay, and we talked about we did not talk about ELISA, right? Okay, we haven't talked about ELISA yet. Okay, and then we have few chapters left here. Their metabolisms are also left. It has been about 5.5 hours. Okay, so yeah, we are going with good speed. Okay, and I hope you are bearing with me. Okay, I am just saying, like, you have to just listen to the facts carefully. Just practice your just pause the video and practice the pathways, okay, along with, so that you can have a good revision of your biochemistry. Okay, so let's now continue with our biochemistry revision. Okay, let's now discuss the another topic. Let's now go to another chapter. Okay, so those who are just watching the video, so I have a big salute to them, so they are bearing up to this. Okay, and let's move forward. We have a lot to cover today. Okay, so let's do the lot. Okay, let's move on to another chapter. Let's start with our heme metabolism. Okay, heme metabolism. So what is heme? Your porphyrins and iron combine to produce your heme, and heme, when degrades, produces your iron and bile pigments. Okay, so what are porphyrins? Porphyrins have the formula C20H14N4. C20H14N4, this is a formula porphyrin. These are cyclical compounds. Okay, cyclical compounds composed of 4 pyrrole rings. Okay, 4 pyrrole rings are present and HC breezes are present. Okay, HC breezes and 4 pyrrole rings are present in your porphyrin. Okay, so let's look at the structure of the porphyrin. This is the structure of your porphyrin. Okay, so you can see the 4 pyrrole rings here. Okay, 1, 2, 3 and 4. Okay, these are your pyrrole rings and these are connected by your HC breezes. Okay, these are your HC breezes which are connecting the 4 pyrrole rings. Okay, so heme is a tetrapyrrole. Okay, Heme has two types here, heme B and heme C. Okay, this heme B and heme C, there are of many types like A and E are there. Okay, A, B, C, D, E are there. But heme B and heme C, these are predominant. Okay, these are predominant. Okay, so this is a porphyrin ring, and you have 4 pyrrole rings present over here. You have 4 pyrrole rings present over here. Okay, this is your porphyrin. Okay, what are the importance of the important heme proteins? So what are important heme proteins? So hemoglobin, first one, is the oxygen transport in blood. Your myoglobin, which is the oxygen storage, which does the oxygen storage in the muscles. Okay, your cytochrome C, which is utilized in the ETS. Your cytochrome P450 in your hydroxylation of xenobiotics, you saw cytochrome P450 is the heme enzyme we did. Yes. Then your catalase, which causes your H2O2 degradation, and your tryptophan pyrrolase, okay, which causes your oxidation of your tryptophan. Okay, tryptophan pyrrolase causes oxidation of the tryptophan. Talking about the heme biosynthesis, it occurs in most of the mammalian cells except in mitochondria. Okay, most mammalian cells except in mitochondria. 85% occurs in the bone marrow, 15% in the hepatocytes. Okay, so these are the sites in which the heme biosynthesis is going on. Okay, and if we talk about the pathway of the heme biosynthesis, okay, this is the pathway of the heme biosynthesis. Okay, so first what happens is that the succinyl CoA combines with the glycine. Succinyl CoA combines with the glycine to in the presence of ALA synthase enzyme. Okay, ALA synthase enzyme, in which the CoASH and CO2 are removed. Okay, this is converted into gamma ALA. This is converted to gamma ALA. This is called the gamma aminolevulinic acid. Gamma aminolevulinic acid. This gamma aminolevulinic acid in the presence of ALA dehydratase is converted into your, what is removed, dehydratase is there, and it is converted to a porphobilinogen. This porphobilinogen, okay, this is acted upon by UPG1 synthase, UPG1 synthase, your uroporphyrinogen 1 synthase. Okay, and here 4NH3 is removed, and your hydroxymethylbilane is formed. UPG1 synthase, hydroxymethylbilane is formed. This hydroxymethylbilane is now acted upon by UPG3 co-synthase. Okay, and your uroporphyrinogen is formed. This uroporphyrinogen, then in the presence of UPG decarboxylase, okay, your coproporphyrinogen is formed. Okay, by the release of your CO2 molecules, and coproporphyrinogen in the presence of coproporphyrinogen oxidase. Okay, coproporphyrinogen oxidase. Okay, where CO2 is removed, your protoporphyrinogen is formed. Okay, protoporphyrinogen. This protoporphyrinogen in the presence of protoporphyrinogen oxidase. Okay, protoporphyrin is formed, and protoporphyrin pyrrolylase act. Okay, there is a chelation of your iron. Iron is incorporated, forming your heme. Okay, forming your heme. Understood? And this heme has a negative feedback. This heme has a negative feedback to a succinyl CoA and glycine. Okay, we'll read this in enzyme. So you can just remember the mnemonic. Some doctors palpate heart under cover. Okay, some doctors palpate heart under cover. Okay, and this produces pure heme. Okay, so initials S is the succinyl CoA, doctors D is your the delta aminolevulinic acid, or gamma D stands for delta, that's why it is given. Okay, P is a porphobilinogen, H stands for the hydroxymethylbilane. Okay, U stands for the uroporphyrinogen, C stands for your coproporphyrinogen. Okay, P stands again for your protoporphyrinogen, and again another P for protoporphyrin, and heme for heme. Okay, heme for heme. Okay, after this let's discuss a lot about your porphyria. Okay, about the porphyrias. Okay, so talking about your ALA synthase. Okay, if there is the deficiency of ALA synthase, okay, you will suffer from sideroblastic anemia. Okay, if you suffer from deficiency of ALA dehydratase, you will have plumboporphyria, also called as the DOS porphyria. Okay, and UPG1 synthase, you will suffer from acute intermittent porphyria, which is important, we will discuss. And UPG3 co-synthase, you will suffer from congenital erythropoietic porphyria, which is called as Gunther's disease. Okay, or Gunther's disease, congenital erythropoietic porphyria. And then UPG decarboxylase, okay, you suffer from porphyria cutanea tarda. Okay, and your CpG oxidase, you will suffer from hereditary coproporphyria. Then PPG oxidase, your variegate porphyria, and your phyllochelitase deficiency will lead to your protoporphyria. Okay, understood? So you can just remember the mnemonic: space problems have vitamin, please. Okay, just a nonsense statement, just remember. Okay, space problems. S for okay, you have SPACE now. You have SPACE. Okay, S for your sideroblastic anemia, P for your plumboporphyria, A for acute intermittent, C for your congenital erythropoietic. Okay, E is here absolute. Okay, this is E is it doesn't have any meaning here. Okay, space is there, and the problem. Okay, I talked about problem. Okay, problem stands for your porphyria cutanea tarda. Porphyria cutanea tarda. Okay, have vitamins please, have and vitamins and please, have vitamins please. So this stands for your porphyria cutanea tarda. Okay, then your hereditary coproporphyria, H for your hereditary coproporphyria, B for variegate porphyria, and P for your protoporphyria. Okay, so mnemonic is your space problems, have vitamin please. Okay, which is common in porphyria cutanea tarda. This is the most common type of porphyria. Okay, all porphyrias are X-linked. All porphyrias are autosomal dominant. All porphyrias are autosomal dominant. While two porphyrias are X-linked. Okay, which one? Your sideroblastic anemia, and this one. Okay, this one, and another porphyria is there which is not shown in this diagram. Okay, these are X-linked. Okay, you just remember your sideroblastic anemia. Okay, sideroblastic anemia. And Gunther's disease, this is autosomal recessive. Just remember, this is autosomal recessive. Okay, your Gunther's disease, which was congenital erythropoietic porphyria, is autosomal recessive. Okay, so porphyrias can be inherited or acquired porphyrias. Okay, inherited or acquired. Inherited may be hepatic or erythrocytic. Okay, hepatic, you can remember the mnemonic hepatic porphyrias. ACP sir, PCT देने high court गए. Okay, VIP ACP sir. Okay, VIP, you have the ACP in your CID. Okay, ACP sir. Okay, so he is now a medical student. Okay, giving a PCT exam or part completion test. Okay, but he is giving a part completion test in your high court. Okay, in your high court, not in your medical college. Okay, so just a nonsense statement made by me. Okay, so VIP means your variegate porphyria. Okay, and ACP sir. Okay, ACP, you can remember ACP for your acute intermittent porphyria. Okay, acute intermittent porphyria. And PCT is porphyria cutanea tarda. Porphyria cutanea tarda. Okay, and high court stands for your hereditary coproporphyria, your HC. Okay, hereditary coproporphyria. And we have erythrocytic ones like your congenital erythropoietic porphyria and protoporphyria. Okay, and acquired ones like your renal failure, liver disease, tumors, and toxic materials. This also cause your porphyrias. No special names for them. Plumboporphyria is due to PB poisoning. Some special facts I have written. Acute intermittent porphyria is due to UPG1 synthase defect. Okay, treatment is a hematin or heme. Okay, in this what will happen in acute intermittent porphyria, there will be neuropsychiatric manifestations like your increased serotonin and increased tryptophan. Okay, increased serotonin and increased tryptophan will occur. Okay, so due to acute intermittent porphyria, there will be neuropsychotic manifestations, your abdominal pain, and lab diagnosis will show increased porphobilinogen. Okay, and increased ALA in urine. Okay, and discussing about congenital erythropoietic porphyria, Gunther's disease. Okay, this is a uroporphyrinogen 3 co-synthase defect. Okay, due to which the porphyrin accumulates. This porphyrin accumulates in the presence of UV light. It forms reactive oxygen species. Okay, the porphyrin in the presence of UV light forms a reactive oxygen species, and this reactive oxygen species will cause a lysosomal damage and skin lesions. This ROS will cause a lysosomal damage as well as skin lesions. Okay, and this is causing a vampire or werewolf type of appearance, of the person. Okay, just look at this. This is a patient of congenital erythropoietic porphyria. You can treat it by hematin and antioxidant vitamins for reactive oxygen species neutralization. Okay, you can give vitamin A, C, and E. You can advise the patient to use sunscreens and cytochrome P450 inducer drugs. You can give cytochrome P450 inducer drugs. So congenital erythropoietic porphyria will cause a vampire or werewolf appearance. Okay. Another thing we have to talk, that is heme catabolism and bilirubin metabolism. Okay, site where it is occurring, macrophages of a reticular endothelial system like liver, spleen, bone marrow is occurring. Okay, in this what happens, the hemoglobin splits into heme and globin. This globin goes to your amino acid pool, but what happens to the heme? Heme is acted upon by heme oxygenase. Okay, oxygenase, the dioxygenase enzyme, where O2 is added, NADPH is added, your CO is released and your iron is released. Okay, so heme in presence of heme oxygenase converted into a biliverdin, green colored pigment biliverdin, and in presence of biliverdin reductase is converted to bilirubin, which is a yellow pigment. Okay, this is unconjugated. Okay, and now it is acted upon by albumin. Okay, which forms a albumin bilirubin complex. Okay, which goes to the liver. Okay, and how it is taken, it is uptake, it is first taken up by the liver. How the albumin bilirubin complex reaches the liver, okay, where the albumin and bilirubin separate. This bilirubin goes via facilitated diffusion, okay, into your enterocyte cells, okay, and this ligandin and protein Y keeps bilirubin in your solubilized state, okay. Ligandin and your protein Y keeps your bilirubin in your solubilized state, okay. Okay, so your glutathione S transferase, which is also called as a ligandin, and your protein Y, this keeps the bilirubin in a solubilized state. Then what is the conjugation procedure? So unconjugated bilirubin in the presence of UDP glucuronidase transferase. We know this from our discussion previously from the genobiotics. Okay, where the UDP glucuronidase is added, forming the bilirubin monoglucuronide. This bilirubin monoglucuronide with the presence of a UDP glucuronidase transferase again is forming the bilirubin diglucuronide, which is called as a conjugated bilirubin. Okay, and this conjugated bilirubin is going here. Okay, this UDP glucuronidase transferase, if there is deficiency, so we will see the formation of Crigler-Najjar syndrome. I have told in the genobiotics metabolism also. It is increase in unconjugated bilirubin. Type 1 will is a complete defect, while type 2 is the partial defect of UGT. Okay, Crigler-Najjar syndrome. Okay. What is the fate of a conjugated bilirubin? The conjugated bilirubin, some part of it will enter the bile canaliculi by MRP2 mediated active transport. Okay, there is a protein called as MRP2. Okay, this MRP2 will cause your active transport of your will help to enter the conjugated bilirubin, will help the conjugated bilirubin to enter the bile canaliculi. Okay, this MRP2 helps to enter the conjugated bilirubin into the bile canaliculi by active transport, from where it goes to ileum and colon, and in the presence of colonic bacteria, this conjugated bilirubin is changed into urobilinogen. Urobilinogen breaks down and changed into stercobilin, which is excreted in feces, which gives the color to the stool, and urobilin, which is excreted out in your urine. Okay, and mutation of this MRP2, this MRP2 that is mutated. If this MRP2 is mutated, you will see Dubin-Johnson syndrome. This is increase in the conjugated bilirubin. Okay, where there is black color of liver. This is due to the MRP2 mutation. And some of the conjugated bilirubin, they take part in portal circulation via MRP3. Okay, they take part in portal circulation via MRP3. So there is reuptake by hepatocytes then, okay, via the OAT. What is OAT? Your organic anion transporter protein. Okay, we have two types, like OAT1B1 and OAT1B3. These are the common types, okay, of OAT. OAT is your organic anion transporter protein. Okay, so you have OATP1B1 and OATP1B3. Okay, if there is the mutation of the OATP1B1 and OATP1B3, okay, you will see the syndrome which is called as a Rotor syndrome. Okay, if you want to know the full form MRP, it is your multi-drug resistance like protein. Okay, and this Rotor syndrome will show increase in conjugated bilirubin. Okay, if there is defective uptake of bilirubin in the liver, there is defect in this part, if there is defective uptake of the liver of bilirubin by the liver, okay, then we will suffer from a disease which is called as Gilbert's disease. Okay, this Gilbert's disease is due to defective uptake of a bilirubin by the liver. Okay, so it will increase your unconjugated bilirubin. Yes. Okay, these are your porphyrias. I have discussed all the porphyrias. This is summary of this. Okay, so after heme metabolism, let's move on to another part of the heme metabolism, that is your hemoglobins and the related disorders of the hemoglobin. Okay, so let's talk a bit about your hemoglobins. Hemoglobin is a tetrameric protein. Okay, adult hemoglobin HbA contains alpha to beta 2 in 90% of population, and HbA2 contains alpha 2 and delta 2 subunits. Okay, in 2.5% of population. The majority is alpha to beta 2. And fetal hemoglobin, this contains alpha to gamma 2. Okay, in less than 2% of population also, and in the fetus also, HbF, which has high affinity to oxygen dissociation curve, which will cause your left sip of your oxygen hemoglobin dissociation curve. This is your HbF. And glycated hemoglobin is called as HbA1c. Okay, this contains your alpha to beta 2 glucose. Okay, which increases in diabetes mellitus. Hemoglobin derivatives, like a normal hemoglobin derivatives and abnormal derivatives. Okay, so we have normal derivatives, we have oxyhemoglobin like Hb plus O2. Okay, we have your deoxyhemoglobin like your Hb and no O2 is there. Okay, we have your carbaminohemoglobin like that is your HbCO2. Okay, CO2 is non-covalently bound to your globin chain. Abnormally, we have your methemoglobin, that's your methemoglobin. where Fe3 plus is present in the place of your normal Fe2 plus. Okay, so in this hemoglobin, Fe3 plus is present in place of your normal Fe2 plus, which will lead to your cyanosis and all other symptoms. Your your carbonyl hemoglobin HbCO or HbCO. This CO bounds to your Fe. Okay, in CO poisoning or your smoking. Okay, so this is your carbonyl hemoglobin. In your sulfomoglobin, okay, sulfomoglobin in your H2S poisoning, and your cyanoglobin in your cyanide poisoning. Okay. Talking about the disorders, we have the sickle cell anemia, which is an important case-based question also. Okay, and it is important as short note question also. It can come as a long question also. Okay, the sickle cell anemia and hemoglobin can come combinedly as a long question. Okay, the sickle cell anemia is an autosomal recessive condition. This is an autosomal recessive condition. Okay, mutation type is your missense point mutation. Important, missense point mutation. Okay, and glutamate is replaced by your valine. At which point? At the 6th position of your beta globin chain. It replaces by valine at the 6th position of your beta globin chain. Okay, and this causes localized stickiness on the surface of RBC. Okay, so RBC will polymerize and the shape will be like sickle shape. Okay, sickle shape. Okay, which will occlude the microvasculature and cause intense pain. Okay, intense pain will be there. The molecular basis of SCD is that in CTC the point mutation will occur, which will convert it into CSC. Okay, if CTC is there, so GAG. Okay, the mRNA will contain your GAG. Okay, and glutamate will be the protein, and the glutamate is normally present in HbA. Okay, and here there is the mutation from T to A mutation, by which the GUG will be formed. GUG code for your valine. Okay, GUG codes for your valine, and this valine is abnormal. Okay, valine is non-polar non-polar amino acid. Okay, glutamate is a acidic amino acid, so it is polar and negatively charged. Okay, so HbA is polar, negatively charged and soluble, which is normal. Okay, but your HbS is non-polar, less soluble and more precipitable. Okay, so precipitation of hemoglobin occurs. This is the oxyhemoglobin, this is a deoxyhemoglobin, and if sticky patches are there like this, so it is a oxyhemoglobin S. The sticky patches with the deoxyhemoglobin is called deoxyhemoglobin S. Okay, this is the structure of deoxyhemoglobin S. If there is decreased supply of O2, that is increased deoxyhemoglobin S, so polymerization of deoxyhemoglobin S will occur. Okay, like this they will polymerate, and this RBC will be sickle shaped, and there will be occlusion of blood vessels. Okay, clinical symptoms. What are the clinical symptoms? There will be anemia, pain, fatigue, jaundice, hands and feet swell up, and infections. The complications: your acute chest syndrome will be there due to the vessels blockade. Your acute chest syndrome will be there. The splenomegaly will be there. The spleen has to break down the blood vessels. Okay, the splenomegaly will be there, break down the RBCs, but that as due to the sickling. Okay, the splenic capillaries, which have smaller diameter. Okay, they are blocked, causing splenomegaly. Pain crisis. Okay, acute pain crisis due to blockade of your microvasculature, and strokes and priapism. Priapism is a painful erections. Okay, then lab diagnosis, we have the sickling test. Okay, in the peripheral blood smear by adding sodium metabisulfite or sodium diothionate, and this is added, sodium metabisulfite and sodium diothionate. Okay, sodium diothionate. Okay, and observed under the microscope. Sickling test. Then your hemoglobin electrophoresis test, HbS, which moves slowly in electrophoresis, but HbA, this moves very fast in your electrophoresis. Okay, and HPLC is high pressure liquid chromatography, and a capillary electrophoresis test. Okay, these are 4 tests: sickling test, HB electrophoresis, HPLC, and your capillary electrophoresis. These are for a lab diagnosis of your sickle cell anemia. Treatment: hydroxyurea, blood transfusion, stem cell therapy, and gene therapy. Okay, hydroxyurea, blood transfusion, stem cell therapy, and gene therapy. These are your treatments for your sickle cell disease. Then you have thalassemia. We can have alpha globin chain deficiency, which is alpha thalassemia, or beta globin chain deficiency, which will cause beta thalassemia. Okay, alpha thalassemia has 2 genes on chromosome number 16 coding for alpha globin. Okay, there is the mutation in the 2 genes. Okay, so there is decreased quantity of your alpha globin production. Okay, there are 4 types of carriers in your alpha thalassemia. 4 types of carriers. One is a silent carrier. Okay, you have 4 alpha genes. Okay, you have like 2 genes on chromosome number 16 which are coding for alpha globin. Okay, so these are the chains of a hemoglobin. So if there is only 1 chain mutation, silent carrier, okay, there are no symptoms. If there is 2 chain mutation, alpha 1, alpha 2, or alpha 1, alpha 3, okay, you can have mild anemia. Okay, if there is hemoglobin H disease, okay, like your 3 chains are mutated. Okay, so there is microcystic hypochromic anemia, microcytic hypochromic anemia, smaller RBCs, and your iron is less. Okay, and hydrops fetalis is a fetal death. This is fetal death due to the accumulation of your blood, your water or your fluid in your brain, accumulation of fluid in your brain. So hydrops fetalis. Okay, next hemoglobin bars. Hemoglobin bars are formed. Okay, there's mutation in all the alpha chains. Okay, so in the chains for alpha globin, these are not coded, so there is decreased quantity of your alpha globin production. Okay. Then beta thalassemia, chromosome number 11 gene mutation. The clinical features may be anemia, jaundice, hepatomegaly, splenomegaly, frontal bossing, maxillary overgrowth, and hair on end appearance on X-ray. Okay, skull X-ray has an hair on end appearance. Okay, why this skeletal symptoms are that? Due to decreased hematopoiesis in the bones. Okay, decreased hematopoiesis will lead to this frontal bossing, maxillary overgrowth, and hair on end appearance. Okay. Thalassemia can be two types. A beta thalassemia major due to defect in both genes, the homozygous, okay, and a beta thalassemia minor, defect in a single gene, this is heterozygous. Okay, so in beta thalassemia major you will see there is in both thalassemia, in all thalassemias in fact, okay, you will see there is increased HbF levels and increased HbA2 levels, okay, understood. Other treatment, supportive you have your supportive case, like your supportive therapy, symptomatic treatment and all. Blood transfusion can be done, iron chelating agents can be given, and stem cell transplant is the way. Okay, lab diagnosis, how will diagnose in the lab? By hemoglobin electrophoresis, your high pressure liquid chromatography, your DNA analysis for mutations, and increased HbF and increased HbA2. These are specific for your beta thalassemia. Okay, so here we completed our hemoglobin, and your finally your heme metabolism is now complete. Okay, let's move on to the next chapter. Okay, so we are talking about an important chapter in your biochemistry, that is your enzymology. Okay, enzymology and your clinical enzymology. So as it is an important chapter, so let me teach you by writing. Okay, so enzymes are your biocatalysts. Okay, these are your biocatalysts. These are colloidal in nature. And these are thermolabile. Okay, and these are proteinaceous in nature, proteins, except your ribosome, and this ribosome, okay, this is called as your peptidyl transferase. Okay, this is your nucleic acid in nature. Okay, and this is specific in action. The enzymes are your specific in action. So we have the classification of enzymes. We have the enzyme commission numbers. Though it is a 4-digit number. The enzyme commission number is a 4-digit number. Okay, given by your IUBMB. IUBMB is the International Union of Biochemistry and Molecular Biology. Okay, so for example, enzyme commission number, for example, 2611. First of all, 2611, this is your SGOT. 2612, okay, this is your SGPT. Okay, this is your AST, and this is your ALP. Okay, then again 3131, this is your ALP. 3131 is your ALP. Okay, and your 127, this is your lactate dehydrogenase, your LDH. Okay, LDH. And your 3131 is alkaline phosphatase. Okay, I've written. Okay, then your 41213. Okay, this is your aldolase. Okay, and your 6511, this is your DNA ligase. These are some of the enzyme commission numbers, okay, which are given by the IUBMB, okay, enzyme commission numbers. And talking about your what is this numbers? What are these numbers? Okay, this 2611. Okay, two represent your class of enzyme. Okay, so SGOT it is belonging to your enzyme class 2. This is your transferases. Okay, then 6 is a subclass, 1 is a subclass, and this one again is your individual enzyme number. Okay, individual enzyme number. Okay, so if you have classification, so we have enzyme you have EC enzyme number. Okay, enzyme commission number 1, 2, 3, 4, 5, 6, and 7. 7 is recently added, okay, in 2018. Okay, so you can remember the mnemonic OTHLIL, OTHLIL. Okay, OTHLIL, OTHLIL, and 7 you can remember, this is your translocase enzymes. Okay, this is newly added. Okay, and what is this OTHLIL? This O is the oxidoreductase enzymes, T is your transferase enzyme, H is the hydrolase enzyme. Okay, this is the hydrolases enzyme. Okay, L is the lyases enzyme, I is the isomerases, L is the ligases, and 7 is the translocases. Okay, so oxidoreductases, this catalyze oxidation reduction reaction. Okay, transfer of H plus and electron transfer. Okay, this also includes classes like your dehydrogenases. dehydrogenases, and your oxidases, your peroxidases like this, okay. Another enzyme, for example, you can remember oxidoreductase, that is your alcohol dehydrogenase. Enzyme commission number is 1111. Okay, just remember 1111 is your alcohol dehydrogenase. Okay, understand? Okay, so it belongs to one class, the oxidoreductase, subclass acting on alcohol. Subclass is your acceptor is NAD plus or NADP plus, and one is the it is an individual enzyme name. Okay, then you can also remember a lactate dehydrogenase. It is also oxidoreductase. Okay, this has your commission number 11127. Okay, this is lactate dehydrogenase. Then about class 2, the transferases. This transfer the group other than H plus, because H plus transfer is done by oxidoreductases. So transferases transfer group other than oxidoreductases. For example, your enzymes for your transamination reaction like SGOT and SGPT, like aspartate is there. Aspartate combines with alpha ketoglutarate in the presence of SGOT. Okay, this is converted into your glutamate. This is converted to glutamate plus oxaloacetate. Okay, oxaloacetate and glutamate. And for example, the alanine in the combines with alpha ketoglutarate in the presence of SGPT. This is converted into your pyruvate. The glutamate is fixed glutamate and a pyruvate. Okay, so we told alanine and alanine can be converted, enter the cycle by converting it itself into pyruvate. Okay, just remember the glycogenolysis. Okay, that not the glycogenolysis, your glyconeogenesis. Just remember the pathway, that is your glyconeogenesis. Okay, you will get to know. Then another transferase is your this is 1, this is 2. Another transferase is your hexokinase. Which group it will transfer? It will transfer phosphate group. Okay, so glucose in the presence of your hexokinase, it is converted to glucose 6 phosphate. Okay, glucose 6 phosphate, and this requires your ATP. Okay, and then about talking about your transferases is done. Then talking about your hydrolases. Hydrolases, this catalyze the hydrolysis of bonds. Okay, this can break the bonds by using your water. This water will help to break the bonds. Okay, like the digestive enzymes. Okay, digestive enzymes. For example, you can remember not a digestive enzyme, but alkaline phosphatase, ALP. ALP is an example of your hydrolase class, ALP. Okay, ALP is enzyme commission number 3131. Then liases. What are liases? This can also break the bonds. This can also, which bond? They can break CC bond, CO bond, or CN bonds. This can break these bonds. Okay, this can break these bonds. These are called as a liases. For example, aldolase. Aldolase is a liases. Okay, aldolase is a liase. Enzyme commission number is 41213. No need to remember much. I have written it also either. 41213. Okay, this is also a liase. And isomerase, isomerase, your phosphotriose isomerase, which is converting on DHAP and PGAL. Okay, we saw the reaction before. And okay, isomerases, so these are responsible for transfer of your intramolecular atoms. Okay, for interconvergence of your isomerism. These are helpful in isomerase. Okay, this includes your racemases. Okay, this has many classes like your racemases. Okay, epimerases, then cis-trans isomerases, and mutases. Okay, mutases also. Okay, for example, you can write in your isomerase, you can give the example of your triose phosphate isomerase, that is converting your DHAP, then DHAP into your PGAL. Okay, this is your triose phosphate isomerase, TPI. Okay, and you can give the example of your glucose 6 phosphate converting into your fructose 6 phosphate by your phosphohexose isomerase. Okay, and then you have ligases. Catalyze the joining of two molecules by utilization of ATP. Okay, like the DNA joining enzyme, that it helps to join two nucleotides. Okay, it helps to join the two nucleotides using a 3-5' phosphodiester linkage we saw from the molecular biology. Okay, and the example of your ligases is your DNA ligase. Okay, then you have translocases. Translocases is catalyzing the translocation of ions across the membrane. The translocation of ions. For example, your ATPase. Okay, sodium potassium ATPase. So sodium potassium ATPase, that has the enzyme commission number 72213. Okay, so you can just add in this list. Okay, sodium potassium ATPase, that have enzyme commission number 72213. This is your sodium potassium ATPase. Okay, just let me revise you a bit. Okay, this table. Okay, 2611 is SGOT, 2612 SGPT, 3131 ALP, 11127 is LDH, 41213 is aldolase, 6511 DNA ligase, 1111 alcohol dehydrogenase, 721213 is sodium potassium ATP. So this table you can remember for Viva questions, for targeting or Viva questions. Okay, so then talking about a mechanism of enzyme action, what is the mechanism by which the enzymes have their action? Okay, by the enzymes, what is the mechanism by which the enzyme are acting? Okay, what is the mechanism? Okay, talking about the mechanism of enzyme action. So enzyme action, they enzymes, what they do, they lowers the activation energy, they lower the activation energy required for the reaction. Okay, activation energy for the reaction, like this is a substrate. Okay, and this is a product, this is a formation of your product. Okay, the product is always less energy than your substrate. Okay, this is your product. Okay, and this state, this height is your energy required for the conversion, and this state is called as your transition state. Okay, so what enzymes do, this is the reaction without enzymes. Okay, and with enzymes what happens, this will convert like this. Okay, with enzymes, like you can do like this. Okay, the same product will be found, but less energy will be taken. The less energy will be taken in the presence of, if enzymes are present, if enzymes is present. Okay, if enzymes is present, the activation state is lowered. Okay, so enzymes will decrease the energy barrier in the reaction and lower the activation energy. They will lower the activation energy, decrease the activation energy. Okay, and transition state, what is transition state? It is an intermediate state and high energy state. This is a state of high energy and less stability, because if energy is high, then stability is low between the substrate and the product. Okay, so enzyme will favor the formation of your transition state. Okay, then talking about enzyme substrate complex formation, here what happens is that the enzyme and the substrate, they form a complex, when the enzyme is going to act. So for explaining the enzyme substrate complex formation, okay, we have theories. Okay, we have theories like a lock and key model. First is the lock and key model, or your Fisher template theory. So in this theory, what it is told that the active site of an enzyme, it is rigid and pre-shaped. Okay, it is shaped like that. Okay, and only specific substrate can bind to the active site of the enzyme. Okay, so but it failed to explain your enzyme kinetics. Okay, it could not explain the enzyme kinetics. Then we have the induced fit model. Okay, which is called as the Koshland's theory. So Koshland's theory had the notion that active site of an enzyme is not rigid. Okay, it is flexible. Okay, so the substrate will induce a conformational change in the active site, and this conformational change will favor the formation of your enzyme substrate complex. Okay, this is called as the induced fit model. Then we have another theory which is called as a substrate strain theory. Third is a substrate strain theory. So according to the substrate strain theory, again the active site is flexible. Okay, and the substrate is bringing out the conformational change in the active site, and in the course of this, the substrate is also strained. Okay, Okay, there is the strain within the substrate also. Okay, and this strain will cause the cleavage of bonds, and the bonds will get attached to the, due to the cleavage of bonds, there will be free bonds, and these free bonds will favor the attachment to the enzyme. Okay, understood? This is there. Afterwards, there is a mechanism of catalysis. Okay, what is catalysis? Catalysis, you know. Okay, so what are the theory of the mechanisms of catalysis as thought? Okay, one is called as acid-base catalysis. So there in the active site, okay, you have acidic and basic groups. Okay, and these acidic and basic groups are basically attacking the substrate. Okay, if here is the enzyme, okay, then acidic and basic groups are present, the acidic group, basic group, like this. Okay, and these are causing the substrate to attach here and form your enzyme substrate complex. Okay, and another theory is that after acid base catalysis, we have another catalysis that is called as your covalent catalysis. So covalent catalysis theory, it states that there are positive and negatively charged groups in your active site. Okay, so if here is the enzyme, okay, so you have the positive and negatively charged groups, okay, in the present in the enzyme. Okay, and if there is a substrate present, it will react. Okay, this will react and favor your formation of enzyme substrate complex. Okay, then you have an effect called as a proximity effect. Okay, proximity effect. Proximity effect is related with the entropy. Okay, it is related with the entropy. Okay, so what happens if the enzyme and the substrate, they come at a bond forming distance. Okay, if they come very near, then there won't be formation. Okay, if they're in a sufficient bond forming distance, you must have read this bond forming distance about this in inorganic chemistry. Okay, and this will result in the formation of your enzyme substrate complex, and this catalysis will occur. Okay, and again there is a theory which is called as the substrate strain catalysis. The substrate strain catalysis, and this theory is again similar to a substrate strain theory, the strain development in the substrate. Okay, related with this. Okay, understood. Next we'll talk about some cofactors. Okay, we'll talk about some cofactors. Okay, what are cofactors? And not cofactors specifically, we are talking about the coenzymes. Okay, to be specific, we're talking about the coenzymes. Okay, so the coenzymes, these are non-protein enzymes. Okay, small molecules or metal ions. Okay, that participate directly in substrate binding and catalysis. Okay, substrate binding and catalysis. These coenzymes are also called as co-substrates or a secondary substrate. Okay, like your lactate to pyruvate, some co-substrate is acting. Okay, your NAD plus. Okay, like that. I will give you examples later on. Okay, so for an enzyme, this is a hollow enzyme. Means the whole enzyme. Okay, whole enzyme is made up of two parts. One is the apoprotein part. Not the apoprotein. The apoenzyme part, which is the proteinaceous part. Okay, and another part is a coenzyme part. Or a cofactor part. Okay, or a prosthetic group is there. Okay, this is a non-protein part. So every enzyme has a protein part as well as a non-protein part. Okay, talking about coenzymes. Okay, these are non-protein. These are organic. Okay, they have low molecular weight, and they are dialysable. They are easily dissociable. Okay, talking about cofactors. What is cofactors? So cofactors have weak association with your apoenzyme. Weak association also. These cofactors are also called as a metal activated enzymes. Okay. Example is your ATPase. ATPase, which requires Mg2 plus and Ca2 plus. So is Mg2 plus and Ca2 plus. This act as cofactors. Talking about what are prosthetic groups. Prosthetic groups are usually metal ions. Okay, these are usually metal ions. Okay, so usually these are metal ions. Okay, and these have tight association. This is important, tight association to apoenzyme. Okay, and the enzymes to which these are attached are called as the metalloenzymes. Okay, example of prosthetic groups are like zinc. Zinc is the prosthetic group of superoxide dismutase we saw in your xenobiotics, in your antioxidants, and your carbonic anhydrase also. Okay, then molybdenum is of xanthine oxidase. Okay, and selenium of your glutathione peroxidase. Okay, so these three are your prosthetic groups. Okay, so if we just classify the coenzymes on the basis of precursor molecules, we have B and B complex and non-B complex. Okay, and on the basis of required metal ions and basis of group transfer, we can classify the coenzymes. Okay, let's discuss some B complex vitamins. Okay, let's discuss some B complex vitamins and their coenzymes. Okay, vitamins and their coenzymes. These are important. Okay, and the enzymes too, of which they are coenzymes. Okay, of the enzyme too. So vitamin B1, starting from vitamin B1 is a coenzyme TPP. Okay, and the enzyme is your transketolase. Okay, then B2 we have the coenzyme FMN and FAD. These are of your amino acid oxidase, the succinate dehydrogenase. Okay, then a B3 we have NAD plus and NADP plus. Okay, these are of your LDH, your PDH, your alcohol dehydrogenase, etc. Okay, then of B5 we have CoA, coenzyme A, CoA. This is of your PDH complex. Okay, PDH. And we have of B6 we have PLP. B6 we have PLP. Okay, like SGOT, SGPT of this. Okay, then B7 we have biocytin. Okay, biocytin is the coenzyme of the enzyme acetyl CoA. this is not the biocytin, this is also biotin. You can write this biotin. Okay, so in some books it is written biocytin, so I have written like this. Okay, acetyl CoA carboxylase. Then of B9, THF is the coenzyme. THF is the coenzyme, and the function of THF is to transfer one carbon metabolism. Okay, on one carbon metabolism. Okay, it doesn't have any enzyme such like that. Okay, B12 has no, like, it has adenosyl B12 and methyl B12. You can write this. Okay, but not specifically coenzymes are there. Okay, just remember this enzymes and the coenzymes. Okay, and we have some these are all your B complex coenzymes. Okay, then we'll discuss about some non-B complex coenzymes. Non-B complex coenzymes. For example, your SAM, your UDP, your PAPS, your ADP. Okay, ADP, UDP, PAM, your SAM. What is SAM? Your S-adenosyl methionine, which will help in a transmethylation reaction. Okay, transmethylation reaction. Your UDP. Okay, this is uracil uridine diphosphate. Okay, this will transfer of glucose and galactose is done by them. Glucose and galactose. Okay, in the glycogen synthesis. Okay, we have talked about UDP earlier. PAPS is the active sulfur donor, as I have told in your sulfurylation reaction. Okay, active sulfur donor, like in your GAG synthesis, GAG synthesis. Okay, your synthesis of your mucopolysaccharides. Okay, understood. And your ADP is transferring the phosphate group. Okay, this is transferring the phosphate group. Okay, so SAM, UDP, PAPS and ADP, these are non-B complex coenzymes. Okay, there are some metal ions we have talked about, like zinc is the metal ion of the enzymes. Which enzymes zinc is there? The carbonic anhydrase, your superoxide dismutase, is lactate dehydrogenase also. Okay, your alcohol dehydrogenase also, your ALP. Okay, and your RNA polymerase also. Okay, so just remember which zinc is acting as a cofactor. Okay, of which enzymes. Okay, then we have the copper. It is of your cytochrome oxidase. Okay, of your tyrosinase, your superoxide dismutase, your lysyl oxidase. We talked about lysyl oxidase sometimes later. You remember, collagen synthesis and all. Lysyl oxidase. Okay, selenium, it is of your glutathione peroxidase. MO of xanthine oxidase. MG of your kinase, enolase. Okay, and your glucose 6 phosphatase. And MN it is of arginase. Just remember arginase. Okay, talking about active site. What is the active site? Active site is the region of an enzyme as the substrate binds and undergoes a chemical reaction. Okay, so active site, most common active site, most common amino acid. Okay, there is a question sometimes asked, the most common amino acid at your active site. Okay, it is your serine. It can be asked in the viva. Okay, and generally this active site has a 3D cleft or crevice. Okay, and a substrate binding site and a catalytic site. Okay, substrate binding site and a catalytic site. This substrate binds to the active site by weak non-covalent bonds. Okay, talking about the factors affecting enzyme activity. So what factors are affecting enzyme activity? Factors affecting your enzyme activity. So first of all, we have the enzyme concentration, which will directly affect your enzyme activity if substrate is available. Okay, so rate of reaction is directly enzyme concentration. So rate of reaction is directly proportional to the enzyme. I am writing ROR. Okay, so if you tell the velocity of the reaction and this is enzyme concentration. Okay, so you will have such type of this graph. Okay, second factor is the substrate concentration. Okay, if you go on increasing your substrate concentration, initially you will get velocity is directly proportional to substrate concentration, but afterwards your enzyme is exhausted. Your enzymes are exhausted and the graph comes to a plateau phase. Okay, so if you take the velocity and the substrate concentration, this is an important graph. Okay, initially this happens and then you get the plateau phase. Okay, the velocity at which so this is the maximum velocity, this is the Vmax and this is the Vmax. Okay, the maximum velocity of the reaction. Okay, and where the point at which where there is half Vmax, and you extrapolate the graph to the x-axis. Okay, and so the velocity of the reaction, okay, the substrate concentration at half Bmax, substrate concentration at half Bmax is called as your Km. Okay, Km is your Michaelis-Menten constant. Okay, this is your Michaelis-Menten constant. Okay, also called as, this is important, also called as your Briggs-Haldane constant. Okay, this other name is important, sometimes examiner may confuse you. Okay, so it is the substrate concentration to produce the half of maximum velocity. Okay, and the Km is the indicator of the affinity. The Km is the indicator of the affinity of the enzyme. Like Km is indirectly proportional to your affinity. More the Km, less the affinity of the enzyme towards the substrate. Okay, and Km value is constant. It is constant for a particular enzyme. Constant for particular enzyme. Okay, understood. So we have an equation which is called as a Michaelis-Menten equation. This is called as a Michaelis-Menten equation. Michaelis-Menten equation, that is v naught. Okay, initial velocity. This is the initial velocity. It is equal to your Vmax into S by Km plus S. Okay, this is the Michaelis-Menten equation. Okay, another thing I want to tell you that if you just reverse the Michaelis-Menten equation, like you take 1 by V naught. Okay, you take 1 by V naught. So if you take 1 by V, okay, so what will happen? Km plus S by your Vmax into S, okay. So you can separate this term Km by Vmax into 1 by S plus 1 by Vmax. You can do this, yes. So this comes in the format of this, let me mark this as y equal to mx. So this is m, your Km by Vmax, this is m, your slope, okay, into x is your 1 by S, x-axis, okay, y equal to mx plus c. So curve, what is the curve of y equal to mx plus c? Okay, y equal to mx plus c comes like this, okay. So here we take 1 by V, here we take 1 by S, okay, the reverse ones, okay. And if you extrapolate the graph, you get minus 1 by Km in this axis, okay. So you can get the Km from here, okay, and this point represents 1 by Vmax, okay. So this plot, this inverse plot, okay, double reciprocal plot, this is called as a double reciprocal plot, or your Lineweaver's-Burke plot, or your Lineweaver-Burke plot, okay, or a double reciprocal curve, okay. And the significance is that you can calculate the Km and Vmax at the initial velocity, okay. I can calculate the Vm and Kmax at the initial velocity by using a double reciprocal plot, okay, understood. This is a Lineweaver-Burke plot. Okay, this we done. Substrate concentration is done. Okay, third property which will affect it is the product concentration. If the product is increased, it will decrease your enzyme activity. Okay, the product will inhibit, okay, by your feedback inhibition. Okay, so product will like inversely affect your enzyme activity. Then fourth is the temperature. So there is an optimum temperature at which there is a maximum enzyme activity. So if you take velocity and if you take temperature, okay, the curve will be bell-shaped curve. This is a bell-shaped curve, okay. And this temperature at which there is maximum enzyme activity, temperature at its maximum velocity of reaction, this is called as the optimum temperature, okay. For most of the enzymes of the body, we have optimum temperature about 37 degrees Celsius. At about 50 degrees Celsius, there is denaturation of the enzymes occur, okay. At 70 degrees Celsius, there is inactivation of enzyme. At 70 degrees Celsius, there is inactivation of enzymes, okay. So bell-shaped curve is there, and the theory for this temperature relation is the collision theory. So there is the collision theory. If there is increased temperature, there are increased collisions between enzyme and your substrate, okay. And this increased collision will lead to the formation of your enzyme substrate complex, understood. And okay, and Q10 value, what is Q10 value? This is the increase in enzyme activity, by increasing temperature by 10 degrees Celsius, okay. So enzyme activity is nearly found to be double, okay, in your physiological cases, okay. So this is your Q10 value, okay. Then talking about another factor after temperature is your pH, okay. pH is also like your temperature thing, okay. And there is an optimum pH at which there is maximum enzyme activity, okay. If you take your velocity and pH, okay, there's an optimum pH about 6.8 is the optimum pH, okay, for your enzymes, okay. But for exceptions are always there. Exceptions, what are the exceptions? Your pepsin is optimum pH for pepsin is 1 to 2. Okay, for pH pepsin is 1 to 2. Your acid phosphatase, this is acidic enzymes act in the acidic environment. Phosphatase is your 3 to 5, 3 to 5, and your alkaline phosphatase, it is about 9 to 10. It is about 9 to 10. Okay. Then we have after pH, we have another factors. We have after pH another factors. 6. Okay, metal ions. Okay, what are the effect of the metal ions? So metal ions increase the reaction velocity, like chlorine increases the reaction velocity of your amylase. Okay, your magnesium and calcium of your ATPase, your zinc of your SOD. Okay, carbonic anhydrase and all. Okay, then we have effect of inhibitors, and inhibitors decrease the velocity of the reaction. Okay, inhibitors decrease the velocity of the reaction. So what are the factors that are affecting your enzyme activity? Factors affecting enzyme activity, we have enzyme concentration, directly proportional to the rate of reaction, substrate concentration in which you obtain this type of curve. Okay, Michaelis-Menten equation, Lineweaver-Burke plot is discussed. The product concentration, which will inversely affect in enzyme activity. Temperature, there is an optimum temperature present. Okay, and the pH, then optimum pH present. Metal ions, which will like increase the enzyme activity, and inhibitors, and we have some inhibitors present. Okay, so these are the factors affecting enzyme activity. Okay, and now let's discuss another topic, which is enzyme inhibition. This is enzyme inhibition. Enzyme inhibition, we have a reversible type of inhibition, reversible, or we have irreversible inhibition. Okay, reversible can be your competitive inhibition, where the enzyme is a substrate analog of the product, or it can be non-competitive. Okay, or it can be uncompetitive. And this irreversible in type of inhibition, okay, this can be a suicide inhibition. Okay, or it can be a feedback inhibition. Okay, so talking about your enzyme inhibition, so inhibitor is substance which can decrease your enzyme velocity. Okay, in this reversible inhibition, the inhibitor bind to the enzyme by your non-covalent forces, but in irreversible, the inhibitor binds by covalent interactions. So it can be reversed, it cannot be reversed. Okay, talking about a competitive inhibition, inhibitor is a structural analog of the product. Okay, your substrate, if the substrate looks like this, your inhibitor will also look like this. Okay, these are structural analogs. Okay, these are the structural, and this is the inhibitor. Okay, structural analogs. Okay, and the inhibitor competes with the substrate to bind with the active site. Okay, there is competition to bind with the active site, and if the inhibitor binds, then there will be enzyme inhibition. Okay, in this, what will happen to the curves? Okay, if I talk about your competitive inhibition, okay, so if you see the V and S curve, the V velocity and substrate curve, okay, if there is no inhibitor is like this. Okay, and if the inhibitor is present, okay, the curve like becomes like this. Okay, here what happens, here the Km value will increase, okay, and the half Vmax remains constant. Okay, Km will increase and your Vmax will remain constant. In your competitive inhibition. And if you want to see the Lineweaver-Burke plot as well, okay, so you can see like this. 1 by V is there, 1 by S is there. You can get this in spotting. Okay, so if inhibitor is present, if there is no inhibitor, let's first do. Okay, this is minus 1 by Km. Okay, if there is no inhibitor. Okay, but if inhibitor is present, okay, this Km increases. Okay, so this is minus 1 by Km dash. So which one is bigger? In negative side, your minus 1 by Km dash must be bigger. Okay, so if there is inhibitor present. Okay, so this is the plot for Lineweaver-Burke plot. Okay, what are the examples of a competitive inhibition? So examples you can write your malonate, which inhibits your succinate dehydrogenase. You know this. Okay, then your allopurinol. This inhibits your xanthine oxidase in case of gout. Okay, in treatment of gout, allopurinol is used. Okay, then ethanol. This is inhibiting an alcohol dehydrogenase. Okay, in your methanol poisoning it is used. Okay, then your methotrexate drug is there, Okay, this is inhibiting a dihydrofolate reductase, DHFR enzyme. Okay, and your statins are there, which reduce cholesterol by inhibiting your HMG-CoA reductase enzyme. Okay, and your dicumarol is an oral anticoagulant which will inhibit your vitamin K epoxide reductase. Okay, these are your examples. Talking about a non-competitive inhibition. Okay, so enzyme has two sites, your allosteric site and your active site here. Okay, see, this is the active site, is the active site, this is the allosteric site. So the inhibitor binds to the allosteric site. So the inhibitor will bind to the allosteric site, and by binding to the allosteric site, it will modify your active site, and this active site will be like of other conformation. There will be conformational changes. Okay, so when the substrate comes to bind, it cannot bind the active site. Okay, so there is no competition. There the inhibitor will do its job, okay, independently. Okay. So there is no structural similarity between substrate and inhibitor. Okay, no competition is there, and the inhibitor, it binds to the allosteric site and induces conformational modifications to prevent the substrate from binding to the active site. Okay, understood. So in this process, if you just talk about the graphs, V and S graph, okay, and another graph we will see your 1 by V and 1 by S graph. Okay, in V and S graph, what happens? This is without inhibitor, okay, but this is when inhibitor is present. Okay, so Vmax is your decreased, your Km value is still constant, your Km is same, so here in your non-competitive, your Km is same and your Vmax is decreased. Okay, this is important. Okay, and in a Lineweaver-Burke plot, what happens is that this Km is constant, okay, and with an inhibitor, the slope will increase. Okay, slope represents your 1 by Vmax. So 1 by Vmax is increasing, Vmax is decreasing, so 1 by Vmax must increase. So there is increase in slope. Okay, this is with inhibitor present. Okay, and this is without inhibitor, this is inhibitor absent. Understood? So this is the Lineweaver-Burke plot in this case. Okay, and what are the examples? Like a fluoride, this inhibits your enolase, enzyme. Okay, so fluoride bulb is read in glycolysis, that it is used in your collection of your blood sample in your blood glucose estimation. Okay, because you have sodium fluoride and your potassium oxalate in 3:1 ratio and all. Okay, we read about that. Okay, then your complex 4 is inhibited by the cyanide. Okay, the cyanide, which will inhibit your complex 4 of ETC, the inhibitor complex 4 of ETC. Okay, and then arsenite was inhibiting which one? Your alpha ketoglutarate dehydrogenase. Okay, we saw it in the Krebs cycle. Okay, and the PBAGHG, the heavy metals, okay, these are inhibiting the serine proteases. This is also an example, serine proteases. Okay, then iodoacetate is inhibiting the glyceraldehyde 3-phosphate dehydrogenase, D3PDH. Okay, these all examples are of your non-competitive inhibition. Okay, then we have uncompetitive inhibition. In this case, your Km also decrease, your Vmax also decrease. Okay, your inhibitor binds with the enzyme substrate complex to form ESI complex, enzyme substrate and inhibitor complex. Okay, there is no competition at all. Okay, there is no competition at all. Example is there is only one example in this case. Okay, the one example is your placental ALP. This is inhibited by phenylalanine inhibited by placental ALP. Okay, understood. After this, let's talk about irreversible inhibition. Okay, covalent binding is there. Okay, covalent binding is present here in the irreversible inhibition. Okay, so we can have a suicide inhibition. Okay, suicide inhibition, the inhibitor itself is converted to a more potent form by the enzyme it is inhibited. Okay, like your allopurinol is converted by xanthine oxidase by allozanthine, and this allozanthine now inhibits your xanthine oxidase. Due to which this allopurinol, okay, decreases your uric acid level. Okay, this is called as suicide inhibition. Okay, the inhibitor itself is converted into more potent form by the enzyme that it is inhibited. Okay, so allozanthine is been converted to more potent form, that is your allopurinol again. Okay, so this is one example, and the second example is aspirin, which is inhibiting a cyclooxygenase COX. Okay, so aspirin is a COX-1 COX-2 inhibitor. Okay, and your DFMO, what is DFMO? DFMO is your difluoromethylornithine. Okay, this inhibits your ornithine decarboxylase enzyme. Okay, then talking about a feedback inhibition, you know about the feedback inhibition, the final product of the pathway. These are inhibiting your rate limiting step, like a succinyl CoA. okay, because succinyl CoA plus glycine, okay, these were being converted by ELA synthase. ELA synthase was converting them to gamma ELA, okay, which will be giving a heme, and this heme will inhibitor ELA synthase enzyme, okay. And if the feedback inhibition this occurs at the genetic level, it is called as the feedback regulation. It's called as the feedback regulation. Understood? Okay. After this, we can have enzyme patterns in your liver disorders. Okay, in the liver disorders, you can see some enzyme patterns, some increased enzymes and all. Okay, which is a part of your clinical enzymology. Okay, it's a part of your clinical enzymology. Okay, so let's talk a little bit about clinical enzymology now. Okay. Yes, so talking about the clinical enzymology, we have first a term called as multi-enzyme complex. Okay, multi-enzyme complex is what? When all the enzymes of a reaction sequence, okay, are arranged in a multi-enzyme complex. Okay, all the reactions of a reaction sequence are arranged in a multi-enzyme complex. Okay, so that the product of the first reaction may be transferred to the second enzyme and so on. Okay, like A plus B combines to form C. Okay, and this C again gets transferred to C plus D. This C forms E, and this E again gets combined with F to form the G. Okay, so this is a multi-enzyme complex, like a fatty acid synthase complex. Okay, we'll see in the fatty acid synthesis. Okay, that is the multi-enzyme complex. In the pyruvate dehydrogenase complex, remember pyruvate dehydrogenase complex we discussed in our carbohydrate metabolism. Okay, there was many enzymes like this, E1, E2 and E3, which were forming the last 2.5 ATP at the end. Okay, what is ping pong reactions? The reactions of product are formed before all substances have reacted. Okay, products are formed before all substances have reacted. It's called as ping pong reactions, like your aminotransferases catalyze your ping pong reactions. Just an example. Then isoenzymes. What are isoenzymes? Physically distinct form of the same enzyme activity. Okay, physically distinct from the same enzyme. Okay, same enzyme, physically distinct from different structures, but function are identical. Okay. What is the process of formation? Your true isoenzymes are the product of different genes. Okay, like a salivary and pancreatic amylase, these are true isoenzymes. They are the product of different genes, entirely different genes. Okay, and second one, all different forms are in the same individual. Okay, like your isoenzymes of LDH. Okay, 5 varieties, LDH 1, 2 to 5. Okay, these are present in all individuals. Then we have a type called as allozymes. Allozymes, they result from differences in the single allele at the same genetic locus. Okay, the gene is same, the genetic locus is also same, but there is difference in the single allelic difference. So we call it allozyme. Okay, like your G6PDH has 400 different forms. Okay, produced by the same locus on X chromosome. Afterwards, polymorphism in isoenzymes due to variation in single locus, okay, occur with more than 1% in population. Okay, we call it as polymorphism. You must have heard about polymorphism in your 12th class. Okay. Properties of isoenzyme. What are the properties? That isoenzymes show tissue localization. Okay, like H4 is present in your heart, M4 is present in your skeletal muscles. Okay, they show tissue localization. We'll see what is H4 and M4. Okay, they show different mobility in electrophoresis. Okay, they are mobile to different extents. They have heat stability, different heat stability. Like your BALP is denatured by your heat. Your bone ALP is denatured by your heat. And then there is a thermolabile ALP also, which is not denatured. One may be sensitive to inhibitors. Okay, like a tartrate labile ACP is sensitive to tartrate. Okay, the different Km values, like glucokinase has less Km while hexokinase has more Km. Then on cofactor requirement, mitochondrial ICD, okay, it is NADP plus dependent, while cytoplasmic ICD is NADP plus dependent. Okay, talking about lactate dehydrogenase, it is told it is a multi-enzyme complex. It is a tetramer of 4 subunits. Okay, this yellow one are the muscle type, blue one are the heart types. Okay, so talking about LDH5, LDH5 is M4. M4 means all the muscle types are there. Okay, this is present in muscle. Okay, all the muscle types means present in muscle. You can remember like this. Then LDH4, LDH4 is a H1M3. Okay, more the muscle types and less heart type. Okay, more muscle type, less heart type. This is present in your liver. Then LDH3, H2M2. Okay, this you can remember, it is present in your brain. Okay, because you can remember like this, that brain has more, the same amount of blood as the same amount of brain tissue or the muscle. Okay, you can just remember like a silly mnemonic. Okay, or you can remember this mnemonic too. You can take a screenshot of this mnemonic, and I have no time to discuss now. Then LDH2 is H3M1, more heart type. Okay, because heart you can relate with your blood vessels. Okay, but it will have more blood means RBC. Okay, and LDH1 is H4, heart type, so it is present only in heart. Okay, and as you go down, the mobility increases. So which is more mobile? The LDH1, then LDH2, 3, 4 and 5. LDH5 is the least mobile. Okay, Okay, and about an abundance, okay, about the abundance, LDH2 is the most abundant, afterwards LDH1, LDH3, LDH4 and LDH5. Okay, there's a reversal pattern here. Okay, but when it is flipped, okay, it is called as a flipped pattern. Flipped pattern is of diagnostic importance. When it is flipped, like LDH1 is more than the LDH2, it is seen in a myocardial infarction. It is seen in a myocardial infarction. So LDH is a marker of your myocardial infarction too. Okay. Then talking about functional enzymes and non-functional enzymes. Functional enzymes are actively secreted into the plasma, while non-functional enzymes, they come out of cells due to normal wear and tear. Okay, they come out of cells spontaneously. Okay, and these are used for the diagnosis of disease. Talking about biomarkers, the biomarkers topic is important. Biomarkers are the substances which usually show the presence of a biological origin material which indicates a particular disease. Okay, these are substances which indicate a particular disease. Talking about cardiac biomarkers which show your cardiac disorders, we have cardiac troponins, okay, or is it TNI and TNT, cardiac troponins, creatine kinase, CKMB, and myoglobin. Okay, myoglobin is a non-specific cardiac marker. Then we have BNP, brain natriuretic peptide, which is also the indicator of congestive cardiac failure. This can come as MCQ questions also. BNP, brain natriuretic peptide, congestive heart failure, the cardiac failure. And let's talk about enzyme profiles in the liver disorders. Okay, in the liver disorders, like your alanine aminotransferase, ALT is there, ALT, okay, which is also called as your SGPT, serum glutamate pyruvate transaminase. Okay, so coenzyme is PLP, and the normal serum levels in the male is 13 to 35 international unit per liter, and females 10 to 30 international unit per liter. Okay, just remember the values of your ALT. 13 to 35 in males and 10 to 30 in your females. So you can well write 10 to 35 if the ALT levels are asked. Okay. And there is increase in level up to 300 to 1000 international unit per liter in case of acute hepatitis. Okay, and liver disorders, in liver disorders your AST, ALT increases. Okay, and ALT increases more than the AST. ALT increases more than the AST. Talking about 50 to 100 international unit per liter of ALT, this indicates chronic liver disorders. Chronic liver disorders like in your cirrhosis, your hepatitis C, and your NASH. Okay. How you estimate it? Okay, first you take alanine plus PLP. Okay, you add serum at 37 degrees Celsius. Okay, serum has your transaminase ALT. Okay, and this combines with your alanine to form your pyruvate, and pyruvate in the presence of your dinitrophenylhydrazine will give you a reddish brown hydrates. Okay, and intensity of red brown color will be the amount of pyruvate which will be proportional to the amount of your ALT also. Okay, so this is the estimation of your ALT. Talking about AST, aspartate aminotransferase, which is also called as SDOT, or the serum glutamate oxaloacetate transaminase. Okay, the coenzyme is PLP. Normal serum levels are 8 to 20. Okay, international unit per liter. 8 to 20 international unit per liter. Okay, it is used as a marker of myocardial ischemia in older days. Okay, and now it is used as a marker for hepatic injury. Okay, hepatic injury, liver disorders. And how you can estimate it? You put serum in a solution of aspartate and PLP. Okay, and at 37 degrees Celsius, then the serum has your transaminase, okay, your AST, and it combines with the aspartate to form oxaloacetate in the presence of your that dinitrophenylhydrazine. Okay, this DNPH. Okay, dinitrophenylhydrazine. Okay, it gives your reddish brown hydrazones. Okay, there it was the production of hydrates, but here it is hydrazones. Okay, and the intensity of the hydrazone gives the AST concentration. Then we talk about alkaline phosphatase. Alkaline phosphatase optimal pH is 9 to 10, as we discussed in our enzyme chapter. Okay, it is activated by Mn and Mg. This also we discussed previously. Production is from osteoblast of bone. Okay, osteoblast of bone, this produces your ALP. Okay, how can do the assay of ALP? Then serum ALP plus disodium phenyl phosphate. Okay, DPP, disodium phenyl phosphate, at the pH 10 will give you phenol. Okay, and phenol in the presence of your antipyrine, your amino antipyrine. Okay, phenol in the presence of amino antipyrine and potassium ferricyanide will give you a purple color. Okay, will give you a purple color. This is a assay. And reference serum value is your of that ALP is your 40 to 125 international unit per liter. Okay, ALP is 40 to 125 international unit per liter. And the infective hepatitis, alcoholic hepatitis, hepatocellular carcinoma. Okay, it moderately increases up to 2 to 3 times. Okay. In extra hepatic obstruction, like obstructive jaundice, it is increased to a greater extent. Okay, and in bone disorders, these are just limitless, like drastically increase in levels of ALP. Bone disease like your Paget's disease, your rickets, osteomalacia, osteoblastoma, metastatic carcinoma, bone, hyperparathyroidism. In this, it increases drastically. Okay. And isoenzymes of ALP, what are the isoenzymes? So we have alpha 1 ALP, okay, which increases in obstructive jaundice. Alpha 1 ALP, which increases in obstructive jaundice. Then alpha 2 heat labile ALP, okay, heat labile ALP means it is stable at 56 degrees Celsius but denatures at 65. Okay, just it is not thermally stable. Okay, then we have alpha 2 heat stable ALP, alpha 2 heat stable ALP. Okay, which is a placental origin. Just remember, alpha 2 heat stable ALP is a placental origin. It is not destroyed even at 65 degrees Celsius and it's inhibited by phenylalanine. Okay, phenylalanine inhibits the alpha 2 heat stable ALP. Okay, then we have pre-beta ALP, which is a bone origin, Okay, this is also heat labile, and this your heat labile beta pre-beta ALP. Okay, it is the marker of your bone diseases. Okay, talking about another marker, that's a nucleotide phosphatase or your 5-dex nucleotidase. Okay, which is active at the pH 7.5. Okay, it converts your 5-dex nucleotidase. Okay, and this in the presence of HOH converts into nucleosides. Okay, nickel ions are inhibiting the 5-dex nucleotidase. Okay, or your nucleotide phosphatase. It is an ectoenzyme present on the cell membrane. Okay, so it is present on the cell membrane, not inside the cell membrane. Normal NTP level is about 2 to 10 international unit per liter, 2 to 10 IU per liter. Okay, and it moderately increases in your hepatitis. Okay, and highly increases in your biliary obstruction. Okay, highly increases in your biliary obstruction. Talking about levels, okay, we saw your ALT levels or normal ALT levels. Okay, you can say 10 to 30 international unit per liter. Okay, afterwards we had the levels of your AST levels. Okay, AST levels were about 8 to 20 international unit per liter. Okay, and the ALP levels, these were about 40 to 125 international unit per liter. Then you saw your NTP levels. Okay, these are about 2 to 10 international unit per liter. Okay, we will get another other values also. Let's see, these values are important. These are asked in a viva. Okay, talking about the gamma glutamine transferase, which is a marker for your alcoholic liver damage, GGT. Okay, so GGT is used in the synthesis of your glutathione. It has 11 isoenzymes, monitoring of your metabolic syndrome, your non-alcoholic fatty liver disease, and your non-alcoholic steatohepatitis. Okay, NASH. NASH is also non-alcoholic steatohepatitis, non-alcoholic fatty liver disease, your NAFLD. Okay. Reference value is 10 to 30 international unit per liter, 10 to 30 international unit per liter. Okay, so you can write GGT levels also. It is about 10 to 30 international unit per liter. Okay, the same values as your ALT. Okay, then clinically it is important because of sensitivity to alcohol abuse. Okay, the GGT levels are clinically important due to sensitivity to alcohol abuse. GGT level indicates the amount of alcohol that is intaken by a person. Okay, then a prostate specific antigen, which is called PSA. Okay, prostate gland epithelium releases your PSA, which is secreted into a seminal fluid and used to break the coagulum. Okay, the semen coagulates after some time, so it is used to break the coagulum. Okay, it is a serine protease. In blood, it is bound to alpha 2 macroglobulin and alpha 1 antitrypsin. It is bound form. Okay, and the reference serum value is about 1 to 5 international unit per liter. 1 to 5 international unit per liter. Values above 10 will indicate your prostate cancer. Values above 10 indicate your prostate cancer. So you can add another thing also. You can add your what is prostate specific antigen. It is about 1 to 5 international unit per liter. Okay, then afterwards acid phosphatase. Okay, acid phosphatase or your ACP, which acts at pH of 4 to 6. Okay, secreted by your RBC, WBC, platelets and prostatic cells. Okay, inactivated by your tartaric acid. So tartaric acid was inhibiting acid phosphatase. We saw in the isoenzymes part. Okay, and increases also in case of a prostate cancer, and highly increases in case of a bone metastasis also. Okay, bone metastasis is an important tumor marker also. We talked about your acid phosphatase in the tumor markers also. You can just go and check out the previous parts. Okay, then you have cholinesterase. Okay, cholinesterase. So this was your acid phosphatase. Afterwards we will have cholinesterase, your acetylcholinesterase, which breaks down the acetylcholine enzyme. Okay, and it is present in the nerve endings and RBCs, acetylcholinesterase. Okay, this is also called as your true cholinesterase or type 1 cholinesterase. Okay, and the cholinesterase level in RBC will be proportional to the reticulocyte count. Okay, so it indicates the reticulocyte count. Okay, understood. Then afterwards there is organophosphorus insecticides. Okay, like a parathion. Okay, this irreversibly inhibit your acetylcholinesterase in your RBC. Okay, so measurement of CHE levels is done to determine the amount of exposure to organophosphorus poisoning. Okay, so acetylcholinesterase is important in organophosphorus poisoning. And then you have pseudocholinesterase, that is your type 2 cholinesterase. Okay, which is non-specific and produced mainly by your liver cells. Okay, and the succinylcholine, this is hydrolyzed by the liver cholinesterase within 2 to 4 minutes. Okay, and prolonged choline apnea. Okay, prolonged choline apnea may result in a nightmare of the anesthetist. Okay, prolonged choline apnea may lead to your nightmare of the anesthetist. Okay, this is of diagnostic importance. I have no time to explain this phenomenon also. Okay, talking about the glucose 6 phosphate dehydrogenase G6PD. Okay, this is the dimer. Okay, it is important to HMP shunt. Okay, you have read about this in HMP shunt. Okay, production of your NADPH, for the production of your NADPH, where in the HMP shunt also NADPH production is occurring. Okay, normal value in RBC is about 6 to 12 international unit per gram of hemoglobin, Okay, if there is deficient G6PDH, okay, if there is deficient G6PDH, the RBC lifespan is reduced and there is no more manifestations. But if there is deficient G6PDH plus your exposure to your oxidizing drugs like aspirin, primaquine, sulfa drugs, your mepacrine, okay, this is all drug exposure. To deficient G6PDH will lead to your hemolytic anemia. Okay, will lead to hemolytic anemia, and hemolytic anemia due to faba beans is called as a fabuism. Okay, and G6PDH deficiency also protects the person from your falciparum. So in hemolytic anemia due to your G6PDH, okay, falciparum resistance is there. Okay, then we have to talk about two other markers, that is an amylase like your pancreatic amylase, salivary amylase. Okay, this increase in your pancreatitis. And then a lipase, which is a triglyceride, which converts triglyceride to monoglyceride and free fatty acid. Okay, which increases in your acute pancreatitis. So lipase is an indicator of your acute pancreatitis. Okay, then we have enolase. Enolase is a glycolytic enzyme, we know. Okay, so the neuron specific enolase, the NSE, neuron specific enolase, is acts as a tumor marker. Okay, we saw in your previous discussions. Okay, it acts as a tumor marker and measured by your radioimmuniscent assay and your ELISA. Okay, ELISA we'll discuss in a while. Okay, then about the ceruloplasmin, the ferrooxidase. Okay, reference serum value is about 25 to 30 milligram. This is not international unit, this is about milligram per deciliter, 25 to 50 milligram per deciliter. Okay, ceruloplasmin, which increase in inflammatory reactions, collagen disorders, in malignancies and in pregnancy. Okay, ceruloplasmin increases. Then enzymes which act as therapeutic agents like streptokinase is there, okay, and urokinase is there. These both are used in the myocardial infarction, okay, for treating myocardial infarction. Okay, and your trypsin and lipase, these are used in patient with defective digestion. Okay, asparagine is used as an anti-cancer agent. Okay, then enzymes used for diagnosis of glucose oxidase to estimate your glucose concentration. Urea is to measure the quantity of ammonia in urea in our biological fluid, and restriction endonuclease in recombinant DNA technology. Okay, these are used for your diagnosis. Okay, and markers for muscle disease, we have CKMM, your creatine kinase, MM, which is present in muscle, your AST, not specific. Okay, but the ALP, it is the earliest to rise in muscle disease. Okay, it is also not specific. Okay, so these are your markers for your muscle diseases. Okay, so after this clinical enzymology, okay, after finishing this clinical enzymology, let's move on to another part of your enzyme topic, that is a regulation of enzymes. Okay, regulation of enzymes, let's talk about them. Talking about the regulation of enzymes, first let's see the allosteric regulation. Okay, so we have allosteric activation, which enhances your enzyme activity, and allosteric inhibition, which represses your enzyme activity. So the modifier binds to your allosteric site and increases and decreases the enzyme activity alongwise. Okay, so the modifier is not a substrate analog because it is not competitive inhibition. Okay, and it is partially reversible. The Km increases by this and Vmax decreases. Okay, and the most allosteric enzymes have a quaternary structure, and the kinetics of allosteric modification governed by the Hill's equation, and you get a sigmoid curve if you plot the enzyme kinetics. Okay, like your ALA synthase, which is inhibited by your heme, okay, and the citrate synthase inhibited by your ATP, and the PFK inhibited by your AMP and ATP. PFK is stimulated by your AMP and inhibited by your ATP. Okay, these are your allosteric regulation. Okay, about feedback inhibition, like A plus B, you have an enzyme. Okay, this enzyme you produce C. Okay, and C will inhibit the enzyme. Okay, like inhibition of hexokinase by glucose 6-phosphate. Okay, so glucose from glucose 6-phosphate converts to glucose 6-phosphate, and the hexokinase enzyme is there. And this hexokinase is again inhibited by glucose 6-phosphate. Okay, understood. In purine synthesis also, you will see first step, the carbamoyl phosphate plus your aspartate. In the presence of your aspartate, trans carbamylase forms the carbamoyl aspartate. Okay, and the last step, CTP is produced. So the CTP will inhibit your aspartate trans carbamylase. Okay, in the purine synthesis, this is also an example of a feedback inhibition. Okay, afterwards, talking about a homotropic effect. Okay, the enzyme is in a tense state in the absence of the substrate, but when there is the presence of substrate, the enzyme comes into a relaxed state. Okay, this is called as a homotropic effect. Then your induction or derepression, derepression. Okay, we have you must have read about a lactosoperon. We talked about a lactosoperon in the molecular biology today only. Okay, and so you have an inducer and a repressor. The operator site is there in the repressor. Okay, so inducer, it binds to the operator site. Okay, inducer prevents the binding of the repressor protein to the operator site. Okay, like that, and induces this. and biosynthesis will occur, like the transcription of your structural genes will occur. Okay, like a lack of operon. Okay, like a glucokinase is induced by insulin, the allosynthesis is induced by barbiturates. Then repression acts at the genetic level, gene level. It is the delay in production of the effects and reduces the number of enzyme molecules. It reduces the number of enzyme molecules. Okay, so regulator gene, this produces aporepressor and a co-repressor, which form the holorepressor. Okay, and this holorepressor binds to your operator region, okay, and prevents the transcription. So heme is produces if the heme is produced in plenty, it acts as a co-repressor and shuts up the operator site. The further production of allosynthesis will be stopped. Okay. Then we have another thing which is called as a covalent modification. Covalent modification is increases or decreases the activity, which can increase or decrease the activity by formation of a covalent bond. Okay, so here is the substrate and the enzyme. Okay, in the between there is a formation of covalent bond between the enzyme and the substrate. Okay, this bond when cleaved produces your enzyme and the substrate. Okay, so what are the examples of zymogen activation by partial proteolysis? Okay, this is a irreversible covalent modification. Okay, and it is a reversible reaction except the zymogen activation, which is an irreversible part. Okay, and commonest type is a protein phosphorylation. Talking about reversible covalent modifications, you have phosphorylation, dephosphorylation, you have methylation, you have adenylation, ADP ribosylation, and acetylation. Okay. So talking about enzymes active in phosphorylated state versus enzyme activate in a dephosphorylated state. Okay, you can remember the mnemonic GC Panda hero. Okay, GC Panda is a person who is the hero of your film you have made. Okay, in the phosphorylated state. Okay, so G stands for glycogen phosphorylase, C is for citrate lyase, P is for phosphorylase kinase, H is for HMG CoA reductase kinase. Okay, and you're active in a dephosphorylated form is your glass key. Okay, so you can remember glycogen synthase, and key is for key enzymes of glycolysis. Okay, so glass key, you can remember glycogen synthase, key from key enzymes of glycolysis. Okay, hadpa. Okay, hadpa is a Hindi term. Okay, so you had a glass key, okay, which you took forcefully. Okay, glass key, hadpa. Okay, hadpa you can remember HMG CoA reductase. Here in phosphorylated state it was HMG CoA reductase kinase. Okay, but in a dephosphorylated state it is HMG CoA reductase with acetyl CoA carboxylase and pyruvate dehydrogenase. Okay, then what is ADP ribosylation? This ADP ribose is added to an enzyme or a protein. Okay, like your cholera toxin and your pertussis toxin, this act via ADP ribosylation. Okay, remember we learned your molecular biology of the diphtheria toxin. Okay, and the ADP ribosylation of EEF2 ADP ribosylation is acting by it. Okay, remember? Yes. Afterwards, then ADP ribosylation of G3PD. Okay, ADP ribosylation of your G3PDH. Okay, this enzyme will cause inhibition of your glycolysis. Okay, then what is stabilization? It is a prevention of wear and tear of enzyme. Okay, because due to wear and tear the enzymes will get degraded. So the stabilization is done due to prevent the wear and tear of enzymes. Okay, like a degradation of tryptophan pyrrolase. Okay, the degradation of tryptophan pyrrolase is stabilized by your tryptophan. Okay, the phosphofructokinase is stabilized by your growth hormone, your papain, in the urease. This both are stabilized by your glutathione. Okay, talking about another term is the compartmentalization. It is the difference in the localization of an enzyme. Like one enzyme may have mitochondrial part and the cytoplasmic part also. Okay, mitochondrial and the cytoplasmic form. Okay, difference is in your localization. Okay, like a heme synthesis enzymes, your urea cycle and your gluconeogenesis pathways. Okay, there is compartmentalization of your enzymes. Okay, enzymes are present at different locations so as to regulate the enzyme activity. Okay, so this completes your whole enzyme topic. Okay, now let's move on to the next chapter. Let's discuss your protein metabolism. Okay, so here is the structure of a protein. You know the basic structure of a protein. Okay, you know the basic structure of a protein. You have a carbon and you have a hydrogen attached to it. You have COOH group attached to it. You have an H2 group attached to it. Okay, and you have the R group. R group is a variable group. Okay, so you have 20 amino acids. Okay, which make up the most of the proteins. You have 20 amino acids making up the protein. Okay, and glycine is the simplest amino acid. You know. Okay, that is H group present in the place of R group. Okay, so it has no alpha carbon or asymmetrical carbon. We talked about asymmetrical carbon in your carbohydrate chemistry. Okay, and human body has mostly L amino acids are found. Okay, but exception is there D-serine and D-aspartate. These are also found in your human brain tissue. If we classify the amino acid based on side chain, based on polarity, based on glucogenic or ketogenic action, okay, or it is based on your nutritional requirement. Based on nutritional requirement, it is essential amino acids. Okay, then a semi-essential amino acids and your non-essential amino acids. Okay, we'll discuss about them. Okay, ketone bodies, these are acetoacetate and beta hydroxybutyrate. So these two are called your ketone bodies. Okay, just remember. Based on your side chains, we have amino acid aliphatic side chain. Okay, aliphatic group is present when the carbon chain is present. Okay, so which have aliphatic side chains? Your glycine. Okay, your I love bases. You can remember I love bases. Okay, your isoleucine, your leucine, and your valine. Okay, these are branched chain amino acids. Your glycine and alanine. These are also aliphatic amino acids but unbranched. Okay, so in aliphatic you can have your branched chain amino acid, that is your isoleucine, leucine, and valine, and your unbranched amino acid, that is your glycine and alanine. Amino acid with OH group is a serine and threonine. There is a OH group present. With the side chain having sulfur atoms, you have cysteine and methionine. Okay, with acid groups present, glutamic acid and aspartic acid. With basic groups, you have glutamine and asparagine. Okay, from the acid you made the amines. Okay, so it became basic. And histidine and arginine, these are also basic amino acids. Amino acids is aromatic ring. This is a phenylalanine, tyrosine, and tryptophan. This is asked much. With the heterocyclic ring, you have histidine. Okay. Here are the structures of amino acid. You can see the glycine is the simplest amino acid, represented by GLY. Okay. Then is your alanine, where R group is your CH3 group, methyl group present in the R group. Okay. Then you have valine. Valine is having a branch point here. A branch point is here. Okay. This is a branch chain amino acid, branch chain aliphatic amino acid. Then you have a leucine. Okay. This is having also branched. Okay. CH2 is more than as compared to your valine. Okay. Then your isoleucine is there. Okay, then the proline is there. Proline is an amino acid. Proline forms the fibrous structure of collagen. We saw in collagen synthesis. Yes, proline and hydroxyproline. Understood? And proline is also called as an amino acid. Because the imidazole ring. This is the imidazole ring which is present. Okay, then you have phenylalanine having the this benzene atom. Okay, the benzene group. Okay, and the tyrosine is having the phenol group here attached to this. Okay, tyrosine is slightly polar amino acid. And the tryptophan has an indole ring. Okay, look at this. This is called as a indole ring. Okay, and the blue color to urine, if the tryptophan is not metabolized, the urine is blue color. Okay, and this is called as a blue diaper syndrome. Okay, and then you have a lysine. Okay, lysine is called as an epsilon amino acid because this is an epsilon amino group. Okay, so you can remember epsilon amino group is present in your lysine. Just these are facts. Okay, this is called as epsilon amino group. Okay, understood? And you have now arginine. Arginine has guanidium group present. Okay, guanidium group is present in arginine. And histidine of a heterocyclic ring containing carbon, nitrogen, as well as hydrogen. Okay, heterocyclic ring is present in histidine. Okay, this lysine, arginine, and your histidine, this LHA or LAH, this are you call your positively charged amino acids. Okay, and your aspartate, which forms your aspartic acid, glutamate, which forms your glutamic acid. Okay, if you add NH2 in your aspartate, okay, you will get asparagine, which is your basic amino acid. And if you also add NH2 in your glutamic acid, okay, you will get glutamine, which is also a basic amino acid. Okay, and then you have cysteine has a sulfur hydryl or thiol group present, so it is a sulfur containing amino acid. Okay, and then you have methionine has this S bond. Okay, S, and this methionine can make your cysteine. Okay, so methionine is essential amino acid while cysteine is a non-essential amino acid. Okay, we have 20 amino acids. We saw the structure of 20 amino acids. Okay, recent discovery is we have the 21st amino acid, that is your selenocysteine. Okay, selenocysteine coded by UGA codon. Okay, and there is 22nd amino acid also. Teachers may love to ask these questions. Okay, so as to know if you are updated with the recent things and we are reading standard textbooks. Okay, this is given in a Harper. Okay, and you can find in any other resources you are using. Okay, so 21st amino acid is your selenocysteine and the 22nd amino acid is your pyrrolysine. Okay, coded by UAG. These are coded by stop codons, please note. Okay, based upon polarity, you have non-polar amino acids which you can remember by gap-blim T, this mnemonic, and you have polar amino acids. So non-polar, these are your glycine and alanine, and P stands for phenylalanine and proline, V is for valine, L is for leucine, I is for isoleucine, M is for methionine, and T is for tryptophan. Okay, these are non-polar amino acids. Okay, non-polar. Okay, these don't have any positive and negative charges. Then a polar amino acid. Okay, which are uncharged, which may be acidic chain and which may contain basic chain. Okay, uncharged amino acids, these are your serine, threonine, tyrosine, asparagine, glutamine, cysteine. Okay, and just don't remember this all like this. Okay, you have acidic amino acids, acidic chain, aspartic acid, glutamic acid, you know this, and a basic side chains. Okay, LAH I told. Okay, lysine, arginine, and histidine, and rest all amino acids are your uncharged amino acids. Okay, Then if you condense two molecules of cysteine and cysteine, okay, the SS bond is formed. Cysteine S cysteine is a cysteine. Okay, cysteine. Okay, cysteine is the least soluble of all amino acids. Based on polarity of non-polar or you have polar amino acids. Okay, non-polar is called as hydrophobic amino acid. Polar is called as hydrophilic amino acid. Okay, so branched amino acid like your isoleucine, valine, and your leucine. These are your non-polar amino acids. You have aromatic amino acids like your phenylalanine, tyrosine, and tryptophan. These are also non-polar amino acids. Okay, then simple amino acids like lysine and arginine, alanine. Okay, these are also non-polar amino acids. And then you have sulfur containing amino acid methionine, which is also a non-polar amino acid. Then we have polar amino acid, that are charged amino acids. Okay, charged and uncharged. So you have in positively charged amino acid, you have LAH, your lysine, arginine, and histidine. How can you remember that? These were your basic side chains, and basic means your negative charge only. Basic means your negative charge. So you can remember from this. Basic means a positive charge. What I'm saying, basic means a positive charge. Okay, there must be positive charge in this. So these all will be positively charged. Okay, and negative charge means acidic amino acid, glutamic acid, aspartic acid, and all the uncharged amino acids. These are there. Okay, you know this. Okay, histidine is a polar amino acid, and this is an exception here. Okay, histidine is a polar amino acid, and this is a exception here. Okay. Based on metabolism, we can have your glucogenic or your ketogenic amino acids, glucogenic or ketogenic amino acids. So glucogenic, you have phenylalanine, you have isoleucine, you have tyrosine, and tryptophan. Okay, you can remember PIT, PITT, phenylalanine, okay, isoleucine, tyrosine, and tryptophan. Okay, but isoleucine is the most branched amino acid, so it is the most non-polar. It is the most non-polar amino acid also. Okay, ketogenic, you can remember there is leu always lies, Okay, leucine and lysine are your ketogenic amino acids. Okay, except your leucine and lysine, all other are glucogenic. Okay, some are also glucogenic plus ketogenic. Okay, so some amino acids are there like that also. Okay. Okay, based on the nutritional requirement, you can have essential amino acids and non-essential amino acids. Okay, essential are your about 8 amino acids are essential and 2 amino acids are semi-essential. Okay, this become essential at the time of growth only when they are needed. Okay, this become essential. These are added to the essential class. Histidine and arginine, and histidine, these are your semi-essential amino acids. Essential, you have isoleucine, leucine, tryptophan, lysine, methionine, valine, threonine, and your proline. Okay. And non-essential or rest all the amino acids. 21st amino acid is a selenocysteine, as I told earlier. This is a structure of selenocysteine, not much important. Okay, coded by your UGA codon. Okay, UGA codon, and specified by an unusual tRNA called as tRNA sec. Okay, SEC, difference of selenocysteine. It is a good antioxidant, and it is a polar, acidic, and strong nucleophilic. Okay, diseases due to SEC. SEC also causes your diseases like your Kissan's disease. Okay, cancer prevention. This also helps in the cancer prevention. Thyroid hormone regulation. Deficiency will lead to lean body mass, premature aging, and heart diseases. Okay. Then uses of amino acid. What are the uses? Uses in the nerve transmission. You use amino acids. These are building blocks of your proteins. Biosynthesis of porphyrins. Okay, purines, pyrimidines, and your urea. These are all synthesized. Okay, these are your synthesized peptide hormones. Okay, and this make up your hormone releasing factors, your neuromodulators, neurotransmitters. These all have your amino acids. Okay, it is essential for your blood coagulation. Okay, blood coagulation. And tyrosine is the precursor of a thyroid hormone. Glutamate is the precursor of a gamma aminobutyric acid, or your GABA. Okay, just remember, glutamate is the precursor of GABA. Tyrosine is the precursor of thyroid hormone. Okay, ornithine and citrulline, these are your intermediates in the urea biosynthesis. Your homocysteine, homoserine, and glutamate semialdehyde. Okay, these are your intermediates in metabolism of protein and amino acids. Okay, homocysteine, homoserine, and glutamate gamma semialdehyde. Okay. And there are some potentially toxic L-alpha amino acids. This is a table of your Harper textbook. Okay, like in homoarginine. This is implicated in neurolethyrism. Okay, then your beta ODAP, that is your beta and oxalyl diaminopropionic acid. This is also indicated in your neurolethyrism. Okay, then your BAPN, or your beta and glutamyl aminopropionine nitrite. Okay, this is the glutamine derivative of beta aminopropionine nitrite. This is osteolethyrosin. Okay, this lethyrism is the disorder that is odoratus, lethyrus odoratus, your sweet pea. Okay, this is all are caused by this. And you have the ornithine analog, which is called as your 2,4-diaminobutyric acid, or your alpha gamma diaminobutyric acid, which is the ornithine analog. Okay, and it inhibits your ornithine transcarbamylase activity. Okay, OTC enzyme will see in your protein metabolism, which will lead to your ammonia toxicity. Okay, and then your L-beta methyl aminoalanine, okay, which is a neurotoxic amino acid present in the cycad seeds. Okay, which is possible risk factor of a neurodegenerative disorders like amyotrophic lateral sclerosis. Okay, AMS, of which Stephen Hawking was a patient. Okay. And neuroletharism, talking a little bit about, it is a profound neurological disorder. Okay, there is a spastic paralysis in the legs. Okay, and it occurs widely in your famines. Okay, where there is increased consumption of Lathyrus seeds, Lathyrus odoratus, sweet pea seeds. Okay, so there is neuroletharism caused. Okay, so this was all about the classification of amino acids. Let's go on to the next part. Talking about the properties of functional group of amino acids are positive, negative, or zero net charge. They can have these three types. Okay, so physiological pH. Okay, physiological pH is about 7.4. Okay, imidazole group of histidine and guanidine group of arginine exist as resonance hybrids. They exist as resonance hybrids for the increased stability, like histidine and your arginine. They exist as resonance stable structures. Okay, resonance stable structures. And we have a Jeter ionic form. Jeter ionic form means there is no net charge, or it is electrically neutral. Okay, no net charge but ionized neutral species. Okay, this have your positive and negative signs. Okay, but this the net charge will be 0. Okay, and these are your isoelectronic species. Jeter ionic forms. Okay, what is the isoelectric pH, also called as the PI? At isoelectric pH, okay, this is a pH midway between the pKa values of ionization in both sides of the species. Okay, let's say, have I given an example for this? But there's no example. You can just remember like this. This is isoelectric pH. Okay, it is the pH. This is a pH midway between pKa values. Okay, if there is the pKa value of COOH. pKa value of COOH for alanine is about 2.35. The pKa value of your amine group is about 9.69. Okay, so isoelectric pH will be Pi equal to pK1 plus pK2 by 2. Okay, you just sum this by 2 is just 6.02. This is the isoelectric pH for alanine. Okay, at isoelectric pH of alanine, the alanine will show no mobility. Okay, no mobility in electrophoresis, minimum solubility, maximum precipitability. Okay, at isoelectric pH generally the proteins get precipitated. Okay, and minimum buffering capacity. Okay, isoelectric pH, no mobility, minimum solubility, maximum precipitability, and minimum buffering capacity. For lysine, this is Pi is equal to pK2 plus pK3 by 2. Okay, and knowledge of Pi is important for it sets the condition for electrophoretic separation. Okay, Okay, and it is also used in your this underlies your chromatographic separations. Okay, so Pi is useful in your electrophoresis and your chromatography too. Okay, and the pKa of a dissociable R group will depend upon location within a protein. Neighboring groups can increase or decrease the pKa. Okay, so pKa value is not much constant. It depends upon a neighboring group. Okay, neighboring group will, if it goes into localization, if it is getting the electrons are getting localized. Okay, your resonance hybrids are formed, and this can this can change your pKa values. Okay, soluble about the solubility of protein. These are soluble in the polar solvent like water and ethanol. Okay, but insoluble in non-polar solvents like benzene, hexene, and ether. Okay, absorption of light. Okay, proteins don't usually absorb your visible light. Your amino acids don't usually absorb your visible light. They are generally colorless. Exceptions are there like tyrosine, phenylalanine, and tryptophan, the aromatic amino acids. Okay, these absorb high lambda UV light, high wavelength UV light, about 250 to 290 nanometer. They can absorb the light. Okay, they can absorb the light. So you can see the curves of the tryptophan, the tyrosine, and the phenylalanine. Okay, this is also a part of your Harper textbook. That's why I have covered. Okay, then this exhibit your chemical reactions. Okay, due to COOH group, formation of esters, amides, and anhydrases are there. Due to NH2 group, your acylation, amidation, and esterification reactions are there. Okay, so they exhibit chemical reaction due to the COOH group or due to NH2 group. Due to COOH group, they show the formation of esters, amides, and anhydrates, and due to NH2 group present in them, they will show the reactions like acylation, amidation, and esterification reactions. Okay, these charged R groups, they stabilize protein conformations via ionic interaction or your salt bridges. Okay, this is called as your charged relay system. So these are all your physical properties. These are all your properties of your amino acids. Let's discuss about another things. Talking about the structures of proteins, the primary structure, the secondary, the tertiary, the quaternary structures. So primary structure is basically your amino acid sequence, the amino acid residues. Okay, and like lys, leu, tyr, and gln. Okay, this is lysyl, leucyl, tyrosyl, and glutamine. Okay, this is tetrapeptide. Okay, so these are amino acid sequences. So these are called as primary structure of proteins. Okay, which will have a N terminal and a C terminal present. Okay, which will have a N terminal and a C terminal. Glutathione, is a tripeptide, participates in metabolism to your xenobiotics. Okay, understood. After we have non-protein amino acids like your D-phenylalanine, and your ornithine, which is present in antibiotics, tyrosidine and gramicidin. Antibiotic and gramicidin antibiotic. Okay, where non-protein amino acids like your D-phenylalanine and your ornithine are present. What is peptide bond? Peptide bond exhibits your partial double bond character. PDBC. Okay, partial double bond character, or your PDCH. this all from your previous classes or organic chemistry. Okay, this nitrogen, this gets localized and like this moves like this. The nitrogen, let me show you. This unpaired electron will be given here, and this double bond will shift over the oxygen. It is more electronegative, so negative charge will develop over oxygen and positive charge will be developed over here nitrogen. Okay, so this is called as a partial double bond character, and this now can't rotate. This rigid rotation, semi rigidity. Okay, your rigid rotation or semi rigidity is conferred by the peptide bond. So this is the peptide bond formation. Okay, and semi-rigidity, this determines the manner of folding of proteins and peptides. Okay, based on the rigidity, okay, which will determine the manner of the folding of proteins and peptides. So this is the semi-rigid structure. There is the distance between adjacent alpha carbon atoms is 0.36 nanometers. Okay, the peptide bond is uncharged at normal physiological pH. The peptide bond is uncharged. Okay, at normal physiological pH. Understood? And we have few angles. These are your psi angle and your phi angle. Okay, the angle between your nitrogen and the carbon. Okay, the nitrogen and the carbon. This is called as your psi angle. This is called as a psi angle, and the angle between carbon and carbon, this is called as a phi angle. Okay, understood? So these are few angles. And the life of a protein, how a protein is born. Protein is born by a translation. Then there is post-transcriptional modifications like a selective proteolysis and all. Okay, ages by oxidation, deamidation, and denaturation. Can die as forming constituent amino acids. Okay, and again these constituent amino acids are taken to do a translation, and after from translation again dies, and the cycle goes on. Okay, which contributes to the amino acid pool in the body. Okay, so this is a life cycle of a hypothetical protein. You can take a screenshot here. Done? Yes. About the purification of proteins, we have the chromatographic techniques like a size exclusion chromatography, or ion exchange chromatography, or hydrophobic interaction chromatography, or affinity chromatography. Okay, recent advance we know HPLC, or high pressure liquid chromatography. Okay, another thing is that it is called as Stokes radii. Okay, this is the effective volume occupied by a protein as it rapidly tumbles in free solution. Okay, you put an amino acid in a free solution. Okay, so how much volume it effectively takes? It is called as a Stokes radii. Okay. Understood? So these are given in a Harper, so I have covered this all. Okay, then ion exchange chromatography, and is a protein. Okay, at given pH, a positive charge, negative charge. Okay, if there is positive charge, okay, then it adheres to the negative charge functional groups like a carboxylates and sulfates, and if it is negative charge, this adhere to beads with positively charged like a tertiary and quaternary amines. Okay, then protein purity is assessed by a polyacrylate gel electrophoresis page, SDS page, or a page in the presence of anionic detergent sodium dodecyl sulfate. Okay, then we have the contribution of Frederick Sanger, who took insulin, reduced the SS disulfide bonds between A and B chains, okay, and cleaved each chain using the protein cleaving enzymes, okay, and treated a Sanger's solution. What is Sanger's reagent? Sanger's solution. Sanger's reagent is your 1-fluoro-2,4-dinitro benzene. 1-fluoro-2,4-dinitro benzene. Okay, the Sanger's reagent, it reacts with alpha amino acids of amino terminal residues, and the peptide content is determined. This is called as the Sanger's method. Okay, and he got Nobel Prize for complete sequence of insulin and techniques of DNA sequencing. Okay, 2 Nobel Prizes. Then we have an Edman reagent. What is Edman reagent? This is phenyl isothiocyanate. This is called as Edman's reagent, phenyl isothiocyanate, which is selectively used to label the amino terminal residue of the peptide. Which terminal? Amino terminal. Okay, and it is now replaced by mass spectrometry, which is an advanced technique again. Mass spectrometry means to analyze the metabolites, used to analyze the carbohydrate, the lipid, post-translational modifications. Okay, increment of mass to a protein, and etc. Okay, so you use mass spectrometry. Okay, this is the process from mass spectrometry. Laser is irradiated and the quantities are analyzed. Okay, yes, and then we have a tandem mass spectrometry, in which 2 mass spectrometers are used in series. This is even more advanced. Okay, tandem mass spectrometry, MSMS or MS square it is called. Okay, and it is used to screen the blood samples from newborn for the presence of amino acid, fatty acid, and metabolites. Okay, we use tandem mass spectrometry. Okay, understood? And what is proteome? This is the identity, abundance, and the state of modification of the entire suite of proteins expressed by a particular cell at a particular time. This is called as a proteome. Okay, so this was about peptide bond and purification of proteins. Let's move on to the next topic. Okay, so let's talk about higher orders of structure of proteins. Okay, some diseases are there which impede the protein maturation, like a genetic deficiencies, like a Creutzfeldt-Jakob's disease, the CJD. okay, which is the human analog of your bovine spongiform encephalopathy. This is also a genetic deficiency which will impede the protein maturation. Another disease is a Scrapie disease, Alzheimer's disease, and the BAC disease, mad cow disease, which I told now. And nutritional deficiencies like your scurvy and your Menkes syndrome, your Menkes disease or Menkes kinky hair syndrome. Okay, these are the diseases which impede protein maturation. Talking about conformation, this is a specific 3D arrangement. Okay, this is a special relationship of every atom. Okay, and configuration is retained during interconversions. Okay, a typical peptide has about 10 to the power 50 different conformations. 10 to the power 50 different conformations. There are 4 orders of protein structure. The primary order, that is sequence of amino acids which we talked, the peptide bond formation and all. Secondary structure, which has about 3 to 30 residue arrangements, okay, which are geometrically folded ordered units. The secondary structure may be alpha or beta, your alpha helix or your beta pleated sheets. Okay, like a alpha helix is like this and a beta pleated sheet is like this, bonds in between them. Okay, then a tertiary structure is the mature polypeptide, and quaternary structure, okay, and which is the number and types of polypeptide and the spatial arrangement in the space. Okay, let's talk about secondary structure. It is alpha helix formation and a beta pleated sheet. Okay, the alpha helix, it has the psi angle of your minus 57 degree, and your phi angle is your minus 57 degree. Okay, and the psi angle is minus 47 degree. We talked about the psi and phi angles in the previous discussion. Okay, and one complete turn will have about 3.6 amino acid residues. Okay, the pitch of the helix is about 0.54, is about 0.54 nanometers. Okay, and in proteins only right-handed helices are present. In proteins only right-handed helices are present. Okay, so for your determination of your alpha helix and beta pleated, we use the Ramachandran plot. So this is the Ramachandran plot. You can see, okay, this is a curve, okay, plotted by Ramachandran, where we plot your psi, okay, we plot your psi with relation to your phi. Okay, the psi and phi are thus important. Okay, we plot your psi in relation to your phi, and we plot the Ramachandran plot, okay, for determining if it is alpha helix or beta pleated. Okay, it is also used in collagen, or determining the structure of the collagen. Okay, so you can see the alpha helix, there is 0.54 nanometers pitch. Okay, this is the pitch of the helix, and this is between N and C you have 0.15 nanometer. Okay, if you take transfer section, this will look like this. Okay, and stability of alpha helix is due to hydrogen bond, which is the intramolecular hydrogen bond, okay, between oxygen atoms of peptide, carbonyl, and H of peptide bond. Okay, and H and N of the fourth residue. Okay, is form a H bond, intramolecular H bond. Then your van der Waals attractions also increase the stability of your alpha helix. Okay, understood. So in proline there is a peptide bond, nitrogen will lack a H atom. Okay, peptide bond nitrogen will lack an H atom, so it is incapable of forming a hydrogen bond with a carbonyl oxygen. Okay, so it produces a bend. Okay, so you can find a bend in your proline. Okay, and your glycine it has a small R group, okay, which will produce bends in the alpha helix. Okay, so glycine is every third amino acid in your collagen is your glycine. Okay, and it produces the bends. Okay, it can be accommodated in a small space. Then have amphipathic helices, which are both hydrophilic and hydrophobic groups projecting on the opposite sides, which will create channels and pores. Okay, so these amphipathic helices are mainly forming a translocon channels and protein channels, which are allowing specific substances in polar molecules to pass through the cell membrane. Okay, talking about a beta pleated sheet, these amino acid residues form a zigzag or pleated pattern. Okay, where R groups project in opposite directions. Okay, and intramolecular H bonds are present and are responsible for the stability of the beta sheet. Okay, alpha sheet were stabilized by your in your intramolecular. Here intermolecular hydrogen bonds are present between the carbonyl oxygen and your amide hydrogen. Just see, in carbonyl oxygen and hydrogen you can have intermolecular H bonds, intermolecular H bonds. So beta sheet can be your parallel beta sheet, or your anti-parallel beta sheets are also there. Okay, this is your anti-parallel beta sheet. So it is arranged like this, then again this alternate manner. Okay, and the parallel beta sheets, these are arranged in the same type of manner. Okay, parallel beta sheets, and most beta sheets have right-handed twist only. Okay, beta barrels, what are beta barrels? These are clusters of twisted strands of beta sheet, Okay, talking about turns and bends, these are not much important, as your beta turns and bends are there. Okay, the beta turn is usually present in the proline and glycine, if the examiner tries to ask. Okay, talking about loops are regions that contain certain residues beyond the minimum number of residues necessary to connect adjacent regions of secondary structure. Okay, so these loops have certain residues, like a protein is there. Okay, so these are the loops, and this have a crowding of the residues. Okay, these are crowding of the residues which induces the loop, okay, in the secondary structure. Okay, and these loops also serve important functions like biological roles or catalysis. Okay, so these are your higher order structures. Let's talk a bit about color reactions. We have the biuret test, which is a general test for your proteins. Okay, here in alkaline medium, okay, violet color is produced due to the formation of this complex. Just see the complex. Okay, and it requires at least 2 peptide bonds to give a positive test. Then we have nin hydrogen test, which is a general test for all alpha amino acids. Okay, we take amino acid and 2 mole of nin hydrogen solution. Okay, and the aldehyde CO2 and the color of Ruhmann's purple is present. Okay, and there is some like exceptions like proline and hydroxyproline, this produce yellow color, okay, in hydrogen test. And asparagine and glutamine, this produce brown color in your hydrogen test. Then you have xanthoprotic test, which is answered by your aromatic amino acids like tyrosine, phenylalanine and tryptophan, and reagent usage is a concentrated HNO3. The Millan's test, your this is for your tyrosine. Millan's test is for your tyrosine. Then the Rosenheim's test is for your tryptophan, okay, tryptophan. And Hopkins-Cole test is for your your glyoxylic acid is used, okay, and it is also answered by your tryptophan. Okay, so for tryptophan we have the Rosenheim's and we have the Hopkins-Cole's test also. And Sakaguchi test is for your arginine. So these are your color reactions of proteins. Okay, and now let's jump to our protein metabolism. Okay, so talking about your protein metabolism, first you have a urea cycle or your ornithine cycle or your Krebs-Henselt cycle. Okay, so just see the urea cycle and whichever point I am pointing. Okay, so first what happens, your CO2 and NH3 combines in the presence of enzyme, which is your carbamyl phosphate synthetase, CPS1. Okay, carbamyl phosphate synthetase 1. Okay, and forms your carbamyl phosphate with the utilization of 2 ATP, which is converted to 2 ADP plus PI. Okay, then this carbamyl phosphate in the presence of your ornithine trans carbamylase synthase. Okay, this is carbamyl phosphate converted to citrulline. The citrulline in the presence of your arginosuccinate lyase converts into arginosuccinate. This arginosuccinate in the presence of your citrulline in the presence of arginosuccinate synthetase, not the lyase, synthesis of arginosuccinate occurs. This arginosuccinate in the presence of your arginosuccinate lyase, now arginosuccinate will break down, okay, and release your arginine, and fumarate is also released. So you must have remembered that pathway in the Krebs cycle. I have written that the fumarate is also produced by this, okay, and this fumarate goes on to produce your malate. This fumarate produces your malate afterwards, okay. And this arginine then goes, and arginase is acted upon by the arginine. Okay, this arginine then converts into your ornithine. Okay, in this step your S2 is utilized and your urea is given out. Okay, so ornithine is converted to ornithine and your urea and this ornithine is again acted upon by ornithine transcarbamylase to give you citrulline. Again. Okay, so this was your Krebs cycle, and let's talk about the disorders of your Krebs cycle. So first, talking about regulation, your N-acetyl glutamate, this is a allosteric activator of CPS1, just a regulation part. Okay, and talking about your steps, okay, energetics. Okay, talking about energetics, so in step 1 2 ATP are used. In step 1 this 2 ATP are used, okay, and in step 3 also 2 ATP are used, okay, but in step 4 just see fumarate to malate, and this malate will go into electron transport chain, okay, ETS, and produce 2.5 ATP. Okay, this fumarate to malate conversion occurs in the, where NAD plus change into NADH plus H plus. This NADH, okay, when it goes into an electron transport chain will produce 2.5 ATP. Okay, so there will be production of a 2.5 ATP. Okay, so 4 ATP are used and 2.5 ATP is produced. Okay, so there is net consumption of 1.5 ATP. This is your energetics. Okay, talking about the disorders of your urea cycle. So if there is CPS1 defect, then you will suffer from hyperammonemia type 1, where there will be increase in your ammonia levels. If there is the decrease in your OTS, if deficiency of OTS will occur, then you will suffer from your hyperammonemia type 2. Okay, hyperammonemia type 2, where there is NH3 accumulation also. Okay, and if there is deficiency of this arginosuccinate synthetase, will suffer from citrullemia. Okay, citrullemia, there will be accumulation of citrulline, and arginosuccinate lyase deficiency will lead to your arginosuccinic aciduria. Okay, where arginosuccinate levels will increase. So arginosuccinic aciduria, and then your arginase deficiency will lead to arginemia, where there will be increased arginine levels. Okay. And another genetic disorder is that if there is mutation of ORNT gene, then there is decrease in the ornithine translocase enzyme activity, okay, and this will be deficient, and this will lead to HHS syndrome, in which there will be increase in arginine, ornithine, and citrulline. These three, or ornithine, arginine, citrulline, these three are increased. Okay, so this completes the urea cycle. Then let's talk about phenylalanine and tryptophan metabolism. Not the tryptophan, tyrosine metabolism. We'll talk about the tryptophan metabolism after this. Okay, Okay, talking about phenylalanine and tyrosine. Okay, so phenylalanine in the presence of an enzyme, phenylalanine hydroxylase, it's an infamous enzyme. Okay, this will lead to the conversion of a tyrosine. Phenylalanine converted into tyrosine in the presence of a phenylalanine hydroxylase, where in this reaction O2 is consumed, H2O is given out. Okay, and the tetrahydrobiopterin, the BH4 tetrahydrobiopterin is converted to a dihydrobiopterin. Okay, and after this step, tyrosine is generated. Tyrosine now goes into the pathway by transaminase enzyme. Tyrosine in the presence of a transaminase enzyme is converted into a para-hydroxyphenylpyruvate, and in this step alpha ketoglutarate is changed into glutamate. So it is mainly a deamination reaction occurring here. So tyrosine in the presence of transaminase converts into a para-hydroxyphenylpyruvate. This para-hydroxyphenylpyruvate again in the presence of a hydrolase, gets converted into a homogentisate. In this step, ascorbate is vitamin C is changed to a dehydroascorbate, CO2 and H2O. Okay, and the para-hydroxyphenylpyruvate changed into a homogentisate. Then this homogentisate is acted upon by homogentisic acid oxidase, which is also a famous enzyme. Okay, where oxygen is added. Oxidase enzyme. Okay, homogentisate is changed into 4-mallyl acetoacetate. Okay, then this 4-mallyl acetoacetate is acted upon by an isomerase enzyme, that is a mallyl acetoacetate isomerase. Okay, where it gets converted into a 4-fumaryl acetoacetate, and this in the presence of a hydroxylase enzyme gets converted into fumarate and acetoacetate. It splits into fumarate and acetoacetate. And fumarate will give you your glucose, acetoacetate will give you your fat. Okay, understood? Okay, another pathways are there, some supplementary pathways due to which the tyrosine will be able to synthesize your melanin and your epinephrine and your norepinephrine. Okay, so this tyrosine will synthesize many things. Okay, will synthesize many things. You can remember the mnemonic MEDNET. So med is your melanin. Okay, D is your dopamine, N is your norepinephrine, E is your epinephrine, and it will also synthesize the thyroid hormones T3 and T4. Okay, tyrosine is the precursor of the thyroid hormones T3 and T4. Understood? So afterwards, tyrosine is acted upon by tyrosinase. And this tyrosinase acts upon tyrosine to convert it into DOPA. Okay, DOPA, and this DOPA is again acted upon by tyrosinase to convert it into dopaquinone. This dopaquinone splits into leukodopachrome and your red melanin. And this leukodopachrome, okay, this after subsequent steps becomes your dopachrome. Okay, and this dopachrome will lead to the synthesis of your black melanin. So I have skipped many steps here, okay, which are not important at all for your prof examinations. Okay, you can just write this pathway for the synthesis of your black melanin and your red melanin. And if the tyrosine is acted upon by your tyrosine decarboxylase enzyme, then it is converted into a DOPA. Okay, the same thing. Your tyrosinase and your tyrosine decarboxylase convert the tyrosine into a DOPA. Okay, then it is acted upon by dopa decarboxylase, okay, which will cause the formation of a dopamine, and dopamine in the presence of hydroxylation of dopamine in the presence of vitamin C will convert into a norepinephrine, and this norepinephrine, that is SAM, your methyl transferase. Okay, your methyl group will be transferred, okay, which will cause the formation of epinephrine. Okay, so you made dopamine also, you made epinephrine, norepinephrine, and you made epinephrine also, you made melanin also. Okay, so you synthesized all of this. Okay, this is the pathway. Talking about some of the disorders. So talking about disorders, if the phenylalanine hydroxylase is deficient, you will suffer from phenylketonuria, very famous disorder. Phenylketonuria is an autosomal recessive disorder. It may have type 1, type 2, type 3, type 4 and type 5, but type 1 is called as a classical phenylketonuria, where there is deficiency of phenylalanine hydroxylase. Okay, type 2 and 3 are due to a BH2 reductase, and type 4 and 5 are due to a BH2 biosynthesis. This is not much important, just remember the type 1. Okay, here type 1, the phenylalanine accumulates, okay, which will convert into a phenylpyruvate. Okay, this phenylpyruvate is due to conversion of excess phenylalanine. Okay, and this phenylpyruvate will split into phenyl lactate and phenyl acetate, and this phenyl acetate will give you a mousy order of urine. So there is mousy order of urine in phenylketonuria. Okay, and also due to phenylalanine accumulation, tyrosine won't be synthesized. Okay, because the phenylalanine hydroxylase was converting phenylalanine into tyrosine. Okay, so phenylalanine is accumulating in excess and tyrosine is deficient. Due to deficiency of tyrosine, tyrosine synthesizes many things. Okay, like your epinephrine, norepinephrine and all. Okay, so there will be no production of melanin also. Okay, there will be hyperpigmentation, and many other things are affected, like your norepinephrine, your epinephrine, these are also not produced. Okay, So there will be CNS problems, your mental retardation will occur. Okay, hypopigmentation due to lack of synthesis of melanin. Okay, seizures and tremors. These all are CNS problems. These are due to the deficiency of epinephrine, norepinephrine, and your dopamine. Okay, epinephrine, norepinephrine, and your dopamine. Okay, due to which it is seen in your phenylketonuria. Okay. Understood. And what about the test for identifying a phenylketonuria? So you have the screening test like your Guthrie's test, your bacterial assay inhibition test. Then you have confirmatory test, that is your tandem mass spectrometry. Your prenatal test you can do by your DNA probes. Okay, and other like your FeCl3 test for your phenylketonuria. And how can you treat it? Phenylalanine diet must not be given, like your meat and poultry must be avoided. Okay, and your tyrosine supplements can be given to compensate for the tyrosine deficiency, and behavioral therapy for your neurological manifestations. Okay. After this, we have the disorder which is transaminase deficiency, which will cause tyrosinemia type 2. Okay, and the hydroxylase deficiency, which will cause a neonatal tyrosinemia. Okay, and then homogentic acid oxidase deficiency, which will lead to alkaptonuria. So you have to discuss to alkaptonuria also. Alkaptonuria is an autosomal recessive disorder. PKU is also autosomal recessive. Okay, cases are 1 in 20,000 where tyrosinase enzyme is deficient, which will lead to a melanin secretion decrease. Okay, and hypopigmentation. Okay, so there will be photosensitivity. Okay, photophobic, the person will be photophobic. Skin cancer incidences will be there, and hypopigmentation will be there in your sorry, what I am discussing in your alkaptonuria. I was talking about albinism. So okay, look at the albinism also. Okay, if the this tyrosinase deficiency is there, then there will be albinism and vitiligo. Vitiligo is the white patches over the skin. Okay, and this tyrosinase deficiency will lead to your albinism. So I was talking about albinism. Albinism is autosomal recessive, 1 in 20,000 cases where the tyrosinase enzyme is deficient. There will be hypopigmentation and photosensitivity along with skin cancers. Okay, and talking about your another disorder which we had to discuss, alkaptonuria. It is called as a black urine disease. Which will lead to arthritis in adults. Arthritis in adults are caused due to alkaptonuria. Okay, which is asymptomatic in childhood. Okay, but it will lead to arthritis in adults. Why arthritis is caused? Because there is decrease in the homogentic acid oxidase, due to which this homogenticity accumulates. Okay, and in the presence of polyphenol oxidase enzyme, it is converted into a benzoquinone acetate. Okay, this benzoquinone acetate polymerizes and forms alkapton. Okay, this alkapton in bone, cartilage, tissues, and tendons, it accumulates. Okay, causing ochronosis. Okay, ochronosis, like your pannus formation is there. That is your formation of plaques. Okay, like which will cause a painful arthritis in adults. Okay, and this is asymptomatic in childhood, and the urine will be dark on standing. So it is also called as a black diaper syndrome. Okay, there was a blue diaper syndrome, remember? Okay, where you have read it, just comment in the comment box. Okay, and urine will be dark on standing. Okay, so there will be black stains on the diaper. Okay, and you can know the neonate is suffering from your alkaptonuria. Okay, and there will be discoloration of the skin, sclera, and the cornea. Test is your FeCl3 test and your silver nitrate test, which will be positive. FeCl3 and silver nitrate, which will be positive in your alkaptonuria. Okay, and we had to discuss tyrosinemia type 2 also, which was due to deficiency of a tyrosine transaminase. Okay, which is also called as a Rickner-Hanhart syndrome. Just remember this point, Rickner-Hanhart syndrome. And here we have DEM. Okay, DEM syndromes. You have dermatitis, eye lesions, and mental retardation. Okay, and then your tyrosinemia type 2 is there. Okay, which is also called as the tyrosinosis. Okay, where there is the deficiency. Tyrosinemia type 1 we haven't seen the pathway yet. So here, if you have deficiency of mallyl acetoacetate isomerase and your hydroxylase, acetoacetate isomerase and your hydroxylase, you will suffer from tyrosinemia type 1. So this is the tyrosinemia type 1. It is called as a tyrosinosis. That is decreased mallyl acetoacetate isomerase and your hydroxylase. Okay, it will lead to your diarrhea, vomiting, and cabbage odor from your skin. Okay, yeah. So I was telling that cabbage odor from skin is noted in your tyrosinemia type 1. Okay, talking about another disorder. Okay, I hope I have covered all the disorders from now. Okay, another miscellaneous disorder, that is your Parkinson's disorder. If there is decreased dopamine in your brain, okay, there is the loss of neurons from your basal ganglia, from your nigrostriatal pathway. There is the loss of your dopaminergic neurons, as you have read in your physiology. Okay, there is lethargy, tumors, muscle rigidity, mask-like face, and involuntary movements. Okay, and you can treat this by L-dopa and carbidopa. Like in Parkinson's disease, you have many cardinal features like your cogwheel rigidity is there, your lead pipe rigidity is there. Okay, and the mask-like face, your statue-like stature. Okay, mask-like face, statue-like stature is there. Tremors, you will show pill rolling tremors or your drum beating tremors. Okay, and treatment is by L-dopa or your carbidopa, okay, which are dopamine analogs. Let's talk about the tryptophan metabolism. Tryptophan metabolism. So, two pathways. One is a kynureninase pathway. One is a serotonin pathway. And other one is a kynureninase pathway. So here you take tryptophan, okay, then put tryptophan pyrrolase. Take tryptophan, put tryptophan pyrrolase, okay, add O2, okay, O2 is added, and N-formyl kynurenin is formed. This N-formyl kynurenin in the presence of formamidase, the formate is removed. Okay, from this formate is removed, and then you form again your kynurenin. Okay, this kynurenin is formed. This kynurenin is hydroxylated by kynurenin hydroxylase, which will convert it into 3-hydroxykynurenin. Kynurenin is converted into 3-hydroxykynurenin by kynurenin hydroxylase enzyme. Okay, and this 3-hydroxykynurenin, okay, this is again acted upon by kynureninase, which is converted into your 3-hydroxyanthanylate. This 3-hydroxyanthanylate again, in the presence of oxidase, it is oxidized to 2-amino-3-carboxymuconate semi-aldehyde, very long name, just remember, 2-amino-3-carboxymuconate semi-aldehyde. Okay, this compound in the presence of picolinate carboxylase, it is converted into picolinate. Okay, or if it is like stagnantly just left undisturbed, then it changes into quinolate, this quinolate into NMN. Okay, and this NMN is the source of your NAD plus and NADP plus. Okay, so NAD plus and NADP plus, these are your the precursor is your tryptophan. Okay, and this 2-amino-3-carboxymuconate semi-aldehyde also in the presence of a decarboxylase enzyme changes to 2-amino-muconate semi-aldehyde, okay, which will give your acetoacetyl, okay, and this acetoacetyl will give your fat. Okay, talking about another pathway, that is a serotonin pathway. Just a clinical here in the previous pathway, okay, that your 3-hydroxykynurenin, okay, in the presence of a B6 deficiency, okay, this is converted into your 3-hydroxykynurenin converted into a xanthurenate despite the conversion by kynureninase is converted into 3-hydroxyanthanylate, okay, which is normal, but here the 3-hydroxykynurenin is deviating due to B6 deficiency and going to form xanthurenate, and this xanthurenate is excreted out in urine. Okay, this is pathological. Okay, talking about serotonin pathway, we have the tryptophan. Okay, tryptophan will synthesize the serotonin, and how we will see. About tryptophan will convert by the enzyme tryptophan hydroxylase into your 5-hydroxytryptophan, obviously, and O2 is converting into H2O, and BH4 is converting into your BH2. This 5-hydroxytryptophan in the presence of your decarboxylase enzyme, where CO2 is removed, decarboxylase enzyme where CO2 is removed. Okay, this is converted into 5-hydroxytryptophan, converts into a serotonin. Okay, serotonin, and the serotonin in the presence of acetylase enzyme forms your N-acetyl serotonin. The serotonin in the presence of your acetylase forms the N-acetyl serotonin, and this N-acetyl serotonin in the presence of your methyl transferase, is converted into your melatonin. So serotonin, serotonin and your melatonin, this both precursor is your tryptophan only. Okay, the tryptophan also forms a serotonin and your melatonin. Okay, understood. Serotonin, what's the importance? It is synthesized in the brain, mast cell and the platelets. Okay, it is helpful in the BP and temperature regulation, and it is an excitatory neurotransmitter in your brain. Okay, and melatonin regulates the sleep-wake cycle. Synthesis is your pineal gland. Okay, and melatonin is helpful. It's a hormone. It is helping in regulating a circadian rhythm in the sleep-wake cycle. Okay. The disorders of tryptophan metabolism, we don't need to go into detail. Okay, disorders are the Hartnupp's disease is there, carcinoid syndrome is there, and the xanthurenate in urine. These three are your disorders of your tryptophan metabolism. Understood. So afterwards we have your glycine metabolism here. This is also important. Okay, here glycine is the simplest amino acid. So glycine is how it is formed. Okay, how is the form? Okay, so just see this pathway. We have to discuss this pathway, glycine metabolism. Okay, so glycine, okay, it is formed from a serine by the action of a serine hydroxymethyl transferase. This forms a glycine. Okay, serine forms a glycine, and then you have threonine. Aldolase acts upon the threonine, converting it to glycine. This is one pathway. Okay, then you have NH3, CO2, and N5, N10 methylene THF. Okay, this is acted upon by glycine synthase enzyme. It will form a glycine. Okay, and then your glyoxylate. This glyoxylate in the presence of your transaminase. This also forms a glycine. Okay, understood. And what are the degradation products of glycine? So talking about the degradation products of glycine, so glycine degrades to again give you serine, okay, and the serine gives a pyruvate, and pyruvate gives a glucose, okay. So glycine also gives a glucose, so glycine is called as a glucogenic amino acid also, okay. So glycine degrades to form serine, serine to pyruvate, pyruvate to glucose, understood. And glycine also degrades to form again this N5-N10 methylene THF, NH3 and CO2, okay. And glycine in the presence of an oxidase enzyme, again converts into a glyoxylate, okay. In the presence of transaminase it was becoming glycine, and in the presence of oxidase again glyoxylate, okay. So glycine in the presence of oxidase converts into a glyoxylate, and this glyoxylate, okay, splits into oxalate and fermate. Oxalate is excreted out in urine, and the fermate is a 1 carbon compound which will like form a 1 carbon metabolism, which will be useful in a 1 carbon metabolism, okay. Glycine also there are some conjugation reactions with glycine, okay. We saw the conjugation with the glycine, okay. Let's discuss them. Okay, so glycine is forming a glutathione, okay, glutathione, which is maintaining the RBC membrane integrity. This is called as gamma glutamyl cysteine glycine, okay. This is the gamma glutamyl cysteine glycine. It's a glutathione, okay. And glycine is conjugating with chenodeoxycholic acid, okay, in forming a glycochenodioxycholate, and glycine is conjugating with cholic acid to form the glycocolate. Glycine is conjugating as a benzoyl CoA, glycine, benzoyl CoA plus glycine is a hippuric acid which is excreted out in urine, okay. Then we have the glycine plus arginine, okay, which forms a guanidinoacetate, and this guanidinoacetate in the presence of SAM, okay, creates your creatine. Guanidinoacetate forms in the presence of SAM, your methyl transferase, okay, forms a creatine, okay. Creatine changes into creatine phosphate, and creatine phosphate changes into creatinine, okay. So if you just add, if you just remove a water molecule from the creatine, you get creatinine, okay, and this creatine is removed in your urine, okay, understood. So this and again glycine plus your succinyl CoA in the presence of ALA synthase, we discussed the heme metabolism, okay. This is helpful in your heme synthesis. And glycinuria, it is the defective renal absorption of glycine, okay. Defective renal absorption of glycine causes a glycinuria, okay. This is whole your glycine metabolism, okay. Then let's talk about the metabolism of sulfur containing amino acids, okay. Sulfur containing amino acids, let's talk about their metabolism. Let's take this highlight, yes. Metabolism sulfur containing amino acids. So, sulfur containing amino acids are cysteine and methionine. Okay, so first there will be transmethylation reaction in which the methionine is acted upon by methionine adenosyl transferase, MAT enzyme, methionine adenosyl transferase, where 1 ATP is utilized completely, okay, and methionine forms at 5 adenosyl methionine. This 5 adenosyl methionine, okay, this is acted upon by methyl transferase, which will remove the CH3 group, okay, and form a 5 adenosyl homocysteine, okay. So this 5 adenosyl methionine is forming a 5 adenosyl homocysteine. This 5 adenosyl homocysteine is now acted upon by homocysteinase, okay, where the adenosyl group is removed and homocysteine is formed. And this homocysteine is again acted upon by a methionine synthase enzyme, okay, where the methyl THF is converted into THF, okay, and B12 is used in this reaction, your homocysteine to methionine, okay. We'll see in the vitamins also B12 use here, the homocysteine to methionine in the presence of B12 and methionine synthase, okay. This is the conversion, okay. So this is a cycle of transmethylation reaction that is occurring, okay. And what are the other other transmethylation reaction, okay, like your guanidinoacetate in the presence of SAM, it is converting to creatine, we saw above. The norepinephrine in the presence of SAM was converting to epinephrine, also we saw above, okay. And this epinephrine in the presence of your CH3 again SAM, this is forming the metanephrine, okay. Then N-acetyl serotonin to melatonin conversion, we also saw that above. Ethanolamine to choline, okay, and carnosine to anserine, this is also a transmethylation reaction, okay. Then what happens after this transmethylation reaction is done, okay, we'll have second step, that is your cysteine synthesis, So what happens in your cysteine synthesis, your homocysteine and your serine combine, in the presence of cystathione beta synthase, very special, and the cystathione beta synthase, okay, and form a cystathione. This cystathione again in the presence of cystathionase splits into cysteine plus homoserine, okay. There was homocysteine and serine, okay, which are now forming cysteine and homoserine. This cysteine, there will be 2 molecular condensation we discussed in protein metabolism to form cysteine, and this homoserine, there will be deamination decarboxylation to form a propionyl CoA, and propionyl CoA goes into gluconeogenesis we discussed in a carbohydrate metabolism to form a glucose again, okay. There is an important clinical here, that cystathione beta synthase, if there is deficiency of cystathione beta synthase, we will suffer from homocysteinuria. The important disorder is homocysteinuria, okay. So there is increased homocysteine in the blood, which will interfere with the collagen formation, and accumulates of LDL will occur, and coronary artery disease will be seen, okay. So in homocysteinuria, you will see your coronary artery disease, okay. Afterwards, the treatment of there are some clinical features of your homocystinuria. So you will have brittle bones, okay, and the skeletal abnormalities, okay, brittle bones, your myopia will be there, your failure to thrive, learning problems, abnormal blood clotting, megaloblastic anemia, and ectopia lentis. Sometimes in clinical case ectopia lentis are given, okay, which are seen in your homocystinuria, and the treatment is a B6 B12 folic acid, okay, and avoiding of methionine in your diet, okay, B6 B12 and your folic acid supplements, okay. And the test for your homocystinuria are your enzyme assay, okay, cyanide nitroprusside test, okay, for your cysteine concentration, cyanide nitroprusside test for your cysteine concentration, which will help to diagnose your homocystinuria, okay. Understood? So these are about your homocystinuria and the cysteine metabolism, okay. And let's see about the degradation of cysteine now. Degradation of cysteine, how cysteine is degraded, okay. So cysteine converts into your pyruvate, okay, and pyruvate gives you your glucose back. Pyruvate gives you your glucose, okay. So cysteine degrades into pyruvate and pyruvate to glucose, okay. And cysteine also forms mercaptoethanolamine. When it degrades, it forms mercaptoethanolamine, okay, which in presence of vitamin B5, your pantothenic acid, okay, converts into your CoA, acetyl CoA, that coenzyme A, okay. Then cysteine also degrades into citric acid and sulfides, okay, sulfides and citric acid. This citric acid will form your taurine, okay, and this sulfides will form your PAPs, okay. PAPs is called as an active sulfur donor, okay, which will donate active sulfur. And so this is taurine and your PAPs is taurine is a degradation product of your cysteine only, and PAPs is also the degradation product of your sulfides or your cysteine, okay. Then cysteine also degrades to form glutathione, okay. Understood? So these are your degradation of your cysteines, okay. Let's talk about your metabolism of a branch chain amino acid, metabolism of a branch chain amino acid, okay. Where we talked about glucogenic and ketogenic amino acids. So leucine is a ketogenic amino acid, valine is a glucogenic amino acid, and your isoleucine is a glucogenic plus your ketogenic amino acid, okay. So how their branch chain amino acids are metabolized. So we have valine, leucine, and isoleucine, which are acted upon by transaminase, okay, transaminase, and which are converted into corresponding alpha keto acids, okay, which are converted into your corresponding alpha keto acid. Okay, the valine, leucine, and isoleucine are converted into a corresponding alpha keto acid by transamination reaction. This corresponding alpha keto acids, these are reacted upon by branch chain alpha keto acid dehydrogenase enzyme. Okay, branch chain alpha keto acid dehydrogenase enzyme, okay, which will convert it into the corresponding acyl CoAs, okay, like isovaleric acyl CoA, okay, and your valeryl acyl CoA, okay, this all. And this corresponding acyl CoAs are then acted upon by acyl CoA dehydrogenases, okay, to form succinyl CoA, to form acetoacetate, okay, and to form succinyl CoA plus acetoacetyl CoA plus acetyl CoA, okay. This can form any of the compounds like a succinyl CoA, or this can form a acetoacetate, or this can form a mixture of a succinyl CoA and acetyl CoA, okay. We have an important clinical here, 3 clinicals specifically, okay. This transaminase deficiency will suffer from hypervalinemia, okay, transaminase, your valine transaminase, if the valine transaminase is deficient, will suffer from hypervalinemia. And if there is branch chain amino acid deficit, okay, this enzyme is deficient, the branch chain alpha keto amino acids dehydrogenase deficiency, or branch chain alpha keto acid dehydrogenase deficiency, a important disorder is there, which is called as maple syrup urine disorder, or MSUD, okay. So MSUD is caused due to your branch chain alpha keto acid dehydrogenase deficiency, but there is accumulation of alpha keto acids in your brain, okay, accumulation of alpha keto acids in the brain, which will lead to neurological manifestations, okay. The urine order, the order of urine will be maple sugar like or your burnt sugar type of urine, okay. You can diagnose this test from a tandem mass spectroscopy, okay. And treatment will be protein restricted diet, you should give protein restricted diet, okay. Then intermittent branch chain ketonuria, okay, this is the partial deficiency of branch chain keto acid dehydrogenase. There's complete deficit, then MSUD will occur. There's intermittent deficiency, then if partial deficiency, then intermittent branch chain ketonuria will occur, okay. And the isovaleric acidemia, this is due to defect in isovaleryl CoA dehydrogenase, understood? So with this we finish our protein metabolism and our protein chemistry, okay. Now let's move on to the next chapter, okay. Let's move on to another important chapter, that is your mineral metabolism, okay. So major elements which are present more than 100 milligram per day, which are called as the major elements, and the trace elements which are present less than 100, which are required less than 100 milligram per day, are called as your trace elements, okay. So you can remember the mnemonic corn pk sodium chloride mange, okay. So here you have calcium, okay, phosphorus, potassium, sodium, chlorine, and magnesium, okay. And the trace elements like your iron, zinc, copper, iodine, selenium, and molybdenum. Talking about calcium is important as a mineral, okay. So calcium is one of the important minerals. So in body, 1 to 1.5 kg of calcium is there. 99% is present in your bones and teeth. 1% is present in your extracellular fluid. Okay, daily requirements you have 800 milligram in your adults, 1500 milligram in your children, okay, and 1500 milligram in the pregnant, lactating and menopausal women. Okay, so in pregnancy, lactation and menopause, more calcium is needed. Okay, we saw this in your diet also today. Okay, and sources are milk, eggs, milk products, cereals, pulses, fish and meat. Milk, eggs, milk products, cereals, pulses, fish and meat. This have calcium. Okay. What are the functions? Okay, you can just remember the mnemonic BCCI. BCCI means have heart. BCCI means have heart. Okay, you can remember. Okay, so B for bones and teeth. These are present in bones and teeth and mineralize. Okay, cause mineralization of bones and teeth by formation of a hydroxyapatite. Okay, C is for muscle contraction, bind to actin and myosin and cause muscle contraction. Another C is for coagulation. Okay, and I is for integrity of cell membrane and maintaining the integrity and permeability of the cell membrane. Okay, M is for the secondary messenger. They act as secondary messenger in the IP3 DAG pathway. Okay, they do enzyme activation. Okay, activation of ATPase, adenyl cyclase, PDH, PK. Okay, pyrokinase, glycine synthase, etc. They cause their activation. Okay, N is for your nerve impulse transmission. Okay, S is for the secretory processes like endocytosis and your exocytosis. Okay, H is for your hormone release like insulin, PTH, vasopressin release. Okay, and H is again for your heart, which prolongs the systole of the heart. Okay, so these are your functions of your calcium, and the normal calcium level is 9 to 11 milligram per deciliter. 9 to 11 milligram per deciliter, where 50% is present in your ionized form, 40% is present in your protein bound form, and 10% in bicarbonate and phosphate form. Okay. How it is regulated? So vitamin D, okay, it increases the absorption of calcium from renal tubules, okay, and increase the bone mineralization. The PTH level, they cause demineralization of bone, and calcitonin causes mineralization of bone. Okay, so absorption of calcium factors which increase the absorption of calcium are your vitamin D, PTH, lysine, arginine, and low pH. But factors that decrease your absorption are your phytates, oxalates, phosphates, free fatty acid, and your higher pH. Okay, these are the factors that decrease the absorption. These are factors that increase the absorption of calcium. Okay, then you have hypocalcemia and hypercalcemia. Hypocalcemia is less than 9 to 11 milligram per deciliter of calcium, and hypercalcemia is more than 9 to 11 milligram per deciliter of calcium. Okay, then hypocalcemia, you will suffer from convulsions, okay, arrhythmia, spasm, tetany, and stridor. Okay, these are the clinical features. And what are the causes of hypocalcemia? Rickets, osteomalacia, osteoporosis, and decreased PTH. These are the causes of your hypocalcemia. Okay, and hypercalcemia, there will be pain in abdomen and bones, nausea, weakness, and confusion. Okay, pain in abdomen and bones, nausea, weakness, and confusion. Okay, and the causes of are your PTH tumor, bone tumor, and metastasis, and Paget's disease, mainly the bone disorders. Okay, this bone is destroyed. Okay, and calcium is released in large quantity, which causes your hypercalcemia. Okay. What is tetany? Tetany is your calcium levels when it is less than 7 milligram per deciliter. Okay, in tetany you will see three main signs. That is your carpopedal spasm, that is a flexion of MCP joint and extension of ICP joint. Okay, and this Bostick's sign. Okay, this Bostick's sign is a facial twitching or tapping of the facial nerve. You tap the facial nerve. Okay, then there will be twitching of the face. Okay, spasmatic twitching of the face. Okay, and your Trousseau sign. Okay, Trousseau sign is your carpopedal spasm will occur. Okay, carpopedal spasm, this one will occur on application of your BP cuff. Okay, on application of your BP cuff. Then let's come to phosphorus. Phosphorus total body content is 1 kg, and calcium was more. Your 1.5 to 2 kg was there, but 80% was in bones and teeth, 10% in the muscles and RBC, and 10% bound to other compounds. What are the sources of your potassium? Milk is one main source. Eggs, milk products, vegetables, cereals, the sources of your potassium. Function, what is the function of your potassium? So potassium function is synthesis of your nucleotides. Okay, nucleotide synthesis. Then component of nucleic acids, mainly DNA and RNA. In mainly DNA and RNA, potassium is present. Okay, then sorry, not the potassium, phosphorus. Okay, phosphorus is present. Okay, and this phosphorus causes activation of various enzymes by phosphorylation. You know the enzymes activated by phosphorylation. You know the enzymes that are activated by phosphorylation. Like I said, your mnemonic, that is a GC Panda hero. Okay, so you can remember the activation of which enzymes are activated by phosphorylation. And formation of high energy compounds like ATP, GTP, CTP, and creatine phosphate. These are your high energy compounds. Okay, which are formed by a phosphorus. Okay, the formation of bone and teeth, formation of phospholipids and phosphoproteins. Okay, it also acts as intracellular buffer. We first saw in acid-base balance today only, and in your acid-base balances. Serum phosphorus level is about 3 to 4 milligram per deciliter. Serum calcium level was 9 to 11 milligram per deciliter, but serum phosphorus level is your 3 to 4 milligram per deciliter. In blood, phosphorus is present in 30 to 40 milligram per deciliter. Okay, just 10 times more in blood. Disorders are hyperphosphatemia, which is caused in hypoparathyroidism. Parathyroid gland is not secreting its hormone, that is parathormone. Okay, so there is hyperphosphatemia. And chemotherapy and vitamin D, increased vitamin D will cause your hyperphosphatemia. Hypophosphatemia is caused due to malabsorption of phosphorus, malnutrition, and a vitamin D deficit. Okay. Talking about magnesium. So RDA in males it is about 350 milligram per day, in females 300 milligram per day. Sources are your vegetables, milk, meat, and cereals. Okay, what are the functions of your magnesium? It forms your bones and teeth. Okay, and activation of enzymes like glucokinase, hexokinase, pyruvate kinase, adenyl cyclase, these are activated in the presence of your magnesium. Okay, and it also it has also neuromuscular functions. And magnesium deficiency will lead to neuromuscular irritability, convulsions, and spasms. Okay. Talking about sulfur is a component of your cysteine, and the methionine, sulfur containing amino acids. Okay, which is also important for the protein synthesis, SH bond orientation. Okay, synthesis of GTPs, not GTPs. Okay, these are not your GTPs. Okay, these are your glutathione, your taurocolate. Okay, and P for PAPs. Okay, and S for your dermatan sulfate, keratan sulfate, chondroitin sulfate, and heparin, your mucopolysaccharides. Okay, this helps in synthesis of glutathione, taurocolate, PAPs, and your this sulfates and heparin. Okay. It is important for your detoxification and important in transmethylation reaction, or SAM. SAM is the active sulfur donor. Okay, PAPs is the active sulfur donor, and it also uses SAM. Just a minute. Okay, so there was a phone call. Okay, so it is a component of a cysteine, and methionine. Okay, important for protein synthesis. Your GTPs are your glutathione, your taurocolate, your PAPs, and all this sulfate, dermatan sulfate, keratan sulfate, chondroitin sulfate, and your heparin. Okay, it is important for detoxification and transmethylation reactions. Okay, let's now go to your sodium. Sodium is also important. Your calcium is important. Your sodium is now and the next important thing. Okay, the RDA required daily allowance is the 5 gram per day. So you require about 5 gram per day of sodium. Okay, sources are your cereals, table salt, milk, eggs, and pulses. Okay, cereal, table salt, milk, eggs, and pulses. Function is the maintenance of acid-base balance. Nerve impulse transmission is important thing. Maintenance of osmolarity, plasma osmolarity of the body. Like sodium potassium pump, sodium potassium ATPase, and absorption of your glucose, galactose, and amino acids requires your sodium transporter. Okay, so just remember we talked about the your sodium and glucose, symporter, and your sodium amino acid symporter and all. Okay, I talked about in cell, remember? Yes. So regulation of sodium levels is via aldosterone and via ANF. Okay, let's see how the regulation occurs. Okay, if there is decreased blood volume and decreased BP, so it stimulates your JG apparatus, juxtaglomerular apparatus. This will secrete your renin. Renin will convert your angiotensin 1 to angiotensin 2. Angiotensin 2 will stimulate your adrenal cortex. Adrenal cortex will cause the release of aldosterone, and this aldosterone will cause sodium reabsorption from a distal convoluted tubule. Okay, and the sodium reabsorption will restore your blood volume and BP. Okay, and the ANF, let's see what is the ANF effect. So if there is increased blood volume or increased BP, so there will be stretching of the right atrium of the heart. Okay, which will release your ANF from the wall of the right atrium, and this ANF will decrease the aldosterone and decrease the ADH, antidiuretic hormone. Okay, and increase the sodium excretion. Sodium is excreted out. Due to excretion, the blood volume and BP will be reduced. Okay, this is excessive BP and blood volume. Distribution is 4000 milliequivalent. Okay, sodium is distributed 4000 milliequivalent in the body, 75% in exchangeable form and 25% in tissues. External and the normal plasma sodium concentration is about 135 to 145 milliosmol per kg. This value is important. 135 to 145 milliosmol per kg. There is decrease less than 135 hyponatremia. Is more than 150, you will get hypernatremia. Okay, hyponatremia. Let's talk about plasma osmolarity. Okay. So how will diagnose your hyponatremia cause? If there is first see the plasma osmolarity. Okay, the normal plasma osmolarity is about 280 to 290 milliosmol per kg. Okay, if this is there, then you will see it will say pseudo hyponatremia. Okay, if the plasma osmolarity is if there is hyponatremia and the plasma osmolarity is also decreased. Okay, so check your total body water. If there is excess of total body water, okay, so you can have renal failure, heart failure, liver cirrhosis, nephrotic syndrome. If there is normal body water, you'll see SIADH, hypothyroidism, hypoadrenalism, diuretic uses and all. If there is decreased body water, okay, like in diarrhea, sweating, burns, vomiting, metabolic alkalosis, mineralocorticoid deficiency and all. Okay, and if the plasma osmolarity is increased, this may be due to your glucose accumulation, hyperglycemia, or your mannitol or your uremia, uric acid or mannitol. These are osmotically active, which will drag your water and increase the plasma osmolarity. Okay, understood, and will cause your hyponatremia. Okay, Talking about your hypernatremia, okay, then also you will check the total body water. That is excess of total body water. Okay, this may occur in your hypoaldosteronism. Okay, hypoaldosteronism. Then Cushing syndrome and IV fluid infusion. If there is normal body water, this may be occurred in a diabetes insipidus. So in diabetes insipidus, there is normal total body water, but hypernatremia will be there. Okay, there is less body water, as in a diarrhea, sweating, and decreased water intake. Okay, will suffer from hypernatremia. Okay, so these are your causes of your hyponatremia, and these are your causes of your hypernatremia. Okay. Afterwards, let's discuss about the potassium. Okay, potassium, this is also important. The RDA is 3 to 4 gram per day. RDA is 3 to 4 gram per day. As per phosphorus was also RDA required was 3 to 4 gram per day. Okay, potassium. But sources are bananas are good sources of potassium, oranges, apples, beans, legumes, nuts, tender coconut, etc. This is the function. It is the major intracellular cation. Sodium is the major extracellular cation, and potassium is the major intracellular cation. Regulates the ICF volume. Required for a repolarization, you know. Okay, contraction of heart, nerve transmission, component of a sodium potassium pump also, and secretion of HCl via H plus K plus pump. So potassium is secreted by the H plus K plus pump. Okay, and activates protein kinase also. It also activates your protein kinase. Okay, potassium also activates your protein kinase. Regulation, how is I can regulate aldosterone and corticosteroids. These both hormones are basically increasing the excretion of potassium. Okay, so this aldosterone and corticosteroids, this will reduce your blood potassium, potassium levels in your blood, and this insulin, this will increase the cellular uptake of potassium. So insulin causes hyperkalemia. Okay, insulin causes your hyperkalemia. Okay, this is important. You must have read in physiology also. Normal serum potassium level is 3.5 to 5 millimole per liter. 3.5 to 5 millimole per liter, and the intracellular concentration is about 160 millimole per liter. 160 millimole per liter. Okay, and this normal potassium level 3.5 to 5 millimole per liter. If that is less than 3.5, hypokalemia. More than 3.5 is hyperkalemia. Okay. Let's come to iron. Iron is again another important thing. So important things are your calcium, your sodium, and your iron. Okay, iron is also important. Okay, what are the proteins which contain iron? Your hemoglobin, myoglobin, xanthine oxidase, tryptophan pyrrolase, your cytochrome P450, your catalase. Okay, these all proteins have your iron. Okay, hemoglobin, myoglobin, xanthine oxidase, tryptophan pyrrolase, cytochrome P450, and your catalase. Okay, the functions of iron. Okay, so you can just remember this. Tamil Nadu Medical Education Director announced part completion test. You can remember the functions by this. Okay, T stands for the transport of oxygen and CO2. M is for your myoglobin, component of myoglobin in the muscle. E is for your ETC, it is helpful in the electron transport chain. Okay, D is for your detoxification of cytochrome P450. Okay, A is antioxidant, iron acts as antioxidant. Okay, P is the purine degradation, it is also used in purine degradation, and T is the tyrosine metabolism, it is also used in your tyrosine metabolism. Okay, and required daily allowance is your for males it is 10 mg per day, for females it is 20 mg per day, and the pregnant and lactating women must take 40 mg per day of iron. Okay. So females have more RD of iron due to the menstrual loss of iron. Okay, so they require more. What are the sources of iron? That are green leafy vegetables, pulses, cereals, liver, meat, jaggery, dry fruits. These are your sources of your iron. These are sources of your iron. How is the iron is absorbed and transported and stored? So we have the dietary iron in Fe3 plus form. Okay, these are taken by duodenal cytochrome B, which convert it into Fe2 plus. Okay, Fe2 plus is taken up by the enterocyte using the transporter, that is DMT1. Okay, we have the DMT1 transporter. Okay, which will cause the uptake of your Fe2 plus into the iron, into the enterocyte, liver, your gastric mucosa cells. Okay. And this here it is again it stays in the equilibrium. Fe2 plus and Fe3 plus stay in your equilibrium state. Okay, and then we have the ferroportin will transmit the iron out. Okay, transfer the iron out of the cell, Fe2 plus, and then we have the celluloplasmin which will carry the iron. Okay, and celluloplasmin convert the iron to Fe3 plus form. Okay, Fe3 plus form. So this transport is called as the ferroportin. So we have the DMT1 transporter and a ferroportin transporter which are transporting the iron. Okay, this hepcidin. Okay, this hepcidin will inhibit your ferroportin. This is important. Inhibit ferroportin and regulate the iron levels. Okay. Okay, we have Fe3 plus apotransferrin. Okay, which forms a transferrin. Transferrin is a transport form of iron. Okay, transferrin partly of it is utilized, and other part is your stored in your liver, which is called as a hemosiderin. Okay, so hemosiderin is a storage form of iron. Transport form is a transferrin, and storage form is a hemosiderin. Okay, and the iron is utilized and reutilized again and again. Okay, less than 1 milligram per day is excreted in sweat and bile. Okay, regulation of iron level is at mucosal level, which is called as the mucosal block theory. Okay, mucosal block theory. So regulation of iron level at mucosal level is called as mucosal block theory. What are the clinical things? The iron deficiency anemia, of course. What are the causes? If there is increased utilization of iron or decreased intake of iron or blood loss or chronic diseases or miscarriages, okay, your miscarriages that which occurs due to very like blood loss is increased in the miscarriages, okay. So what are the symptoms? You will see microcytic hypochromic anemia, where the RBCs will be like small sized, and hypochromic anemia. And sluggish or dull child, that child will be dull due to decreased ATP production and all. Fatigue and weakness will set in, okay. And lab diagnosis, you will see decreased hemoglobin in blood, okay. Increased TIBC, okay, increased TIBC, decreased serum iron, decreased serum ferritin, okay. And MCV and MCH, the mean corpuscular volume and mean corpuscular hemoglobin will decrease, okay. Just your TIBC is increased, okay. And what is the treatment? Your oral iron therapy, that is 100 to 200 milligram per day of iron you can give as a therapeutic measure, okay. And increase iron consumption, you can advise the patient to take these substances like green leafy vegetable, pulses, cereals, meat and jaggery and all, okay. And IV iron injection is also given. Then we'll talk about hemosiderosis. This is the increased absorption of iron, due to which there is the accumulation of hemosiderin, okay. And this is mainly found in the Bantu tribe, okay. This is a localized to a Bantu tribe, okay. It is also called as a Bantu siderosis, okay. And hemochromatosis is the excess iron deposition in your pancreas, in your skin and in the liver, okay. If there is increased iron deposition in pancreas, it will impair your glucose tolerance. In the skin, it will cause yellow brown discoloration, the bronze pigmentation of your skin, okay. And in the liver, it will lead to liver cirrhosis. These three things, impaired glucose tolerance, yellow brown discoloration and liver cirrhosis, if they are combined, it is called as a bronze diabetes. So the examiner may ask what is bronze diabetes, so you can write this, okay. Let's talk about zinc. Zinc is also a bit important, okay. Zinc is also a bit important, not much important of course, okay. So zinc is also a trace element, not required in much quantities. Amount in body is about 2 gram. Okay, 2 gram, has 60% is in skeletal muscles, 30% bones. Okay, rich areas of prostate gland and hippocampus of brain, which are zinc rich. Okay, sources are milk, meat, eggs, shellfish, nuts and all. Okay, RDA adults 10 milligram per day, pregnancy and lactation 10 to 20 milligram per day. The biochemical functions, it is a component of 300 enzymes, more than 300 enzymes contain your zinc, okay, like your carbonic anhydrase, carboxypeptidase, superoxide dismutase, alcohol dehydrogenase, ALP, RN polymerase, okay. So we have read this in enzymes chapter also today, and acts as anticoagulant, not anticoagulant, antioxidant, like a superoxide dismutase, okay, scavenges the free radical, okay. This is also helping in wound healing, okay, storage and release of insulin is done by zinc. Vitamin A level maintenance, okay, we will see, and reproduction, it has a role in reproduction too, okay. So what will be the deficiency manifestation? So you will see acrodermatitis enteropathica, okay, which is the dermatitis and inflammation of finger, toes and mouth. Then you have alopecia, pattern baldness. This is not blindness, this is a pattern baldness. Pattern baldness, okay. Pattern baldness is also caused due to your zinc deficiency, okay. Okay. Then poor wound healing, impaired spermatogenesis and neurological symptoms. These all are occurring due to deficiency manifestations of zinc. Let's talk about copper. Copper is another important thing. So calcium, sodium, copper and iron, these are important. Copper is also a trace element. Amount in body is about 100 milligram, okay. And sources of meat, veggies, fruits and nuts, okay. RDA adults will require 2 to 3 milligram per day only. Infants will require 0.5 to 2 milligram per day of copper, okay. It has many functions, like it acts as a cofactor for enzymes like a cytochrome oxidase, your catalase, your lysyl oxidase, celloplasmin, superoxide dismutase, allosynthetase. We have discussed all this, so you can, it is now easy for you to know. So I discussed your mineral metabolism towards the end, okay. And it is utilized in the ETS, okay. Cytochrome oxidase complex is there in the ETS, okay. It is also used in incorporation of Fe in ferritin, okay. Incorporation of Fe in your ferritin, so we require copper for this, okay. And the heme synthesis by allosynthetase will require your copper, okay. And lysyl oxidase, which is utilization of formation of your cross links in collagen, formation of vessel walls, this also requires your copper. And antioxidant, superoxide dismutase, okay. Superoxide dismutase, it also contains copper. What are the diseases? Wilson's disease, it is a favorite question of the examiner, okay, in Viva. Wilson's disease, it is a hepatolenticular degeneration, where there is mutation in copper carrying ATPase. So ATPase 7B is mutated, due to which there is the copper can't come from liver to bile. ATPase 7B has the function of moving the copper to the bile, okay. So in Wilson's disease, copper can't come into the bile, and the liver, it accumulates, there is the liver accumulation of copper, okay, which will damage the lenticular nucleus of brain too, which will cause hepatic damage of course, and also damage the lenticular nucleus of brain too. You must have read in neurophysiology, okay. In your lenticular nucleus of brain also you have read, about the deposition of copper, which will cause your Wilson's disease. Okay, for the clinical features, there will be seen, you will see your Kayser-Fleischer ring, a special thing, your Kayser-Fleischer ring. This Kayser-Fleischer ring is a golden ring, okay, due to the accumulation of copper in the membrane of cornea. Okay, in the membrane of cornea, you see a golden ring, which is called as a Kayser-Fleischer ring. Okay, so if you want to see here, just see there is a golden ring. You can see in the periphery of your cornea, membrane of cornea, there is a golden ring, which is called as a Kayser-Fleischer ring, which is seen in your Wilson's disease. Okay. And this also other things like the jaundice, okay, because there is liver damage, okay, fatigue, stiffness and tremors, as lenticular nucleus of brain is also damaged. Okay, then the Menkes disease or your Menkes kinky hair syndrome, this is a X-linked recessive disorder. Okay, this is the mutation of ATPase 7A, which is the function of transporting your copper from intestine to your liver. Okay, this is mutated due to which there is intestinal accumulation of your copper. Okay, mutation, intestinal accumulation of your copper. Okay, and treatment, you can treat this by penicillamine, which will causes chelation of copper. Okay, penicillamine can be used in the treatment of your Menkes disease. Okay, talking about iodine, iodine is also important because it is related with the thyroid hormones and all. Iodine is also important. Okay, so iodine, the RDA is about 100 to 150 mg per day. This is a microgram. Okay, in adults. Okay, in pregnancy you require 200 microgram your iodine per day in pregnancy. Okay, what are the sources of iodine? Drinking water, seafood, table salt, fresh fruits and vegetables. Okay, these all are your sources of iodine. Okay, and the iodine content of crops in hilly areas is less. Okay, so there is the more chances of your goiter occurring in hilly areas. Okay, the functions of iodine is the production of T3 and T4. Okay, T3 and T4 will increase in metabolic rate. Okay, increase the protein synthesis, increase lipolysis, maintain water and electrolyte balance, and increase the glucose level. Okay, so in iodine produces a T3 and T4, which has a variety of functions. Okay, deficiency is the iron deficiency goiter, called as a simple endemic goiter, and cretinism. Okay, talking about iron deficiency goiter, what are the clinical manifestations? You can get a clinical case on this iron deficiency goiter. So there is swelling in the front of the neck, the thyroid gland swells up. Okay, so there is increase in number of cells of thyroid cells. There is weight gain due to I2 deficiency, or T3 T4 decreases. Okay, T3 T4 has the function of lipolysis, so lipolysis won't occur, due to which there is increased fat accumulation. Okay, the person will feel tired, sluggish and dizzy. Okay, because T3 and T4 regulate the BMR of the body, so the person will feel tired, sluggish and dizzy. Okay, flaky dry skin and hair loss will occur. Flaky dry skin, hair loss, heavy periods. Okay, and increased cold sensitivity, because I2 has the function of generating a body heat. Okay, due to loss there is increased cold sensitivity. Okay, the person will feel cold. Then cretinism is the congenital iron deficiency syndrome. You can remember the mnemonic SME. Okay, SME for this. Okay, subject matter expert. Okay, S is for short stature, and M is for mental retardation, and E is for early puberty, which are three of the cardinal features of your cretinism. Okay, understood. After your iodine, let's discuss your fluoride. Okay, fluoride, RDA is about less than 2 ppm per day. We don't require much fluoride. Less than 2 ppm per day is sufficient. Sources are your drinking water. Drinking water contains as much as fluoride you need. Okay, found to the functions, your formation of water resistance, fluoroapatite. Okay, your enamel, if your hydroxyapatite layer of enamel is there, okay, but we require water resistance in our enamel, so there is formation of fluoroapatite, and this fluoroapatite is formed by your fluorine and fluoride. Okay, it protects the enamel from bacterial acids and important for bones and teeth. Okay, this fluoride ions inhibit enolase enzyme, preventing glycolysis. We have read about 3 times today. Okay, because this point is there in many chapters. Okay, like glycolysis, and again in your enzymes and all. Then grade of IL, is glucose estimation. NAF is K plus oxalate, and there is a 1:3 to prevent the consumption of glucose. This fluoroacetate, this inhibits your aconitase enzyme. This we saw in the TCA cycle. Okay. Disorders, with fluorine deficiency and the fluorine toxicity. So fluorine deficiency less than 0.5 ppm in pregnancy and early childhood. Okay, this will cause your dental caries. Okay, there is decreased fluorine, it will cause your dental caries. Less than 0.5 ppm will cause dental caries. And with toxicity, like if there is more than 2 ppm, okay, so we have mild gastrointestinal symptoms. We have more than 5 ppm, we have dental fluorosis, like the teeth will start yellowing, the mottling and discoloration of teeth. We have more than 20 ppm is a very high amount. Okay, then we have skeletal fluorosis. It will affect the bones directly and cause a genovalgum. Genovalgum, the bowing of the legs, knock-knee disease. The legs will be like this. Okay, the legs will be like bowed down. Okay, so genovalgum or your knock-knee disease will be caused due to fluorine toxicity more than 20 ppm. Okay, so here we finish our mineral metabolism. So here mineral metabolism was a theoretical chapter. Basically, you have to memorize your facts, RDAs and all. Okay, just make a chart. I will say just make a chart. Okay, just write the RDS, and you have to just tick the chat when you read. Okay, by that you can just remember your mineral metabolism. Okay, it is easy chapter. You can write the functions. If you have read the whole biochemistry, the functions are easy for you. Okay, so afterwards let's move on to the next chapter. Okay, so look which other chapters we have completed till now. Okay, so cell is complete, your enzymes are completed, carbohydrate chemistry and metabolism is now complete, your amino acid chemistry and metabolism is now complete, your heme metabolism we have discussed. We have discussed your minerals. Okay, we have discussed your minerals, and this small chapters are there, some small chapters are there, not much are there. Okay, and have we discussed anything else? Yes, we have discussed our chromatography. Okay, and now let's move on to the next chapter. Let's discuss the lipid chemistry and the lipid metabolism. Okay, so after discussing the mineral metabolism, we will come to lipid metabolism. So lipids are heterogeneous group of organic compounds, insoluble in water and soluble in a polar solvent like benzene, ether, CCl4 and hot alcohols. Essential fatty acids are called as PUFAs, fat soluble vitamins, A, D, E, K vitamins, and lipophilic micronutrients are there which are formed by the lipids. Okay, impact of omega-3 fatty acid is that they are more preferred and they are used as dietary supplements for chronic diseases, CVH diseases, rheumatoid arthritis and dementia. Okay, and we have some non-polar lipids, okay, which act as electrical insulators and help in the action potential traveling. Okay, non-polar lipids, we'll see the action potential travels in the myelin sheath. Okay, the sphingomyelin, we'll see, and these are stored in the adipose tissue. We have the Blue's classification of lipids. So Blue was a scientist who classified the lipids into simple lipids, your complex lipids, your derived lipids, and miscellaneous lipids. Simple lipids are a combination of fatty acid and alcohol. This comprise of fats, oils, and waxes. Okay, complex lipids have fatty acid, alcohol plus a special group. Okay, this phosphate group, phospholipids, and sulfate group, sulfolipids. Okay, amine group, aminolipids. Okay, carbohydrate present, your glycolipids. Okay, then we have the sphingolipids, we have the sphingophospholipid. Okay, and you have the lipoproteins also. Complex lipids also have comprise lipoproteins. We have the HDL, LDL, VLDL, and chylomicron. We will also see their metabolisms also. Okay, and derived lipids, these are simple lipids plus your complex lipids. So derived lipids like your fatty acid, like your glycerol, like fatty aldehydes and ketone bodies. These are your derived lipids. And miscellaneous lipids like your steroid hormones are there, fat soluble vitamins are there, and carotenoids are there. Okay. So alcohol may be of two types. Your simple lipids, okay, fatty acid and alcohol. So one component is your alcohol. Alcohol may be two types, your glycerol, okay, or a sphingosine. Okay, and the sphingosine are generally amino alcohols which are derived from a serine. Okay, serines. So this is a structure of a sphingosine. Just you don't need to mug up the structure, just see it. Okay, sphingosine. What are the uses of your fatty acids or lipids? Functions. So fatty acid acts as lung surfactants. The surfactant lung, that is lecithin, we'll discuss about it. Okay, this maintain the homeostasis of the body, regulatory and coenzyme functions they have. Okay, the hormones, vitamins and intracellular messengers, they act as hormones, vitamins and intracellular messengers. They act as fuel molecules and are stored as triacyl glycerols. They do protein modification. They act as thermal barriers and cushioning between your organs. Okay, they're the building blocks of your phospholipids, glycolipids and your cholesterol. Okay, these are your constituents of your cell membrane. Okay, these act as a hydrophobic barrier, okay, between the aqueous part of the cell and the organelles. Okay, fatty acid is a storage form of energy. Okay, and they produce electrical insulators like your galactoceramide and your cholesterol. Okay, and the triacid glycerol is same as your triglyceride, just don't be confused in between them. Okay, talking about your fatty acids, what are fatty acids? Fatty acids are your long chain, they comprise of your long hydrocarbon chain in a terminal carboxylate group. Like this, a long chain is there and a terminal carboxylate group is there. Okay, which is the polar end and it is hydrophilic, water loving. And the non-polar end comprise of a fatty acid tail. Okay, hydrocarbon tail. Okay, hydrocarbon tail. So non-polar end, it is hydrophobic. Okay, and classification based on number of carbon atoms. We have short chain fatty acid which are oxidized in mitochondria containing C2 to 4. Okay, medium chain C16 to 14, then a very long chain fatty acid, your long chain fatty acid from C14 to C20 to 22. Okay, and very long chain fatty acid, the VLCFA. Okay, this comprise more than C22. Okay, more than 22 carbon atoms. Okay, in this chain part, more than 22 carbon atoms in this chain part. This short chain is medium chain, this long chain, these are oxidized in a mitochondria, but this very long chain fatty acid is oxidized in a peroxisome. We have read about the syndrome, your Zellweger syndrome, yes, which is the deficient oxygen of your the oxidase and all. Okay, Zellweger syndrome is functional, there is the peroxisomal wrong targeting of your peroxisomal enzymes. We have read in your protein targeting. Okay, due to which your very long chain fatty acid accumulate in the brain and all and cause neurological symptoms. Classification based on number of carbons of saturated and unsaturated. Unsaturated may be three types. Your MUFA having one double bond, your monounsaturated fatty acids having one double bond, polyunsaturated fatty acids having more than one double bond, and your icosanoids. What are icosanoids? These are all unsaturated fatty acids, okay, which are your arachidonic acid derivatives. Okay, arachidonic is a fatty acid, so they're the arachidonic acid derivatives. They comprise of your lipoxygens, your leukotrienes, your prostanoids, and your thromboxanes. Okay, thromboxane is also a lipid derivative. Omega-3 fatty acid is preferred over MUFA. Okay, omega-3 fatty acid are more like good than your MUFA. Okay, and this PUFA and MUFA, these both unsaturated fatty acids are generally cardioprotective. Okay, they are good for your body, but saturated fatty acids, these are bad for your body. We have read also in the nutrition chapter. Okay, so you have to take more and more unsaturated fatty acids rather than your saturated fatty acids. Okay. Based on synthesis, we have your essential fatty acids and your non-essential fatty acids. Okay, essential fatty acids are like your omega-3 fatty acids, like your alpha-linolenic acid. Okay, alpha-linolenic, not linoleic. Okay, don't get confused. Alpha-linolenic acid, your timnodonic acid, and your cervonic acid, DHA. Cervonic acid is also called as DHA, or your docosahexaenoic acid. Okay, which causes the brain development in neonatal period. Okay, so omega-3 are your alpha-linolenic, your timnodonic, and your cervonic acid. Okay. And your omega-6 fatty acids are there, which are also essential fatty acids. Okay, these are your linoleic acid, your gamma linolenic acid. Okay, alpha linolenic is your omega-3, but your gamma linolenic acid is your omega-6 fatty acid, and your arachidonic acid. So omega-6 is your linoleic, your gamma linolenic, and your arachidonic. Okay, understood? Where more essential is your linoleic acid over your linolenic acid. Okay, because linoleic acid can form your linolenic acid later. Okay, understood? After this, let's talk about non-essential fatty acids. So, omega-7 fatty acid and omega-9 fatty acid. That is a palmitoic acid. Omega-7 is a palmitoic acid, and omega-9 is the oleic acid or the elladic acid. Oleic acid or the elladic acid. Okay. The sources of essential fatty acids, linoleic acid. Okay, linoleic acid, where you can get it from your safflower oil. Safflower oil is the richest source of your linoleic acid. This is given in Harper. Okay, then corn oil, then sunflower oil, then soybean oil. Okay, safflower is the richest source. Then from corn oil, from sunflower, then from your soybean. And linolenic acid is obtained from a soybean oil, arachidonic acid from animal sources. Okay, and DHA or your cervonic acid from your breast milk. Okay, so this DHA is necessary for the brain development in neonatal period. So you must have seen your complaint. Okay, the complaint and the drink that the neonates drink or the children drink, it has DHA. Okay. Disorders due to essential fatty acid malformations, or your due to deficiency of your essential fatty acids, or the decreased metabolism. Also, we have acrodermatitis enteropathica, dry scaly dermatitis. Okay, and cystic fibrosis, your Jorgen-Lanson syndrome, your Crohn's disease, and cirrhosis. Acrodermatitis, cystic fibrosis, your Jorgen-Lanson syndrome, your Crohn's disease, and your cirrhosis. Structures. Let's discuss about some structures. So stearic acid has 16 carbons. Just remember this fact. Your oleic acid has a cis part and a trans part. So this is a cis oleic acid. You must have read about cis and trans in optical isomerism. This is a cis oleic acid. There is a kink due to the double bond. Okay, and there is a trans oleic acid. There is a trans oleic acid. Okay, cis and trans. And alpha linoleic acid. This is of 18 carbon and 3 double bond. Okay, this icosapentaenoic acid. Okay, EPA, icosapentaenoic acid. This is of 20 carbon and 5 double bond. Okay, and docosahexaenoic acid, the DHA or the cervonic acid. Okay, this has 22 carbon and 6 double bond. Okay, so you can remember 18, 20, 22, 2, 2 increase, and 3, 5, 6. Okay, your alpha linoleic, your icosapentaenoic, and your docosahexaenoic. Okay, and linoleic acid, has 18 carbon and 2 double bond. 18 carbon and 2 double bond. Arachidonic acid has 20 carbon and 4 double bond. This is important. Linoleic, just remember the linoleic, the arachidonic. Okay, and your DHA is also important, and your alpha linoleic, this is 18 carbon, 3 double bonds. Okay, so omega fatty acids, there are omega-3 and omega-6. Omega-3 we told, alpha linolenic, timnodonic, and cervonic acid. This timnodonic acid is also called as EPA, which we saw earlier. EPA, icosapentaenoic acid. And omega-6 are your GLA. GLA is your gamma linolenic acid. It is also called as your GLA. Okay, linoleic acid and your arachidonic acid. Arachidonic acid is a precursor of your icosanoids. Okay, icosanoids, which increase the CV disorders and increase degenerative disorders. Okay, and your cervonic acid, the DHA, it will decrease the degenerative disorders, decrease cardiovascular risk, decrease your inflammation, decrease your platelet adhesion. Okay, and one triglyceride produces about 9 kilocalorie per gram of energy. You know about this calorific values from the nutrition chapter. Okay, and triglycerides and glycogen is the storage form of carbohydrates, and the storage form of fats is a triglyceride. Okay, and 1 gram of fat store will give more energy than the hydrated glycogen. Okay, if you talk about the nomenclature of fatty acids, so we write the nomenclature like this: 18:1 omega 9. Okay, 18:1 omega 9. So what is this 18:1 omega 9? This refers to that 18 carbon, it has number of carbon atoms is 18. Okay, and one double bond is there, omega 9 means the position of double bond, 9 means the position of double bond when numbering starts from the methyl end, it is the position of double bond. So double bond is present at your 9th position. Okay, 18 carbons, number of double bond, and the numbering when the numbering starts from the methyl end. Okay, and alternative way you can represent it like this also: 18:1 delta 9. So 18 is the number of carbons, 1 is the number of double bond. Okay, and 9, this 9 is the double bond location, but the numbering starts from a carboxyl end. Now the numbering starts from a carboxyl end. Okay, when it is written like this, the numbering starts from carboxyl end. When it is written like this, the numbering starts from a CH3 end. Okay, if the length of fatty acid increases, the melting point also increases due to London dispersion forces. You must have read about in your inorganic chemistry. And unsaturated fatty acid, they have a lower melting point due to weak intermolecular interaction. Okay, so unsaturated fatty acid, they have lower melting point, so they are more fluidy in nature than a saturated fatty acid, so they have lower melting point. And cis and trans, about cis, this is a cis form, this is a trans form. Okay, and the trans form is bad for health, like your reheated oils and your pies, your cheeseburgers, french fries, banaspati ghee. Okay, these are bad for health. Okay, talking about fats and oils, these are glycerol plus fatty acid, and there's a liquid in form. And waxes, they have higher monounsaturated fatty acids. You remove fast. Okay, so they are solid in form. Okay, they are solid in form. So your waxes generally are saturated parts, so they are not healthy for the intake purposes. All fatty acid of physiological importance. Talking about this. Okay, first we have your formic acid. It has 1 carbon. Okay, and 0 double bond. Okay, acetic acid 2 carbon, 0 double bond. Propionic acid 3 carbon, 0 double bond. Butyric acid 4 carbon, 0 double bond. Capric acid will be 10 carbon. Okay, 10 carbon and 0 double bond. Capric acid 10 carbon and 0 double bond. Okay, and this 4 to 10 carbon fatty acids, these are found in the milk. Okay, this 4 to 10 butyric acid, starting from butyric acid to the capric acid, between we have many other fatty acids which are not much important for you. Okay, so butyric acid up to your capric acid, these are found in the milk. Okay, then palmitic acid, these are 16 carbon and 0 double bond, palmitic acid. But palmitoleic acid has 16 carbons, but 1 double bond at the 9th position. So palmitoleic acid is the 16 carbon and 1 double bond in 9th position. Stearic acid has 18 carbon and 0 double bonds, but oleic acid has 18 carbon and 1 double bond. The numbering which we are doing here, 18:1 omega 9, this refers to your oleic acid. This was the numbering of your oleic acid. Okay, if you can note. I didn't tell you at that time because first I had to discuss the number of double bonds and all. Okay, your oleic acid has 18 carbon and 1 double bond at the 9th position. Okay, linoleic acid has 18 carbon and 2 double bond at the 9th position and the 12th position. Okay, and alpha linolenic acid has 18 carbon and 3 double bond. Okay, at 9, 12 and 15 position. 9, 10, 11, 12 and 15 position. Okay, then arachidonic acid, that is 20 carbon and 4 double bonds, present at 5, 8, 11, 14 positions. 5, 8, 11, 14. 5, 8, 11, 14 position. Okay, and lignoceric acid 24 carbon, 0 double bond. Nervonic acid 24 carbon, 1 double bond. This two you can skip. Okay, just remember this: linoleic acid 18:2. Okay, alpha linolenic 18:3 and arachidonic 24. Okay, understood. Then the stearic acid 18:0, oleic acid 18:1. Okay, palmitoleic acid is 18:1. Okay, and your palmitic acid is 16:0. Just remember this. Okay, and the linoleic acid, this is a precursor of omega 6 arachidonic acid. Okay, which is helping the prostaglandin synthesis. And alpha linolenic acid is a precursor of omega 3 fatty acid. Okay, which are essential for the dietary essentials. Okay, so alpha linolenic acid is a precursor of omega 3 fatty acids. Okay, understood. Up to this you have understood. Let's discuss about complex lipids. What about complex lipids? Is the fatty acid glycerol, it's connected to a phosphate group. Phospholipids, sulfate group is sulfolipids. Amino group is aminolipids. Carbohydrate group is glycolipids. Okay, so fatty acid and sphingosine, here the alcohol is your sphingosine. Okay, plus phosphate group. Okay, here the alcohol was a glycerol and this were formed, but here now the alcohol is a sphingosine, and if you combine the phosphate group, this is the phosphosphingolipids, and fatty acid, sphingosine and carbohydrate group, this is your glycophospholipids. Understood? Glycosphingolipids, this is not phospholipids, that is glycosphingo lipids. Understood? After let's talk about your phospholipids. Phospholipids are polar ionic compounds. Okay, fatty acid, glycerol, and phosphoric acid plus nitrogenous or non-nitrogenous bases are there. Okay, alcohol may be glycerol or may be sphingosine. This is a glycerophospholipid. This is called as sphingophospholipid. Okay, if there is a glycerophospholipid, this may be a nitrogen containing glycerophospholipid, or it can be non-nitrogen containing glycerophospholipid. Okay, so nitrogen containing glycerophospholipid is your LCP. LCP what is? Lecithin, cephalin, and a phosphatidyl serine. Lecithin, your cephalin, and your phosphatidyl serine. Okay, these three are nitrogen containing. Non-nitrogen containing, you have phosphatidyl inositol, phosphatidyl glycerol, diphosphatidylglycerol. glycerol. Okay, phosphatidyl inositol, phosphatidyl glycerol, and diphosphatidylglycerol. glycerol. Okay, understood? And okay, so this is a thing. So LCP, this lecithin, cephalin, phosphatidyl serine, this signal your apoptosis process, and this phosphatidyl serine is present in your cell membrane. And if the concentration of cell membrane increases of phosphatidyl serine, okay, if its concentration increases in your cell membrane, okay, it will lead to a cell death. Talking about lecithin, it's a phosphatidylcholine. Your name is a phosphatidylcholine. It is a lung surfactant. Okay, example is a dipalmitoyl lecithin. Dipalmitoyl lecithin, most commonly found. Okay, like 3 carbons are there. One is attached to a palmitic acid, another one palmitic acid, another one is attached to a phosphate group, and the choline group. Okay. So important clinical is the NRDS, neonatal respiratory distress syndrome. This is seen in premature babies in which the lecithin production haven't started yet. Okay, so what happens is that there is sudden respiratory failure, and the neonate generally dies in your generally dies in your birth and after birth. Okay, so how can you treat it? Breathing support, MgSO4, and steroid therapy can be used to treat your NRDS. Okay, talking about cephalin, phosphatidyl this membrane is present in the cell membrane part, and phosphatidyl serine, this is present in the outer leaflet of cell membrane. Okay, which will signal your apoptosis pathway. Okay, if there is if the phosphatidyl serine is is normally present in your inner leaflet. If it is present in your outer leaflet, then it will cause apoptosis. If it is present in the outer leaflet, then it will cause your apoptosis. Okay, cell death. Then the phosphatidyl inositol, it is a non-nitrogenous base. Okay, so fatty acid, and one is connected to IP2. Okay, so if you add phospholipase C to it, then you will form diacylglycerol DAG and IP3. This IP3 DAG pathway, remember we talked about the IP3 DAG pathway, and which was helping in the calcium was helping in it. Okay, we previously discussed about the mineral metabolism. So this is the IP3 DAG pathway, which act as a secondary messengers in your hormonal action. Okay, talking about cardiolipids. Cardiolipids are first isolated from your cardiac muscles. Okay, in the mitochondrial membrane of the cardiac muscles, you have cardiolipids. It is the only antigenic phospholipid. So VIBA in VIBA it may be asked, the only antigenic phospholipids. These are your cardiolipids. Okay, understood? So it comprises of your phosphatidic acid, your glycerol, your fatty acid O and P. This is a phosphatidic acid. Okay, and again glycerol, then again a phosphatidic acid. Okay, 3 glycerol molecules are present here. Okay, understood? Then we have a Barth syndrome, BARTH, Barth syndrome. Barth syndrome is a THZ gene mutation due to which your cardiovascular myopathies occur. It is X-linked and only present in your males. Okay, we have a Barth syndrome in your cardiolipids. Okay, cardiolipids. So that's all about your up to cardiolipids we have discussed. Okay, let's now talk about some sphingospholipids. Okay, and the sphingolipids. Okay, let's talk about them. Okay, so let's discuss the sphingolipids. These are amino alcohols called as amino alcohols. Okay, and this have a fatty acid, sphingosine, and a sphingosine group. Okay, so these are synthesized from a serine. This is the structure of a sphingosine molecule. You can see the structure of a sphingosine. Okay, this if it's expanded, it looks like something like this. It's just sphingosine. Okay, and if the fatty acid is added to the sphingosine, it is called as a ceramide. So what is ceramide? Sphingosine plus a fatty acid. Okay, and then again, if the phosphate group is attached, okay, the phosphate group is attached, and the choline group is also attached. Okay, so you have the sphingosine. Okay, you have the sphingosine plus your fatty acid is attached, so ceramide is there. So ceramide plus your phosphate group plus your choline group. Okay, so it is called as a sphingomyelin. Okay, it is present in the cell outer plasma lemma. Okay, present in the myelin sheath and present as lipid rafts. Okay, lipid rafts. So sphingomyelin is the only phospholipids with a sphingosine present. So only phospholipids with a sphingosine present is a sphingomyelin. Okay, talking about sphingolipids, you can have sphingomyelin or your sphingoglycolipids. Okay, sphingomyelin, it can be like one example, just your stearic acid. Okay, let's talk about them. Okay, and like this. Okay, we have a sphingoglycolipid like fatty acid, sphingosine, and a carbohydrate group is present. Okay, carbohydrate group, it can have 4 types, like a cerebroside, okay, containing your ceramide, and a monosaccharide. In this carbohydrate part, you have a monosaccharide, okay, and a sulfatide in which your ceramide is there. Ceramide means the fatty acid and sphingosine, okay. Fatty acid sphingosine is there, your monosaccharide is also there, then sulfate group is also there, okay. Then we have the globoside in the fatty acid and sphingosine, the ceramide is there, and the oligosaccharide is attached to the carbohydrate in the place of a carbohydrate group, okay, like a galactosyl ceramide, okay. And then you have gangliosides you can have ceramide, your fatty acid and sphingosine present, okay, your oligosaccharide plus your sialic acid, it is also called as a NANA, okay. What is NANA? N-acetyl neuraminic acid, which is also called as a sialic acid, okay. So this based on the number of sialic acid attached, okay, that can be of 4 types, your GM, GD, GT, and your GQ, okay, your GQ, okay. So this can be GM1, GM2, and GM3, okay. In which the GM has isomers, okay, the GM has isomers, okay, and based on the structure, the smallest one is the GM3, okay, which migrates the farthest towards the cathode, okay. Talking about sphingoglycolipids, these are present in the nervous tissue and participate in the cell-to-cell interactions, okay. GM1, talking about GM1, acts as a receptor for cholera toxin, okay. Your ganglioside 1, okay, the GM1 acting as a receptor for a cholera toxin, like a Vibrio cholerae, okay. It is a AB5 multimeric protein, okay. This will act as a receptor for cholera toxin, due to the cholera toxin comes and stimulates the CAMP synthesis, okay. This CAMP synthesis will cause the release of various CAMP dependent channels, like sodium, chloride, potassium, HCO3, and water, okay. This will cause the release, and there will be massive watery diarrhea, okay. And talking about lysosomal storage disorders, we have the sphingolipidosis and we have the mucopolysaccharidosis, the GAGs, like Hunter disease and Hurler's disease. We have talked about both of them, okay. Talking about sphingolipidosis, okay, we have to discuss. These are your defect in degradation of lipids, okay. There is a defect in degradation of lipids, okay. These are a group of lysosomal storage disorders only, okay, which are inherited deficiencies of your lysosomal enzymes, okay. So we can have GM1 deficiency, okay, the GM1, okay, GM1 sphingolipidosis, or a beta galactosiderosis, okay, or a GM2, which is divided into Tay-Sachs and a Sandhoff's disease. We have the Krabbe disease, the Gaucher disease, Niemann-Pick disease, the Farber's, Fabry's, and the metachromatic leukodystrophy, the Wallmann's disease. Wallmann's disease is not a sphingolipidosis, but it is a lysosomal storage disorder. But these are sphingolipidosis and again lysosomal storage disorders also, okay. And Wallmann's is not a sphingolipidosis. Talking about a GM1, the GM1 is broken down by the beta galactosidase into GM2, okay. And if there is the defect in the beta galactosidase, okay, it is called as a beta galactosidosis, okay, or a GM1 sphingolipidosis, okay. If there is the defect of beta galactosidase, then how will be the patient present, okay. Patient will present with low set ears, depressed nasal bridge, long philtrum, and frontal bossing. Is there, okay, and low set ears are there, you can see, okay. And clinical effects will be the blindness, angiokeratoma, hepatosplenomegaly, mental retardation, okay, and macular cherry spot. This is specific for macular cherry spots are seen in various lysosomal storage disorders, okay, or your sphingolipidosis, okay. And then this GM2 is broken down into GM3 by beta hexosaminidase, okay. And this genetic defect in beta hexosaminidase, okay, which has 2 isoforms, the A and B, okay. Based on this, we have the Tay-Sachs disease or a Sandhoff's disease, okay. The beta hexosaminidase has 2 isoforms, your A isoform and a B isoform, okay. This A isoform is 1 alpha 1 beta, and this B isoform is 2 beta, okay. Based on this, we have Tay-Sachs disease or we have your Sandhoff's disease, okay. This Tay-Sachs disease is due to the defect in alpha subunit. Due to defect in alpha, only hexosaminidase A deficiency will occur, okay. There will be defect in hexosaminidase A, okay. So cherry red spot will be there, neurological problems, macrocephaly, and hyperacusis, okay. And in the Sandhoff's disease, the B is mutated, okay. There's a genetic defect in the B subunit, okay. B subunit is present in both A and B, okay. So there is beta hexosaminidase A and B defect. The same problems will be there: cherry red spot, neurological problem, macrocephaly, hyperacusis. Same problems will be there, and along with it, hepatosplenomegaly, bone deformities, and cardiac abnormalities will be there also, okay. So you can just see this disorder, okay. Talking about other disorders like a Krabbe disease and a Gaucher disease, okay. So GM1 and GM2 beta galactosidase, and beta galactosidase deficiency is a GM1 sphingolipidosis, okay. And GM2 to GM3 by beta hexosaminidase, we have Tay-Sachs and Sandhoff. From GM3 we get beta glucocerebrosidase. This beta glucocerebrosidase is converted to ceramide, converted to ceramide by beta glucocerebrosidase enzyme. Okay, beta glucocerebroside is converted to ceramide by beta glucocerebrosidase enzyme, and deficiency of beta glucocerebroside will cause your Gaucher's disease. Okay, and Gaucher's disease is non-neural disorder. It is a non-neural disorder. Okay, and your sphingomyelin is also degraded by sphingomyelinase, which will also convert it into ceramide. Okay, and if there is a deficiency of your sphingomyelinase, we call it as Niemann-Pick disease. Okay, and your beta galactocerebrosidase is also converted into a ceramide by beta galactocerebrosidase. Okay, deficiency will call it as your Krabbe disease. Okay, so Krabbe disease and Gaucher's disease differences. Krabbe disease is seen in your 5-month-old child, and your Gaucher's disease in a 3-year-old child. Okay, and Krabbe disease, the patient will present with developmental delays, and with time condition will deteriorate. Okay, opisthotonous posture, upright posture is there, and clenched fist. The patient present with opisthotonous posture and clenched fist. This is specific for this. Okay, and there is no hepatosplenomegaly. And neurological deficits will be there, as it is a neural disorder. Okay, and globoid cell inclusions will be there. Because in neural tissue, beta galactocerebrosidase will accumulate. This will lead to enlarged macrophages, which are called as a globoid cell inclusions. Okay, and in Gaucher's disease, it will occur in a 3-year-old child. Okay, 3-year-old child. Severe bone pain will be there, and pancytopenia, anemia will be there, early bruising, abdominal distension. Okay, then we have the Erlenmeyer flask. What is Erlenmeyer flask? It is the abnormality in the lower limbs. Okay, abnormality in the lower limbs is called as a Erlenmeyer flask. Okay, so you can see the Erlenmeyer flask here. Okay, Erlenmeyer flask deformity. Okay, Erlenmeyer flask deformity, and there will be no intellectual deficit because it is a non-neural disorder. Okay, and no cherry red spot. Okay, so there is no cherry red spot in case of your Gaucher's disease. Okay, you can get a short note on your Gaucher's disease. Okay, this is important, and you can also get a short note on your beta galactosidosis. This is also important. Okay, and your Tay-Sachs disease. Sandoff's is not much important. Tay-Sachs disease you can get a short note. Okay, and about treating Gaucher's disease, you can do enzyme replacement therapy like tildeglucarase, venia-glucarase, and imiglucerase. These all enzymes are used. These are recombinant beta glucosidases. Okay, then you can have oral substrate reduction therapy like miglustat. Okay, you can use this, and bone marrow transplant. These are not much important treating Gaucher's disease. You can get a short note. This is important for a short note purposes. Your Niemann-Pick disease, or your as per your prof is concerned, but these disorders are asked extensively in a PG entrances, so I have just taught them in detail. Okay, Niemann-Pick disease is the biochemical defect. Okay, in sphingomyelinase enzyme. The sphingomyelinase enzyme was converting your sphingomyelin to your ceramide. Okay, so it is the deficiency of your biochemical defect in the sphingomyelinase. Okay, due to which the sphingomyelin accumulates, causing hepatosplenomegaly. Okay, foam cells. This is very important in your Niemann-Pick. Foam cell or zebra body inclusions. If the question is asked, clinical case is given. This is important for clinical case also. So if your foam cells or zebra body inclusions are added, or lipid laden macrophages are given, okay, then you think about the foam cells, and the foam cells mean Niemann-Pick disease. Okay, and macular cherry red spot will be there. Okay, Fabry's disease, this is X-linked recessive and occurs in males only. Okay, and Fabry's crisis is another thing, which is agonizing pain in your proximal extremities. Okay, you will have pain in your extremities. Okay, and what is the triad of Fabry's disease? Fabry's disease has a triad, that is peripheral neuropathy, your angiokeratoma, skin eruptions, and hypohidrosis. Okay, these are your triad of your Fabry crisis. Okay, and other things are corneal clouding will be there, opacity will be reduced. Okay, then Maltese appearance in urinary segments will be there. Treatment is by enzyme replacement therapy like your Fabrazyme and your Riplagal. Okay, then about Farber's disease, this is a acid ceramidase defect. When there will be painful swollen joints in 18 months old baby. Okay, this occurs in neonates. Farber disease. Okay, painful joints in your baby, acid ceramidase defect. Then you have metachromatic leukodystrophy. There is the aryl sulfatase defect. Okay, there will be dementia. Okay, and central and peripheral demyelination as well as ataxia. Ataxia will occur, and the demyelination will also occur. Then you have the Ullmann's disease, WOLMAN, Ullmann's disease, where there will be acid lipase defect. Okay, acid lipase defect. It is not a sphingolipidosis. It is a lysosomal storage disorder. Okay, the only lysosomal storage disorder. Okay, that is not a sphingolipidosis. That is a Ullmann's disease. Okay, whereas the effect of acid lipase will be deficient. Okay, due to which there will be cholesterol ester degradation. The cholesterol ester won't be degraded, which is called as a cholesterol ester storage disorder, CERD. Okay, and it will cause your vomiting and watery green diarrhea. Watery green diarrhea is the characteristic feature of your Ullmann's disease, the characteristic feature. Hepatosplenomegaly and the calcification of adrenal gland. This is also specific for your Ullmann disorder and failure to thrive. Okay, this will be the effects of your Ullmann's disease. Okay. So we finished your sphingolipidosis and your sphingolipids, about sphingolipids in much detail, which are just more than enough for your prop examinations and more than enough for a PG entrances too. Okay. Let's talk about some miscellaneous concepts in the lipid chemistry. One is a liposome. You can get a short note on liposome. So there's a spherical vesicles shaped like this. Okay. At the center you can have a drug or protein or enzyme or a gene which is called as your cargo. Okay, the cargo. Okay, so cargo is any drug, any protein or any enzyme or any gene which is carried by the liposomes. How it is formed? It is formed by the sonication of a mixture of phospholipids and cholesterol. The mixture of phospholipids and cholesterol are irradiated by the sonic rays. Okay, what is the function? Act as a carrier, cargo for drug, proteins, enzymes and genes. Applications is in cancer chemotherapy, gene therapy, vaccines and diagnostic imaging. Okay, and the oral preparation of sucrosomal iron, oral preparation of the sucrosomal iron. Okay, these are introduced to avoid gastric irritation. So this is a therapy. Size is about 0.025 mu to 0.25 micron. Okay, 0.025 micron to 2.5 micron. Types liposomes are multilamellar vesicles, MLV, or unilamellar vesicles, ULV. So multilaminar or onion-like structures. Unilamellar may be large unilamellar or small unilamellar vesicles. Okay. A little bit about your surfactant. Okay, so it is made up of lipids and as well as proteins. Okay, you can get a short note on your surfactant also. So 85% of lipids is your dipalmitoyl lecithin, it's the main component. Okay, and 10% is your phosphatidyl glycerol is also present. Plasma proteins are also present, 50%, and rest are apolipoproteins, SP, A, B, C, and D. Okay, SP, what is SP? A surfactant associated protein. SP is a surfactant associated protein. Then your respiratory distress syndrome, RDS, respiratory distress syndrome, it occurs in your infants. Your neonatal respiratory distress syndrome, it is also called. Apnea will occur, cyanosis will occur, grunting, nasal flaring, and your tachypnea. Okay, and there will be ground glass appearance due to alveolar atelectasis. Okay, if you see the alveoli under the microscope, you will see ground glass appearance. Okay, because the walls of alveoli have been damaged, there is alveolar atelectasis. Okay, and this is the defect in the DPL biosynthesis. Okay, pathway, that is your dipalmethyl lecithin is not synthesized. Okay, understood. And you have another syndrome which is called as a meconium aspiration syndrome. Where if there's meconium staining amniotic fluid, which is indicate your fetal distress syndrome. Okay, meconium is the which will destroy the surfactant and increase the surface absorption rate. Okay, understood. So this is a meconium aspiration syndrome. Let's talk about a pulmonary alveolar proteinosis, PAP, pulmonary alveolar proteinosis. These are syndromes, especially these are some clinical facts which I am telling you. Okay, so in your alveoli proteinaceous materials are accumulated and the infants die within 1 year of life. Neonatal hyaline membrane disorder, also called as NRDS. Again, it is an NRDS. Okay, so waxy layer of hyaline membrane line the surface of alveoli, causing a ground glass appearance, as I told earlier. Okay, to check your fetal lung maturity, this is important. To check your fetal lung maturity, use LS ratio, lecithin sphingomyelin ratio. Okay, so the normal LS ratio is about 2:1. Okay, if it is less than 2:1, then the lungs may be surfactant deficient. Okay, then the fetal lung haven't matured yet. The lung is surfactant deficient if the LS ratio is less than 2:1. Normal is your 2:1. Okay. Saponification, again I think the triglycerides plus alkali, if there is hydrolysis, then glycerols and soaps are produced. Okay, saponification number is the number of milligrams of potassium hydroxide which is required, KOH, which is required to saponify at least 1 gram of fats. This is the number of milligrams of KOH required to saponify 1 gram of fats. Talking about iodine numbers, it is the number of grams of iodine, not atoms, number of grams of iodine taken up by 100 gram of fat. This is the number of grams of iodine taken by 100 gram of fat, and it is an index of degree of unsaturation. Okay, more the iodine number, more is the degree of unsaturation. Okay, talking about rancidity of fat, rancidity means unpleasant taste and smell. Okay, you can have hydrolytic rancidity and have oxidative rancidity. These two are there. Okay, hydrolytic rancidity is due to partial hydrolysis of triacylglycerol molecules, but oxidative rancidity is due to partial oxidation of unsaturated fatty acids, due to which free radicals are formed, your epoxides and peroxides are formed. Okay, and this will damage your food, causing oxidative rancidity. Okay, so these were some miscellaneous topics. Let's move on and read a little bit about cholesterol before going to the fatty acid metabolism. Okay, Okay, so cholesterol have cyclopentanoparahydrophenanthrene ring, CPPD ring. Okay, cyclopentanoparahydrophenanthrene ring, where we have 3 cyclohexane rings and 1 cyclopentane ring. 3 cyclohexane rings are called as A, B, C, and 1 cyclopentane ring is D. Okay, so have I drawn the structure of your cholesterol? Okay, let's see the structure of cholesterol again. Okay, so here you have the structure of cholesterol. Okay, so A, B, C, and D are there. Okay, 3 cyclohexane rings. Okay, this OH group is attached at the 3rd position. There is a double bond between 5th and 6th positions. Okay, 1 cyclopentane ring is there, D. Okay, and then to it attached is a chain of carbon atoms. Okay, the formula is C27H5OH. It has 27 carbons. C27H5OH. One double bond is present between the C5 and C6. One double bond is present between the C5 and C6, and the OH group is at 3rd position. OH group is present at the 3rd position, and the several centers of asymmetry. Okay, it has many isomers. Okay, like your cholestanol and your coprostanol. These are isomers of your cholesterol. Okay, what the function? It modulates the fluid state of cell membrane. It insulates the nerves, bile acid and bile salt synthesis, steroid hormone synthesis, and 7-dehydrocholesterol is converted to vitamin D3 in the skin, and OH group esterified at the 3rd position is forming a cholesterol esters. Okay, this cholesterol OH plus this fatty acid, when it is acted upon by L-cat enzyme. Okay, L-cat enzyme, because cholesterol is OH group and fatty acids and esterified by L-cat enzyme lecithin cholesterol acyl transferase, this form a cholesterol ester. Okay, what are the clinical correlations? Excess of cholesterol will lead to atherosclerosis, which can cause a myocardial infarction or a cerebral infarction. Okay, or a cardiovascular accidents and heart attacks. Heart attack risks is increases. Okay, due to excess of cholesterol. So this was just a basic discussion about cholesterol, and let's now move with no further delay to the lipid and to the fat metabolism. Okay, let's discuss the fat metabolism, lipid metabolism. So basically, we are into the beta oxidation of fatty acids. Okay, which will occur in 3 steps. First one is the activation of fatty acid, then it's a transport, then beta oxidation proper. Beta oxidation proper will have also 4 steps: your oxidation, hydration, oxidation, and thiolytic cleavage. Okay, so talking about beta oxidation, it will occur in the mitochondria of muscles, adipose tissue, and liver. Alpha oxidation occurs in the peroxisomes and ER, and omega oxidation, okay, this will occur in your ER only. Okay, so first, what is activation? The fatty acid is first activated. This fatty acid in the presence of thiokinase, and where ATP is utilized, it is changed to acyl adenylate. This acyl adenylate in the presence of thiokinase again, where CoASH gives your AMP. Okay, and this is from your acyl CoA. This is forming your acyl CoA. This acyl CoA is transported. This can't cross your mitochondrial membrane like this directly. Okay, it is first converted. This acyl CoA is converted by addition of carnitine by carnitine acyl transferase CAT1 enzyme. This CAT1 enzyme, this acyl CoA is converted to acyl carnitine. Okay, and this acyl carnitine can easily cross the mitochondrial membrane by the help of your enzyme translocase, and this acyl carnitine is again your carnitine is removed by CAT2 enzyme. Okay, and it is again converted into your acyl CoA. So acyl CoA thus comes into your mitochondria. This is a transport mechanism. And beta oxidation proper, this is the pathway. The acyl CoA in the presence of acyl CoA dehydrogenase, where FAD converts into FADH2. This is converted into trans-enoyl CoA. Okay, trans-enoyl CoA. This trans-enoyl CoA again in the presence of enoyl CoA hydratase. Okay, enoyl CoA hydratase, where water is added. This is converted into beta hydroxyacyl CoA. This beta hydroxyacyl CoA in the presence of beta hydroxyacyl CoA dehydrogenase. Okay, it is converted to ketoacyl CoA, where NAD plus changed into NADH plus H plus. Okay, and 2.5 ATPs are generated. So beta hydroxyacyl CoA in the presence of beta hydroxyacyl CoA dehydrogenase is converted to ketoacyl CoA. This ketoacyl CoA in the presence of thiolase converts into acyl CoA plus acetyl CoA. Acyl CoA plus acetyl CoA mixture is there, and from this acetyl CoA, this goes into a TCA cycle. Okay, generating your energy. Okay, there's a clinical here that if the toxin from your unripe ackee fruit is there, from the unripe ackee fruit a toxin is released, which is a hypoglycin A toxin is released, and this hypoglycin A inhibits your acyl CoA dehydrogenase, causing Jamaican vomiting sickness, This is given in Harper. Then due to the deficiency of acyl CoA dehydrogenase in your infants, there is the sudden infant respiratory death syndrome, SIDS. Okay, and the death occurs due to blockage of a beta oxidation pathway. Okay, talking about your this is that. Then odd chain fatty acid. What about the odd chain fatty acids? They will give the acetyl CoA plus propionyl CoA. Okay, they don't give your acyl CoA. This fatty acid oxidation will occur, and acyl CoA plus acetyl CoA won't be given. Your acetyl CoA plus your propionyl CoA is given. Okay, so propionyl CoA, this is converted by propionyl CoA carboxylase enzyme, propionyl CoA carboxylase in the presence of ATP, biotin and CO2 to methyl manolin CoA, and this methyl manolin CoA in the presence of methyl manolin CoA mutase, okay, methyl manolin CoA mutase and B12 presence, it is converted into succinyl CoA. This succinyl CoA can go into your TCA cycle now. Okay, so odd chain fatty acids have this type of generation of your ATP. Okay, talking about energetics, let's take a palmitic acid. Palmitic acid is 16 carbon. Okay, if there is a 16 carbon fatty acid, there will be 7 FADS2 generated and 7 NADS2 generated. Okay, 7 FADS2 and 7 NADS2 generation. Okay, if you just do the sum, the FADS2 will give 1.5 ATP, the NADS2 will give your 2.5 ATP. So the ATPs are there. Okay, and there will be formation if there is 16 carbon present, then there will be formation of your 8 acetyl CoA. 8 acetyl CoA, this 8 acetyl CoA as 1 acetyl CoA in Krebs cycle gives you 10 ATP. So 8 acetyl CoA will give you 80 ATP. So 10.5 ATP, 17.5 ATP plus 80 ATP will give you 108 ATP. And what ATP is utilized also here? 1 ATP is utilized, and let's say 2 ATPs are utilized here. Okay, 2 ATPs are utilized here. Okay, on this activation step. So minus 2 ATP is 106 ATP, and stearic acid thus gives you 120 ATP. You can calculate from this. Okay, what is the regulation? So if you are well-fed state, in the well-fed state, okay, the beta oxidation is inhibited. How? In the well-fed state, we have increased insulin and decreased glycogen. Okay, and this increased insulin will stimulate your fatty acid synthesis. This fatty acid synthesis occurs, fatty acid synthesis starts occurring and will increase in manolin CoA. This increased manolin CoA will inhibit your CAT1 enzyme, and if this CAT1 is inhibited, okay, then your beta oxidation won't occur. Okay, as you are well-fed state, as you are in a well-fed state, you have no dearth of ATP. Okay, so there won't be beta oxidation so as to produce more ATP. Okay, but in fasting state, you know you require your ATP. Okay, your fasting state, so there is stimulation to your beta oxidation. How? Increased glycogen will inhibit your fatty acid synthesis, and inhibition of fatty acid synthesis, we will discuss the fatty acid synthesis in a while. Okay, and inhibition of your fatty acid synthesis will cause a decreased manolin CoA, and decreased manolin CoA will stimulate your CAT1 enzyme, and this CAT1 enzyme will cause your transport of this acyl CoA and will stimulate your beta oxidation. Okay, talking about Repsum disease. Repsum disease is the deficiency of phytanic acid oxidase. Okay, in alpha oxidation, this is a defect of alpha oxidation. So it is a disorder for alpha oxidation, and there is decrease in your phytanic acid oxidase, where you will see neurological manifestations, retinitis pigmentosa, okay, and ataxia. Okay, due to accumulation of phytanic acid in brain, the neurological manifestations will occur. Okay, then a Zellweger syndrome is there. Zellweger syndrome is also called as your cerebrohepatorenal syndrome. Cerebrohepatorenal syndrome, we have discussed your Zellweger syndrome back in your protein targeting. Okay, in the cerebrohepatorenal syndrome, as brain, liver and kidney, all three are affected. Brain, liver and kidney, all three are affected. Okay, here due to absence of your functional peroxisome, your functional peroxisomes are absent. So there is effect to the very long chain fatty acid oxidation. Okay, the VLCFS, this will accumulate in these parts, your brain, liver and kidney, and will affect this will cause also a neurological manifestations and ataxia type of causes. Okay, and liver damage will also be there. Okay, now let's discuss the fatty acid synthase complex. So let's talk about fatty acid synthase complex. It is a multi-enzyme complex. We know what is multi-enzyme complex in your enzyme discussion. It has 7 enzymes and 1 acyl carrier protein. All enzymes are present in equimolar concentration and derived from the same gene. Okay, so there are 3 units: a condensation unit, a reducing unit, and a releasing unit. Okay, the condensation unit will contain Ks-ATMT. Okay, just remember Ks-ATMT. Okay, what is Ks? Ketoacyl synthase. Okay, AT will be the acyl transacetylase, and MT is manolil transacetylase. Okay, reducing unit has DR, Kr, ACP. Okay, just remember DR, you know the DR. Okay, Kr and ACP. Okay, just to remember this. Okay, DEER. Okay, what is DEER? Your dehydratase. Okay, then ER is your enoyl reductase, Kr is your ketoacyl reductase, ACP is your acyl carrier protein. Okay, one carrier protein, acyl carrier protein. Okay, so dehydratase, okay, then your enoyl reductase, then your ketoacyl reductase, and your acyl carrier protein. And releasing unit has only one enzyme, that is thioesterase. Okay, thioesterase only. Understood? And the condensation unit, you know, Ks-ATMT, releasing unit DRKRACP, and the releasing unit thioesterase. Just remember all the enzymes of the fatty acid complex. Okay, afterwards let's start with the de novo synthesis of fatty acid, where these enzymes will be used. Okay, so de novo synthesis, the rate limiting enzyme is your acetyl CoA carboxylase enzyme. Okay, So first we'll see the transport of acetyl CoA, how the acetyl CoA transported. So acetyl CoA combines with your oxaloacetic acid to form the citrate. This is the step of your Krebs cycle only. Okay, this citrate can now cross the membrane. Okay, this acetyl CoA can't cross the mitochondrial, the membrane of the mitochondria. Okay, so it transforms itself into citrate and crosses the membrane. The citrate crosses the membrane. The citrate comes and ATP citrate lyase will act, okay, which will break the citrate into your acetyl CoA and your oxaloacetic acid again, ATP citrate lyase. So we get the acetyl CoA like this. Okay, transport is there. Then we will have activation of acetyl CoA. Okay, the acetyl CoA comes here, then there will be activation. Okay, so the acetyl CoA in the presence of your acetyl CoA carboxylase, the rate limiting enzyme. This is the rate limiting enzyme, acetyl CoA carboxylase, in the presence of ATP, biotin, and CO2. Okay, it gets converted into malonyl CoA. Malonyl CoA. Okay. Afterwards, this acetyl CoA plus ENZ CYS SH, ENZ CYS SH. Okay, this will in the presence of acetyl transacetylase. Acetyl transacetylase, you know, okay, KS80MT we are learning. Okay, so this is the AT condensation unit only. It condenses acetyl CoA and ENZ CYS SH to form acetyl ENZ, okay, and CoA SH and CoA SH. Then afterwards, the manonyl CoA also reacts with the ACP SH. Okay, the ACP SH. Okay, in the presence of malonyl transacetylase, okay, MT. So we have the MT now, and MT was also of your condensing unit. Okay, and you form your manonyl ACP and CoA SH, manonyl ACP and CoA SH. Okay, so you have the manonyl ACP and the acetyl ENZ. Okay, this both will react now, acetyl ENZ plus manonyl ACP, in the presence of your ketoacyl synthetase, your KS, first enzyme, KS, ketoacyl synthetase, and form your beta ketoacyl ACP. This beta ketoacyl ACP in the presence of ketoacyl reductase of your second part, that is your reducing part. Okay, you will get beta acyl ACP converts into beta hydroxyacyl ACP, okay, in the presence of ketoacyl reductase, where NADPH changes to NADP plus, okay, and then this beta hydroxyacyl ACP in the presence of dehydratase, okay, will form your enoyl ACP. Okay, so beta hydroxyacyl ACP in the presence of dehydratase forms your enoyl ACP. This enoyl ACP again in the presence of enoyl reductase, okay, this is also a enzyme of your reducing unit, okay, where NADPH plus H plus combines to form NADP plus. Okay, this forms your butyryl ACP. This is enoyl ACP forms your butyryl ACP, and butyryl ACP in the presence of thiokinase enzyme, after 6 cycles will give you the palmitate. Understood. And regulation, the acetyl CoA carboxylase enzyme is activated by citrate, is also activated by insulin, okay, and it is inhibited by your glucagon. Understood. So this is the regulation part. So you can now make the palmitate, which is a fatty acid or lipid. Okay, let's talk about your cholesterol. Cholesterol is a derived lipid. We learned a few important points about the cholesterol, like it is a 27 carbon compound, C27H45OH. OH is present in the third position. Double bond is there between fourth and fifth carbon. Okay, so this is the structure. We talked about the structure too in the lipid chemistry, which we discussed today only. Okay, this is the precursor of steroid hormones, fat soluble vitamins, component of the cell membrane, fluidity buffer, acts as the fluidity buffer. Okay, makes the membrane fluid or solid according to the needs. Okay, fluidity buffer, and transport of substances across cell membrane. This is also done by your cholesterol. Okay. Let's talk about the synthesis of cholesterol, how the cholesterol is synthesized. So this is synthesized in the cytosol and ER of all of these organs. Okay, so first what happens is a long pathway. Okay, but trust me, that pathway is easy. Okay, the pathway is not difficult like that. Okay, okay, learn this much. Fatty liver is there. Okay, I know the video is getting very long, but we are covering each and every point that are required for your prof examination or your PG is concerned. Okay, so synthesis of cholesterol, first 2-acetyl CoA is there. Okay, in the presence of thiolase enzyme, it gets converted into acetoacetyl CoA. But CoA-SH is taken out. Okay, this acetyl CoA converts into acetoacetyl CoA. The 2 units are condensed in the presence of your thiolase. Okay, then this acetoacetyl CoA in the presence of HMG CoA synthase. Okay, HMG CoA synthase, where acetyl CoA gets again converted into CoA-SH. Okay, this gets converted into beta hydroxymethyl glutaryl CoA, or just you can say HMG CoA. This HMG CoA in the presence of your HMG CoA reductase. Okay, HMG CoA reductase, which is the rate limiting enzyme. Okay, it's a rate limiting step or a committed step of this cholesterol synthesis. Okay, HMG-CoA reductase. And where NADPH plus H plus change into NADP plus, it forms a mevalonate. It forms a mevalonate. Okay, this mevalonate in the presence of kinase, forms a 5-mevalonate. Again, in the presence of kinase, it forms 5-pyrophosphomevalonate. 2 phosphates are added again. In the presence of again kinase, it forms 3-phospho, 5-pyrophosphomevalonate. Okay, again, so 3 steps of ATP, that kinase activity are there. Okay, mevalonate, 5-mevalonate, 5-pyrophosphomevalonate. Okay, and 3-phospho, 5-pyrophosphomevalonate. Okay, then we'll use decarboxylase. The CO2 is removed, forming the isopentenyl pyrophosphate. Isopentenyl pyrophosphate. Okay, and on isomerase action, it is converted into dimethyl allyl pyrophosphate. Okay, this isopentenyl pyrophosphate in the presence of isomerase is converted into dimethyl allyl pyrophosphate. And this dimethyl allyl pyrophosphate, in the where IPP is added, it forms geranyl pyrophosphate. Again IPP is added, it forms farnesyl pyrophosphate. Again IPP is added, again FPP is added. Okay, and the NADPH plus H plus is converted into NADP plus, and your squalene is formed. Squalene. Okay, squalene, 30 carbon compound squalene. Then you have action of your EHC, epoxidase, hydroxylase, and your cyclase, which will convert the squalene to your lanosterol. Okay, which is also a 30 carbon compound. And on this lanosterol, HCOOH and 2CO2 gets removed from 30 carbon. 3 carbons are getting removed. Okay, like HCOOH and 2 carbons of this. Okay, and it is forming a cholesterol, 27 carbon compound. Okay, so this is the whole pathway how the cholesterol is synthesized. Okay, a little bit complex pathway, but if you can just practice once, you can easily write. Okay, how it is occurring. HMG-CoA reductase, this is inhibited by your glucagon. Okay, by phosphorylation, this is inhibited by your glucagon, and this is inhibited by statins also. So statins are used as a blood cholesterol reducing lowering agents or blood cholesterol reducing agents. Okay, because the statins are inhibiting this step. Okay, so the whole pathway won't occur and the cholesterol formation won't occur. Okay, so cholesterol will be reduced. Okay, and insulin and thyroxine, these are stimulating the pathway HMG-CoA reductase. Okay, okay, talking about your degradation of cholesterol. So cholesterol is degraded into bile acids. It may be degraded into vitamin D, may be degraded into your steroid hormones. Okay, so bile acid, first let's talk about bile acid. So cholesterol gets converted into your 7-dehydroxycholesterol. Hydroxycholesterol. Okay, cholesterol gets degraded into 7-hydroxycholesterol, which splits to give your cholic acid and chenodeoxycholic acid. Your cholic acid and your chenodeoxycholic acid. This cholic acid combines with your glycine to form glycocholic and taurine to form taurocholic acid. Okay, understood? And this chenodeoxycholic acid, this gets combined with your glycine to form chenodeoxycholic acid and the taurine to form your taurocholic acid again. Okay, so this, these are called as the primary bile acids. Okay, and this can in further course can form lithocholic acid, which is your secondary bile acid. Talking about vitamin D, how vitamin D is formed from cholesterol. Okay, so 7-dehydroxycholesterol is there in our skin. In the presence of UV rays, it is converted into a cholecalciferol. Okay, cholecalciferol. This cholecalciferol, if 25-hydroxylase enzyme is acted upon by cholecalciferol. Okay, this is from your liver. Okay, and your 7-hydroxycholecalciferol is done. Okay, formed, and this 25. Not 7, the 25. Okay, 25-hydroxycholecalciferol is formed. This 25-hydroxycholecalciferol is acted upon by 1-alpha-hydroxylase from your kidney. Okay, this enzyme comes from your kidney and forms a 125-dihydroxycholecalciferol. Okay, this 125-dihydroxycholecalciferol is called as a calcitriol. Okay, and it is the active form of your vitamin D. Understood? After this, talking about steroid hormones, how they are formed. The cholesterol can split into your aldosterone or your cortisol, or can form your sex steroids like a testosterone, progesterone, and estrogen. Okay, we have separate pathways forming this. Okay, you will read it in physiology. Okay, these are also little bit bigger pathways, but these are more related with the physiology part. Okay, so I am not discussing it here. Okay. Talking about the ketone body metabolism, how the ketone bodies are metabolized. Okay, ketone bodies like acetone, acetoacetate, and beta hydroxybutyrate is there. These are the ketone bodies. You just see the structures. This is acetone, acetoacetate, and beta hydroxybutyrate. Okay, ketone bodies. And the clinical importance of ketone bodies is that in uncontrolled diabetes mellitus, okay, there is decreased glucose level, okay, which is increased lipolysis and increased acetyl CoA, forming a ketone bodies. Then in starvation, you will have decreased glucose level, Okay, just remember what I'm telling. Uncontrolled diabetes mellitus, it is occurring, okay, in decreased glucose level due to uncontrolled diabetes mellitus. But in if there is only diabetes mellitus, okay, the glucose level are high, but the uncontrolled ones, the glucose levels decrease at a exponential rate. Okay, so in starvation you have decreased glucose. Okay, in decreased glucose, increased fatty acid consumption will be there due to decreased glucose. Lipolysis will increase. Your acetyl CoA will be formed more and more, and your ketone bodies will appear in urine. So in DM and your starvation, in both, the ketone bodies will come in your urine. Okay. Okay, so how you synthesize your ketone body? Let's talk about the ketone body synthesis and the ketone body utilization. Okay, ketogenesis and your ketone body utilization. So this acetyl CoA and acetyl CoA, these are condensed to acetyl CoA in the presence of thiolase to form acetoacetyl CoA, which in the presence of your HMG CoA synthase, not your HMG CoA lyso reductase. Okay, it is where HMG CoA synthase. Okay, the same step we are doing as in your cholesterol synthesis. HMG CoA synthase to form HMG CoA by addition of your acetyl CoA. Okay, and now we don't add your HMG CoA ligase, if your HMG CoA reductase. Okay, if we have added HMG CoA reductase, then here mevalonate will be have formed, but here we are adding HMG CoA ligase. Okay, it is converting it into acetoacetate, which is the primary ketone body. Acetoacetate is the primary ketone body. And this acetoacetate can produce a beta hydroxybutyrate, okay, by the enzyme beta hydroxybutyrate dehydrogenase, where NAD plus is changed into NAD plus, NADH plus is changed into NAD plus. Okay, beta hydroxybutyrate dehydrogenase and beta hydroxybutyrate is formed. This acetoacetate on spontaneous decarboxylations, many decarboxylations will occur. Okay, CO2 will be released like this. Okay, and acetone will be formed at the end. Okay. And talking about ketone body, how they're utilized. Okay, and the beta hydroxybutyrate is the predominant ketone body. Okay, this beta hydroxybutyrate, again in the presence of beta hydroxybutyrate dehydrogenase, where NAD plus is changed into NADH plus H plus. Okay, this converting into acetoacetate. This acetoacetate in the presence of thiophorase, important enzyme thiophorase. Okay, thiophorase, but succinyl CoA changes into succinate. Okay, it is converting it into acetoacetyl CoA. Acetoacetate in the presence of thiophorase gives you acetoacetyl CoA. Okay, but the succinyl CoA changing into a succinate. Okay, so acetoacetyl CoA, this acetoacetyl CoA, on adding CoA as such, you get acetyl CoA and acetyl CoA. Okay, so two acetyl CoAs are produced. Okay, and this two acetyl CoA can again go into ketone body synthetic pathways and form a basic ketone body. Okay, so you have a balance of this ketone body formation and ketone body utilization. About the energetics, this beta hydroxybutyrate gives you 21.5 ATP. Okay, and this acetoacetate will give you 19 ATP. Okay, you don't have to go into detail for this calculations. Okay. Then ketonemia. Ketonemia is the increased ketone body in the blood, and ketonuria is the increased ketone body levels in your urine. Okay, this both when combined is called as your ketosis. Like in diabetic ketoacidosis or your diabetes. Okay, ketosis will occur. And the test for ketone body, is the Rothera's test, which will form a purple ring at the interface. Okay. Talking about the chylomicron metabolism, this is also important. The chylomicrons, they have a density of about less than 0.95 gram per liter, comprised of 1% protein, 99% lipids like triglyceride and cholesterol. Okay, they have the apoproteins present in them like B48 apoprotein, ApoC2, ApoA, and ApoE are present in the chylomicrons. So site of synthesis is your intestine. Okay. And from the GIT, okay, this exogenous triacylglycerol is taken to the liver and adipose tissue, okay, by the chylomicron in the chylomicron form. Okay, what is the metabolism? That synthesis occurs in the intestine. Okay, this is the nascent chylomicron. Just see, okay, it has a triglyceride, it has a cholesterol present, B48 is present, and your A is present. B48 is present, and A is also present. Okay, it moves through the circulation and matures, taking out your ApoC2 and ApoE from your HDL. Okay, so HDL donates your ApoC, okay, and your ApoE to the chylomicron. So this is a mature chylomicron. B48 is there, okay, E is there, A is there, and C is there. Then mature chylomicron has ApoC2, which activates this C2. The C2 will activate a lipoprotein lipase enzyme, okay, and which converts a triglyceride to free fatty acid, and glycerol. Okay, and this free fatty acid is taken up by peripheral tissues, and this glycerol is taken up by the liver. The remnant, so it fulfilled its like action by transporting a triglyceride. Okay, and the remnants are taken up by your liver, okay, and acted upon by hepatic lipase. It is taken up by your liver. This is the remnant. Okay, this is the remnant. And the deficiency of lipoprotein lipase will cause a disorder which is called as your type 1 hyperlipoproteinemia. Okay, type 1 hyperlipoproteinemia. So this is the whole chylomicron metabolism. Let's talk about your VLDL, IDL and LDL metabolism. Okay, this VLDL contains APO, B100, C1, C2 and C3, and this LDL contains APO, B100 plus cholesterol only. Okay, so VLDL is synthesized in your liver. Okay, liver. Your chylomicron is being synthesized in your intestine, but VLDL is synthesized in your liver. Okay, this is a function for transporting endogenous triglyceride from liver to tissues, from liver to your tissues. Okay, understood. So this is the TAG and C. TAG and C are there. Okay, B100 is there. TAG, C and B100 is there here. Okay, this is matured by acquiring ApoC2 and ApoE from the HDL. Okay, HDL again gives you ApoE and ApoC2. Okay, and B100 is there, of course. This is acted upon by lipoprotein lipase. Okay, this lipoprotein lipase. Mature VLDL, which is this part. Okay, this is converted to IDL. Is converted to intermediate density lipoprotein, IDL. Okay, and lipoprotein lipase will cleave your ApoC2 and TAG. So your IDL will have B100. Okay, C will be there, E is there, and your and E is there. Okay, this is only your IDL. Okay, the C and C is cleaved, ApoC2 is cleaved, and your TAG is now cleaved. Okay, and this IDL, okay, if it also loses its ApoE, then only B100 and C are there, which is called as your LDL. Okay, so LDL will have only C present and only B100 present. Okay, this is your LDL. And LDL is up to LDL moves via the clathrin mediated transport. Okay, it binds to your clathrin proteins. Okay, this red proteins are your clathrin proteins. Okay, and it is going like this, forming an endosome and going inside the cell membrane. This is called as a receptor mediated endocytosis. Okay, and defect of this clathrins, if there is the defect of clathrins, okay, then you will suffer from a disorder which is a type 2 hyperlipoproteinemia. Okay, and this clathrin receptors are again recycled by and broken down by lysosomes into your cholesterol, which is stored in the tissue and plasma membrane. Okay, so this is the whole process. Disorders I have discussed. Disorders were first one where if there is abnormal ApoE, then another disorder is there, that is type 3 hyperlipoproteinemia. Okay, and one disorder I discussed that deficiency of lipoprotein lipase will cause a type 1 hyperlipoproteinemia. Okay, and type 3 will be caused by the abnormal ApoE. Okay, and here also type 2 will be caused by a due defect to the clathrin receptors, due to his LDL transport is altered, okay, or disturbed. HDL is a good cholesterol. Synthesis is in the liver and small intestine. Apoproteins, you have ApoA1, A2, C1, C2, C3, D and E. Okay, 30 to 60% proteins and lipid 40 to 90%. Okay, function is to take the cholesterol from tissues to the liver for detoxification. Okay, so it takes the tissue, it reduces the cholesterol level in the tissues. So it is also called as good cholesterol. Okay, so this is present in a discoidal form, discoidal HDL. Okay, this is esterified by your LCAT enzyme, okay, and this will form your spherical HDL, which is the mature form. Okay, this is present in a discoid form, okay, like this, and it becomes now a spherical form. Okay, and the cholesterol is transferred to the LDL and VLDL remnants. Okay, so it loses this cholesterol through cholesterol ester transferase protein, CETP. Okay, so CETP are transferring the cholesterol from HDL to LDL and VLDL via CETP, cholesterol ester transferase proteins, and the remnant is taken up by liver using your scavenger receptors. Okay, and LDL due to very low or deficiency of LDL will have these disorders, as LDL is a good cholesterol. Okay, so deficiency of LDL will cause diseases like a Tangier disease, your fish eye disease, and ApoA1 deficiency. Tangier disease, fish eye disease, or your ApoA1 deficiency. Talking about a fatty liver at last. Okay, this is reversible accumulation of triglyceride globules in your liver. Just note it is reversible, so it can be reversed. Okay, if you maintain a healthy lifestyle. Okay, types are alcoholic fatty liver and non-alcoholic fatty liver. So alcoholic, you have ethanol that will be causing by alcohol dehydrogenase will be converting to acetaldehyde, and acetaldehyde will be converting to acetic acid. In this step, your NADH is increased. So NADH you will convert your OAA to malate. Okay, this is due to more NADH. Okay, and OAA levels are decreased. So acetyl CoA cannot be metabolized. Okay, so acetyl CoA goes into a TG and cholesterol synthesis due to excess of acetyl CoA, and due to this triglyceride and cholesterol synthesis, your fatty liver is formed. Like a fatty liver first occurs, then it is invaded by your scar tissues invaded, okay, forming fibrosis of the lung. Okay, and this, if untreated for long period of time, will cause you lung cirrhosis. This is the alcoholic liver damage, non-alcoholic fatty liver. Okay, then you have the non-alcoholic fatty liver syndrome also, that is caused due to increased triglyceride in diet. You are taking a high fat diet, okay, or there is defect in the lipotropic factors. We will see the lipotropic factors here, okay, and the mnemonic there. Okay, so lipotropic, just let me tell you, lipotropic factors are the factors which increase mobilization of triglycerides from the liver, okay, from the liver. So if you are taking the defects in the lipotropic factors, so from there will be increased deposition of triglycerides in the liver, okay. And what are the things you can remember by College Board Veterinary Medical Institute, Gurgaon South, okay. So college is for choline, okay, board is for the betaine, okay, veterinary is for the vitamin B12 and vitamin E, medical for your methionine, I for inositol, okay, G for your glycine, okay, and S for your serine and selenium, okay. So what are the risk factors of fatty liver? You have obesity, high BP, diabetes mellitus, steroid therapy, and hepatic infections, which are causing your fatty liver. Chief complaints are your nausea, vomiting, hepatomegaly will occur, jaundice and weakness, pain in right upper abdominal region. Diagnosis, you can diagnose by gamma glutamyl transferase. Your specific for alcoholic fatty liver is gamma glutamyl transferase. We talked about a lot in today's session, okay. Your liver ultrasonography, liver USG, your CT scan, okay, and elevated lab diagnosis will be elevated levels of triglyceride, elevated cholesterol levels, okay, elevated LDL levels, and decreased HDL levels, okay. And how can you treat it? You can ask the patient to take a low fat diet, low calorie diet, okay, and exercise, and obesity must be decreased, and avoid the alcohol intake. So these are all about your lipid metabolism. We finish our lipid metabolism here, okay. Let's talk about some other chapter. Let's talk about the ELISA. It is your enzyme-linked immunosorbent assay for detection of your antigen or your antibody. ELISA sometimes is asked as a long question, but not very like frequently, but rarely it comes as a long question. Or you can get a short note on ELISA too. Okay, it depends on the antigen-antibody reaction. Okay, as it is immunosorbent. Okay, it uses your antigen-antibody reaction. Okay, and what are the antigens? Like protein, peptide, hormones, bacterial antigens, and viral antigens. Okay, and ELISA can be used for quantitative and qualitative analysis of antigen and antibody. Can be used for qualitative and quantitative analysis for antigen and antibody. Okay, so in immunoassays you can have three types: radioimmunoassay, your ELISA, and your CLIA, chemiluminescent immunoassay. Okay, RIA, okay, ELISA, and your CLIA. Okay. And what is the disadvantage of radioimmunoassay? Is that radiation risk will be there. Okay, and the reagents are like radioactive, so they have shorter lifespan. Okay, ELISA, it has no radiation risk. Okay, shelf life is also longer, and easy automation is there. Okay, so it is used for estimation of hormones like your T3, T4, and TSS. These are estimated by your ELISA. Okay, detection of your antibody against your HIV viruses. Okay, it's the HIV, and used for SARS-CoV-2 infection detection too. Okay, in SARS-CoV-2 infection detection too. What is CLIA? CLIA is the chemiluminescent immunoassay that is ultra sensitive, ultra specific, and easy automation is there. So it has partly replaced ELISA in few institutions. Okay, but now also ELISA is prevalent. Okay, so ELISA, what's the definition? It is the immunochemical method which implies the use of antigen antibody reaction in order to detect antigen or your antibody. Okay, understood? So this is called as your ELISA. And the antigen is first adsorbed on a solid phase support. Okay, this is like fixed to a surface, okay, by PVC or polystyrene. Like a solid phase support is a PVC or polystyrene surface. Okay, and this process is called as passive adsorption. Okay, like either you can fix your antigen like this, and directly capture your antibodies, or you can fix your antibodies, okay, which will indirectly capture your antigen. Okay, what are the enzymes involved? Okay, we have the enzymes like your, so we have enzymes like horseradish peroxidase, HRP, ALP, beta glucosidase, glucose oxidase, and ACHE. These are your enzymes utilized in ELISA. What are the linking substances? You can have glutaraldehyde, okay, or you can have periodate or melamide. 4 types of ELISA are there based on your detection. You have direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Okay, so in direct ELISA, if you attach your antigen to the PVC surface, okay, to which your primary antibody binds to. Okay, and this primary antibody is linked to your enzyme which will convert your substrate to colorful product. Okay, and this substrates, this may be tetramethyl benzidine, okay, or a amino salicylic acid, or OPD, or orthophenylene diamine, or a aminodiammonium salt, azino-diammonium salt. Okay, your TMB, amino salicylic acid, your OPD, or your azino-diammonium salt. You can just remember TMB, tetramethyl benzidine. Okay, Then indirect ELISA, where you fix the antigen, okay, and the primary antibody attaches to it, which is the antibody of interest, okay, and then secondary antibody, this also attached to your FC portion of your primary antibody, and the secondary antibody is now linked to your linked to your enzyme, okay. So here there is formation of more products. So you can see this causes amplification of your signals, and these are used to detect antibodies against the HIV. Indirect ELISA is employed. Then you have sandwich ELISA. So antigen is sandwiched between two antibodies, okay. First you have the captured antibodies, so you are fixing antibodies now, okay, and antigen of interest is fixed here, okay, and then the primary antibodies are attached here, okay. There is a sandwich formation, and this primary antibodies are again being recognized, and the secondary antibodies attached to the primary FC region of your primary antibodies, leading to even more production of products, okay. These are amplifying the signal to a very higher extent, okay. And okay, this is the and competitive like ELISA, you fix the small antigens, okay, okay. So competitive ELISA, talking about it is also called as the inhibition ELISA, to determine the concentration of T3 and T4. So what you do is you fix your antibodies like this, okay, and your serum antigens, you fix your serum antigens over the antibodies, okay, and your conjugated antigen, that it should be labeled with your enzyme, your conjugated antigen, okay. So as the conjugated antigen, they will attach to your serum antigens, okay, and these are also washed, okay. So concentration of T3 will be proportional to inversely proportional to the color intensity, okay, how much they are retained, it is dependent upon that, okay. So if you didn't understand, you can just go through the text, okay, as ELISA is a very small topic, so I am not wasting much of my time in it, okay. So this is about your competitive ELISA, okay. You can take a screenshot if you want, okay. So now let's move on to our next chapter, okay. Now let's learn about the nucleotide metabolism, okay. So this is also a metabolism which is given, okay. So you must be saturated by metabolisms now, so I am just covering the chapters like ELISA and the small chapters, okay, in between, so that you don't get saturated with all those enzymes and cofactors as well as the ATP and NADPH, okay. So let's read the nucleotide metabolism, then we'll go to a more interesting chapter, okay. I will teach you that. So nucleotides, what are nucleotides? You have nucleoside having the ribose sugar and the nitrogenous base, okay, and a phosphate group, okay. So nucleotide is a ribose sugar plus nitrogenous base plus phosphate group, okay, or it is called as a nucleoside monophosphate, okay, like adenine, adenosine, adenylate to 5'-AMP is the nucleotide, okay. Guanine, guanosine, guanylate 5'-GMP is the nucleotide, understood like that. What is the importance of nucleotides? Mnemonic is a chyme glycoregulation. This is a mnemonic, okay. Chyme, SCHIM is scheme, okay. Just you can remember scheme and glycoregulation, just a nonsense mnemonic, okay. Just remember. S is the synthesis of a DNA and RNA. C is the coenzymes, it is present in the coenzymes NAD plus, NADP plus, FAD, FMN, CoA. Hormone action, CAMP and CGMP, you must have knowing that it is utilized in the hormone action. Then I is the intermediates, okay. It generates metabolic intermediates like UDP glucose and your CDP acylglycerol, okay. Then methyl group donor, it acts as methyl group donor like your SAM, okay. E is energy for high energy compounds like ATP and GTP, okay. Glycom is important for your glycoprotein synthesis, okay, like UDP glucose and UDP galactose. And regulation means regulates the metabolic pathway. The CAMP regulates the metabolic pathway. Synthetic nucleotide analogs are there, which are producing by altering the heterocyclic ring, okay. You alter the heterocyclic ring and produce synthetic nucleotide analogs, okay, which are analogs to your purines, pyrimidines, and nucleotides and nucleosides, okay. So what are the synthetic nucleotide analogs? First, they act as anti-cancer agents like a 6-mercaptopurine, 5-fluorouracil, 6-azaguanine, 6-azacitidine, 6-azauridine, iodouracil, arabinosyl adenine, and arabinosyl cytidine, okay. This arabinosyl adenine is used in neurological diseases and viral encephalitis, okay. Then we have allopurinol drug, which inhibits the xanthine oxidase, which was converting a hypoxanthine to xanthine, and due to this inhibition, the xanthine can convert into uric acid, okay, and uric acid will decrease, okay. So it is used in a gout treatment. So allopurinol drug is your used in gout treatment, is a drug of choice of your gout treatment, okay. And allopurinol inhibits the xanthine oxidase. This is a competitive or suicide inhibition, okay. We talked about this in your enzymes part also today, okay. Then we have AZT, okay. AZT, which is called as zidovudine. This is a thymidine analogs, okay, which inhibit HIV reverse transcriptase and decreases the viral load. So it is AZT. Zidovudine is also used as an anti-cancer drug, okay. So this was all about your nucleotides, introduction to nucleotides, okay. Let's now move on to the nucleotide metabolism. Okay, so let's talk about the purine metabolism, purine nucleotides. Okay, so the sources of carbon and nitrogen are important. This is sometimes asked as a short note. Okay, so we have the purine ring like this, purine ring like this. Okay, so N1 is given by your aspartate, N1 is given by aspartate. Okay, and the carbon 2 is comes from N10 formyl THF, N10 formyl THF. Your carbon 3 your nitrogen 3 and your nitrogen 9. Okay, 3 and your 9. Okay, these are from your glutamine, 3 and 9 from your glutamine, 8 from your N5 and 10 methylene THF. Okay, 8 from your N5 and 10 methynyl THF, and this all. Okay, C5 C4 and your C5 C4 and your nitrogen 7. This is not carbon 7, nitrogen 7. Okay, this is from your glycine, this is from your glycine. Okay, so N1 comes from your aspartate. Okay, and N3 and N9 comes from your glutamine. Okay, N7 comes from your glycine. And if we talk about carbons, your C2 comes from your N10 formyl THF. Okay, and your C8 comes from your N5 and 10 methynyl THF. Okay, and this carbon 4 and 5 comes from your glycine. Okay, just remember this. Let's talk about de novo synthesis of your purine. Okay, de novo synthesis, this is also a long pathway. Okay, so you have to just practice, and there is a short pathway too. It is called the purine salvage pathway. Okay, and we'll talk about the degradation of purine nucleotides and about some disorders also. Okay, disorders also. So talking about your de novo synthesis. Okay, first ribose 5-phosphate is there, ribose 5-phosphate, which in the presence of PRPP synthase, ribose 5-phosphate gets converted into phosphoribosyl pyrophosphate. Okay, so ribose 5-phosphate in the presence of PRPP synthase reduces the phosphoribosyl pyrophosphate in the presence of your ATP. Okay, and then you have the enzyme that is your G80. Okay, G80 enzyme, and due to which it converts into phosphoribosylamine. Okay, the phosphoribosyl pyrophosphate converts into phosphoribosylamine in the presence of G80 and your glutamine. Okay, glutamine, understood? And this phosphoribosylamine, it's again converted into a glycinamide ribosyl 5-phosphate, in the presence of a synthetase enzyme. In the presence of your synthetase enzyme and the presence of a glycine and ATP, it is converted into a glycinamide ribosyl 5-phosphate. Okay, and this glycinamide ribosyl 5-phosphate in the presence of transferase, okay, and 5 and 10 methanyl THF, this is also present. Okay, and this glycinamide ribosyl 5-phosphate in the presence of transferase gets converted into formyl glycinamide ribosyl 5-phosphate. Okay, transferase will catalyze a formate group here. Okay, so formyl glycinamide ribosyl 5-phosphate. Okay, again synthetase is there. Okay, and glutamine and ATP are there. So again synthetase, and this synthetase will form formyl glycinamidine. Okay, your mide was there. Okay, midine ribosyl 5-phosphate again. Okay, again, and again synthetase will be there, ATP. Okay, this will form amino imidazole ribosyl 5-phosphate. Okay, amino imidazole ribosyl 5-phosphate, or your AIR. Okay, AR. Okay, so this formyl group, this formyl glycinamidine is converted to your amino imidazole. Okay, and ribosyl 5-phosphate is there constant. And this amino imidazole ribosyl 5-phosphate in the presence of a carboxylase enzyme, and a CO2 is added. This forms amino imidazole carboxylate ribosyl 5-phosphate. Okay, just CO2 is added and carboxylate becomes. Okay, and this amino imidazole carboxylate ribosyl 5-phosphate in the presence of synthetase again, and aspartate and ATP gets converted into amino imidazole succinyl carboxamide ribosyl 5-phosphate, this compound. Okay, so what has happened, this aspartate has been converted in succinyl. Okay, this has added a succinyl group here, and the synthetase has added a succinyl group here from the aspartate. Okay, and we form amino imidazole succinyl carboxamide ribosyl 5-phosphate. Okay, and this breaks down in the presence of a lyase. Okay, there are bond cleavage in the presence of a lyase in which fumarate is released, and you form amino imidazole carboxamide ribosyl 5-phosphate. This succinyl group is taken out. Okay, you form amino imidazole carboxamide ribosyl 5-phosphate. This amino imidazole carboxamide 5-phosphate again in the presence of transformylase enzyme. This is converted into your formamino imidazole carboxamide ribosyl 5-phosphate. Okay, this amino imidazole was there. Okay, this gets converted into your formamino imidazole. Okay, this amino is converting into your formamino. Okay, and formyl THF and transformylase enzyme. Okay, this formamino imidazole carboxamide ribosyl 5-phosphate. Again, there is cyclization. Okay, cyclase enzyme will act. Okay, and H2O will be released, and you form your IMP. Your iron monophosphate. Okay, this IMP in the presence of your synthetase enzyme, Okay, this will form your adenyl succinate. Then you have adenyl succinase enzyme, which will break down it and release your fumarate. Okay, and produce your AMP. AMP acts in the presence of kinase to give your ADP, and ADP in the presence of kinase again gives your ATP. Okay, and this IMP in the presence of your IMP dehydrogenase, okay, gives your xanthosine monophosphate. And this xanthosine monophosphate in the presence of your synthetase again will give you GMP. Okay, and this GMP has inhibitory action of IMP dehydrogenase. Okay, so dehydrogenase, okay, where NAD plus is changed into NADS plus S plus, okay, and this will give you xanthosine monophosphate. This xanthosine monophosphate in the presence of synthetase will give you GMP. This GMP in the presence of kinase will give you GDP, okay, and this GDP in the presence of kinase will give you GTP, okay. And ribonucleotide ADP and GDP, these are your ribonucleotides, okay, and enzyme will be ribonucleotide reductase enzyme, okay, and NADPH, which will change it into ribonucleotides, the ADP and GDP to deoxyribonucleotides, okay, so you can synthesize the deoxyribonucleotides like this. Understood? So this was the whole pathway, okay. So we have some important things like a PRPP synthase. This is inhibited by your ADP, okay, activated by your GDP, okay, and this GAT enzyme, this is inhibited by your IMP, AMP and GMP, okay. Understood? And another thing is to note that in 3, 5, 6 and 8 steps, in 3, 5, 6 and 8 steps, okay, in this steps your synthetase enzyme is used, okay, in this steps your synthetase enzyme is used, okay. You can just note your 3, okay, and your 5, okay, and 6 and again 8 in the main pathway, okay, and the subsequent pathway, yeah, from IMP, okay, you again use your synthetase, and in your production of your GMP or GTP, okay. Now from IMP you again use your synthetase here, okay, xanthosine monophosphate to GMP, okay, synthetase is used, okay. This you have understood. So this is a de novo synthesis. And what are the inhibitors of purine synthesis? So you can have inhibitors like a sulfonamide, methotrexate drug, 6-mercaptopurine, azathioprine, and a mycophenolic acid. So the sulfonamides, methotrexate, 6-mercaptopurine, where azathioprine, and a mycophenolic acid. These are your inhibitors of your purine synthesis, okay. Then we'll talk about purine salvage pathway, okay. This pathway is very long. Purine salvage pathway is a shortcut pathway, okay, and it is used by your brain and RBCs for nucleotide synthesis, okay. So nucleotide synthesis is from your degraded DNA molecules, okay. Just let's see how it is occurring, okay. You have the substrates like your adenine, your hypoxanthine, your unusual base hypoxanthine, and your guanine, okay. So you have the degraded DNA molecule giving your adenine, hypoxanthine, and adenine, okay, guanine, hypoxanthine, adenine, okay. So in adenine, okay, enzyme will act APRT. What is APRT? Your adenine phosphoribosyl transferase, APRT will change your PRPP into PPI, and form your AMP. Okay, such hypoxanthine will by the enzyme presence HGPRTase is the hypoxanthine, okay, guanine, okay, hypoxanthine guanine phosphoribosyl transferase, okay, hypoxanthine guanine phosphoribosyl transferase, PRPP to PPI. This will also give your IMP. And this guanine in the presence of your HGPRT, the same enzyme as this, okay, this will also give you GMP, okay. So you can form AMP, IMP and GMP from this all, okay. And then from your IMP, AMP and GMP, from your AMP you put kinase to form ADP, and from kinase to put ATP, okay, and GMP you can put kinase to get GDP, and again kinase to get GTP, okay. So you can form any of them, okay. Understood? So after this there is a deficiency, okay, important clinical case also, HDPRTase deficiency here in both the cases will lead to your Leschnian syndrome, okay, Leschnian syndrome, which affects males only, okay. This syndrome affects males only, it is X-linked, okay, and affects males only. Clinical features are that the patient will be aggressive, okay, and it also self-mutilating activity, like the patient tries to harm himself, okay, and there will be mental retardation and hyperuricemia, okay. So you can see this self-mutilating activity, the patient tries to constantly bites his fingers to meet his protein demands, okay. And self-mutilating activity like scratches and pulling of hair, and this all are seen, okay. Self-mutilating activity is important in case of Leschnian syndrome, okay. And hyperuricemia, because there is decreased HGPRTase, okay, due to which there is increased PRPP, and increased PRPP will favor your de novo synthesis, forming your uric acid, okay. You can see from a de novo synthesis, okay, here in the your purine salvage pathway and all, okay, the uric acid concentration is increased, okay. So you have hyperuricemia, okay, and this hypoxanthine and the guanine, okay, gets converted into your uric acid, okay. And there is gout also. So how you can treat it? Allopurinol, which is inhibitor xanthine oxidase, and you can treat the hyperuricemia by this, okay. So the main thing is the hyperuricemia only, so you have to treat find the drug for the hyperuricemia and give allopurinol to the patient, okay. Then how the purine nucleotides are degraded, so uric acid, and just I was saying that uric acid was a product of your purine nucleotide degradation only, like your AMP is converted by nucleotidase. AMP is a nucleotidase to adenosine, it is a nucleoside now, okay. And this IMP to your inosine, okay. And GMP is directly converted by the nucleotidase, okay. This is not nucleosidase, this is nucleotidase only, I have made a mistake here, This is not nucleosidase, this is your nucleotidase only. Okay, so GMP is converted into your guanosine. This is converted into your guanosine. Okay, and how your H2O is converted into your PI in this process. Okay, you can remember this. And AMP can be converted into your IMP by AMP deaminase enzyme. Okay, AMP deaminase. So AMP can be converted into your IMP. Okay, and this IMP can form your inosine. And this adenosine also can form your inosine by adenosine deaminase, and your inosine. Okay, this nucleoside phosphorylase will phosphorylate, will cleave the phosphate group, okay, and form hypoxanthine. Okay, this step is important. Your hypoxanthine in the presence of xanthine oxidase, will form your xanthine. Okay, and this xanthine in the presence of xanthine oxidase again will form your uric acid. So uric acid is the final product of your purine nucleotide degradation pathway. Okay, and what about GMP? GMP is converted by nucleotidase to guanosine. Okay, it is not interconvertible into AMP or IMP. Okay, then this guanosine changes into guanine. Okay, where the PI changes into ribose 1 phosphate. Okay, and this guanine enters the pathway by converting itself into xanthine. Okay, and again the pathway xanthine to xanthine oxidase uric acid. Okay, this is the purine nucleotide degradation, which is easy pathway as compared to your de novo synthesis. So I will suggest for de novo synthesis, make a flowchart. Okay, make the flowchart and stick in your table, because the pathway needs regular revision. Otherwise you will forget the whole pathway, and once you forget one intermediate, the whole pathway you will forget. Okay, so that important things are there. Okay, so you can just stick a chart for this. Okay, talking about the disorders, we have gout to discuss. Okay, gout is important, which is your increased uric acid concentration. Okay, due to the monosodium urate crystals are deposited in your joints. Okay, and the most common joint affected is the metatarsophalangeal joint. Just look at this. Okay, in 90% of cases, the metatarsophalangeal joint is affected. Okay, and other sites, maybe knee joint, ankle joint, and wrist joint, this can also get inflamed. Okay, due to the monosodium urate crystal deposition. Okay. After this, what are the symptoms? You have swollen, painful joints. Okay, excruciating pain. Podagra gout is where great toe is involved. This is a podagra gout. Okay, and you can have renal stones also. Normal serum uric acid level in males, it is 5 to 7 milligram per deciliter. In females, it is 2 to 5 milligram per deciliter. Okay, if it exceeds 7, the uric acid deposits in joints and renal tubules. Okay, if it deposits in joints, it can lead to your this podagra gout and swollen, painful joints and all. And if it deposits in your renal tubules, it can lead to your kidney stones. Okay, your renal stones. So renal stones is also a symptom of your gout. What are the causes? You can have primary causes of gout. Okay, then secondary causes. Based on this, you can tell it as primary gout or secondary gout. Under the primary causes, increased PRPP. Okay, what happens due to increased PRPP? Due to this, your purine synthesis increases. Okay, your purine nucleotide synthesis will increase. Okay, and more synthesis and more degradation. More degradation means your more formation of uric acid. Okay, then increased GAT, another the second enzyme in your purine nucleotide synthesis, increased GRT or HGPRTase deficiency. Okay, so HGPRTase deficiency will cause the formation of your gout. Okay, so decreased HGPRTase deficiency. Okay, so what happens is that let's see the pathway. Okay, if you decrease the HGPRTase, your PRPP will increase, and the PRPP will accelerate your de novo synthesis. Okay, causing the production of your more uric acids. Okay, and increased glutathione reductase is also a primary cause. Secondary cause, you can have lymphoma, leukemia, psoriasis, renal failure, and chemotherapy. Okay, lymphoma, leukemia, psoriasis, renal failure, and chemotherapy. How can you treat the gout? By allopurinol, which will inhibit your xanthine oxidase by suicide inhibition. This is a case of your suicide inhibition. Okay, and you can also treat by NSAID drugs, non-steroidal anti-inflammatory drugs. Okay, and what is the lab diagnosis of gout? It is that your synovial fluid aspiration of affected joint must be there. Okay, and what is the most definitive test is the examination of negative birefringent monosodium urate crystals under the microscope. Okay, examination of birefringent. Okay, negative birefringent monosodium urate crystals under microscope. This is a definitive test of your gout. Okay, and uric acid level increases also indicating your gout more than 7 milligram per deciliter. Okay, and this alcohol, this ethanol will increase your gout attack. How? Okay, ethanol converts into acetaldehyde and acetic acid. We have discussed this pathway millions of times. Okay, in which NADH is produced. This increased NADH will cause the monosodium urate crystals, which are in soluble form, to form your uric acid, which are insoluble. Okay, this monosodium urate crystals to form uric acid, and this uric acid will deposit in the gout. Okay, precipitating the gout attack. So alcohol will precipitate your gout attack. will increase your gout attack. We have discussed about the Lesnian syndrome in the self-mutilating activity, and HDPR test deficiency was there. Okay, then you can have severe combined immunodeficiency disorder, your SCID. Okay, due to the deficiency of your adenosine deaminase. Okay, and you know that adenosine deaminase was involved in your purine nucleotide degeneration, or this adenosine deaminase, due to which your adenosine will tend to accumulate and inosine won't be formed. Okay. So you will see decreased uric acid level in this case, and purine nucleoside phosphorylase PNP deficiency, where there will be decreased uric acid level. Okay, this is also immune disorder. Okay, so this was about your purine metabolism. Let's talk about your pyrimidine metabolism now. Okay, talking about your pyrimidine metabolism. So you can have pyrimidines like a TMP, CMP, and a UMP. Okay, your pyrimidines are your thymine, cytosine, uracil. Okay, forming a TMP, CMP, and UMP. Okay, so pyrimidines have only one ring. Okay, in contrast to your purines, which have two rings. Okay, what are the sources of this carbon and nitrogen atoms? So first talking about an N1. Okay, this N1, C5, C6, and your C4. Okay, C4, 5, 6, and a nitrogen 1. These are contributed by your aspartate. And the CO2 contributes to the C2, and glutamine forms the N3. Okay, so what is the process of synthesis of your pyrimidine, that metabolism? What is the de novo synthesis of pyrimidine? Okay, then we'll see about the degradation procedures. Okay, about synthesis, CO2 and glutamine combine, your glutamine and CO2 combine in the presence of CPS2. Okay, CPS2 is a carbamoyl phosphate synthetase 2 enzyme. Okay, and 2 ATP. This gets converted into a carbamoyl phosphate. This carbamoyl phosphate in the presence of a aspartate trans carbamylase, aspartate trans carbamylase, okay, gets converted into a carbamoyl aspartate. Okay, while aspartate is changed to your PI. Okay, this is added to this carbamoyl part. Okay, and this carbamoyl aspartate in the presence of a dihydroorotate. Okay, dihydroorotate, where water is removed. Okay, H2O is removed. This forms a dihydroorate. And this dihydroorate, this is an important step, please focus. This dihydroorate in the presence of a dihydroorate dehydrogenase is converted into your orotate. Okay, orotate. So conversion of dihydroorate and orotate, it is there, where NAD plus is changed into NADS plus H plus. Okay, and your orotate in the presence of your OPRtase. This step is also important. Your OPRtase. Okay, orotate in the presence of your OPRtase, where the PRPP is changed to your PPI. Okay, this orotate is changed to your orotatase monophosphate OMP. Okay, orotate is changed into your OMP. Okay, by OPRtase enzyme. And this again acted upon by OMP decarboxylase. You can have decarboxylation of this OMP. Okay, and form your uridine monophosphate UMP. Okay, this OMP in the presence of your OMP decarboxylase forms your UMP. Okay, and this UMP in the presence of your kinase forms your UDP. Okay, this UMP in the presence of a kinase forms your UDP. Okay, and this UDP in the presence of again kinase. Okay, again kinase forms your UTP. Okay, and UTP in the presence of a CTP synthase can form your CTP. Okay, and this UDP in the presence of your NADPH ribonucleotide reductase, this forms a DUDP. And this in DUDP, it can your addition of water can occur and form your DUMP. And this DUMP in this, your thymidylate synthase enzyme may be like involved. Okay, and this DUMP is converted to your DTMP. Okay, what is the method of degradation of this? Okay, this CMP is degraded by nucleotidase to cytidine, this UMP to uridine. Okay, and this DTMP to your thymidine. Okay, and this cytidine and uridine, these are interconvertible by your deaminase. This cytidine, okay, there is a deamination of a cytidine. Okay, you will form uridine. Okay, and this uridine and your thymidine that is formed. Okay, these are acted upon by phosphorylase enzyme to convert it into uracil and thymine respectively. Okay, this uridine is converted into uracil and thymine is converted into your thymine. Thymidine is converted into your thymine by phosphorylase enzyme. Okay, and this uracil again from dihydrouracil, this thymine will form dihydrothymine, this dihydrouracil will form beta alanine, this dihydrothymine will form beta amino isobutyrate. Okay, and this beta alanine will form your acetyl CoA, and this beta amino isobutyrate will form a succinyl CoA. So acetyl CoA and succinyl CoA, these are the end products of your pyrimidine metabolism and pyrimidine degradation. Okay, talking about orotic aciduria. What is orotic aciduria? It is an autosomal recessive disorder. Okay, if you have this enzyme deficit here, OPRtase. Okay, OPRtase enzyme deficiency. Okay, so orotate phosphoribosyl transferase, that is OPRtase enzyme deficiency. Okay, and your OMP decarboxylase deficiency also, this can induce your OMP decarboxylase, OPRtase and your OMP decarboxylase. If both deficiencies are there, you will have orotic aciduria. Okay, where the orotic acid crystals are accumulated. Okay, and these are excreted out in urine. Okay, and if a megaloblastic anemia will be there and immunodeficiency will be there. You can treat by giving an oral uridine. Okay, oral uridine can be salvaged to form a UMP, and by this UMP can form all other pyrimidines by the process. Okay, So this was about your pyrimidine metabolism and your whole nucleotide metabolism. Okay, now let's move on to another chapter. After your purine and pyrimidine metabolism, let's go on to your biological oxidation, which occurs in your inner mitochondrial membrane. Okay, you have the complex one, which is your NAD-CoQ oxidoreductase complex. Okay, so what happens? Your NAD H plus, this converts into your NAD plus. Okay, and your electron is given to your FMN, due to which it changes into your FMNH2, and then the electron goes to your FES. Okay, FES gives the electron to CoQ. Okay, and this CoQ will change into your QH2. Okay, so this is the first procedure. Okay, this one. And your complex 2 is a succinate CoQ reductase or a succinate dehydrogenase complex. Okay, so there is succinate to fumarate conversion, due to which the electron released is accepted by your FAD. Okay, from FAD it goes to FADH2, and from FADH2 to CoQ. The CoQ again changes into your QH2. Okay, so electron is coming from FES also, and electron is also coming from your FADH2. Okay, to the CoQ, due to which the CoQ is changing into your QH2. Okay, and then from complex 3 is your CoQ cytochrome C oxidoreductase complex. Okay, due to which the CoQ, okay, it is giving electron to a cytochrome B. Cytochrome B is giving to your FES to the cytochrome C1, and again to the cytochrome C. What is the sequence? Your B, FES, C1 and the C. Okay, and then your complex 4 is there. Complex 4 is the cytochrome C oxidase complex. Okay, due to which the electron from your cytochrome C is given to a cytochrome AA3. Okay, and from the cytochrome AA3, okay, the electron goes here, and the half O2 plus 2H plus, this half O2 plus 2H plus. Okay, this gain the electron to change into H2O. Okay, so these are the things that are occurring. Okay, and this is the whole structure. You can take a screenshot of this first, then I am explaining it. Done. Okay, so what happens? This is a complex 1, this is a complex 1. So what happens? Your NADH plus H plus is converted to your NAD plus. Okay, electron goes to the FMN. From FMN it goes to the FMNH2, from FMNH2 to the FES, and from FES to the CoQ. Okay, and by this your 4H plus is getting out to the mitochondrial lumen. Okay, this is the inner mitochondrial membrane, it's the outer mitochondrial membrane. Okay, so it is going it is your inner one and it's the outer one. Okay, and the electron is going towards the lumen. It is the H plus is going towards your lumen. This is the lumen, this must be the lumen of your mitochondrial matrix. Okay, and complex 2 is there, where succinate to fumarate conversion will take place. Okay, the electron will move to a FAD, which will convert into FADH2, from FADH2 to FES. Okay, and from FES again to your CoQ, from FES again to the CoQ. Okay, and from CoQ the electrons will move down like this. Okay, and the electron acceptor will be cytochrome B. Okay, cytochrome B, and cytochrome B will give your electron to the FES, and by FES it gives the electron to a cytochrome C1. Okay, from cytochrome C1 it moves to a cytochrome C. Cytochrome C is the mobile electron carrier. It is the mobile electron carrier. Okay, in this procedure 4H plus go due to the proton gradient created. Okay, due to the proton gradient 4H plus move like this. Okay, and from the cytochrome C it is accepted by a cytochrome AA3. Okay, cytochrome AA3. Okay, due to which your half the electron is given, so due to which your half O2 plus 2H plus gets your electron and converts it into your H2O, and in this process 2H plus go into this membrane. Okay, into the lumen. Okay, so complex 5 is ATP synthase. We will discuss the ATP synthase separately. Okay, so you can now appreciate the movement of your electrons. Okay, there are few inhibitors of your electron transport chain, that is a rotenone and barbiturates. This will inhibit your complex 1. Rotenone and barbiturates inhibiting the complex 1. Your malonate and carboxyl will inhibit your complex 2. Okay, malonate and carboxyl inhibiting the complex 2. You know this succinate dehydrogenase. This is succinate dehydrogenase complex now. Okay, and malonate is inhibiting a succinate dehydrogenase. Remember. Okay, so this is the same complex as you read in your Krebs cycle. Okay, so try to correlate. And then this complex 3 is inhibited by antimycin. This antimycin will inhibit your complex 3. Okay, and H2S, azide, CO and Cn minus, this all will inhibit your complex 4, or will inhibit your complex 4. Okay, what is PO ratio? PO ratio is the amount of inorganic phosphate consumed for ATP production per molecule of OTP consumed, per molecule of O2 consumed. Okay, this is the NADH. It has PO ratio of 2.5. Okay, so NADH it is giving a 2.5 ATP and FADH2 is giving a 1.5 ATP. Talking about ATP synthase, which is comprised of two parts, your F0 and your F1 particle. Okay, which is also called as a Fernandez-Morans particle. I should write here. Okay, if it is not written, your Fernandez-Morans, particles. Okay, which are present over the cristae, of the mitochondria, present over them. Okay, so this F0 F1 particles or ATP synthase, okay, this is the smallest motor, and proposed by the Paul Boyer. Okay, and this synthesizes the ATP. Okay, so this is the structure of ATP synthase, where we have an F1 subunit, okay, and the F0 subunit is embedded in the inner mitochondrial membrane. Okay, we have the F0 subunit like this. Okay, F0 subunit, which is inhibited again by your oligomycin. Oligomycin inhibits your F0 subunit. Remember, oligomycin inhibits your F0 subunit. And this F1 subunit has a gamma handle. Okay, the handle, this tail-like structure is called as a gamma handle. Okay, and we have 3 alpha and 3 beta subunits. So we have 6 subunits like 3 alpha and besides them 3 beta. Okay, this is a structure of your ATP synthase. Let's talk about a chemiosmotic hypothesis. What is the chemiosmotic hypothesis? If there is electron movement across the complexes, so this will cause your H plus ions to move out. Like a 4H plus from complex 1, 4H plus from complex 3, and 2H plus from complex 4. And this movement of H plus will cause a proton gradient. Okay, and this proton gradient, if H plus moves to F0 subunit. Okay, how the ATP synthase works, let me talk about this. Okay, when the H plus moves through the F0 subunit, like when the ATP synthase moves through the F0 subunit like this in this subunit, okay, it causes rotation of a gamma handle. This will rotate. Okay, due to movement of electron like this gamma handle will rotate, and due to this rotation of the gamma handle, okay, there will be conformational change in the beta subunit. Okay, there will be conformational change in the beta subunit, and this conformational change, like your beta subunit has 3 conformations. One is open, okay, one is a loose, and one is a tight. Okay, so these are 3 conformations. This conformational change will lead to ATP formation. Okay, this is called as a binding change mechanism. Okay, so you can be asked a short note on your binding change mechanism or a binding change mechanism. Okay, so what is this mechanism? If the conformational change is open, okay, this will release the ATP. Okay, and loose means binding of ADP and PI. Okay, and tight means the synthesis of ATP. So first there will be loose conformation where the ADP and PI will bind. Okay, then will be tight conformation to synthesize the ATP. Okay, and then be O conformation to release the ATP. So what is the sequence? Your loose will be there, then tight will be there, then open will be there. This will be a sequence of binding, and after this your ATP will be released. Okay, understood? This is your chemiosmotic hypothesis. And this proton gradient is disrupted by your valinomycin and gramicidin. This valinomycin and gramicidin disrupt the proton gradient created by your H plus. Okay, understood? Next, we have to talk about uncouplers. What are uncouplers? We will see. You can get a short note on uncouplers too. Okay, these are the substances, the physical or chemical substances, which can increase the permeability of your inner mitochondrial membrane. Okay, due to which your oxidative phosphorylation, this one, your H plus synthesis. Okay, your ATP synthesis. Okay, this will be delinked from your ETC. Okay, ETC will occur at its rate, and your oxidative photophosphorylation will occur at other rate. Okay, so there will be production of heat in place of production of ATP. The production of heat due to excess accumulation of ATP. Okay, and there will be no oxidative phosphorylation. So there will be production of very heat production will be there in the body. Okay, what are the types of your uncouplers? So we have physical uncouplers and chemical uncouplers. Physical, you have like thyroxine, you have long chain free fatty acid, unconjugated bilirubin, and thermogenin. Thyroxine, long chain fatty acid, unconjugated bilirubin, and thermogenin, a brown fat in newborn. Chemical like uncouplers, like a 2,4-dinitrophenol, okay, 2,4-dinitrocresol, your calcium, dicumarol, pentachlorophenol, and aspirin. Okay, calcium, dicumarol, pentachlorophenol, aspirin, 2,4-DNP and 2,4-DNC. Okay, these are your chemical uncouplers. Talking about high energy compound, what are high energy compounds? These are the compounds which on hydrolysis give minus 7 kilocalorie per mole or more energy. Okay, this give minus 7 kilocalorie per mole or more, even more than this energy. Okay, what are the high energy compounds? You can get an short note on high energy compounds. Okay, you can just define this and write all of the examples. Okay, all the examples. So all the examples, your PPI is there, your ATP, GTP, CTP, and UDP glucose, your acyl CoA, your acetyl CoA, your succinyl CoA, your creatine phosphate, your PEP, acyl phosphate, carbamoyl phosphate, and your BPGA, biphosphoglyceric acid. These all are your high energy compounds. On hydrolysis of this will give you more than 7 kilocalorie per mole of energy. Okay, what is SLP, or substrate level phosphorylation? This is the synthesis of ATP without electron transport chain. Just by hydrolysis of your high energy compound. Okay, so in SLP or high energy compounds, the high energy compounds will be utilized for your ATP synthesis. Okay, electron transport chain won't be involved. Your ATP synthesis will occur just by hydrolysis of your high energy compound. Remember, we talked about your SLP steps in your glycolysis. Yes. Yes, succinyl CoA in the presence of thiokinase gives you succinate, okay, where ATP synthesis is there. 1,3-bisphosphoglycerate, this is in the presence of PGK, okay, your ATP synthesis, your phosphoglycerate is synthesized, okay, in the presence of kinase enzyme, okay. And another example is a PEP. PEP in the presence of your pyruvate kinase is forming a pyruvate, PEP to pyruvate, okay. This was also, this both are your SLP steps in your glycolysis. Understood? Next we have some subtle pathways, okay, subtle pathways, because your malate, okay, because your aspartate, specifically your aspartate, your malate aspartate subtle is about your aspartate. This aspartate, it can't cross, okay, it can't cross from cytosol to your mitochondria, okay. Aspartate can't cross directly, so it is first converted into your malate, and malate process again gives you aspartate, okay, which can come back, okay. So what happens is that your aspartate is changed in a transamination reaction by AST, okay. AST converts it into OAA, okay, oxaloacetic acid. We saw the transamination reactions, yes. And this OAA in the presence of a malate dehydrogenase will give you a malate, okay. And here NADH plus H plus is changed to NAD plus, okay. This malate will go into your mitochondria, This malate will again convert into your OAA, the malate dehydrogenase, where NAD plus is again transferred to your NADH plus H plus, the reverse step, okay. This OAA in the presence of your AST is converted into your aspartate, and this can like go back to the cytosol, okay. So this is called as a malate aspartate subtle, okay, malate aspartate subtle. Understood? And this is present in your heart, liver and kidneys. About your glycerol 3-phosphate subtle, which is present in your brain and skeletal muscles, okay. Malate aspartate in the heart, liver, kidneys, and glycerol 3-phosphate subtle in the brain and skeletal muscles, okay. So cytosol and mitochondria we have. So first of all, your DHAP is converted into glycerol 3-phosphate, by the enzyme glycerol 3-phosphate dehydrogenase, okay, where NADH plus H plus is changed into your NAD plus. This glycerol 3-phosphate can cross the membrane, okay, and form here it can be back converted into your DHAP. It can be converted into DHAP again, okay. And this DHAP in this step, the enzymes used will be glycerol 3-phosphate dehydrogenase only, okay. And here FAD is converted to your FADH2. So that is the generation of 1.5 ATP here, okay. There will be generation of 1.5 ATP here. And in malate aspartate subtle, what happens? Your NADH plus H plus is there, so the generation of a 2.5 ATP will be here, okay. And if you just take the whole reaction, then the whole ATP generated will be 0 and 0 here. But we write malate aspartate subtle that generates 2.5 ATP, okay. And your glycerol 3-phosphate subtle will generate your 1.5 ATP, okay. These are your subtle pathways. What are the enzymes involved in ATS? All are your oxidoreductases, okay. So I have different classes of oxidoreductases, like oxidases enzymes are there, like cytochrome oxidase, okay. Oxidases are removal of this hydrogen is removed and accepted by oxygen and water formed, okay. Oxidases are generally water forming. The cytochrome oxidase is an example. And the dehydrogenase is also present, which will transfer your H from one substrate to another, like a succinate dehydrogenase, glycerol 3-phosphate dehydrogenase, lactate dehydrogenase, acyl CoA dehydrogenase, write a host of examples, okay. And then your hydroperoxidases are there, like your peroxidase and your catalase, okay. Peroxidase, you know what is the action of peroxidase, and catalase, you know, H2O to H2O plus O2. And oxygenases, like your monooxygenases, like your phenyl hydroxylases, and dioxygenases, like your homoxanthinic acid oxidase, and your tryptophan pyrrolase, these are dioxygenases, okay. So basically this was a part of enzyme which I had left, okay. So I am teaching it here in biological oxidation, so you can just relate, okay, what is happening, okay. So after this biological oxidation, let's move on to another chapter. So just to see what we have completed, okay. So we have completed our lipid chemistry now, and biological oxidation, and nucleotide metabolism also, okay. So we have the vitamins left, and this small chapters that are left, okay. Vitamins and the small chapters, okay. So let's start with the immunity part now, okay. Let's start with the immunity part. Yeah, so let's read our immunology, okay. So immune system protects us from infections and foreign substances, you know all these things, okay. Internal degradation of pathogens, okay. B cells are producing antibodies, T cells are destroying infected cells, self non-self discrimination, gut flora maintenance, these all are functions of the immune system, okay. You have the primary immune organs, your secondary immune organs. Your primary are central immune system organs, called as your thymus and the bone marrow. This thymus and bone marrow, these are your primary organs, okay. Secondary are peripheral immune organs, like your spleen, lymph nodes, and your gall and your malt is there, okay. Gut associated, mucosal associated lymphoid tissue and all, or Pierce patches and all. Tertiary lymphoid structures, these are your abnormal structures developing in the peripheral organs. These are abnormal, okay. So primary and secondary are physiological, while tertiary is abnormal. Then we have thymus. Thymus have cortex and a medulla. This is the thymus gland. Just see the thymus gland present above heart and cortex. Immature T cells are present in the cortex. Okay, so the thymocytes migrate from the bone marrow to the cortex of the thymus gland. Okay, where the immature thymocytes are converted into a mature thymocytes. Okay, they acquire some receptors called as the TCRs. Okay, they acquire this type of spring type of receptors called as the TCR, your T cell receptors. Okay, this is called as the maturation of your T lymphocytes. Okay, so talking about the your cortex. Okay, so this is a cortex having cortical epithelial cells, and nerve cells are also present for providing nutrition to the thymocytes. Okay, the T lymphocytes. Okay, and your medullary epithelial cells are present in the medulla part. Okay, and as age increases, the structure and function of the immune system of your thymus gland, this decreases. Okay, your DJOS syndrome is there. Your DJOS syndrome, it is a congenital defect in the development of thymus. Okay, congenitally there is defect in the development of thymus. Okay, due to the deletion of long arm of chromosome number 22. Due to the deletion of long arm chromosome number 22. Okay, your T cell count decreases in this, and cell mediated immunity is lost. Okay, then talking about bone marrow, it is a hematopoietic cell generation organ. Okay, where the proliferation and differentiation of B cells occur. Okay, B lymphocytes occur, and this also secretes cytokines. Talking about lymph nodes, lymph nodes are bean shaped nodular structures which have an outer cortex, which have a middle layer, and an inner medulla. Okay, outer cortex, middle layer, and inner medulla. Outer cortex has B lymphocyte, macrophage, and dendritic cells. Okay, and the middle layer, the paracortex, has your T lymphocytes, and the inner medulla, has a plasma cells which will provide antibodies. Okay, so this is about your lymph nodes. Okay, and we have some cells like your antigen presenting cells. What are these? These are your dendritic cells, your B cells, and your macrophages. Okay, which present, this can trap antigens with association with MHC, your major histocompatibility complex. Okay, this will stimulate your T cells, and T cell will stimulate your B cells. Okay, so MHCs have a role, your APCs have a role in B cell, in your antibody production too. So we'll see about that. Just lymphoid follicles. What are lymphoid follicles? So primary follicle and secondary follicle. Primary follicle beta cell aggregates, and secondary follicle have a germinal center. You might have seen in your histology classes. Okay, where you can where you draw the germinal center. Okay, these are the light staining organs, light staining parts of your lymphoid follicle. Okay, and the secondary follicles are generated only after exposure to your immunogen. Okay, and this contain B cells as well as the plasma cells. Talking about spleen. Okay, spleen is a ovoid structure which is present in the left side of your abdominal cavity. You must have read about these anatomical things in your anatomy classes. The spleen has red pulp and a white pulp. Red pulp, in its red pulp, venous sinuses are present. Okay, in which the old and defective RBCs are removed. Okay, and this in this venous sinuses you can have macrophages, which are a part of a reticular endothelial system, which will remove these dead cells and the RBCs. Spleen is also called as the graveyard of the RBC. Okay, where the RBCs are destroyed after 120 days. Okay, then lymphocytes are present in the red pulp, granulocytes, erythrocytes, and platelets are also present. Okay, and this white pulp, this is located along the branches of a splenic artery. Okay, and in white pulp your pulse is present. What is pulse? Your periarterial lymphatic sheath is present, which is T cell rich. Okay, and this also are helpful in the presentation of antigen to the T cell. Cells of immune system. What are the cells? You have B lymphocytes, your B lymphocyte B cells, or you can say your CD19 cells or the CD20 cells. Okay, these are many names: CD19, CD20, or a B cells or a B lymphocytes, whatever you may call. This develop from your bone marrow, and this comprises of 20% of your lymphocytes. This produce antibody. Antibody production is the main function of your B cells. Okay, B cells in circulation, okay, produce your IgM and IgD. While in your tissues, they produce G, E, and A, IgG, IgE, and IgA. And human B cell markers, these are important: your CD19, your CD20, and your CD21. Okay, these are your human B cells markers. What are these CDs? We will talk about this. Then about T lymphocyte. T lymphocyte are T cells or your CD3 cells. You can call them CD3 cells. This develop from your bone marrow only. Okay, but mature in thymus. Okay, in this B cells, B stand for bursa. Okay, in your birds. Okay, the B cells were first isolated from birds. Okay, and this was produced by the primary lymphoid organ of the birds, that was called as a bursa of Fabricius. Okay, and so it is in humans also, it is named as your B cells. Okay, and this T cells, this mature in your thymus, that's why they are named as T lymphocytes or T cells or your CD3 cells. This T cells may be of three types: your killer cells or your cytotoxic T cells, which are also called as your CD8 plus T cells, your T helper cells, which are called as your CD plus 4 T cells, CD4 plus T cells. Okay, and the regulatory T cells, which are also called as Treg cells, Treg cells. Okay, these killer T cells, these are mainly destroying the pathogens, okay, by forming your pores in the cell membrane of the pathogen. Okay, this provide anti-tumor immunity. These T helper cells, these are secreting some substances like your IL-2, interleukin-2, gamma interferons, TNF alpha, and your IL-4, 5, 10, and 13, which are secreted by your T helper cells. Okay, and this IL-2 gamma interferons and the TNF alpha, they stimulate your NK cells, your natural killer cells, and your TK cells. Okay, this substances will stimulate your TK cells. So these are basically helping the killer T cells to destroy the pathogen. Okay, T helper cells may be of two types: TH17 cells and the TFH cells. Okay, TH17 cells and TFH cells. Okay, this TH17 cells, this produce IL-17. This provide interleukin 17. Okay, which confer antifungal and antibacterial immunity. And this TFH cells, follicular helper T cells, these have stimulated germinal center formation in your lymph nodes. Okay, understood? What is CD? CD means a cluster of differentiation antigens. These CD antigens are used as a surface marking of your lymphocytes. Okay, these are present over the surface of the lymphocytes and act as identification of the lineage of cells of which CD are there. Okay, so you know from the previous discussion, if there is CD19, CD20, or even CD21, okay, this will indicate that is there B cell present, and if there is CD3, this will indicate your T lymphocytes are present. Okay. Then regulatory T cells, okay, these are your negative regulators of your immune system. Negative regulators are your repressors of your immune system. Okay, this will secrete your IL-10, interleukin 10, and TGF beta. Okay, TGF beta. Okay, which will inhibit your T cells, Okay, so regulatory T cells, so suboptimal functioning of your regulatory T cells will cause your autoimmunity, because regulatory T cells are generally repressive. Okay, these are generally inhibiting. Okay, these are generally inhibitory. So that if there is suboptimal functioning, if there is no control, so you can have autoimmune disorders. Okay, you can have autoimmune disorders. NK cells are natural killer cells which are non-phagocytic cells. Okay, this kill the tumor cells and viral infected cells and derived from the bone marrow. This NK cells have two receptors: your inhibitory NK receptor. So one is called as inhibitory NK receptor. Okay, and if a healthy cell is there, healthy cell will express your MHC1. Okay, MHC1 receptor. So if there is the interaction between inhibitory NK cell receptor and your MHC1 receptor, there won't be any killing. But if this NK cell like it has another receptor, if there is no MHC receptor over the tumor cells, like infected cells, okay, this infected cells may be caused due to cancer or herpes simplex virus, okay, or Epstein-Barr virus or chickenpox viruses. Okay, if they are inducing a cell to lose its MHC receptor, okay, so NK cell will start releasing your antibodies, and this antibody will kill the cell. Talking about neutrophil, neutrophil is another cell of your immune system, which are also called as a polymorphonuclear leukocytes, okay, which is the indicator of inflammation also. Okay, these are most abundant WBCs and develop in the bone marrow. Okay, this confer phagocytic killing, this act via phagocytosis, okay, and also generate antimicrobials. So neutrophils have two type of granules, like a primary granules having acid hydrolases, lysozymes, and myeloperoxidases, or a secondary granules having lactoferrin or a lysozyme. The lactoferrin and your lysozyme are present in secondary granules. Okay, and if these granules are released, it will cause the death of the pathogen. Okay, and pyrogenic infection is infection which induces pus formation. Okay, so don't be confused. What is pus? Pus is the live plus dead neutrophils. Okay, the accumulation of dead neutrophils, if they are live or dead doesn't matter. Okay, in acute inflammatory processes, so if there is accumulation, there is the pus formation. Okay, the pus formation, and this is seen like acne and all, acne vulgaris and all. Your pus infection, pus formation is there. See. Then you have another cells, your monocytes are there, your macrophages are there. You know about all this. Okay, your monocyte, when they reach the tissues, they differentiate into macrophages. Okay, and these are the mononuclear phagocytes in the blood. Okay, and this macrophages, monocyte from macrophage, many changes are there, like the monocyte will increase in size, there will be increase in number of granules and increased phagocytic activity. Okay, so macrophages are generally your improvised monocytes. Okay, and eosinophils comprise of 0 to 6%, and these are granules rich in basic proteins, which are toxic. Okay, and these are Fc receptors for IgE and IgG. Okay, these are sensitive to the IgE and IgG. Okay, basophils are present in less than 1%. Okay, these are helpful in allergy mediation. And let's talk about the dendritic cells. Okay, dendritic cells, these are antigen presenting cells, okay, which have long membranous extensions, like dendrites of a neuron. Okay, they call it dendritic cells, and these are a bridge between acquired and innate immunity. So this statement is important. This may come as a this is a statement of a Lippincott textbook, so this is a standard statement. Okay, dendritic cells may be of many types, like a Langerhans cells in your skin. Langerhans cells are present in your skin, so I can write this for you. Okay, these Langerhans cells are present in your skin. Okay, and we have the inflammatory dendritic cells, the classical dendritic cells, and plasmacytoid dendritic cells. Talking about complement system, complement proteins are the plasma proteins synthesized in your liver. Okay, liver synthesizes the complement proteins, like many complement proteins are there, C1, C2, and many other. Okay, about 12 complement proteins are there. Okay, which will inhibit the pathogens by cytolysis, by inflammation and phagocytosis. Okay, cytolysis, inflammation producing, and phagocytosis. This is the mechanism where complement system works. Your CD4 to CD8 count ratio is normally, it is 2:1, which is altered in your immunodeficiency diseases. Okay, like in AIDS, your CD4 to CD8 count ratio is altered. Okay, this is of diagnostic importance. Then talking about immunodeficiency disorders, okay, you can have primary immunodeficiency or secondary immunodeficiency. Primary immunodeficiency, like your SCID, the most common cause of SCID, okay, severe combined immunodeficiency, most common cause is your adenosine deaminase deficiency. Adenosine deaminase deficiency, we talked about this in your pyrimidine metabolism, your purine metabolism also. Okay, and another primary immunodeficiency is ataxia telangiectasia. Okay, do you remember where we read this? Yes, in your DNA repair mechanisms, and the Chediak-Higashi syndrome. So the primary immunodeficiencies. And secondary, you have more common. Okay, secondary immunodeficiencies which are more common. Okay, this occur due to causes like HIV/AIDS. So HIV/AIDS is example of your secondary immunodeficiency. Okay, so let's move on to the next part of immunology. Let's talk a little bit about AIDS. AIDS is the causative agent of HIV, which is a retrovirus and replicates using the RNA-dependent DNA polymerase. Okay, HIV-1 is the more common and your more severe form than HIV-2. Okay, transmission is, unprotected sexual intercourse, your blood transfusion, your sharing of needles. Okay, pregnancy and lactation, that is a parenteral route. Okay, and mucosal contact and needle prick injuries, these are common in your healthcare workers. Okay, working in your AIDS prevalent regions, where they have to work very carefully so that the needle doesn't prick. Okay, pathogenesis, what is the pathogenesis? So if you want to see the structure of your HIV, okay, this is the HIV structure. Okay, having GP120, which is a docking glycoprotein, and GP41, which is a transmembrane glycoprotein. Okay, so GP140 is present to the outer membrane. Okay, and GP41 is a transmembrane glycoprotein 41. Okay, and P17, which will form your matrix. This it will line the matrix. Okay, P17, and then another one is a P24, which is present in the capsid. Capsid is the capsule of the DNA in which the genome is incorporated. In the genome is there, it has an RNA genome, and many proteins are there, like integrase and reverse transcriptase enzymes are there. Okay, integrase and reverse transcriptase enzymes are there. Okay, this is the HIV structure, and this GP120 helps to attach it to the CD4 receptor that is present over your T helper cells. Okay, T helper cells will express CD4 receptor, and this GP120 will help the HIV virus to bind it to the CD4 receptor. Okay, and this will produce your viral RNA. Okay, and in case of this is not important. Okay, let's see your viral RNA. This viral RNA comes into your macrophage or your T helper cells. Okay, so you can see the whole process here. Okay, starting from attachment of the virus to the CD4 receptor. This is the green one, is the CD4 receptor present over here. Okay, and the virus gets attached to the CD4 receptor. Okay, this will bind to the co-receptor also. A co-receptor is also present, which is CCR5. Okay, CCR5 or your CXCR4. CCR5 or a CXCR4. Okay, in TH cell you have CCR5, and in your macrophage and monocytes you have CXCR4. Okay, these are bound to the chemokine co-receptor. Okay, then there will be fusion. Okay, the virus will fuse with this and release the viral RNA. Okay, this viral RNA will by some reverse transcription mechanism. Okay, this viral RNA will be converted into viral DNA. Okay, because the HIV has reverse transcriptase, okay, so by the genomic RNA in the presence of a reverse transcription produces a viral DNA. This viral DNA is incorporated in the host cell genome. It's incorporated in the host cell genome by the enzyme, which is your integrase protein or enzyme, you can say. Okay, this viral RNA is incorporated into your healthy host cell genome. Okay, and transcription, when the transcription will occur, it will produce a viral RNA, and this viral RNA will also produce a viral proteins. Okay, this is the viral proteins, and this viral proteins will again combine to form another HIV. So as the host cell replicates, your HIV will also start replication. Okay, another HIV is produced, another HIV will produce. Okay, this whole pathway will go on and produce a host of HIV viruses. Okay, so HIV viruses generally are replicating, okay, by this macrophage. In the macrophage or your TH cells, okay, macrophage is called as your protein, your HIV factory. Okay, HIV factory. When the TH cell count is less than 200 cell per milliliter of blood, 200 cell less than 200 cell per milliliter of blood. Okay, so HIV is said to be your opportunistic infections will set in. Okay, HIV will progress. Opportunistic infections like a Kaposi sarcoma by herpes virus. You have read this Kaposi sarcoma, I remember where. Okay, in your cancer chapter. Okay, then your Hodgkin's lymphoma by Epstein-Barr virus. This was also the example of your infections or your oncogenic viruses. Okay, and this opportunistic infections are due to your HIV only. Okay, what are the diagnosis? You can detect the HIV, your HIV RNA using the polymerase chain reaction. We talked about this in your polymer PCR. Detection of your viral proteins like a P24. You can detect the P24 which is forming the capsid of the virus. Okay, and detection of antibody using immunoassays. Okay, what is the treatment? So you can have many pyrimidine analogs like a zidovudine and AZT. Remember we talked about AZT, zidovudine, which will inhibit a reverse transcriptase, which will prevent a reverse transcription of your viral RNA to viral DNA. This viral RNA to viral DNA can't be formed by the zidovudine and the statibudine. Okay. Understood, which are your pyrimidine analogs. Okay, and you have purine analogs too, like your guanosine analog. Okay, and your adenosine analogs and the inosine analogs. So you will read this more about in your pharma classes in second year. Okay, then fusion and entry inhibitors. These are some drugs like ifrulimumab and maraviroc. Okay, this all, and this protease inhibitors are there, right? Like ritonavir, indinavir, neflinavir, and your ammonavir. Okay, or lopinavir. These are your protease inhibitors. So just remember ritonavir from here, protease inhibitor. Okay, which is preventing this degrading the viral proteins. Okay, and the reverse transcriptase inhibitors like your zidovudine and statibudine. Okay, just remember these three drugs. Okay, and if you want to know more, you can take the screenshot. Yes, done. Okay, let's move on to the next part. Okay, let's talk about antigens. So antigens have epitopes. Epitopes are the immunologically active regions over the antigen which interact with the B cell receptor and T cell receptors. These are mainly foreign substances, maybe the body's own, okay, but possess immunogenicity due to this presence of your epitopes. Okay, antigens on the basis of immunogenicity can be classified into immunogen, which can induce antibody formation, and your hapten, which require a carrier to induce your antibody formation. Hapten require a carrier to induce your antibody formation. And on the basis of your ability to induce antibodies, you can have T cell dependent or you can have T cell independent. Okay, on the basis of source, you can have exogenous antigens and endogenous antigens. Endogenous antigens may be classified into a genoantigen or your alloantigen or your autoantigen. Okay, genoantigen like antigen is produced from different species. Okay, alloantigen like from same species but different individual, and autoantigen is self antigen. Okay, what are the determinants of your immunogenicity? This can come as a short note. So size, larger molecules are more antigenic. Nature of molecules, proteins are more antigenic than the rest. And then foreign antigens. Okay, your genoantigen is more antigenic than your alloantigen, which is more antigenic than your autoantigen. Okay, and heterogenicity. Okay, talking about heteropolymers are more antigenic than homopolymers. Okay, optimal doses, so more antigenic will be there. Route administration, parenteral administration has more antigenicity, like mother to fetus transmission has more antigenicity, and it also depends upon the host's genetic makeup. Talking about the structures of antibodies, this is the important part in this chapter. Okay, antibodies are produced by plasma cells which confer your humoral immunity. Okay, these are antibodies are glycoprotein molecules. So this is the whole structure. We have a variable region here. So this whole part is a variable region, and this whole part is a constant region. Okay, and in variable region also you have this is the light chain of the antibody. This is again a light chain, two light chains, and which are the heavy chains. You can just see this is your heavy chain, and this is also your heavy chain. Okay, so you can have heavy chains and light chains in the antibody. Okay, talking about this VL and VH, these are a variable heavy and variable light chain region, VL, VH. Okay, when you CL and CH, in which one disulfide bond is present. Okay, disulfide bridge is present, so you can have 4 disulfide bridges here, 1, 2, 3 and 4. Okay, and this part, the joint part between this variable region and this constant region is called as your hinge region. This is for its flexibility. Okay, flexibility, the antibody can rotate in various directions. Okay, due to this hinge region. Okay, understood. So you have heavy chains like your gamma, your alpha, okay, mu, epsilon, and your delta. Light chains coded by your kappa and lambda. These are not much important. Okay, this whole region which is above the hinge region is called as FAB. This is FAB. It contains the antigen binding site. AB stands for antigen binding, and below it is called as FC or your crystallizable fragment. Okay, it crystallizes during cold storage. Okay, it crystallizes during cold storage. Then genes for heavy chain is present on chromosome number 14. Okay, chromosome number 14. Genes for light chain, the kappa light chain gene is present on chromosome number 2, and the lambda light chain gene is present on chromosome number 22. Okay, and the light chain genes, these are differentiated to V region, your J region genes, the C region genes, which code for your constant region, and heavy chain genes may be classified into V region, J region, D region. Okay, remember we talked about an example in which the V genes, about 300 V genes were being shown, about 5 J genes were being shown, and about 10 D region genes have been shown. is your coding for the variable chain, okay, in your gene rearrangement. Okay, I am talking about your gene regulation, gene expression, regulation of gene expression in your eukaryotes. Okay, in which there was a phenomenon called as a gene rearrangement. Okay, and this existence of multiple B, D and J regions confer antibody diversity. Okay, random combinations may occur, which I was talking about. Okay, and these things are there. Random association of heavy and light chains may occur. Okay. You know about antigen-antibody reaction interaction. The antigen has epitope, while the antibody has a paratope. Okay, there is the epitope and paratope which are interacting. Okay, interacting by H bonds, your hydrophobic interactions or your van der Waals interactions. Okay, about precipitation reactions, your antibodies and the soluble antigen, these are called as the precipitants. Okay, and this precipitation reaction is helpful in your immunodiffusion and your immunoelectrophoresis for the diagnosis of your amyloidosis and your echinosis. Amyloidosis and your echinococosis. Okay, amyloidosis and the echinococosis. And agglutination reaction. Okay, where the antibody reacts with a particular antigen to form agglutinins. Okay, a projon effect is there. What is projon effect? Excess antibody will inhibit agglutinins reaction. There is excess antibody, it will inhibit agglutinins reaction. This is called as a projon effect. Where this agglutination reaction is helpful in the blood typing. You must have read in the physiology practical about the blood typing, where you determine the blood group of the subject. Okay, by putting that gray top vial. Okay, you have not a gray top vial, your blue top vial and your yellow vial. Okay, all that things are present. Okay, and bacterial infection diagnosis. Then about immunoassays, you have your radio immunoassay RIA, you have your chemiluminescence immunoassay, the ELISA. Okay, you know about the ELISA. Okay, we just discuss your ELISA. Okay, we used this example HRP was the most commonly used, or horse radish peroxidase. Okay, linking substance we used like glutaraldehyde. Okay, periodate and melamide. Types of ELISA we saw: direct ELISA, your indirect ELISA, the sandwich ELISA, and the competitive ELISA or an inhibition ELISA. And color producing ELISA is measured spectrophotometrically. Okay, okay, and what are the functions of antibodies? You know, opsonization is one of the function. But opsonins, these are proteins which mark foreign antigen or eliminating them, like it's a tag. Okay, if a cell comes with a foreign antigen, then opsonins will form a tag like this. Opsonins will form a O tag. Okay, and this O tag will be the marker for your elimination of the foreign antigen. Okay, this they will be recognized by your T cells, and there will be like formation of your phagosome. Then complement activation, the complement proteins and the pathogen antibody complex. Okay, this react and destructed by opsonization or a perforation or a splenic deposition. Okay, then neutralization, like your diphtheria antitoxin, which is neutralizing antibody which neutralizes your diphtheria toxin. Okay, then antibody dependent cell mediated cytotoxicity is there. Okay, what is hypergammaglobulinemia? This is the presence of an excess immunoglobulin in blood. Okay, it occurs in your infections, in your chronic liver disorders, autoimmune disorders like rheumatoid arthritis, and in multiple myeloma. Okay, due to presence of excess like immunoglobulins in the blood. Okay, you know about the autoimmune disorders like myasthenia gravis is there. Okay, your pernicious anemia is there, where the antibodies are present against the intrinsic factor. Okay, or your organ non-specific like systemic lupus erythematosus (SLE). Okay, rheumatoid arthritis or multiple sclerosis. These are there. Okay, SLE, your systemic lupus erythematosus, it is a autoimmune disorder. Okay, in clinical features it includes fever, weakness, rashes, and a characteristic butterfly rash on cheek. This is the characteristic feature of your SLE, systemic lupus erythematosus. Okay, talking about polyclonal antibody, a short note may come of your about a polyclonal monoclonal antibody. Okay, so let's talk about them. This polyclonal antibody, these are synthesized from different clones of plasma cells, okay, which are capable to bind multiple epitopes of same or different antigen. Okay, so monoclonal antibodies are synthesized from single clone of plasma cells, while polyclonal are synthesized from different clones of plasma cells. Okay, monoclonal antibodies are expensive, while polyclonal antibodies are non-expensive. Monoclonal antibodies are difficult to produce, these are produced by hybridoma technology. Okay, hybridoma technique. This is also of diagnostic importance, and the polyclonal antibodies are easy to produce. Okay, and the monoclonal antibodies, these are time consuming and specific for epitope, but polyclonal are not time you're consuming. Okay, what are the applications of monoclonal antibodies? Okay, these are synthesized by hybridoma technology. Hybridoma technology is there, and these are of diagnostic applications like several diagnostic techniques like ELISA, Western blotting. Okay, and immunohistochemistry. Okay, these are applied. Okay, then the monoclonal antibodies are used in the treatment of cancer and rheumatoid arthritis. Okay, talking about Benz-Jones proteins, there are light chain of immunoglobulins excreted in urine in your multiple myeloma. Okay, Benz-Jones proteins are excreted out in multiple myeloma, and we also read that the Benz-Jones proteins are your tumor markers also. Okay, and it is detected by heat test or your coagulation test. Okay, is precipitate at 40 to 60 degrees Celsius. Okay, Then another thing is a Waldenström macroglobulinemia. Okay, this is a malignancy of lymphoplasmacytoid cells which are secreting IgM. Okay, so there is malignancy of IgM associated with lymphadenopathy. Okay, hepatosplenomegaly will be there, and major clinical manifestations are of hyperviscosity syndrome. Okay, that will be increased viscosity of the blood. Okay, so this completes your antigens, antibodies, and your ELISA and your monoclonal and polyclonal antibodies. Just remember the difference. It can be asked, or your short note about your polyclonal or your monoclonal antibodies. Your diagnostic application of polyclonal antibodies, these are used in immunoassays like your ELISA, okay, Western blotting, or your immunohistochemistry and flow cytometry. Okay, and there are also therapeutic agents, okay, like your convalescent plasma therapy, which was used widely in your COVID times. Okay, so this completes your antigen, antibodies, and ELISA. Let's move on to the next part. Okay, so we'll talk about innate immunity now. Innate immunity versus adaptive immunity. So what are the differences? It can come as a question, your short note, or your different type of questions. So what are the cells involved? Your dendritic leukocyte, your natural killer mast cells, dissolved cells confer your innate immunity, while adaptive immunity is governed by your T cells, T helper cells, B cells, antigen presenting cells, etc. Okay, what are the molecules involved in innate immunity? Is just cytokines, complement cells, interferons, okay, acute phase proteins. Okay, and what are the adaptive immunity molecules involved? Antibodies and cytokines. Okay, and action time, innate immunity is an immediate effect, while there is delayed effect in your that is your adaptive immunity or your acquired immunity. Okay, delayed effect. Response occurs rapidly in innate immunity, while it occurs over days to weeks in your adaptive acquired immunity. What is the order of defense? It is the first line of defense as well as the second line of defense. Okay, and this acquired immunity, okay, if some pathogen evades your innate immunity, then it will act. Okay, if the pathogen evades your innate immunity, then the acquired immunity will be acted upon by. Okay, then there is no memory storage in innate immunity. Okay, but there will be immunological memory storage in your acquired immunity, like your antibodies and plasma cells. Your plasma cells are there in this. Your plasma cells are also there, which will store your memory. Okay, so you can write your plasma cells also present in the acquired immunity, okay, which store your memory, which are responsible for your memory storage. Okay, memory. Okay, this is that. Then again, what is happening, if there is subsequent exposure, the immune system does not get altered, but due to memory storage, due to the storage of memory, okay, due to subsequent exposure, the immune response will improve. Like first this will be there, and in the second part this will be the response. Okay, in the third part again this will be the response. Okay, so the subsequent exposures, the immune response gets improved. Okay, and there will be no allergy in your innate immunity, but there will be immediate and delayed allergy. Okay, in your that what I was saying, acquired immunity. Okay, so just look at the cells which are present, your natural killer cells, okay, and your T cells, which are called as your gamma delta T cells. Okay, just remember, natural killer cells, these are both components of innate and acquired. Okay, and APCs, these are components of acquired immunity. Okay, just you can take the screenshot of this. Done. Okay, talking about barriers of innate immunity, we have physical barriers. Like what are the physical components that are protecting us, like the skin, mucous membrane, temperature, interferons, lysozymes, low pH. Okay, epidermis of skin, which has keratinocytes, which will kill the peptides, and which has peptides and cytokines, which will kill the pathogens. Okay, sebaceous glands have fatty acids, which will give you acidic environment for killing. Okay, then you have your sweat gland, peristalsis, ciliary movements, which will remove the pathogens, and tears, saliva, flushing movement of the urine, which will kill the pathogens. Okay, then the chemical barriers, like your chemicals, enzymes and proteins. Okay, lysozyme, which is present in your sweat, tears and saliva, which will cleave the cell wall of the pathogen. Lysozymes cleaves the cell wall of the pathogen. Okay, which is present in your sweat, tears and saliva. Then gastric juice, which contains your HCl, mucus and enzymes, which will kill the pathogen. Your HCl, mucus and enzymes, which will kill the pathogen. Your Helicobacter pylori, like it is neutralizing. Okay, that's neutralizing the HCl and form colonies, forming your gastritis and gastric ulcers. Okay, so Helicobacter pylori is basically evading your chemical barrier. Okay, then phagocytic barriers, you have immune cells. These are the second line of defense in the innate immunity. What are the steps of phagocytosis? If the damaged tissue is there, it will release cytokines and CAMs, cell adhesion molecules. Okay, this will cause the chemotaxis, the movement of your phagocytes towards the infection. Okay, first neutrophils are recruited, then monocytes, then macrophages in the sequence. Okay, then the attachment of surface of pathogens will be there by toll-like receptors, TLRs, toll-like receptors or opsonins. Okay, these toll-like receptors attach to a peptidoglycan of the cell wall, a bacteria, and phagosome will be formed. This phagosome will fuse with lysosome, forming a phagolysosome. And that will be destruction of pathogen by oxygen dependent mechanism or oxygen independent mechanism. Okay, oxygen dependent by activation of NADPH oxidase and myeloperoxidase and generation of reactive oxygen species. Okay, like your O2 minus radical or H2O2. And oxygen independent mechanisms by releasing of lysozymes, lactoferrins or your defensins. Okay, Okay, evasion of phagocytosis, like for example, your Staphylococcus and Listeria, this will secrete leukocidins, which form pores on your phagocytes, and this can evade your phagocytosis. Okay, understood. What are the soluble components of an innate immunity? So you have acute phase proteins like a CRP protein, okay, CRP protein of pentahexanes family. Okay, these are synthesized in your liver again, and induced by TNF alpha, IL-1, and IL-6. Okay, these are your acute phase proteins. And again, we have interferons. Interferons are the glycoproteins also released by viral infected cells. Okay, and interferons may be three types: interferon alpha, interferon beta, IFN beta, and IFN gamma. Okay, IFN alpha by macrophages, IFN beta by fibroblasts, and IFN gamma by your TH cells, NK cells, or your TK cells. Okay. IFN alpha will increase the presentation of antigen, okay, and IFN beta will cause the expression of a class 1 MHC and viral RNA destruction. IFN gamma will cause your delayed hypersensitivity reaction, that is your allergic reactions, and graft rejection. IFN gamma will also do your graft rejection. You know, the graft rejection is the foreign tissue is rejected if it is transplanted. Okay. Talking about cytokines are secreted by our WBCs. We can, if the stress and inflammation, this can induce a cytokine formation. Okay, like your IL-2, IL-4, and IL-5 by the TH cells. Okay, or your IL-1, IL-6, IL-12, or TNF alpha by your macrophages. Okay, and the cytokines bind to the receptor of target organs. Okay, and increase or decrease the signal transduction pathways and alter the expression of various genes. Okay. This IL-1 and TNF alpha, these are inflammation mediating. IL-6 is the secretion of antibody, will do secretion of antibody. IL-2 will do activation of NK cells, and IL-10, this is anti-inflammatory cytokine. This is the anti-inflammatory cytokine. This is important. The levels will decrease during the inflammation. Let's move on to the next part. So let's talk about adaptive immunity now. So you have been observing that I am teaching immunity in a little bit detail, okay, because I am covering the points of your Lippincott textbook, okay, of each and every point of your Lippincott textbook for the immunity purpose, okay, and so I am teaching the immunity in a little bit detail, so bear with me. So adaptive immunity develops during the lifetime of a host. It is acquired, it is highly specific, and development of immunological memory will occur here. Okay, adaptive immunity may be active immunity or passive immunity. Active immunity means it is produced actively by the host immune system, and passive immunity is received. It's received passively. Okay, active immunity is induced by any infection or your vaccination, but your passive immunity is acquired from mother to fetus or your antibody transfer. Okay. So the active immunity is long lasting, but passive immunity is short lasting. Active immunity has a lag period present in it. It works a little bit slow, having a lag period in it. But in passive immunity, it has no lag period. Memory is stored in your active immunity, but no memory storage in your passive immunity. Booster doses are useful in case of your active immunity. Okay, but there is less effectiveness of booster doses in your passive immunity. Okay. And active immunity is not useful in immunodeficient individuals. Okay, but passive immunity is useful in immunodeficient individuals. So you can get a difference between active and passive immunity. So you can just take a screenshot of this. Done. Okay, let's move ahead. What are the mediators of adaptive immunity? So you have antigen presenting cells. Okay, antigen presenting cells, like your this trap the antigens. This can trap the antigen and present it over the surface and the antigen presenting cells, when these are presented, the antigen are presented. Okay, T cell receptor will recognize your presentation, and the T cell will kill them. Okay. This can be proteinaceous, your professional antigen presenting cell, professional or your non-professional. Okay, so best APC is your dendritic cell, your B cell and your macrophages are some. Okay, non-professionals, these are your fibroblast, glial cell, and pancreatic beta cells. Okay, and talking about major histocompatibility complex, the MHC which I was talking about, it's a complex of genes, okay, which help the T cells to identify the pathogens, Okay, so T cell will recognize your MHC, this MHC will activate your T cells, okay, TCR will recognize your MHC, and this MHC will activate your T cell and cause elimination of the pathogens. Okay, so MHC complex in humans is found in chromosome number 6. It is also known as a human leukocyte antigen, HLA. Okay, in the MHC in humans is called as your HLA. Okay. So we have class 1 MHC, class 2 MHC, and class 3 MHC present. Okay, class 1 MHC is expressed on all cells, all nucleated cells, but class 2 MHC is present only on APCs. Okay, expressed only on APCs. Okay, and class 3 MHCs, this code for your complement proteins and heat shock proteins, okay, which are involved in inflammation purpose. Understood? Okay. So TH cells are MHC2 restricted cells, and TK cells are MHC1 restricted cells. Okay, we don't have to go into details of this restriction and all. MHC restriction is a topic that is given in a Lippincott, but you don't need to go into detail. Okay, what is antigen processing? So we have cytosolic pathway in which the endogenous antigens are tagged with a ubiquitin. Okay, ubiquitin tagged antigens are produced, which are degraded in the proteasome. Fragments are created. These fragments associated with the transport associated protein, okay, TAP, transport associated protein, and are transported to the RER, rough endoplasmic reticulum, from which they interact with the class 1 MHC present over the rough endoplasmic reticulum. Okay, so you can see the class 1 MHC is present over all nucleated cells. Okay, so in RER it interacts with the class 1 MHC. Okay, this MHC will be folded by chaperones like your calnexin and your calreticulin, okay, and transported to Golgi, and from the Golgi these are presented in the cell surface. So this is called as your cytosolic pathway of antigen processing. Okay, antigen processing. Then talking about endocytic pathway, exogenous antigen are now induced what we can call your endosome is formed. Okay, these are engulfed, yes, I forgot the term actually, engulfed by the antigen presenting cells. Okay, where the lysosomes will degrade this endosome formation, the phagosome formation. Okay, on degradation these vesicles will be formed, and these vesicles will bind to a class 1 MHC which is present in the antigen presenting cells. So the class 2 MHC is present in the antigen presenting cells and are transported to the cell surface for presentation. Okay, about clinicals, let's discuss some clinicals. Inflammation. Okay, what are the features? Tumor, redness, rubor, heat production, calor, pain production, dolor. Okay, and then will loss of function called as your functiolesa. Okay, and tissue injury will induce your formation of histamines, kinins, leukotrienes. This will call vasodilation, increased blood flow, and increased warmness and redness. Okay, and this edema and swelling will occur due to increased vascular permeability. Increased, the fluid will be leaked out, and this will cause your edema. Okay. And the second stage, there will be margination. Okay, the phagocytes will stick to the vessel wall and travel to the site of injury by sticking to the vessel walls, and there will be phagocytosis. Okay, in the third stage there will be tissue repair. Third stage is the tissue repair. Okay, endotoxins are present in the cell wall of bacteria. They release your IL-1 and TNF alpha, and also your they stimulate the release of your IL-6 and 8. Okay, which are early sepsis, which produce early sepsis. Okay, and IL-6 and 8 produce your late sepsis. Okay, late sepsis. This is not much important, you can just skip this part. So this completes your immunity part. Okay, now let's move on to the next chapter. Okay, some important parts like if Beer's law is there in colorimetry. I am talking about a little bit about colorimetry. Of course, you'll read it in a practical. Okay, so I'm just talking it in very less details. So Beer's law is there. Beer's law, that is the amount of light absorbed by a substance is proportional to the concentration of substance in the solution. Okay, and the Lambert's law state that the amount of light absorbed is proportional to the depth of the path length. Okay, it is the path length. Okay, amount of light absorbed is proportional to a path length traversed by the light in the substance. Okay. Most widely used glucose estimation method, this is a GOD-POD method. Okay, formula for estimation of concentration of unknown solution. This formula is important. Your concentration of test, okay, which you are trying to measure, is your optical density of the test minus optical density of the blank. Okay, and optical divided by optical density of the standard minus optical density of the blank into your concentration of your standard. Okay, you know the standard concentration. Okay, this ODT will measure. Okay, and ODB and ODS will be given in the practicals. The ODS will be given, this ODB will be given, the CS you know. Okay, and you have to calculate your concentration of your unknown solution by this. Okay, what is the application of a colorimetry? What is the application? To estimate your plasma urea, creatine, okay, uric acid, cholesterol, SGOT, SGPT, ALP, calcium, phosphate, albumin, and bilirubin. Okay, this is used in estimation of all of this. Okay, you read in detail in your practicals. Okay, so I am not talking about much detail. Okay, so let's now move to the another part, another chapter. Okay, so again, just a reminder what we have finished. Okay, we have finished our immunity now. Okay, this mechanism of hormone action, this is especially in your physiology. Okay, so I am not talking in much detail about this. You know the mechanisms of hormone action. What are the things you know? Okay, your how the secondary messengers and primary messengers, these are acting. Okay, in biochemistry most probably we won't get any like question in from this. Okay, this is of your physiology. Okay, so you can consider it as done. Okay, and about biochemical technologies, you have electrophoresis and SDS page. Okay, we talked a little bit about SDS page also. We talked about your colorimetry and your ELISA. Okay, and about the quality control and automatic analyzers. Okay, let's talk about them a little bit, I guess. Okay. Okay, so let's discuss a little bit about your quality control in your laboratories. Okay, so clinical chemistry laboratory must have quality control measures, okay, to assess the reliability of each measurement that is performed on a sample. Okay, quality is the conformance to the satisfying needs. Means if you have a reference value, okay, you have a reference value, and to this reference value how much your measured values are being matched. Okay, measured values are being matched. Okay, and how much is the satisfaction of the people to this measured values. Okay, it depends your quality, it depends upon your quality. Okay, so all the laboratories must have quality control measures. Okay, quality control measures, and this may have two types: your external quality control, and your internal quality control. Okay, So external quality control, let's say you have a central reference laboratory which sends a serum sample, okay, containing a known quantity which is analyzed in a small laboratory. If the result obtained in the peripheral laboratory is same as the reference laboratory, okay, so the arrangements are in the peripheral laboratory are said to be reliable, okay. External means there is a central laboratory, okay, central lab which sends it which sends a known quantity, okay. It doesn't disclose the known quantity, known measurement to a peripheral lab, okay, peripheral lab. And in peripheral lab it is analyzed, okay, it is analyzed, and if there is the values match the known quantity, then the peripheral lab is said to be like compatible with its qualities, okay. This is usually done once or twice a month, okay, once or twice it is done in a laboratory, okay. And your internal quality control, okay, it is internal quality control, a reference is made by the internal your peripheral lab only. Peripheral lab will make a reference, okay, and then check its sample for within its reference, okay, and the results are checked on a daily basis, okay. So you do the daily quality control measures in laboratory are called as your internal quality control. So you have external quality control, the internal quality control in a laboratory, okay. Accuracy is the closeness of a result to the true value, okay. If the result is close to the true value, okay, true value, then this is called as your accuracy. This is called as your accuracy, okay. And precision is the reproducibility of the result, okay. It is the your reproducibility of the result, okay. Understood? So how much it is reproducible, okay. And about another thing is the total quality measurement, okay. Total quality measurement, this has about 6 sigma principles and matrices, okay, which is introduced in the laboratory. This is a recent it is involved recently in your healthcare sector, just as in your industries. In industries your TQM is done, okay. It is now involved in your that quality measurement. So you have 5 Qs of quality measurement, okay. 5 Qs of quality measurement. These are your quality planning, okay, quality planning, okay, quality laboratory process, okay, then quality control, then quality assurance, okay, and your quality improvement, okay. So these are your 5 Qs of your quality measurement, okay. These are 5 Qs of your quality measurement, okay. One is called as CRM. What is CRM? It is the certified reference material. Okay, which is just certified, and like your serum urea, okay, it has your uric acid, for suppose your serum uric acid, okay, the reference is your 5 to 7 in males and 2 to 5 in females milligram per deciliter, okay. This is your reference, okay. This is certified, so all the values should be compatible with this reference, okay, compatible with this reference, okay. And control material is a device, solution, or pool specimen which is used in the quality control process, okay. And the calibrator is the material which will test the specimen, okay. Calibrator is the material which will test the specimen, okay. And the calibrator will generate a QC chart, okay. It will generate a QC chart, okay. What is the QC chart? A QC chart is the quality control chart, okay. How much is the quality, okay. The values will indicate if the reference, if the intervals, if the values are acceptable or out of control or unacceptable, okay. Understood? So we have quality charts like your Levy, 2 quality charts like your Levy-Jennings or your LJ chart, Levy-Jennings chart, or you can have your Westgrad's multi-rule chart, okay. Understood? So this if the value is out of limit in a Levy chart, okay, reporting should be stopped and resolve the problem and then repeat the run, okay, of specimens and controls. In the Westgrad multi-rule chart, okay, the same procedure is there but using 2 different controls, okay. The control values are within like 2 second values are acceptable. If the single value exceeds +-2, it is taken as a warning, okay. If one value exceeds +or -3s, okay, so random values are detected as per the rejection rule, okay. So these are just many things which are like not much important. If you go into detail, you can search up this charts in your internet and see, okay. And your another thing is the EQAC is the external quality assurance schemes, okay, which compare the performances of laboratory 1, laboratory 2, laboratory 3, okay. Like you have different medical colleges and different laboratories, different hospitals and different laboratories, so comparing this all, okay, comparing the SCB biochemical laboratory with your clinical biochemical laboratory of MKCG, okay, like that, okay. like this is called as external quality assurance scheme. Okay, and this summary of the reports are sent to the participating laboratories by the control laboratory. Okay, and for each laboratory a SDI is calculated. What is SDI? It is the standard deviation index. Standard deviation index is calculated. Okay, and standard deviation index is equal to your lab result minus group mean, minus your group mean. What is the group mean? The mean result minus your, what is the lab result. Okay, so you can find the deviance of how much quality it is deviating. Okay, how much it is being deviated from the required quality. Okay, and the laboratory safety, you know, good laboratory practice and waste disposal should be there. Your personal protective gear must be worn, like safety glasses, goggles, your gloves and laboratory aprons should always be used. Okay, and for your reagents and chemicals, this may be many grades of purity is there. Okay, so purity must be, like, details must be provided by the manufacturer of how much pure is a like chemical is this. Okay, and laboratory safety is very important, otherwise there may be hazards, like your many hazards are there due to your things. Okay, so this laboratory safety, this is governed by your OSHA Act. OSHA stands for your occupational safety and health act. Okay, this is your OSHA Act, which governs your safety, which governs your standard safety measures for your that laboratories. Okay, understood? So this completes your laboratory safety. Okay, and your quality measurement in your laboratories. Okay, we just talked superficially about these things. Okay, and now let's move on to the next chapter. Okay, so let's talk about your electrophoresis. Electrophoresis is the migration of charged particles through a solution under the influence of an external electric your field. Okay, and Tiselius is the scientist who invented the electrophoresis method. Okay, so the pH, we have the PI. PI is the you know about the PI, your isoelectric pH. Okay, if the pH rises above pH is rising above the PI, then there is negative charge, and negative charge will move towards the positive electrode. And if there is, the pH will be below the PI, then there will be positive charge which will move towards the negative electrode. Okay, negative electrode. What are the factors affecting your electrophoresis? Okay, so electrophoresis is affected by net charge on the molecule. The charge will be affected, net charge, and your size and shape of the particle, Okay, and the movement, okay, the mobility will be inversely proportional to your size, you know this. Okay, inversely proportional to the size. Then it will depend upon the pH of the buffer. Okay, pH, and it will also depend upon the strength of the electric field strength. Okay, we are providing, and also depends upon the properties of the supporting medium and the temperature of the system. Okay, so how is the system? Let me tell you. Okay, so let's say we have this. Okay, we have this, and we have the buffer present over here. We have the buffer like this. Okay, and the positive electrode is dipped over like this, and the negative electrode is dipped over like this. Okay, and the power supply is there. It is connected to your battery. Okay, connected to your battery. Okay, and the bridge is there like this. This is called as a bridge. Okay, and wicks are there to connecting it to the bridge. Okay, and this is the support media. Understood? And the support medium which is there in the lid is comprised of your filter paper. Okay, so this is the horizontal electrophoresis apparatus. Okay, and you can use the support media for this electrophoresis. Support media, you can use a filter paper. Okay, and your cellulose acetate membrane. Okay, cellulose acetate, your polyacrylamide gel, your starch. Okay, then you know your agar and agarose. You talk about this agarose. Okay, so what is the steps that is used in electrophoresis? Okay, so first we'll do the method selection. Okay, and select the appropriate electrophoresis method. There are many types of electrophoresis, like your paper electrophoresis is there. Okay, your cellulose acetate membrane electrophoresis, your agarose gel electrophoresis, your starch gel electrophoresis, and your polyacrylamide gel electrophoresis, all this. Okay, then you prepare the sample. The protein sample may be prepared from a biological sample. Okay, and in either case, the prepared sample is a concentration and a buffer suitable for your electrophoresis. Then you prepare a gel and buffer. Okay, the gel type should suit the properties of the protein. Okay, and buffer selection will also depend upon the gel type and the type of electrophoresis. Okay, then you perform the electrophoresis. Okay, so gels are placed in the electrophoresis cell. Okay, buffer is added and samples are loaded. Okay, and optimum resolution is maintained while maintaining the temperature of the system. Okay, and the staining techniques will, the protein is detected by selecting a staining technique, a correct staining technique. Okay, so this is the whole procedure. You can see the diagrams. Okay, how electrophoresis is done. Okay, talking about the types of electrophoresis, we have your paper electrophoresis, in which Wattmann's paper is used. Okay, for your peptides, for your okay, for your peptides, nucleic acids, okay, nucleic acids, okay, nucleic acids and your nucleotides, okay, this all you use a paper electrophoresis, okay. And for your separation of lipoproteins or separation of your lipoproteins, okay, and your glycoproteins, and your hemoglobins, okay, hemoglobins, the cellulose acetate membrane electrophoresis is done. The cellulose acetate membrane electrophoresis, it is done. Okay, understood. And for the separation of plasma proteins, okay, plasma proteins and your lipoproteins, okay, and your isoenzymes, for example, you have LDH1 to 5, okay, the isoenzymes of LDH, of your CDK, or of your creatine phosphate, okay, creatine phosphate kinase, CK1, CKMB, CKMM, okay, and your ALP, okay. And for your immunodiffusion or immunoelectrophoresis, okay, this is done by the support media is agarose gel, so use agarose gel electrophoresis, okay, agarose gel electrophoresis, okay. For separation of isoenzymes of your LDH and ALP, okay, for separation of isoenzymes for the LDH and ALP, another electrophoresis is also, which is called as your starch gel electrophoresis, okay. And then we use for the separation of plasma proteins and enzymes, okay, we use your polyacrylate gel electrophoresis, okay, understood. After that, we will have some abnormal electrophoretic pattern. So abnormal electrophoretic patterns, okay, we can have abnormal electrophoretic pattern in few diseases, which we are going to see. So just see, this is the normal electrophoretic pattern, okay. So you have the gamma here, you have the beta here, okay, alpha 2 and alpha 1 of albumin, okay. And abnormal pattern, just see, you have in multiple myeloma, okay, you have a prominent M band between your beta and gamma, okay, between beta and gamma you have a prominent M band, okay. So this one is a prominent M band, okay, which will be seen in your multiple myeloma, okay, okay. And in a chronic infection, okay, there will be broad base increase in your gamma region, okay. This gamma region, this was the gamma region, okay, there will be broad base increase in your gamma region, okay. And in a nephrotic syndrome, okay, in a nephrotic syndrome you will have your prominent A2 band, okay. A2 band was like this, okay. In nephrotic syndrome you have a prominent A2 band, okay. This prominent A2 band, you can see your prominent A2 band, okay. So prominent alpha 2, sorry, I'm telling A2, prominent alpha 2 band, this is seen in your nephrotic syndrome, okay, understood. And in your cirrhosis of liver, okay, in a liver cirrhosis, you will see increased gamma globulins and beta, and the decreased albumin, and beta gamma bridging will be there, okay. In the cirrhosis of liver, you can see it is a normal pattern, okay. There will be beta gamma bridging, all other things will remain the same, okay, but there will be beta gamma bridging in case of cirrhosis in liver, okay. And this is also showing on the this is the normal condition of a fibrinogen. In fibrinogen you also get a thin M band, okay. So in cirrhosis you will get increased gamma globulin, okay. In cirrhosis of liver you will get increased gamma globulin, you can see that gamma is this region, okay. And increased gamma globulin will be present in the cirrhosis, gamma globulin will be increased, and beta gamma bridging will be there. And in nephrotic syndrome, prominent A2 band, and in a multiple myeloma you will see the presence of a M band, okay. These are your abnormal electrophoretic patterns, okay. So here we completed our electrophoresis. Now let's go on to the next chapter, okay. So let's discuss in less detail about your SDS based technology. SDS based stands for your sodium dodecyl, okay, sodium dodecyl sulfate polyacrylamide gel electrophoresis, okay. This is also a electrophoresis. So this is also improvisation over electrophoretic techniques. This is also improvisation, okay. So what you have, first of all, okay, you have the sample having the single subunit folded protein and a protein 2 subunits folded, okay. Joined by disulfide bond, you put A and B, Okay, then it is heated with SDS. What is SDS? Is sodium dodecyl sulfate. Sodium dodecyl sulfate. So it is heated with SDS. Okay, it is heated with SDS. Okay, and by heating, your denatured protein with uniform negatively charged, denatured protein will be uniform negatively charged because it is single subunit folded. Okay, and another thing, this will also be a single negatively charged denatured protein. Okay, this is also converted into single negatively charged denatured protein. So that is denaturation. Okay, there is denaturation. Okay, and to cleave this SS bond, this have SS bond present. To cleave this SS bond, we use your beta mercaptoethanol. We use your beta mercaptoethanol to cleave SS bond, to cleave SS bonds. Okay, understood. So this is now loaded into the this is now loaded into a gel. Okay, what are the components of the gel? Okay, gel has two glass plates. Okay, gel has two glass plates, and within them they have a stacking gel and have a resolving gel. Okay, a stacking gel and a resolving gel. Okay, so this two are your gels. Okay, the stacking gel have a pH of your 6.8. Okay, 6.8. Separating gel has a pH of your resolving gel or separating gel. This is also called as separating gel. This has a pH of 8.6. Okay, 8.6. Understood. So at pH 6.8 in the stacking gel, your glycine, which carries no negative charge. Okay, and your separating gel, glycine carries negative charged. Okay, so high molecular weight peptides will move slowly and low molecular weight peptides will move fast. Okay, understood. So this is your whole technology. You load it into the gel, then after you do the electrophoresis. Like electrophoresis done in a chamber in the normal, in a traditional method, but here you do your electrophoresis like this. Okay, electrophoresis is done and it will generate a electrophoretic pattern. Okay, your SDS page will be generated. Okay, you will see the high molecular peptides above. Okay, if this is your cathode here and this is your anode. Okay, then in above region you will see high molecular weight peptides, and in the lower region you will see low molecular weight peptides. Okay, so this whole procedure is based upon the molecular sieving effect. This is based upon your molecular sieving effect. Okay, molecular sieving effect, due to which the high molecular weight peptides will move very slowly. Okay, move very slowly. Low molecular weight peptides are moving very fast. Okay, these are in this direction from cathode to anode. They are moving very slowly and these are moving very fast. Okay, the only thing that is unique in this gel electrophoresis is that SDS page is that these are this stacking gel and resolving gel. These are used. Stacking gel and the resolving gel. These both are used. Okay. Understood. So this is your gel electrophoresis, your SDS page. Okay, what are the applications of SDS page? Applications. So you can get a short note on the applications, or if you or you can get a short note on SDS page. So you can write the application. Okay, to measure the molecular weight of the that proteins by using your molecular weight markers. Okay, so it is used to measure your molecular weight, of the peptides. Okay, to estimate the size, of a protein. Okay, to estimate the size of protein. It is also used in this. Okay, and it is also used in the mapping of the peptide. Mapping of the peptide, and to analyze the size and the number of peptides. To analyze size and number of peptides, size and number of polypeptide subunits. Okay, it is also used in the combination with the Western blotting. You know the snowdrop. Okay, snowdrop in the side of the southern blotting is for your DNA, and northern blotting for RNA. Western blotting is for the proteins. Okay, so it is also used in the combination with the proteins. Okay, with the Western blotting. Okay, to detect the presence of certain proteins. Okay, to detect the presence of certain proteins, it is also used. Okay, certain proteins. It is also used for the analysis of post transcription of the post translational modifications. Okay, and it is used in the diagnosis of, SDS page is used in the diagnosis of your multiple myeloma, okay, myeloma, or your nephrotic syndrome. Okay, then cirrhosis, liver cirrhosis, and chronic infections. Okay, these are used in the diagnosis of these things. Okay, so this completes your SDS page mechanism and your electrophoresis, which are often you can get a short note on this. You have sufficient content to write now. Okay, and these are the biochemical technologies, which I am not going into details. Chemical technology specifically. Okay, as a doctor you must have idea about all this, but you may not know the mechanisms and all, because this you won't have to do in any circumstances. You will not have to ask to do the electrophoresis. Okay, these are the works of your lab technicians and all. Okay, and that too in advanced laboratories, not in a periphery. Okay, so for doctors, just the knowledge is important of these things. Okay, and now we completed our most of the chapters. Okay, and now we'll go to our next chapter. That is your vitamins. Okay, let's go to vitamins. So about the vitamins, starting with the vitamin A. Let's first discuss the flow chart of the vitamin, then we'll make on adding the details. Okay, let's first discuss the flow chart of the vitamins. Okay, so here we have the chart of the vitamins containing all the vitamins and their deficiency, function, active form, other names, and some important notes, okay, which are important for the examinations. Okay, but I will discuss bit more, okay, about all the facts which are important and which are related with the vitamins. Okay, let's first discuss with the folic acid. Let's first start with the folic acid. So folic acid, it is your vitamin B9. Okay, folic acid is called as a vitamin B9. It is your vitamin B9. Okay, it is how it is formed. Okay, so folic acid, how it is formed, it is a pteridine. First your pteridine reacts with the PABA. And from pteroic acid, this forms your pteroic acid, and this pteroic acid plus pteroic acid minus the glutamate, if you do the glutamate, pteroic acid minus glutamate, this will form your folic acid. It is called so because it's the folic acid is present in the leaf of the green vegetable. Folium means a leaf. Okay, it is present in the leaf of your green vegetables or green leafy vegetables. Okay, so it is called as the folic acid. Okay, and folic acid, your how is the folic acid like converted? The folic acid is there. You have the dihydrofolate reductase, which will convert it into dihydrofolate, and dihydrofolate after, and again the same enzyme, it will convert it into a tetrahydrofolate. It is the active form. Of the folic acid. Okay, what are the biochemical functions? Okay, it is transferring one carbon unit, so it is helpful in one carbon metabolism. So the organic compounds which contain one transferable carbon are called as the one carbon compounds. Let's learn about the one carbon metabolism now. Okay, one carbon metabolism. Okay, so discussing our one carbon metabolism in little detail. So what are one carbon compounds? First, one carbon compounds are one transferable carbon. This carbon is transferable. Okay, transferable carbon. Okay, for example, you have a methyl. Okay, you have a methylene. Okay, you have a methynyl. Okay, you have a formyl, and your CH2OS, that is hydroxymethyl. Am I leaving something? Methyl, methylene, methynyl, formyl, and hydroxymethyl, and your formamino compounds also, CHNH. These are your formamino compounds. Okay, these are your one carbon metabolism. Okay, these are transferring like your serine, glycine, choline will transfer methylene, PHF. Okay, and so one carbon metabolism is another thing. Okay, one carbon metabolism has about 5 important reactions. Okay, so you can say that folic acid will have 5 important reactions. That is your conversion of serine to glycine. We have saw this reaction. That's why I'm discussing your vitamins at the very last, because you have read about all these reactions. Okay, then synthesis of thymidylate, the thymidylate synthesis, which is not in our syllabus. The thymidylate synthesis. Then catabolism, histidine. Okay, so histidine catabolism. Okay, and purine synthesis. Okay, folic acid is useful in the purine synthesis. We have talked about this. You can check out the purine metabolism, purine synthesis. Okay, and the synthesis of methionine also, and the methionine synthesis. So these are your actions of your folic acid. Okay, and your deficiency manifestations, okay, will be a megaloblastic anemia and neural tube defects like a spina bifida. Okay, spina bifida is there. Okay, and encephaly is there. Okay, due to the lack of folic acid in pregnancy. Okay, another deficiency manifestations can be there. You can add it. Okay, you can add your megaloblastic anemia is of course there, where there will be formation of macrocytic RBC, and this will cause your anemia. And birth defects. Okay, and due to this deficiency manifestations may be caused to increase demand of your folic acid or your drugs or any drug which is inhibiting your folic acid. Okay, and you can use your homocystinemia also. You can add your homocystinemia in a deficiency manifestations of folic acid. Okay, and talking about a required daily allowance, it is given? No, not given. No problem. Required daily allowance in adults, it is about 200 mg per day. So sources are vegetables, orange, papaya, beet. Okay, vegetables, green vegetables. You should use green vegetables. Okay, and what are the inhibitors of your folic acids? Okay, folic acid inhibitors. Let's talk about your folic acid inhibitors or the antagonists. So talking about your inhibitors, or which drugs are decreasing. Okay, so you have the sulfonamides. Okay, sulfonamides. sulfonamides are the PABA analogs. Okay, these are the analogs of your PABA. Okay, and are antibacterial drugs. Okay, which are competitive inhibition of your folic acid synthesis. Okay, which will inhibit your growth of bacteria. Okay, and another drug is a methotrexate drug. Is a methotrexate. Okay, this methotrexate drug will competitively inhibit your, this will what it will do competitive inhibition of your dihydrofolate reductase, DHFR. Okay, this DHFR is inhibited by the methotrexate drug. This is inhibited by the methotrexate drug, due to which your death of cancer cells will occur. Okay, how can you lab diagnose the megaloblastic anemia? You can do the peripheral smear or the histidine load test, or the serum folate level, or the RBC folate level. Okay, you can use the assay called as the FIGLU assay. Okay, FIGLU assay. What is FIGLU assay? It is your formamino glutamate assay. Okay, formamino glutamate assay. This is used. FIGLU assay is used for your determination of your folic acid, for your lab diagnosis of your megaloblastic anemia or your folic acid. Okay, so in pregnancy also you have to prescribe the patient 500 milligram per day of folic acid. I have talked about this in the nutrition chapter. Okay, so that's all about your folic acid. Okay, and biochemical importance we saw. One carbon metabolism is the most important. So they love asking this. They love asking the active form. Okay, DHFR, the drug inhibiting the DHFR is important. Okay, how it is formed is not much important. What are the synthesis of purine, pyrimidine, methionine, thymidine monophosphate? This is important. What are the deficiency manifestations? Okay, and what are the administration of high levels of folate can mask your vitamin B12 deficiency also. Okay, we'll discuss about a trap called as a folate trap later. Okay, we'll talk about this. After this, related with the folic acid, another vitamin is there, that is a vitamin B12. Vitamin B12 is called as a cobalamin. This is called as a cobalamin. Okay, it is present in bacteria, blue-green bacteria, that is called as a cyanocobalamin. Okay, it has a central cobalt present. And a corin ring. Okay, it has a corin ring, CORRIN, corin ring present. And active form is a adenosyl B12 and a methyl B12. Okay, it is a neurological vitamin, and it is also helping in the action of the extrinsic factor of Castle. It is also called as the extrinsic factor of Castle, and it is also helping in the action of Castle's intrinsic factor. Okay, Castle's intrinsic factor. So what about the digestion, absorption, transport? It is from the dietary B12. Okay, the B12 is released in the stomach. Okay, and the salivary gland also releases a B12. Okay, gastric parietal cells also secrete intrinsic factor, which helps in absorption of vitamin B12. Okay, and in ileum it binds to the receptor which is called as a cobalamin. We have a cubulin receptor, CUBULIN, cubulin receptors. Okay, cubulin receptors are there for binding. It is present in the ileum. Okay, and it is internalized in the ileal mucosal cells and transported in the portal circulation to the tissues. Okay, and how it is produced? It is produced by the microorganisms in the your stomach. Okay, in your intestine, sorry, not stomach. It is produced by microorganisms in the stomach. Okay, so we are talking about the vitamin B12. So vitamin B12 is produced in the microbes of your intestine, and also present in the meat. Okay, fish, this etc. Okay, and the RDA of vitamin B12 is about 1 μg. Okay, what are the main functions and the functions? What are the functions of vitamin B12 functions? So just remember what enzymes we have read, which we are using the vitamin B12. One enzyme which you can remember is acetyl CoA carboxylase activity. Acetyl CoA carboxylase, which was requiring ATP, biotin, and vitamin B12. Understood? Acetyl CoA carboxylase. Then another enzyme was your biochemical function, that is conversion of homocysteine to methionine conversion was there. Homocysteine to methionine conversion was occurring. Okay, here also we required vitamin B12 for a methionine synthase enzyme was there. The methyl THF is converted to THF. Okay, another function is the methyl manonyl CoA mutase. Okay, we are using methyl manonyl CoA mutase enzyme. Okay, okay, which was catalyzing the conversion of a methyl manonyl CoA MMCoA to succinyl CoA. For also vitamin B12 was required. Okay, these three reactions are important. We have read about all the three reactions. Okay, and talking about the deficiency manifestations. What are the deficiency manifestations here? Okay, so active form is a methylcobalamin. It is active form, and deoxyadenosylcobalamin cobalamin 2. Okay, adenosylcobalamin, methylcobalamin, these are active forms. Okay, this acts as a coenzyme for the reaction homocysteine to methionine and methyl manonyl CoA to succinyl CoA. Okay, deficiency manifestations are your nutritional deficiency, that is decreased intake will cause this. Okay, this will cause a malabsorption syndrome also. Okay, malabsorption syndrome. Understood? Okay, so malabsorption syndrome is there, and the main important thing is a pernicious anemia, which is due to autoantibody production. The autoantibodies are produced against the gastric parietal cells. Okay, but that's the deficiency of an intrinsic factor leading to and decreased vitamin B12. Okay, so vitamin B12 and B9 both will produce a megaloblastic anemia. okay, in which the cells are arrested in S phase and cell growth continuously without cell division, and macrocytic RBCs are present. So macrocytic RBCs, okay, and in this your vitamin B12 deficiency, you can see your dementia, okay, dementia and spinal degeneration, or you can write another things like a severe subacute combined degeneration of spinal cord, okay, which is this spinal degeneration, okay, due to defective myelination. It has a function in a myelination, okay. This methyl manonyl CoA, this competes with the manonyl CoA, okay, decreasing the fatty acid synthesis, and fatty acid synthesis decreases means the myelin synthesis is also decreased, okay. And this methyl manonyl CoA is incorporated into your sphingolipids, okay, causing this degeneration of your spinal cord, okay, degeneration of the spinal cord, due to which thus the patient suffers from megaloblastic anemia and neuropsychiatric symptoms. Megaloblastic anemia and your neuropsychiatric symptoms, these are important, okay. And what are the lab diagnosis? How can diagnose lab diagnosis? Okay, you can measure the serum cobalamin level, the serum methyl manonyl acid level, or the serum homocysteine level, and your peripheral blood smear, bone marrow aspiration, you can write, okay. And due to vitamin, the special point I'm discussing about the special point also only, okay. So vitamin B12, the methyl manolic acid, okay, megaloblastic anemia, you know about the megaloblastic anemia, okay, and you know what are the causes about the megaloblastic anemia, okay. And this pernicious anemia is treated with your IM or high dose of oral vitamin B12. The treatment is your high dose of oral vitamin B12, okay. After this, let's move on to your vitamin C, okay, vitamin C. Let's move on to your vitamin C. So vitamin C is called as your ascorbic acid. It's called as ascorbic acid, in which the L-ascorbic acid, levo form of ascorbic acid, is the main, okay, and it is also the active form is also your dehydroascorbate, which is excreted out. Like it is present in the dehydroascorbate form, but it converts into oxalic acid and it is excreted out, okay. What are the sources? What are the sources of your ascorbic acid? Okay, the sources, then you can get the ascorbic acid from Indian gooseberry, okay, your Indian gooseberry, okay, and the citrus fruits, all the citrus fruits like a lemon, okay, and your orange, guava and all, okay. And this is synthesized in the lower animals by uronic acid pathway, okay, and in humans and other primates cannot synthesize the vitamin B12, vitamin C, okay. So vitamin C, it cannot be synthesized, okay, cannot be synthesized due to lack of a enzyme which is called as your due to lack of your L-gluconolactone oxidase. Due to a lack of L-gluconolactone oxidase enzyme, the vitamin C cannot be synthesized in your humans, okay, humans. What are the biochemical functions? So it is an antioxidant. So vitamin A, vitamin C and vitamin E, these act as well antioxidants, okay. And biochemical functions, this causes a coenzyme for hydroxylation of reactions like your hydroxylation in a procollagen, that is your proline to hydroxyproline and lysine to hydroxylysine. These are caused by your vitamin C, okay. So I was telling that it is a coenzyme for hydroxylation reactions, for a hydroxylation of your proline and lysine, okay. So this is one of the function, the antioxidant function, that is it is a free radical scavenger, okay. And vitamin C also has many functions, okay. Let's deal about some the other functions of vitamin C, the functions of vitamin C, okay, functions of vitamin C. So it also prevents your cataract, also decreases your atherosclerotic risk, okay, atherosclerosis risk, okay, decreases your atherosclerosis risk, okay, and coronary artery disease risk, okay. And it is helpful in the oxidation reduction reactions like your cytochrome oxidase was there, okay. And the cytochrome oxidase, this requires your vitamin C also, okay. And it is also helpful in the your aromatic amino acid metabolism, okay. You can just see in your aromatic amino acid metabolism where I had written the tryptophan to 5-hydroxytryptophan has been converted in the presence of vitamin C, and para-hydroxyphenyl pyruvate to homogentisate, okay. This was also done by your vitamin C. Vitamin C is required, okay. It has also immunological functions, okay, like your free radical scavengers and all, okay. And conversion of your methemoglobin to your hemoglobin, this also requires your vitamin C, okay. And it is also helpful in a bone formation, okay, and your steroid hormone synthesis, understood. So these are your vitamin C, okay. Let's discuss your important short note, that is your scurvy, okay. About scurvy, what are the deficiency manifestations of scurvy? You will have swollen gums, okay. Swollen gums are the cardinal features, okay. Generally from vitamins your clinical cases are asked, so I am focusing on the clinicals, okay. The swollen gums, there will be bleeding, okay, and things like petechiae, ecchymosis and hematomas, hematomas. Why these are happening? These are your skin problems. Okay, bleeding tendency increases through fragile vessels because the vessels have your there is a deficiency in the collagen synthesis. Okay, due to the decreased hydroxylation of proline and hydroxyproline, due to which cross links aren't produced. Okay, so this phenomena are occurring. Okay, and you have delayed wound healing. Okay, delayed wound healing. Okay, delayed wound healing. And another thing is called as a scorbutic, your scorbutic rosary. Scorbutic rosary is a beaded appearance of chest. It is a beaded appearance of chest, beaded chest. Okay, due to the prominent costochondral junction. Due to prominent costochondral junction. Okay, junction. And your joint effusion, that is called as a pseudo paralysis. So pseudo paralysis also occurs in your scurvy. Pseudo paralysis. Okay, and your Barlow's syndrome also. Another thing which is called as a Barlow's syndrome. Okay, this is Barlow's syndrome is called as an infantile scurvy. This is called as an infantile. The scurvy in infants, that is called as an infantile scurvy. Okay, just a important thing, like 6 months to 1 year. If it occurs in infants, it is called as a Barlow's syndrome. Okay, and how can you treat the scurvy? You can treat the scurvy by giving high dose of vitamin C. Okay, vitamin C rich diet. Okay, and you can remember the cardinal features, like a pseudo paralysis is there. Okay, scorbutic rosary, that is a beaded appearance of chest due to prominent costochondral junction. This may be asked in a clinical case. Prominent costochondral junction seen in scurvy, and swollen gums are there, petechiae, ecchymosis, hematoma, and delay in wound healing. Okay, and your Barlow's syndrome is the infantile scurvy. Okay, so just remember these facts in your vitamin C. So that's all about vitamin C. And vitamin C, just you see, scurvy is there, and the source spongy gums, loose teeth, and poor wound healing. Okay, okay. These are about your scurvy. Let's go on to the next vitamin, that is a vitamin B6. So B6 is called as a pyridoxine. Okay, it is called as your pyridoxine or a pyridoxal phosphate is the active form. Okay, PLP, which is called as a PLP. Okay, we may have written PLP in various places. Okay, the PLP, it is the active form, and the required daily allowance RDA is about 2 milligram per day. It has three forms, that is your pyridoxine, your pyridoxamine, and a pyridoxal. Okay, your pyridoxine, your pyridoxamine, and your pyridoxal. These are the three forms of a vitamin B6. Okay, and pyridoxine is the alcohol, pyridoxal is the aldehyde form, and pyridoxamide is the amine form. Okay, how can you get vitamin B6? Vitamin B6 is also called the pyridoxal or pyridoxine. Okay, and PLP is the active form. Okay, how can you get this vitamin? You can get this vitamin from your fish, meat, milk. Okay, and from your rice and all. Okay, you can get pyridoxine. What is the biochemical function? It is helping in your transamination reactions. Okay, that is a coenzyme for enzymes. Okay, in amino acid metabolism, it is helping in the transamination reaction. Okay, the activity of your SGOT and SGPT. This requires a PLP. Okay, and your decarboxylation reactions also. Decarboxylation. What are the decarboxylation reaction? Like your let me tell you about some reactions. Okay, we'll be discussing about vitamin B6. Let's discuss a reaction. This is 5-hydroxytryptophan in the presence of your tryptophan pile is converted in serotonin. Okay, serotonin, and this requires your PLP. Okay, and the conversion of dopa to your dopamine. We discussed about these reactions. This also requires PLP. Okay, and another reaction, that is your glutamate to GABA. Okay, all these reactions are in protein metabolism. You can check out. Okay, this also requires a PLP. Okay, so the carboxylation reaction, and you can require PLP in the transsulfuration reaction, that is your homocysteine and serine, which has been converted into a cysteine plus homoserine. Okay, this is a transsulfuration. This also requires your PLP, and your heme synthesis, your succinyl CA. First step is your succinyl CoA plus glycine, which was being converted into your gamma amino levulinic acid. Okay, and in the presence of your ALA synthase, this is also requiring PLP. Okay, this is also requiring a PLP. Okay, and this glycogen to glucose conversion of glycogen phosphorylase, your glycogen in which it was being converted in many steps into your glucose. Okay, glucose by the glycogen phosphorylase. This also requires a PLP. Okay, and another in the kynureninase pathway you can check out. Okay, when the 3-hydroxykynurenin was being converted into your kynureninase by kynureninase. Okay, the enzyme is a kynureninase into 3-hydroxyantranilate. Okay, there also we are requiring PLP. Okay, and if this PLP is deficient, we told this. Okay, we have discussed this. If the PLP is deficient here, okay, if the PLP is deficient, that is a vitamin B6 deficiency. Okay, this 3-hydroxykynurenin will be converted into your xanthurenate. Okay, and the xanthurenate will be excreted in the urine. Okay, so xanthurenate excretion in urine is a vitamin B6 disorder. Okay, So what are the deficiency manifestations of vitamin B6? What are the deficiency manifestations of vitamin B6? Is there given? Okay, it is written rare. Of course, it is rare. Okay, you can have some neurological manifestations like neuropathy is there. Peripheral neuropathy is there. Okay, neuropathy and convulsions. Okay, you can have pellagra-like symptoms also. Pellagra-like symptoms also. Okay, and excretion of xanthocyanate in the urine, and your another symptom which is a sideroblastic anemia. Sideroblastic anemia. Okay, these are your deficiency manifestations. Okay, and your signs and symptoms are your glossitis and your neuropathy. Glossitis is a pellagra-like symptom, and your neuropathy is occurring. Okay, and what are the drugs that causing your B6 deficiency? So there is an important thing to discuss, that is your drugs causing your B6 deficiency. Okay, so you have your drugs isoniazid, okay, which is anti-tubercular drug, INH, then penicillamine, then your cycloserine, and your L-dopa also. These are your drugs which are causing your B6 deficiency. And B6 is used in the treatment of homocystinemia, anemia, and retinal atrophy. Okay, and also in the treatment of schizophrenia or carpal tunnel syndrome also. Okay, understood. So this is all about your B6. Let's talk about your thymine also. Okay, let's talk about your thymine. Okay, what is thymine? The name is a thymine, other name is a thymine. Okay, and the active form is a thymine pyrophosphate, that is called as a TPP. It is the vitamin B1. So vitamin B1 is a thymine. Let's talk about it. So it is also called as an anti-beriberi vitamin, because vitamin B1 deficiency causes anti-beriberi. So it is also called as anti-beriberi or your anti-neuritic vitamins, because beriberi causes neurological symptoms and Wernicke-Korsakoff syndrome. We'll see about that. Okay, so this is your vitamin B1. Okay, what are the functions of vitamin B1? So vitamin B1 is generally, it is the coenzymes. It acts as a coenzyme, okay, of the reactions like pyruvate to acetyl CoA. Okay, it is TPP is there. Then alpha ketoglutarate to the succinyl CoA, TPP is there. Branched chain amino acid oxidation, where the branched chain will be converted into your acyl CoA. Okay, this is also your thing, that is your uses of your vitamin B1. Okay, and this reaction will ribose 5-phosphate plus xylulose 5-phosphate will be converted into pseudohaptalase and glyceraldehyde 3-phosphate. You remember the reaction. Okay, that is of your HMP shunt. Okay, it is used in the HMP shunt pathway. Okay, it also requires a TPP. Okay, this all requires a TPP. Okay, what are the sources? You can get this TPP from rice. Okay, this is present in the outer aileron layer of the rice, of the rice. Okay, and whole wheat, rice and wheat, and you can get it from your the yeast. Okay, all. Okay, this all. And what is the RDA? So RDA of the thymine is about 0.5 milligram. Okay, per 1000 calorie intake. Okay, so it is about 1 to 1.5 milligram per day. So you can write 1 to 1.5 milligram per day for your RDA purposes. Okay, so this is a vitamin B1 RDA. Understood? And what are the deficiency manifestations? So it is a beriberi. Causes of a thymine deficiency may be due to nutritional deficiency due to intake of polished rice. Okay, we are intaking a polished rice, but and removing the outer aileron layer. But this outer aileron layer is mainly rich in your vitamin. It is rich in your vitamin B1. Okay, it is rich in your vitamin B1. So removal of this eating of polished rice will cause this. Okay, and another thing is that a thiaminase enzyme is present. Okay, in tea and coffee, a enzyme which is called as a thiaminase enzyme is present, which will decrease the level of your vitamin B1, okay, and alcoholics also have decreased vitamin B1. Okay, and which the alcohol, the ethanol, which causes decreased absorption and conversion of your thymine to TPP. Okay, understood? And your hyperemesis gravidarum, which causes deficiency in the infant, and diuretic therapy also, which causes the loss of urine. Okay, about beriberi we have to discuss a little bit more about beriberi, because beriberi often comes as a short note. Okay, so beriberi, as of scurvy we discussed. Now we are discussing the short note beriberi. So beriberi we have three types. We have dry beriberi. Okay, we have dry beriberi, we have wet beriberi. Okay, and we have infantile beriberi also. Okay, in this dry beriberi there is mainly weakness. The weakness is present, and there is peripheral neuropathy. Okay, and this is and the wet beriberi. Okay, wet beriberi it will show your cardiomyopathy and dilation of heart. So CVS problems will be there, like your cardiomyopathy. Okay, cardiomyopathy and dilution and your dilatation of heart will be there, and edema will be also there in the wet beriberi. so it is also called as wet beriberi. Okay, and in infantile beriberi it also involves CBS. Okay, and it is if there is acute fulminant form, okay, it is also called as a Shoshan beriberi. Shoshan beriberi, this is also a type of beriberi. Okay, Shoshan beriberi. Okay, and talking about Wernicke-Korsakoff syndrome, you know about Wernicke-Korsakoff syndrome, the Wernicke-Korsakoff syndrome or Wernicke-Korsakoff psychosis, WKP. Okay, it is a chronic alcoholism due to decreased absorption of thiamine causes it. Decreased absorption of thiamine will cause this. Okay, and this due to decreased absorption there will be decreased conversion to TPP. Okay, so neurological manifestations will be there due to decrease in ATP. Your PDH complex is now we have talked about Wernicke-Korsakoff in PDH complex. Yes. So the PDH complex is not working well. Okay, so there is decreased ATP synthesis. Okay, and you know the symptoms, your BOA MP. Okay, I have told you in mnemonic, it is global confusion, ophthalmoplegia, ataxia, memory loss and psychosis. Okay, memory loss and psychosis is called Korsakoff psychosis, and this global confusion, ophthalmoplegia and ataxia belong to Wernicke's encephalopathy. Okay, so it is a combination of two syndromes. What is the treatment for Wernicke-Korsakoff syndrome? You can treat this syndrome by giving it 100 mg per day. Okay, 100 milligram, not 100 per day, 100 milligram per day of your vitamin B1 for 7 days. Okay, and plus your 10 milligram per day maintenance doses. Okay, 10 milligram per day doses afterwards. Okay, this is prescribed for your Wernicke-Korsakoff syndrome and your beriberi. Okay, so this is all about your vitamin B1. We talked a lot about vitamin B1 now. Okay, this is most common in alcoholics. I talked about Wernicke-Korsakoff syndrome. Okay, what are the features? That is tachycardia, vomiting, convulsions, apathy, loss of memory and eye movements. Okay, and this is the cardinal features of your vitamin B1 deficiency. Okay, now let's talk about your niacin. Okay, let's talk about your niacin. So niacin is also called as a nicotinic acid. It is a vitamin B3. Okay, it is a vitamin B3. It is also called as a nicotinic acid or nicotinamide. Nicotinic acid or nicotinamide. There are two forms you can say. Okay, of your niacin. Okay, the active forms are NAD plus and NADP plus. This active or cofactor form are your NAD plus and NADP plus. Okay, and this niacin is synthesized from a tryptophan. Okay, this niacin is synthesized from a tryptophan. So tryptophan, if there is 60 milligram of tryptophan, will produce about 1 milligram of your niacin. 60 milligram of tryptophan will produce 1 milligram of your niacin. Okay, niacin, understood. And what are the biochemical functions? So it is helpful in the electron transport. You know the functions of your NAD plus and NADP plus. These are catalyzing your oxidation reduction reaction, that is a redox reaction. Okay, in your ETC. Okay, like your NAD plus is utilized in your glyceraldehyde 3-phosphate, your lactate dehydrogenase, utilized by pyruvate dehydrogenase, your isocitrate dehydrogenase, all the dehydrogenases they require NAD plus. This is required by all the dehydrogenases. Okay, and this NADP plus, this catalyzes the reactions like your glucose 6-phosphate dehydrogenase, your phosphogluconate dehydrogenase and your malic enzyme. Okay, this is utilized. Okay, and what are the sources of vitamin B3? So vitamin B3 again your fish, cereals, kidneys and liver. Okay, and required daily allowance is about 20 milligram per day is required. 20 milligram per day of niacin is required as your RDA. Okay, and deficiency manifestations is your pellagra. And pellagra is again a short note. Okay, pellagra contains 3 things. Okay, also you can say 4 things. Okay, you can add death too. Okay, so you have dermatitis, diarrhea, dementia and death. So these are called as 4 Ds. 4 Ds of your pellagra. 4 Ds of pellagra. Okay, 4 Ds of pellagra, or your 4D triad of pellagra, or your tetrad of pellagra. Okay, you can say that. Okay, so it has 4 Ds pellagra. So diarrhea will occur, dementia will occur, which will cause the loss of thinking and remembering and reasoning. Okay, and dermatitis, around the sun exposed areas there will be Casal's necklace appearance. Okay, and on hands and feet there is stocking appearance. Okay, let me discuss some more about a pellagra. Okay, so discussing about pellagra. Okay, so what I told that there is 4D features, that is your diarrhea. Okay, diarrhea, and that is your dementia. Okay, dermatitis. Okay, dermatitis. This dermatitis may occur. A special thing that dermatitis is your Casal's necklace. Okay, is your Casal's necklace appearance. CASAL, Casal's necklace appearance around the neck. This is a symptom of your vitamin B3 deficiency. Okay, it looks like this. Okay, so this is your Casal's necklace appearance. This looks like this. Okay, so you can get an image based question also, and you can be asked about your pellagra and all. Okay, and if this pellagra is not treated, it will lead to another D, which is your death. Okay, and what are the causes of this pellagra or niacin deficiency? Okay, so decreased intake is one of the causes. Okay, and your PLP deficiency and all. Okay, another is your Hartnupp's disease. Okay, what is your Hartnupp's disease? This is a decreased decreased absorption of a tryptophan, okay, decreased absorption of your tryptophan, due to which your 60 milligram tryptophan was giving about 1 milligram of niacin, okay, 1 milligram of vitamin B3, okay, vitamin B3, okay. So decreased absorption tryptophan, your tryptophan will decrease your vitamin B3, okay. This is called as your Hartnupp's disease, okay. And your again another syndrome is there, that is a carcinoid syndrome. We had told the names in the tryptophan metabolism, as we are seeing, okay. The carcinoid syndrome, in carcinoid syndrome, the tryptophan, about your about your 60% of your tryptophan is converted to serotonin instead of being giving your vitamin B12, okay. Serotonin, it is deviated towards the serotonin pathway, due to which there is decrease in your tryptophan, and this tryptophan will lead to the decrease in the vitamin B3, okay, and cause your pellagra and all symptoms, okay, all symptoms, okay. And another thing is that, another concept is that, is Jover and maize, okay, Jover and maize, okay. So this some states have this as a staple food, okay. This Jover and maize, these have high leucine content, which will inhibit your QPRT enzyme, okay, which will inhibit your QPRT. QPRT is a quinolinate phosphoribosyltransferase, quinolinate phosphoribosyltransferase enzyme, QPRT enzyme, and this QPRT will not form your tryptophan. For your tryptophan, your NAD was being formed, so it will not form your NAD. And not forming NAD will cause various disorders. Not forming NAD will the glycerolate 3-phosphate, your lactate dehydrogenase, pyruvate dehydrogenase, alpha ketoglutarate, this all enzymes won't be able to work, okay. So this is also like important as per your vitamin B3 is concerned, okay. So you should know this statements, okay. And this is your Kadal's necklace. You can get an image based question on this, okay. So this was about your vitamin B3, okay. Now let's learn about vitamin B2. Vitamin B2, that is your riboflavin, okay. Vitamin B2, that is called as a riboflavin, okay. Riboflavin, okay, it is like the color of riboflavin is yellow, like, okay. It is also called as a Warburg. Yellow, okay, also called the Warburg yellow, okay. The chemistry about the alloxylin and ribitol is there, okay. This is not important, okay. What are the active forms? Active form is your FMN and FAD. Are the active form, and FMN catalyzes the reaction like L amino acid oxidase, okay, and then monoamine oxidase, and it is reacting in the complex 1 of ETC, okay. And FAD is FMN is in your complex 1, and FAD is in your complex 2 of electron transport chain, okay. And FAD is catalyzing various reactions like your xanthine oxidase, okay, your glutathione reductase, okay, then your PDH complex, in your alpha ketoglutarate dehydrogenase complex, okay, then your branch chain keto acid dehydrogenase branch chain keto acid dehydrogenase, it is acting, okay. These all reactions, okay. And your FMN is catalyzing reactions like your L amino acid oxidase, okay, amino acid oxidase enzyme, okay. Then monoamine oxidase, we haven't read this reaction, these are not much important, okay. Monoamine oxidase and all, okay. And it is also used in the complex 1. So these are your functions of your vitamin B2, okay. And deficiency manifestations will cause, it is rare, but it will cause a deficiency manifestation which is called as your araboflavinosis. This araboflavinosis will cause various like features, like it has a glossitis, okay. Glossitis, that is a magenta colored tongue, okay. Magenta tongue, your glossitis, and your cheilosis, okay. Swelling and fistering of lips, okay. And your angular stomatitis is your angle of mouth, okay. The mouth becomes your like stomata like, or your as your stomachoids had that type of mouth, okay. And seborrheic dermatitis, the dermatitis type is your seborrheic dermatitis, okay. And angular stomatitis, and corneal vascularization also, okay. These are your deficiency manifestations of the important disorder, that is araboflavinosis, but this is rare, okay. Understood? Now let's learn about another vitamin, that is your biotin. Let's learn about biotin, okay. So biotin is your vitamin B7, okay. Biotin is a vitamin B7, and it is also called as a vitamin, vitamin H, or it is also called as a coenzyme, R, okay. So it is a vitamin as well as a coenzyme, okay. And this biotin has an active form, biotin, your carboxybiotin, enzyme bound biotin, and a carboxybiotin, this is the active form, carboxybiotin. And from where you get biotin? So biotin you can get from egg yolk. Is an important source, okay. Meat, fish, milk and all, okay. Talking about the RDA, biotin has the RDA of 30 to 100 mg per day. Okay, you should take 30 to 100 mg per day of biotin every day. Okay, this is the required daily allowance of your biotin. Okay, and the biochemical function is in your carboxylation reaction. You know about the carboxylation reaction. Okay, all the carboxylases require biotin. Okay, all the carboxylases require biotin, like your pyruvate carboxylase, which is converting a pyruvate to oxaloacetate. We know about this reaction. Okay, then acetyl acid carboxylase, acetyl CoA carboxylase, okay, which are catalyzing acetyl CO to mannoyl CoA in the fatty acid synthesis, remember? Yes. And we have the propionyl CoA carboxylase, propionyl CO to methyl mannoyl CoA. This was also in the gluconeogenesis. Okay, this is your another reaction, that is your propionyl CoA carboxylase. Okay, carboxylase. Okay, and we have biotin independent carboxylation also, biotin independent carboxylation reactions, which are often asked in the viva. So I am writing some B7. Okay, we are talking about B7, and here we have to discuss a biotin. Okay, which reactions? The carboxylation reactions are biotin independent carboxylation reactions. We are talking about these reactions. Okay, so these reactions are mainly your carbomal phosphate synthesis is CPS1. The CPS1 and 2, these enzymes act by biotin independently. Okay, and then your AR carboxylase, we talked about this AR carboxylase in the purine nucleotide synthesis. This also acts independently. Then your malic enzyme is one of the enzymes which carboxylation occurs in absence of a B7, and your gamma carboxylation, which will see in your vitamin K. So these four are your biotin dependent carboxylation reaction. These are often asked in a viva. Okay, so you must have to tell it. Okay, then afterwards let's discuss another things, like your where in a biotin. Okay, and deficiency manifestation is very rare. Okay, and maybe caused due to prolonged antibiotic therapy. Neurological manifestations, you can see your neurological manifestations. Okay, like your hallucination is there, depression is there, and all. And you can also see your skin rashes and all. Okay, and the antidote is your avidin. Okay, avidin is your antidote. Avidin is the antidote. Avidin has more affinity towards biotin, okay, than the antigen antibody complex. Okay, and the streptavidin is the form of avidin. Okay, this antidote is avidin is used in ELISA, DNA labeling, and DNA proof technologies. Okay, so that's all about consumption of large amount of raw egg whites that contains a protein avidin. It is written. Okay, this is present in your raw egg white, that is the albumin part. Okay, avidin which has more affinity towards your protein. Okay, your biotin, and can induce a biotin deficiency. Okay, it can induce a biotin deficiency. Understood? And this biotin, this avidin can, if there is more consumption, okay, it can induce a biotin deficiency. Okay, understood? So this is about avidin. Let's discuss about vitamin B5. B5, that is a pantothenic acid. Okay, pantothenic acid. So this pantothenic acid is called vitamin B5. Okay, we don't have to discuss in more. Okay, the active form is the coenzyme A. This is the active form. Okay, active form, and it acts as an acyl carrier. Okay, it helps in the acyl carrier. So vitamin B5, the pantothenic acid, acts as an acyl carrier. So it has a function, and the deficiency symptom is very rare. So we don't have to discuss much. Okay, after this water soluble vitamins, we are into the fat soluble vitamins, which are your A, D, E, and K. These all vitamins are important. Okay, let's discuss the vitamin A. We have to discuss the vitamin A a little bit detail. So I'm going to my notes. Okay, so vitamin A, it has vitamins are the similar properties as vitamin A, which have the similar properties as vitamin A. Okay, they are called vitamins. The vitamins of vitamin A are your retinol, your retinoic acid, and beta carotene. Okay, this beta carotene in the presence of dioxygenase changes into retinol. This retinol in the presence of retinol reductase changes into retinol. Okay, and this retinol are esterified again to form retinal esters. Okay, retinal esters which are incorporated into chylomicrons and are transported to the liver. Okay, and these retinal esters are also converted to retinol by the intestinal bus border hydrolases. Okay, and this retinol again is esterified to retinal esters. Okay, incorporated into chylomicrons and go into liver. And in liver, if it is to be released in the bloodstream, okay, it binds to the retinol binding protein. Okay, and it is released into the bloodstream. Okay, and the tissue, the retinol is converted into retinoic acid. okay, which will stimulate the receptors and cause protein synthesis. Okay, so the vitamin A has protein synthesis action. Then your Wall's visual cycle is there, very important, Wall's visual cycle. Okay, here the rhodopsin is there, rhodopsin, which is the combination of 11 cis retinol and your opsin. Okay, in the presence of this light, rhodopsin converts into your opsin and your all trans retinol. Okay, it isomerizes into all trans retinol and opsin is given out. All trans retinol, again in the presence of retinol dehydrogenase, converts into your all trans retinol. Okay, retinol dehydrogenase converts the retinol to the retinol. All trans retinol again in the presence of isomerase is converted to your 11 cis retinol. Okay, and 11 cis retinol in the presence of retinol dehydrogenase is converted into your 11 cis retinol, and this again combines into your opsin in darkness to form a rhodopsin. Okay, so it is the Wall's visual cycle. Okay, so it is the Wall's visual cycle. Then what about the rhodopsin? This rhodopsin in the presence of light, okay, changes into pre-lumirhodopsin to lumirhodopsin to metarhodopsin 1, okay, and this to the metarhodopsin 2. This metarhodopsin 2 induces a transduction will give positive feedback to your phosphodiesterase, which will convert your CGMP to your 5-GMP, which will decrease the CGMP levels, which will close the sodium channels over the rods, okay, and it will cause a hyperpolarization and stimulated visual cortex. Okay, stimulated visual cortex for your visual stimuli. Okay, so this pathway is given in detail in your Guyton textbook, okay, in the physiology. Okay, and importance of vitamin A is in protein synthesis, is in cholesterol synthesis, decreases the keratinization of skin and mucous membrane, it is antioxidant and maintains the germinal epithelium. Okay, what are the sources? The cod liver or shark liver oil, carrots, papaya, your spinach and mango, these are rich in your vitamin A. Okay, RDA is determined in the retinol equivalents. So one retinol equivalent is equal to 1 μg of retinol. One retinol equivalent, 1 μg of retinol. Males require your 1000 retinol equivalent and females 800 retinol equivalent. Deficiency manifestations, we will have visual impairments majorly. Okay, these are of about 8 types as per your WHO. Okay, first is the XN, that is night blindness, is the first symptom of vitamin A deficiency. Then X1A is a congenital xerosis. 1B is a Bitot spot. Bitot spot is a triangular opaque white conjunctival plaque, okay, which is formed at this end of your conjunctiva. It is an opaque white plaque. Okay, you can get the image based question based on this. You can see the Bitot spots in the internet. Then X2 is a corneal xerosis. X3 is the corneal ulcer, the ulceration in the cornea. Okay, and less than 1/3 of cornea, but as it progresses, it will cause X3B, corneal ulceration plus xerotomelicia, which will involve your more than 1/3 of cornea. XS is the corneal scarring, and XF is the xerophthalmic fundus. Okay. Then after, there will be sterility due to vitamin A deficiency, and there will be increased keratinization because vitamin A had that property of decreasing the keratinization, and you can have growth impairments and defects in the protein synthesis also due to deficiency of vitamin A. Treatment is a 2 lakh IU of vitamin A. This can be given for the treatment of the vitamin A manifestations. Okay. Talking about vitamin D. Okay, vitamin D has 2 forms. Okay, your cholecalciferol and ergocalciferol. These are the 2 forms of vitamin D. Vitamin D is chemically called as calcitriol. Okay, it is called as calcitriol, and 2 forms are cholecalciferol and your ergocalciferol, and the active form is your 125 dihydroxycholecalciferol. This is the active form of your vitamin D. Okay, how it is metabolized, you can just see. Okay, for vitamin D, I'm just talking about a metabolism of vitamin D. We're talking about the vitamin D. Okay, and vitamin D, we have yours in the epidermis of skin. We have your dihydrocholesterol, which is converted by UVB rays of sunlight to cholecalciferol, and this is by the enzyme 25 hydroxylase, okay, enzyme which is synthesized from a liver. This is converted into a 25 hydroxy cholecalciferol, okay, and this is the storage and transport form. Okay, this is the storage form or your transport form. Okay, this is storage and the transport form. And this 25 hydroxy cholecalciferol, this in the presence of an enzyme 1 alpha hydroxylase, okay, which is released from kidneys, to your 125 dihydroxy cholecalciferol. That is the active form, that is also called as your calcitriol of vitamin D. Okay, what are the sources? Your fish meat, egg yolk. Okay, what is the RDA of vitamin D? I'm writing RNA many times, okay, because I love molecular biology. Okay, what is the RDA? In children, it is for adults it is about 200 international units, and for children it is about 400 international units. Okay, in pregnancy and lactation also, in children and pregnancy and lactation also, you require 400. Okay, and in old age also, more than 60 years, you require 600 international units. Okay, vitamin D is also called as a hormone. It is also called as a hormone. So the question is asked, which vitamin is called as a hormone? So vitamin D is called as a hormone. Why? Because synthesis is occurring in the skin. Okay, it has target tissues. it has target tissues. In target tissues, like your bone is there, okay, bone, your intestine is there, bone, intestine, and your kidneys, okay, bone, intestine, and your kidneys. So vitamin D in the intestine, it will cause an intestinal absorption of your calcium and phosphate, and the kidney, in the kidney it will catalyze the reabsorption of calcium and phosphate, acting on the renal tubular cells, and in the bone it will help in the formation of bone, the formation of your bone, and the mineralization of bone, okay. So calcium and vitamin D, these are just closely related, okay. And vitamin D has also among the this target tissues, it has calcium regulation, okay. You can write about the calcium regulation on your if you get a short note on your vitamin D, or if you get a long question, you can write in detail of all the pathways, okay, which can regulate your regulation of your calcium, okay. And vitamin D is also helpful in the treatment of psoriasis, and it has also immunomodulatory functions, okay, and prevention of your coronary artery disease prevention, okay, it is also helpful, okay. And deficiency manifestations is your rickets is a short note, which is important short note. And rickets occurs in your children, and the adult form of rickets, what is occurred adult in your adult, osteomalacia, this is the adult rickets. Osteomalacia is the adult rickets. So in rickets, what will be the symptoms? Okay, first you will have your craniotabes, okay. Craniotabes are the craniotables, not tables, tabes, okay. Craniotabes is the soft and pliable skull bones, okay. You have the skull bones are soft, in this case soft, okay. And then have delayed closure of fontanelles, so you will have fontanelles present in the child, okay. Fontanelle is present, okay. Then you will see frontal bossing will be there, that is your prominent forehead, frontal bossing will be there, and your widening of your wrist, bone disorders like your bow legs will be there, your genu valgum and your genu varum, okay. Genu valgum, that is your genu valgum will be there, okay. And another special thing is your rachistic rosary, okay. Rachistic rosary, that is a prominent costochondral junction, that is your prominent costochondral junction, that is called as a rachistic rosary. And another thing is a Harrison's sulcus, Harrison's sulcus, okay. Harrison's is a textbook in your final prof, that is of medicine, okay. Harrison's sulcus is the transverse groove where that's the diaphragm is inserting, that is the groove present in the abdominal cavity, this is called as a Harrison's sulcus, okay. Talking about osteomalacia, what are the symptoms? That the bones will be fragile, there will be fractures and all, there are common fractures. Osteoporosis will set in, that is the porosity of the bone increases, okay. And there is an increase in ALP activity, so you can detect your osteomalacia from increased ALP activity, okay. And if there is the deficient in absorption of your calcium and phosphate from renal tubules, okay, so there is the renal rickets, okay. Renal rickets is also present, okay. Renal rickets. And there is a vitamin D resistant rickets, vitamin D resistant rickets, okay. Vitamin D resistant ricket, that is seen in your Fanconi's anemia, Fanconi's anemia, remember we talked about in the DNA repair mechanisms, okay. Understood? So serum vitamin B levels, if there is less than 30 nanogram per ml, it is insufficient at all, okay. And if there is less than 20, it is deficient, okay. And severe deficiency is less than 10, but if it is more than 150 nanogram per ml, okay, then it is your toxic, okay. It is toxic for the body, okay. And how can you treat it? You can treat the deficiency symptoms like rickets by increasing the exposure to sunlight and giving calcium supplementations, okay. So this is all about your vitamin D. We talked about the vitamin D in details and covered all the points, okay. So vitamin D, agricultural calcium uptake, rickets in children, osteomalacia in adults, soft pliable bones, okay. There will be toxicity, if there is increase in your 150 nanogram per ml. And vitamin D is not a true vitamin because it can be synthesized in the skin, okay. And application of your sunscreen lotions okay, decreases the synthesis of vitamin D, okay. Let's talk about your vitamin K, okay. Let's talk about your vitamin K. K stands for coagulum, okay. Coagulum in German term is a coagulum, okay. And vitamin K is it has three forms, okay. K1, your K2 and your K3, the three forms. Your menadione, your menaquinone, okay. Your menadione is there, your menaquinone is there, and your phyloquinone is there. So phyloquinone is called as a K1, vitamin K1. Vitamin K2 is the menaquinone, and vitamin K3, this is a menadione, okay. Menadione is the vitamin K3. So vitamin K1, K2 and K3 you have, okay. And the sources are like that, okay. What are the biochemical functions? Okay, what are the biochemical functions? Okay, gamma carboxylation of glutamate residues in clotting, okay. They do gamma carboxylation of your glutamate residue, that is they convert your glutamate into gamma carboxyglutamate, which carries an extra negative charge, and this negatively charged clotting factor attached with positively charged calcium ions. Okay, is attached to the positively charged calcium ions. Okay, and this vitamin K also helps in the post transcriptional modifications. These are post translational modifications of clotting factor. Okay, post translational modifications of clotting factor 2, 7. Let me write in numericals: 2, 7, 9 and 10. Okay, so post translation modification clotting factors 2, 7, 9 and 10. Okay, 2, 7, 9 and 10. Understood? So a cycle is there which is called a gamma carboxylation. How it happens? Okay, so here what happens, that your hydroquinone, that is a reduced hydroquinone is a reduced vitamin K. Okay, this is converted by carboxylase to epoxide. Okay, and oxidized vitamin K, okay, which in the presence of a reductase, okay, converts into quinone, and this quinone again in the presence of a reductase converts into hydroquinone. Understood? This is a cycle. Okay, and here in this step, a reduced lipoic acid is converted into your oxidized lipoic acid. Okay, oxidized lipoic acid, and this reductase is called as a vitamin K epoxide reductase. Okay, if this enzyme is deficient, if this enzyme is blocked by two drugs, that is a warfarin and your dicumarol, these are blocking your vitamin K epoxide reductase. Okay, understood? So vitamin K is also role in bone formation and activation of protein C and S, that is your anticoagulant activity. Okay, talking about what is the RDA, this facts are I'm talking, that is about your vitamin K. Okay, so let me just take it separately. Okay, and give the heading vitamin K. And vitamin K, this is also like RDA, the RDA of vitamin K. 70 to 140 mg per day. Sources are the green leafy vegetables, intestinal bacteria and all. Okay, and the deficiency manifestation, there will be hemorrhagic manifestations like ecchymotic plaques. Okay, there will be ecchymotic patches all over the body. There will be red spots and rashes. Okay, and you can just your prophylaxis is 1 mg menadione. Okay, 1 mg menadione is given IM at birth. Okay, I am given IM at birth, so as to prevent your vitamin K deficiency in your future. Okay, how can you lab diagnose the deficiency? Lab diagnosis is to measure your prothrombin time and toxicity of vitamin K. This vitamin K toxicity will lead to hemolysis and your connectors. Understood? So these are all about your vitamin K. We discussed a lot about your vitamin K. Okay, and your vitamin K is thus finished. Okay, let's discuss in one size. Okay, we will discuss in a good amount. Okay, about the vitamin E. Vitamin E is also a fat soluble vitamin, and it is also called as anti-sterility vitamin. Anti-sterility vitamin, and there is no disease. Rare is written. So it is also called as a vitamin in search of disease. It is the vitamin in search of disease. Okay, which includes the tocopherols and tocotrienols. Okay, the RDA about vitamin E, the RDA of vitamin E. The RDA of vitamin E is about 10 mg per day in your males. Okay, in your males, and about 8 mg per day in case of your females. Okay, this is the RDA of vitamin E. Okay, and RDA of vitamin, the tocopherols, the alpha tocopherol, this is the most active form. Okay, this alpha tocopherol is the most active form. Okay, and many tocopherol derivatives are there, like your many tocotrienols are there. Okay, and it is mainly an antioxidant. It is a membrane antioxidant. Okay, it prevents a lipid peroxidation reaction. We also saw in your remember your antioxidant chapter that we discussed from the today. Okay, on that, in that also. Okay, it also prevents the hemolysis of RBCs. Okay, and maintains the integrity. It also maintains the germinal epithelium. Okay, maintains the germinal epithelium that is present in your gonads. Okay, and it prevents your myocardial infarction and prevents your coronary artery diseases. Okay, and this acts synergistically with selenium. Okay, this vitamin E and selenium, these are just acting synergistically. Okay, and this scavenges the free radicals. This is also helpful in the immune responses. Okay, vitamin E also does immune responses. Okay, immune responses. immune responses, and prevent oxidation of your vitamin A, and along with the vitamin C and A, it prevents your cataract formation also, okay, and activates your allosynthetase and dehydratase, okay, it activates your allosynthetase, dehydratase, nucleic acid synthesis, okay, and proper storage of creatine in muscles. This is also the function of your that vitamin E. Okay, if there is deficiency, it will lead to your red blood cell fragility, which will lead to hemolytic anemia. The mild hemolytic anemia will be caused, and some neurological disorders will be there. Like your ataxia. Okay, ataxia is there, and retinopathy, these all are there. Okay, understood. So this is all about your vitamin E, and thus we have a happy ending. Okay, and the video is about 13 hours and 14 minutes. Okay, it has been a long video in which we revised all of the biochemistry, revised each and everything that is given in your textbooks, and is important for your prof as well as your PG entrances. Okay, we talked about the whole biochemistry. We haven't left any of the parts. Okay, the parts I have left are your hormone action, that is inner physiology, so we need not discuss it here. Okay, so that was today. Okay, hello, so I hope you are alive. Okay, so it has been a very long lecture of about 13 hours. Okay, 13 hours and 15 minutes. Okay, and I hope you enjoyed revising the whole biochemistry. If you're watching it in one stretch also, it is gonna be a very good revision. Okay, as you will not retain all the minute details, but you have retained all the major facts which are in your biochemistry as far as the biochemistry is concerned. Okay, so that's all from my side. Okay, I hope you revised all your things, all your stuffs. Okay, and you enjoyed the lecture a lot. Okay, so that's all from my side. If you like the lecture, do like, share and subscribe. I did not tell it. Okay, and that's all from my side. Let's meet again in any other lecture. Okay, so that's all. Thank you and good night.